Insectivorous plants. Chapter Roman 1, Drosera rotundifolia, or the common sundew. Number of insects captured. Description of the leaves and their appendages or tentacles. Preliminary sketch of the action of the various parts and of the manner in which insects are captured. Duration of the inflection of the tentacles. Nature of the secretion. Manner in which insects are carried to the center of the leaf. Evidence that the glands have the power of absorption. Small size of the roots. During the summer of 1860, I was surprised by finding how large a number of insects were caught by the leaves of the common sundew Drosera rotundifolia on a heath in Sussex. I had heard that insects were thus caught, but knew nothing further on the subject. As Dr. Nitschke has given Bot Zeitung 1860 p. 229, the bibliography of Drosera, I need not here go into details. Most of the notices published before 1860 are brief and unimportant. The oldest paper seems to have been one of the most valuable, namely by Dr. Roth. In 1782, there is also an interesting though short account of the habits of Drosera by Dr. Mulder in the Bot Zeitung 1852 p. 540 in 1855. In the Annals des Essenat Bot Tom Roman 3 pp. 297 and 304 Roman 2000 Groenland and Tiarcol each published papers with figures on the structure of the leaves, but Roman 1000 Tiarcol went so far as to doubt whether they possessed any power of movement. Dr. Nitschke's papers in the Bot Zeitung for 1860 and 1861 are by far the most important ones which have been published both on the habits and structure of this plant, and I shall frequently have occasion to quote from them. His discussions on several points, for instance, on the transmission of an excitement from one part of the leaf to another, are excellent. On December 11, 1862, Mr. J. Scott read a paper before the Botanical Society of Edinburgh, page 2, which was published in the Gardener's Chronicle, 1863 p. 30. Mr. Scott shows that gentle irritation of the hairs, as well as insects placed on the disc of the leaf, cause the hairs to bend inwards. Mr. O'Bennett also gave another interesting account of the movements of the leaves before the British Association for 1873. In this same year, Dr. Warming published an essay in which he describes the structure of the so-called hairs, entitled Sur la différence entre les trichomes, Roman 100, extracted from the proceedings of the Soche de Hist Nat de Copenhague. I shall also have occasion hereafter to refer to a paper by Mrs. Treat of New Jersey. On some American species of Drosera, Dr. Burden Sanderson delivered a lecture on Dionea before the Royal Institution, published in Nature June 14, 1874, in which a short account of my observations on the power of true digestion possessed by Drosera and Dionea first appeared. Prof. Gray has done good service by calling attention to Drosera and to other plants having similar habits in the Nation, 1874 pp. 261 and 232, and in other publications. Dr. Hooker also, in his important address on carnivorous plants, Brit Assoc Belfast, 1874, has given a history of the subject, page 2. Gathered by chance, a dozen plants bearing 56 fully expanded leaves, and on 31 of these dead insects, or remnants of them, adhered, and no doubt many more would have been caught afterwards by these same leaves, and still more by those as yet not expanded. On one plant, all 6 leaves had caught their prey, and on several plants, very many leaves had caught more than a single insect on one large leaf. I found the remains of 13 distinct insects. Flies, Diptera, are captured much oftener than other insects. The largest kind, which I have seen caught, was a small butterfly, Chenonympha pamphilus, but the Rev. H. Roman 1000 Wilkinson informs me that he found a large living dragonfly with its body firmly held by 2 leaves. As this plant is extremely common in some districts, the number of insects thus annually slaughtered must be prodigious. Many plants cause the death of insects. For instance, the sticky buds of the horsechestnut Aesculus hippocastanum, without thereby receiving, as far as we can perceive, any advantage. But it was soon evident that Drosera was, page 3, excellently adapted for the special purpose of catching insects, so that the subject seemed well worthy of investigation. The results have proved highly remarkable, the more important ones being, firstly, the extraordinary Fig 1 Drosera rotundifolia leaf, viewed from above enlarged 4 times. Sensitiveness of the glands to slight pressure and to minute doses of certain nitrogenous fluids, as shown by the movements of the so-called hairs or tentacles. The drawings of Drosera and Dionea given in this work were made for me by my son George Darwin, those of Aldrovanda, and of the several species of Utricularia by my son Francis. They have been excellently reproduced on wood by Mr. Cooper, 188 Strand, page 4. Secondly, the power possessed by the leaves of rendering soluble or digesting nitrogenous substances, and of afterwards absorbing them. Thirdly, the changes which take place within the cells of the tentacles when the glands are excited in various ways. It is necessary in the first place to describe briefly the plant. It bears from 2 or 3 to 5 or 6 leaves, generally extended more or less horizontally, but sometimes standing vertically upwards. The shape and general appearance of a leaf is shown as seen from above in Fig 1, and as seen laterally in Fig 2. The leaves are commonly a little broader than long. Fig 2 Drosera rotundifolia, old leaf, viewed laterally enlarged about 5 times. But this was not the case in the one here figured. The whole upper surface is covered with gland-bearing filaments or tentacles, as I shall call them from their manner of acting. The glands were counted on 31 leaves, but many of these were of unusually large size, and the average number was 192, the greatest number being 260, and the least 130. The glands are each surrounded by large drops of extremely viscid secretion, which, glittering in the sun, have given rise to the plant's poetical name of the sundew. The tentacles on the central part of the leaf or disc are short and stand upright, and their pedicels are green towards the margin. They become longer and longer and more inclined, page 5 outwards, with their pedicels of a purple color. Those on the extreme margin project in the same plane with the leaf, or more commonly see Fig 2, are considerably reflexed. A few tentacles spring from the base of the footstock or petiole, and these are the longest of all, being sometimes nearly 14 of an inch in length on a leaf, bearing altogether 252 tentacles. The short ones on the disc, having green pedicels, were in number to the longer submarginal and marginal tentacles, having purple pedicels, as 9 to 16. A tentacle consists of a thin, straight, hair-like pedicel carrying a gland on the summit. The pedicel is somewhat flattened and is formed of several rows of elongated cells filled with purple fluid or granular matter. There is, however, a narrow zone close beneath the glands of the longer tentacles, and a broader zone near their bases of a green tint. Spiral vessels, accompanied by simple vascular tissue, branch off from the vascular bundles in the blade of the leaf, and run up all the tentacles into the glands. Several eminent physiologists have discussed the homological nature of these appendages or tentacles, that is, whether they ought to be considered as hairs, trichomes, or prolongations of the leaf niche, has shown that they include all the elements proper to the blade of a leaf, and the fact of their including vascular tissue was formerly thought to prove that they were prolongations of the leaf. But it is now known that vessels sometimes enter true hairs. The power of movement which they possess is a strong argument against their being viewed as hairs. The conclusion, which seems to me the most probable, will be given in chap Roman 15, namely, that they existed primordially as glandular hairs or mere epidermic formations, and that their upper part should still be so considered, but that their lower. According to Nitschke Bot Zeitung, 1861 p. 224, the purple fluid results from the metamorphosis of chlorophyll. Mr. Sorby examined the coloring matter with the spectroscope, and informs me that it consists of the commonest species of erythrophil, which is often met with in leaves with low vitality, and in parts like the petioles, which carry on leaf functions in a very imperfect manner. All that can be said, therefore, is that the hairs or tentacles are colored like parts of a leaf, which do not fulfill their proper office. Dr. Nitschke has discussed this subject in Bot Zeitung, 1861, p. 241 Roman 100. See also Dr. Warming sur la différence entre les trichomes Roman 100 1873, who gives references to various publications. See also Groenland and Tiarkull, Annal des Essenat, Bot 4th series, Tom Roman 3 pp 297 and 303 page 6. Part which alone is capable of movement consists of a prolongation of the leaf, the spiral vessels being extended from this to the uppermost part. We shall hereafter see that the terminal tentacles of the divided leaves of Roridula are still in an intermediate condition. The glands, with the exception of those borne by the extreme. Fig. 3 Drosera rotundifolia longitudinal section of a gland, greatly magnified from Dr. Warming. Marginal tentacles are oval and of nearly uniform size, viz. about 4/500 of an inch in length. Their structure is remarkable, and their functions complex, for they secrete, absorb, and are acted on by various stimulants. They consist of an outer layer of small polygonal cells containing purple granular matter or fluid, and with the walls thicker than those of the pedicels. Page 7. Within this layer of cells, there is an inner one of differently shaped ones, likewise filled with purple fluid, but of a slightly different tint and differently affected by chloride of gold. These two layers are sometimes well seen when a gland has been crushed or boiled in caustic potash. According to Dr. Warming, there is still another layer of much more elongated cells, as shown in the accompanying section. Fig. 3, copied from his work. But these cells were not seen by Nitschke nor by me. In the center, there is a group of elongated cylindrical cells of unequal lengths, bluntly pointed at their upper ends, truncated or rounded at their lower ends, closely pressed together, and remarkable from being surrounded by a spiral line which can be separated as a distinct fiber. These latter cells are filled with limpid fluid, which after long immersion in alcohol deposits much brown matter. I presume that they are actually connected with the spiral vessels, which run up the tentacles, for on several occasions the latter were seen to divide into 2 or 3 excessively thin branches, which could be traced close up to the spiriferous cells. Their development has been described by Dr. Warming. Cells of the same kind have been observed in other plants, as I hear from Dr. Hooker, and were seen by me in the margins of the leaves of Pinguicula. Whatever their function may be, they are not necessary for the secretion of a digestive fluid, or for absorption, or for the communication of a motor impulse to other parts of the leaf, as we may infer from the structure of the glands in some other genera of the Droseraceae. The extreme marginal tentacles differ slightly from the others. Their bases are broader, and besides their own vessels, they receive a fine branch from those which enter the tentacles on each side. Their glands are much elongated and lie embedded on the upper surface of the pedicel, instead of standing at the apex. In other respects, they do not differ essentially from the oval ones. And in one specimen, I found every possible transition between the two states. In another specimen, there were no long-headed glands. These marginal tentacles lose their irritability earlier than the others, and when a stimulus is applied to the center of the leaf, they are excited into action after the others. When cut-off leaves are immersed in water, they alone often become inflected. The purple fluid or granular matter which fills the cells of the glands differs to a certain extent from that within the cells of the pedicels, for when a leaf is placed in hot water or in certain acids, the glands become quite white and opaque, whereas page 8 the cells of the pedicels are rendered of a bright red. With the exception of those close beneath the glands, these latter cells lose their pale red tint, and the green matter, which they as well as the basal cells contain, becomes of a brighter green. The petioles bear many multicellular hairs, some of which near the blade are surmounted, according to Nitschke, by a few rounded cells which appear to be rudimentary glands. Both surfaces of the leaf, the pedicels of the tentacles, especially the lower sides of the outer ones, and the petioles are studded with minute papillae hairs or trichomes having a conical basis, and bearing on their summits 2 and occasionally 3 or even 4 rounded cells. Containing much protoplasm, these papillae are generally colorless, but sometimes include a little purple fluid. They vary in development and graduate, as Nitschke states, and as I repeatedly observed, into the long multicellular hairs. The latter, as well as the papillae, are probably rudiments of formerly existing tentacles. I may here add, in order not to recur to the papillae, that they do not secrete, but are easily permeated by various fluids. Thus, when living or dead leaves are immersed in a solution of 1 part of chloride of gold, or of nitrate of silver, to 437 of water, they are quickly blackened, and the discoloration soon spreads to the surrounding tissue. The long multicellular hairs are not so quickly affected after a leaf had been left in a weak infusion of raw meat for 10 hours. The cells of the papillae had evidently absorbed animal matter, for instead of limpid fluid, they now contained small aggregated masses of protoplasm, which slowly and incessantly changed their forms. A similar result followed from an immersion of only 15 minutes in a solution of 1 part of carbonate of ammonia to 218 of water, and the adjoining cells of the tentacles on which the papillae were seated now likewise contained aggregated masses of protoplasm. We may therefore conclude that when a leaf has closely clasped a captured insect in the manner immediately to be described, the papillae which project from the upper surface of the leaf and of the tentacles probably absorb some of the animal matter dissolved in the secretion. But this cannot be the case with the papillae on the backs of the leaves or on the petioles. Nitschke has elaborately described and figured these papillae. Bot, Zeitung, 1861, pp. 234, 253, 254, page 9. Preliminary sketch of the action of the several parts and of the manner in which insects are captured. If a small organic or inorganic object be placed on the glands in the center of a leaf, these transmit a motor impulse to the marginal tentacles. The nearer ones are first affected and slowly bend towards the center, and then those farther off, until at last all become closely inflected over the object. This takes place in from 1 hour to 4 or 5 or more hours. The difference in the time required depends on many circumstances, namely on the size of the object, and on its nature, that is, whether it contains soluble matter of the proper kind, on the vigor and age of the leaf, whether it has lately been in action, and according to Nitschke, on the temperature of the day, as likewise seemed to me to be the case. A living insect is a more efficient object than a dead one, as in struggling it presses against the glands of many tentacles. An insect, such as a fly, with thin integuments, through which animal matter in solution can readily pass into the surrounding dense secretion, is more efficient in causing prolonged inflection than an insect with a thick coat, such as a beetle. The inflection of the tentacles takes place indifferently in the light and darkness, and the plant is not subject to any nocturnal movement of so-called sleep. If the glands on the disc are repeatedly touched or brushed, although no object is left on them, the marginal tentacles curve inwards. So again, if drops of various fluids, for instance of saliva, or of a solution of any salt of ammonia, are placed on the central glands, the same result quickly follows, sometimes in under half an hour. Bot Zeitung, 1860 p. 246, page 10. The tentacles in the act of inflection sweep through a wide space, thus a marginal tentacle, extended in the same plane with the blade, moves through an angle of 180°, and I have seen the much reflected tentacles of a leaf, which stood upright, move through an angle of not less than 270°. The bending part is almost confined to a short space near the base, but a rather larger portion of the elongated exterior tentacles. Fig. 4, Drosera rotundifolia, leaf enlarged, with all the tentacles closely inflected from immersion, in a solution of phosphate of ammonia 1 part to 87,500 of water. Fig. 5, Drosera rotundifolia, leaf enlarged, with the tentacles on one side, inflected over a bit of meat placed on the disc. Becomes slightly incurved, the distal half in all cases, remaining straight. The short tentacles in the center of the disc, when directly excited, do not become inflected, but they are capable of inflection if excited by a motor impulse received from other glands at a distance. Thus, if a leaf is immersed in an infusion of raw meat or in a weak solution of ammonia, if the page 11 solution is at all strong, the leaf is paralyzed. All the exterior tentacles bend inwards. See Fig. 4, excepting those near the center, which remain upright, but these bend towards any exciting object placed on one side of the disc, as shown in Fig. 5. The glands in Fig. 4 may be seen to form a dark ring round the center, and this follows from the exterior tentacles increasing in length in due proportion as they stand nearer to the circumference. The kind of inflection which the tentacles undergo is best shown when the gland of one of the long exterior. Fig 6 Drosera rotundifolia diagram showing one of the exterior tentacles closely inflected the two adjoining ones in their ordinary position. Tentacles is in any way excited for the surrounding ones remain unaffected. In the accompanying outline Fig 6, we see one tentacle on which a particle of meat had been placed, thus bent towards the center of the leaf, with two others retaining their original position. A gland may be excited by being simply touched 3 or 4 times, or by prolonged contact with organic or inorganic objects and various fluids. I have distinctly seen through a lens a tentacle beginning to bend in 10 seconds after an object had been page 12 placed on. Its gland and I have often seen strongly pronounced inflection in under 1 minute. It is surprising how minute a particle of any substance, such as a bit of thread or hair or splinter of glass, if placed in actual contact with the surface of a gland, suffices to cause the tentacle to bend if the object which has been carried by this movement to the center be not very small, or if it contains soluble nitrogenous matter. It acts on the central glands, and these transmit a motor impulse to the exterior tentacles, causing them to bend inwards. Not only the tentacles, but the blade of the leaf often, but by no means always, becomes much incurved when any strongly exciting substance or fluid is placed on the disc. Drops of milk and of a solution of nitrate of ammonia or soda are particularly apt to produce this effect. The blade is thus converted into a little cup. The manner in which it bends varies greatly, sometimes the apex alone, sometimes one side, and sometimes both sides become incurved. For instance, I placed bits of hard-boiled egg on 3 leaves. One had the apex bent towards the base, the second had both distal margins much incurved, so that it became almost triangular in outline, and this perhaps is the commonest case, whilst the third blade was not at all affected, though the tentacles were as closely inflected. As in the 2 previous cases, the whole blade also generally rises or bends upwards and thus forms a smaller angle with the footstalk than it did before. This appears at first sight a distinct kind of movement, but it results from the incurvation of that part of the margin which is attached to the footstalk, causing the blade as a whole to curve or move upwards. The length of time during which the tentacles, as page 13, well as the blade, remain inflected over an object placed on the disc depends on various circumstances, namely on the vigor and age of the leaf, and according to Dr. Nitschke on the temperature, for during cold weather, when the leaves are inactive, they re-expand at an earlier period than when the weather is warm, but the nature of the object is by far the most important circumstance. I have repeatedly found that the tentacles remain clasped for a much longer average time over objects which yield soluble nitrogenous matter than over those, whether organic or inorganic, which yield no such matter. After a period varying from 1 to 7 days, the tentacles and blade re-expand, and are then ready to act again. I have seen the same leaf inflected 3 successive times over insects placed on the disc, and it would probably have acted a greater number of times. The secretion from the glands is extremely viscid, so that it can be drawn out into long threads. It appears colorless, but stains little balls of paper pale pink. An object of any kind placed on a gland always causes it, as I believe, to secrete more freely, but the mere presence of the object renders this difficult to ascertain. In some cases, however, the effect was strongly marked, as when particles of sugar were added, but the result in this case is probably due merely to exosmose particles of carbonate and phosphate of ammonia, and of some other salts, for instance sulfate of zinc, likewise increased the secretion. Immersion in a solution of 1 part of chloride of gold or of some other salts to 437 of water excites the glands to largely increased secretion. On the other hand, tartrate of antimony produces no such effect. Immersion in many acids of the strength of 1 part to 437 of water likewise causes a wonderful amount of page 14 secretion, so that when the leaves are lifted out, long ropes of extremely viscid fluid hang from them. Some acids, on the other hand, do not act in this manner. Increased secretion is not necessarily dependent on the inflection of the tentacle, for particles of sugar and of sulfate of zinc cause no movement. It is a much more remarkable fact that when an object such as a bit of meat or an insect is placed on the disc of a leaf, as soon as the surrounding tentacles become considerably inflected, their glands pour forth an increased amount of secretion. I ascertained this by selecting leaves with equal-sized drops on the two sides and by placing bits of meat on one side of the disc. And as soon as the tentacles on this side became much inflected, but before the glands touched the meat, drops of secretion became larger. This was repeatedly observed, but a record was kept of only 13 cases, in 9 of which increased secretion was plainly observed, the 4 failures being due either to the leaves being rather torpid, or to the bits of meat being too small to cause much inflection. We must therefore conclude that the central glands, when strongly excited, transmit some influence to the glands of the circumferential tentacles, causing them to secrete more copiously. It is a still more important fact, as we shall see more fully when we treat of the digestive power of the secretion, that when the tentacles become inflected owing to the central glands having been stimulated mechanically or by contact with animal matter, the secretion not only increases in quantity, but changes its nature and becomes acid, and this occurs before the glands have touched the object on the center of the leaf. This acid is of a different nature from that contained in the tissue of the leaves. As long as the page 15 tentacles remain closely inflected, the glands continue to secrete, and the secretion is acid, so that if neutralized by carbonate of soda it again becomes acid after a few hours. I have observed the same leaf with the tentacles closely inflected over rather indigestible substances, such as chemically prepared casein pouring forth acid secretion for 8 successive days, and over bits of bone for 10 successive days. The secretion seems to possess, like the gastric juice of the higher animals, some antiseptic power. During very warm weather, I placed close together 2 equal-sized bits of raw meat, one on a leaf of the Drosera, and the other surrounded by wet moss. They were thus left for 48 HRS, and then examined. The bit on the moss swarmed with infusoria, and was so much decayed that the transverse striae on the muscular fibers could no longer be clearly distinguished, whilst the bit on the leaf, which was bathed by the secretion, was free from infusoria, and its striae were perfectly distinct in the central and undissolved portion. In like manner, small cubes of albumin and cheese, placed on wet moss, became threaded with filaments of mold, and had their surfaces slightly discolored and disintegrated, whilst those on the leaves of Drosera remained clean, the albumin being changed into transparent fluid. As soon as tentacles, which have remained closely inflected during several days over an object, begin to re-expand, their glands secrete less freely or cease to secrete, and are left dry. In this state, they are covered with a film of whitish semifibrous matter, which was held in solution by the secretion. The drying of the glands during the act of re-expansion is of some little service to the plant, for I have often observed that objects adhering to the leaves, page 16, could then be blown away by breath of air, the leaves being thus left unencumbered and free for future action. Nevertheless, it often happens that all the glands do not become completely dry, and in this case, delicate objects such as fragile insects are sometimes torn by the re-expansion of the tentacles into fragments which remain scattered all over the leaf. After the re-expansion is complete, the glands quickly begin to resecrete, and as soon as full-sized drops are formed, the tentacles are ready to clasp a new object. When an insect alights on the central disc, it is instantly entangled by the viscid secretion, and the surrounding tentacles after a time begin to bend and ultimately clasp it on all sides. Insects are generally killed, according to Dr. Nitschke, in about a quarter of an hour, owing to their tracheae being closed by the secretion. If an insect adheres to only a few of the glands of the exterior tentacles, these soon become inflected and carry their prey to the tentacles next succeeding them inwards. These then bend inwards and so onwards, until the insect is ultimately carried by a curious sort of rolling movement to the center of the leaf. Then, after an interval, the tentacles on all sides become inflected and bathe their prey with their secretion in the same manner as if the insect had first alighted on the central disc. It is surprising how minute an insect suffices to cause this action. For instance, I have seen one of the smallest species of gnats, culex, which had just settled with its excessively delicate feet on the glands of the outermost tentacles, and these were already beginning to curve inwards, though not a single gland had as yet touched the body of the insect. Had I not interfered, this minute gnat would, page 17, assuredly have been carried to the center of the leaf, and been securely clasped on all sides. We We shall hereafter see what excessively small doses of certain organic fluids and saline solutions cause strongly marked inflection. Whether insects alight on the leaves by mere chance as a resting place, or are attracted by the odor of the secretion, I know not. I suspect from the number of insects caught by the English species of Drosera, and from what I have observed with some exotic species kept in my greenhouse, that the odor is attractive in this latter case. The leaves may be compared with a baited trap in the former case, with a trap laid in a run frequented by game, but without any bait. That the glands possess the power of absorption is shown by their almost instantaneously becoming dark-colored, when given a minute quantity of carbonate of ammonia, the change of color being chiefly or exclusively due to the rapid aggregation of their contents. When certain other fluids are added, they become pale-colored. Their power of absorption is, however, best shown by the widely different results which follow from placing drops of various nitrogenous and non-nitrogenous fluids of the same density on the glands of the disc, or on a single marginal gland, and likewise by the very different lengths of time during which the tentacles remain inflected over objects which yield or do not yield soluble nitrogenous matter. This same conclusion might indeed have been inferred from the structure and movements of the leaves, which are so admirably adapted for capturing insects. The absorption of animal matter from captured insects explains how Drosera can flourish in extremely poor peaty soil in some cases where nothing but page 18 sphagnum moss grows, and mosses depend altogether on the atmosphere for their nourishment, although the leaves at a hasty glance do not appear green owing to the purple color of the tentacles, yet the upper and lower surfaces of the blade, the pedicels of the central tentacles and the petioles, contain chlorophyll, so that no doubt the plant obtains and assimilates carbonic acid from the air. Nevertheless, considering the nature of the soil where it grows, the supply of nitrogen would be extremely limited or quite deficient, unless the plant had the power of obtaining this important element from captured insects. We can thus understand how it is that the roots are so poorly developed. These usually consist of only 2 or 3 slightly divided branches from ½ to 1 inch in length, furnished with absorbent hairs. It appears, therefore, that the roots serve only to imbibe water, though no doubt they would absorb nutritious matter if present in the soil, for as we shall hereafter see, they absorb a weak solution of carbonate of ammonia. A plant of Drosera, with the edges of its leaves curled inwards so as to form a temporary stomach, with the glands of the closely inflected tentacles pouring forth their acid secretion, which dissolves animal matter afterwards to be absorbed, may be said to feed like an animal, but differently from an animal. It drinks by means of its roots, and it must drink largely so as to retain many drops of viscid fluid round the glands, sometimes as many as 260, exposed during the whole day to a glaring sun. Page 19. Chapter II. The movements of the tentacles from the contact of solid bodies. Inflection of the exterior tentacles, owing to the glands of the disc being excited by repeated touches or by objects left in contact with them. Difference in the action of bodies yielding and not yielding soluble nitrogenous matter. Inflection of the exterior tentacles directly caused by objects left in contact with their glands. Periods of commencing inflection and of subsequent re-expansion. Extreme minuteness of the particles causing inflection. Action underwater. Inflection of the exterior tentacles when their glands are excited by repeated touches. Falling drops of water do not cause inflection. I will give in this and the following chapters some of the many experiments made which best illustrate the manner and rate of movement of the tentacles when excited in various ways. The glands alone, in all ordinary cases, are susceptible to excitement. When excited, they do not themselves move or change form, but transmit a motor impulse to the bending part of their own and adjoining tentacles, and are thus carried towards the center of the leaf. Strictly speaking, the glands ought to be called irritable, as the term sensitive generally implies consciousness, but no one supposes that the sensitive eplant is conscious, and as I have found the term convenient, I shall use it without scruple. I will commence with the movements of the exterior tentacles when indirectly excited by stimulants applied to the glands of the short tentacles on the disc. The exterior tentacles may be said in this case to be indirectly excited, because their own glands are not directly acted on. The stimulus proceeding from the glands of the disc acts on the bending part of the page 20 exterior tentacles near their bases, and does not, as will hereafter be proved, first travel up the pedicels to the glands, to be then reflected back to the bending place. Nevertheless, some influence does travel up to the glands, causing them to secrete more copiously, and the secretion to become acid. This latter fact is, I believe, quite new in the physiology of plants. It has indeed only recently been established that in the animal kingdom, an influence can be transmitted along the nerves to glands, modifying their power of secretion independently of the state of the blood vessels. The inflection of the exterior tentacles from the glands of the disc being excited by repeated touches or by objects left in contact with them. The central glands of a leaf were irritated with a small stiff camel hair brush, and in 70 minutes several of the outer tentacles were inflected in 5 HRS hours. All the submarginal tentacles were inflected next morning. After an interval of about 22 HRS, they were fully re-expanded. In all the following cases, the period is reckoned from the time of first irritation. Another leaf, treated in the same manner, had a few tentacles. Inflected in 20 in 4 HRS, all the submarginal and some of the extreme marginal tentacles, as well as the edge of the leaf itself, were inflected in 17 HRS. They had recovered their proper expanded position. I then put a dead fly in the center of the last-mentioned leaf, and next morning, it was closely clasped. 5 days afterwards, the leaf re-expanded, and the tentacles with their glands surrounded by secretion were ready to act again. Particles of meat, dead flies, bits of paper, wood, dried moss, sponge, cinders, glass, Roman 100, were repeatedly, page 21, placed on leaves, and these objects were well embraced in various periods from 1 HR to as long as 24 HRS, and set free again with the leaf fully re-expanded in from 1 or 2 to 7 or even 10 days, according to the nature of the object on a leaf, which had naturally caught 2 flies, and therefore had already closed and reopened either once or more probably twice. I put a fresh fly in 7 HRS, it was moderately, and in 21 HRS, thoroughly well clasped, with the edges of the leaf inflected in 2 days and a half. The leaf had nearly re-expanded. As the exciting object was an insect, this unusually short period of inflection was, no doubt, due to the leaf having recently been in action. Allowing this same leaf to rest for only a single day, I put on another fly, and it again closed, but now very slowly. Nevertheless, in less than 2 days, it succeeded in thoroughly clasping the fly. When a small object is placed on the glands of the disc, on one side of a leaf, as near as possible to its circumference, the tentacles on this side are first affected, those on the opposite side much later, or as often occurred not at all. This was repeatedly proved by trials with bits of meat, but I will here give only the case of a minute fly naturally caught and still alive, which I found adhering by its delicate feet to the glands on the extreme left side of the central disc. The marginal tentacles on this side closed inwards and killed the fly, and after a time, the edge of the leaf on this side also became inflected, and thus remained for several days, whilst neither the tentacles nor the edge on the opposite side were in the least affected. If young and active leaves are selected, inorganic particles not larger than the head of a small pin placed on the central glands sometimes cause the page 22 outer tentacles to bend inwards, but this follows much more surely and quickly if the object contains nitrogenous matter, which can be dissolved by the secretion. On one occasion, I observed the following unusual circumstance: small bits of raw meat, which acts more energetically than any other substance of paper, dried moss, and of the quill of a pen, were placed on several leaves, and they were all embraced equally well. In about 2 HRS on other occasions, the above-named substances, or more commonly particles of glass coal cinder, taken from the firestone, gold leaf, dried grass, cork, blotting paper, cotton wool, and hair, rolled up into little balls, were used, and these substances, though they were sometimes well embraced, often caused no movement whatever in the outer tentacles, or an extremely slight and slow movement. Yet these same leaves were proved to be in an active condition, as they were excited to move by substances yielding soluble nitrogenous matter, such as bits of raw or roast meat, the yolk or white of boiled eggs, fragments of insects of all orders, spiders, Roman 100. I will give only 2 instances. Minute flies were placed on the discs of several leaves, and on others balls of paper, bits of moss, and quill of about the same size, as the flies in the latter were well embraced in a few hours, whereas after 25 HRS, only a very few tentacles were inflected over the other objects. The bits of paper, moss, and quill were then removed from these leaves and bits of raw meat placed on them, and now all the tentacles were soon energetically inflected. Again, particles of coal cinder weighing rather more than the flies used in the last experiment were placed on the centers of 3 leaves after an interval of 19 HRS. One of the particles was tolerably well embraced, page 23, a second by a very few tentacles, and a third by none. I then removed the particles from the 2 latter leaves and put on them recently killed flies. These were fairly well embraced in 7-12 HRS, and thoroughly after 20-12 HRS, the tentacles remaining inflected for many subsequent days. On the other hand, the one leaf which had in the course of 19 HRS embraced the bit of cinder moderately well, and to which no fly was given, after an additional 33 HRS Roman 1e and 52 HRS from the time when the cinder was put on, was completely re-expanded and ready to act again. From these and numerous other experiments, not worth giving, it is certain that inorganic substances, or such organic substances as are not attacked by the secretion, act much less quickly and efficiently than organic substances yielding soluble matter, which is absorbed. Moreover, I have met with very few exceptions to the rule, and these exceptions apparently depended on the leaf having been too recently in action, that the tentacles remain clasped for a much longer time over organic bodies of the nature just specified than over those which are not acted on by the secretion or over inorganic objects. Owing to the extraordinary belief held by Roman 1000 Ziegler Comps Rendus, May 1872, p. 122, that albuminous substances, if held for a moment between the fingers, acquire the property of making the tentacles of Drosera contract, whereas, if not thus held, they have no such power. I tried some experiments with great care, but the results did not confirm this belief. Red-hot cinders were taken out of the fire, and bits of glass cotton thread, blotting paper, and thin slices of cork were immersed in boiling water, and particles were then placed every instrument with which they were touched, having been previously immersed in boiling water on the glands of several leaves, and they acted in exactly the same manner as other particles, which had been purposely handled for some time. Bits of a boiled egg cut with a knife, which had been washed in boiling water, also acted like any other animal substance. I breathed on some leaves for above a minute. Repeated the act 2 or 3 times with my mouth close to page 24 them, but this produced no effect. I may here add, as showing that the leaves are not acted on by the odor of nitrogenous substances, that pieces of raw meat stuck on needles were fixed as close as possible, without actual contact to several leaves, but produced no effect whatever. On the other hand, as we shall hereafter see, the vapors of certain volatile substances and fluids, such as of carbonate of ammonia, chloroform, certain essential oils, Roman 100, cause inflection. Roman 1000 Ziegler makes still more extraordinary statements with respect to the power of animal substances, which have been left close to, but not in contact with, sulfate of quinine. The action of salts of quinine will be described in a future chapter. Since the appearance of the paper above referred to, Roman 1000 Ziegler has published a book on the same subject entitled Atonicit et Zoicit, 1874, page 24. The inflection of the exterior tentacles as directly caused by objects left in contact with their glands. I made a vast number of trials by placing by means of a fine needle moistened with distilled water, and with the aid of a lens, particles of various substances on the viscid secretions surrounding the glands of the outer tentacles. I experimented on both the oval and long-headed glands. When a particle is thus placed on a single gland, the movement of the tentacle is particularly well seen in contrast with the stationary condition of the surrounding tentacles. See previous Fig. 6 in 4 cases. Small particles of raw meat caused the tentacles to be greatly inflected. In between 5 and 6 Roman 1000, another tentacle similarly treated and observed with special care distinctly, though slightly changed its position in 10 seconds, and this is the quickest movement seen by me in 2 Roman 1000 s. It had moved through an angle of about 45°. The movement, as seen through a lens, resembled that of the hand of a large clock. In 5 Roman 1000, it had moved through 90, and when I looked again, after 10 Roman 1000, the particle had reached the center of the leaf, so that the whole movement was completed in less page 25 than 17 Roman 1000 s. In the course of some hours, this minute bit of meat, from having been brought into contact with some of the glands of the central disc, acted centrifugally on the outer tentacles, which all became closely inflected fragments of flies, were placed on the glands of 4 of the outer tentacles, extended in the same plane with that of the blade, and 3 of these fragments were carried in 35 Roman 1000 through an angle of 180 to the center. The fragment on the 4th tentacle was very minute, and it was not carried to the center until 3 HRS had elapsed. In 3 other cases, minute flies or portions of larger ones were carried to the center in 1 HR 30s. In these 7 cases, the fragments or small flies, which had been carried by a single tentacle to the central glands, were well embraced by the other tentacles, after an interval of from 4 to 10 HRS. I also placed in the manner just described 6 small balls of writing paper rolled up by the aid of pincers, so that they were not touched by my fingers on the glands of 6 exterior tentacles on distinct leaves. 3 of these were carried to the center in about 1 HR, and the other 3 in rather more than 4 HRS. But after 24 HRS, only 2 of the 6 balls were well embraced by the other tentacles. It is possible that the secretion may have dissolved a trace of glue or animalized matter from the balls of paper. 4 particles of coalcinder were then placed on the glands of 4 exterior tentacles. 1 of these reached the center in 3 HRS 40 Roman 1000, the second in 9 HRS, the third within 24 HRS, but had moved only part of the way in 9 HRS, whilst the 4th moved only a very short distance in 24 HRS, and never moved any farther of the above 3 bits of cinder, which were ultimately carried to the center. 1 alone was well embraced by page 26. Many of the other tentacles we here see clearly that such bodies as particles of cinder or little balls of paper, after being carried by the tentacles to the central glands, act very differently from fragments of flies in causing the movement of the surrounding tentacles. I made, without carefully recording the times of movement, many similar trials with other substances, such as splinters of white and blue glass particles of cork, minute bits of gold leaf Roman 100, and the proportional number of cases varied much in which the tentacles reached the center or moved only slightly or not at all. One evening, particles of glass and cork, rather larger than those usually employed, were placed on about a dozen glands, and next morning, after 13 HRS, every single tentacle had carried its little load to the center. But the unusually large size of the particles will account for this result. In another case, 67 of the particles of cinder, glass, and thread placed on separate glands were carried towards or actually to the center. In another case, 79, in another 712, and in the last case, only 726 were thus carried inwards, the small proportion being here due at least in part to the leaves being rather old and inactive. Occasionally, a gland with its light load could be seen through a strong lens to move an extremely short distance, and then stop. This was especially apt to occur when excessively minute particles, much less than those of which the measurements will be immediately given, were placed on glands, so that we here have nearly the limit of any action. I was so much surprised at the smallness of the particles, which caused the tentacles to become greatly inflected, that it seemed worth while carefully to ascertain how minute a particle would plainly act. Page 27 accordingly measured lengths of a narrow strip of blotting paper of fine cotton thread and of a woman's hair were carefully weighed for me by Mr. Trenham Reeks in an excellent balance in the laboratory in Jermyn Street. Short bits of the paper, thread, and hair were then cut off and measured by a micrometer, so that their weights could be easily calculated. The bits were placed on the viscid secretion surrounding the glands of the exterior tentacles, with the precautions already stated. And I am certain that the gland itself was never touched, nor indeed would a single touch have produced any effect. A bit of the blotting paper weighing 1465 of a grain was placed so as to rest on 3 glands together, and all three tentacles slowly curved inwards. Each gland, therefore supposing the weight to be distributed equally, could have been pressed on by only 11,395 of a grain or 464 of a milligram. 5 nearly equal bits of cot thread were tried, and all acted. The shortest of these was 150 of an inch in length, and weighed 18,197 of a grain. The tentacle in this case was considerably inflected in 1 HR 30 Roman 1000, and the bit of thread was carried to the center of the leaf in 1 HR 40 Roman 1000 again. 2 particles of the thinner end of a woman's hair, one of these being 181,000 of an inch in length, and weighing 135,714 of a grain, the other 191,000 of an inch in length, and weighing, of course, a little more, were placed on 2 glands on opposite sides of the same leaf, and these 2 tentacles were inflected halfway towards the center in 1 HR 10 Roman 1000, all the many other tentacles round the same leaf remaining motionless. The appearance of this one leaf showed, in an unequivocal manner, that these minute particles sufficed to cause the tentacles to bend. Altogether, 10 such particles of hair were placed on 10 glands on several leaves, and 7 of them caused, page 28, the tentacles to move in a conspicuous manner. The smallest particle which was tried, and which acted plainly, was only 81,000 of an inch 203 millimeter in length, and weighed the 178,740 of a grain, or 822 milligram. In these several cases, not only was the inflection of the tentacles conspicuous, but the purple fluid within their cells became aggregated into little masses of protoplasm in the manner to be described in the next chapter, and the aggregation was so plain that I could, by this clue alone, have readily picked out under the microscope all the tentacles which had carried their light loads towards the center from the hundreds of other tentacles on the same leaves which had not thus acted. My surprise was greatly excited not only by the minuteness of the particles which caused movement, but how they could possibly act on the glands, for it must be remembered that they were laid with the greatest care on the convex surface of the secretion. At first I thought, but, as I now know, erroneously, that particles of such low specific gravity as those of cork thread and paper would never come into contact with the surfaces of the glands. The particles cannot act simply by their weight being added to that of the secretion, for small drops of water, many times heavier than the particles, were repeatedly added, and never produced any effect, nor does the disturbance of the secretion produce any effect, for long threads were drawn out by a needle, and affixed to some adjoining object, and thus left for hours, but the tentacles remained motionless. I also carefully removed the secretion from 4 glands with a sharply pointed piece of blotting paper, so that they were exposed for a time naked to the air. But this caused no movement, yet these glands were, page 29, in an efficient state, for after 24 HRS had elapsed, they were tried with bits of meat, and all became quickly inflected. It then occurred to me that particles floating on the secretion would cast shadows on the glands, which might be sensitive to the interception of the light. Although this seemed highly improbable, as minute and thin splinters of colorless glass acted powerfully nevertheless. After it was dark, I put on, by the aid of a single tallow candle, as quickly as possible, particles of cork and glass on the glands of a dozen tentacles, as well as some of meat on other glands, and covered them up so that not a ray of light could enter. But by the next morning, after an interval of 13 HRS, all the particles were carried to the centers of the leaves. These negative results led me to try many more experiments by placing particles on the surface of the drops of secretion, observing as carefully as I could whether they penetrated it and touched the surface of the glands. The secretion, from its weight, generally forms a thicker layer on the under than on the upper sides of the glands. Whatever may be the position of the tentacles, minute bits of dry cork thread, blotting paper, and coal cinders were tried, such as those previously employed, and I now observed that they absorbed much more of the secretion in the course of a few minutes than I should have thought possible, and as they had been laid on the upper surface of the secretion where it is thinnest, they were often drawn down after a time into contact with at least some one point of the gland. With respect to the minute splinters of glass and particles of hair, I observed that the secretion slowly spread itself a little over their surfaces, by which means they were likewise drawn downwards or sideways, and thus one end, or some minute, page 30 prominence, often came to touch sooner or later the gland. In the foregoing and following cases, it is probable that the vibrations to which the furniture in every room is continually liable aids in bringing the particles into contact with the glands, but as it was sometimes difficult owing to the refraction of the secretion to feel sure whether the particles were in contact, I tried the following experiment. Unusually minute particles of glass, hair, and cork were gently placed on the drops round several glands, and very few of the tentacles moved. Those which were not affected were left for about half an hour, and the particles were then disturbed or tilted up several times with a fine needle under the microscope, the glands not being touched, and now, in the course of a few minutes, almost all the hitherto motionless tentacles began to move, and this no doubt was caused by one end or some prominence of the particles having come into contact with the surface of the glands. But as the particles were unusually minute, the movement was small. Lastly, some dark blue glass pounded into fine splinters was used in order that the points of the particles might be better distinguished when immersed in the secretion, and 13 such particles were placed in contact with the depending and therefore thicker part of the drops round so many glands. 5 of the tentacles began moving after an interval of a few minutes, and in these cases I clearly saw that the particles touched the lower surface of the gland. A 6th tentacle moved after 1 HR 45 Roman 1000, and the particle was now in contact with the gland, which was not the case at first, so it was with the 7th tentacle, but its movement did not begin until 3 HRS 45 Roman 1000 had page 31 elapsed. The remaining 6 tentacles never moved as long as they were observed, and the particles apparently never came into contact with the surfaces of the glands. From these experiments, we learn that particles not containing soluble matter, when placed on glands, often cause the tentacles to begin bending in the course of from 1 to 5 minutes, and that in such cases, the particles have been from the first in contact with the surfaces of the glands, when the tentacles do not begin moving for a much longer time, namely from half an hour to 3 or 4 hours. The particles have been slowly brought into contact with the glands, either by the secretion being absorbed by the particles or by its gradual spreading over them together with its consequent quicker evaporation. When the tentacles do not move at all, the particles have never come into contact with the glands, or in some cases, the tentacles may not have been in an active condition. In order to excite movement, it is indispensable that the particles should actually rest on the glands for a touch once, twice, or even thrice, repeated by any hard body, is not sufficient to excite movement. Another experiment showing that extremely minute particles act on the glands when immersed in water may here be given. A grain of sulfate of quinine was added to an ounce of water, which was not afterwards filtered, and on placing 3 leaves in 90 minims of this fluid, I was much surprised to find that all 3 leaves were greatly inflected in 15 Roman 1000, for I knew from previous trials that the solution does not act so quickly as this. It immediately occurred to me that the particles of the undissolved salt, which were so light as to float about, might have come page 32 into contact with the glands, and caused this rapid movement. Accordingly, I added to some distilled water a pinch of a quite innocent substance, namely precipitated carbonate of lime, which consists of an impalpable powder. I shook the mixture, and thus got a fluid like thin milk. 2 leaves were immersed in it, and in 6 Roman 1000, almost every tentacle was much inflected. I placed one of these leaves under the microscope and saw innumerable atoms of lime adhering to the external surface of the secretion. Some, however, had penetrated it and were lying on the surfaces of the glands, and no doubt it was these particles which caused the tentacles to bend. When a leaf is immersed in water, the secretion instantly swells much, and I presume that it is ruptured here and there so that little eddies of water rush in. If so, we can understand how the atoms of chalk, which rested on the surfaces of the glands, had penetrated the secretion. Anyone who has rubbed precipitated chalk between his fingers will have perceived how excessively fine the powder is. No doubt there must be a limit beyond which a particle would be too small to act on a gland, but what this limit is I know not. I have often seen fibers and dust which had fallen from the air on the glands of plants kept in my room, and these never induced any movement, but then such particles lay on the surface of the secretion and never reached the gland itself. Finally, it is an extraordinary fact that a little bit of soft thread 150 of an inch in length and weighing 18,197 of a grain, or of a human hair 81,000 of an inch in length and weighing only 178,740 of a grain 822 milligram, or particles of precipitated chalk. After resting for a short time on a gland should induce some change in its cells, exciting them, page 33, to transmit a motor impulse throughout the whole length of the pedicel, consisting of about 20 cells to near its base, causing this part to bend and the tentacle to sweep through an angle of above 180°, that the contents of the cells of the glands and afterwards those of the pedicels are affected in a plainly visible manner by the pressure of minute particles. We shall have abundant evidence when we treat of the aggregation of protoplasm. But the case is much more remarkable than as yet stated, for the particles are supported by the viscid and dense secretion, nevertheless even smaller ones than those of which the measurements have been given, when brought by an insensibly slow movement through the means above specified into contact with the surface of a gland act on it, and the tentacle bends the pressure exerted by the particle of hair weighing only 178,740 of a grain, and supported by a dense fluid, must have been inconceivably slight. We may conjecture that it could hardly have equaled the millionth of a grain, and we shall hereafter see that far less than the millionth of a grain of phosphate of ammonia in solution, when absorbed by a gland, acts on it, and induces movement. A bit of hair 150 of an inch in length, and therefore much larger than those used in the above experiments, was not perceived when placed on my tongue, and it is extremely doubtful whether any nerve in the human body, even if in an inflamed condition, would be in any way affected by such a particle, supported in a dense fluid and slowly brought into contact with the nerve. Yet the cells of the glands of Drosera are thus excited to transmit a motor impulse to a distant point, inducing movement. It appears to me that hardly any more remarkable fact than this has been observed in the vegetable kingdom, page 34. The inflection of the exterior tentacles when their glands are excited by repeated touches. We have already seen that if the central glands are excited by being gently brushed, they transmit a motor impulse to the exterior tentacles, causing them to bend. And we have now to consider the effects which follow from the glands of the exterior tentacles being themselves touched on several occasions. A large number of glands were touched only once with a needle or fine brush hard enough to bend the whole flexible tentacle, and though this must have caused 1,000-fold greater pressure than the weight of the above described particles, not a tentacle moved on another occasion. 4 to 5 glands on 11 leaves were touched once, twice, or even thrice, with a needle or stiff bristle. This was done as quickly as possible, but with force sufficient to bend the tentacles. Yet only 6 of them became inflected, 3 plainly, and 3 in a slight degree, in order to ascertain whether these tentacles which were not affected were in an efficient state. Bits of meat were placed on 10 of them, and they all soon became greatly incurved. On the other hand, when a large number of glands were struck 4, 5, or 6 times with the same force as before, a needle or sharp splinter of glass, being used, a much larger proportion of tentacles became inflected, but the result was so uncertain as to seem capricious. For instance, I struck in the above manner 3 glands, which happened to be extremely sensitive, and all 3 were inflected almost as quickly as if bits of meat had been placed on them. On another occasion, I gave a single, for page 35, Sybil touch to a considerable number of glands, and not one moved, but these same glands, after an interval of some hours, being touched 4 or 5 times with a needle, several of the tentacles soon became inflected. The fact of a single touch, or even of 2 or 3 touches, not causing inflection, must be of some service to the plant, as during stormy weather the glands cannot fail to be occasionally touched by the tall blades of grass, or by other plants growing near, and it would be a great evil if the tentacles were thus brought into action, for the act of re-expansion takes a considerable time, and until the tentacles are re-expanded, they cannot catch prey. On the other hand, extreme sensitiveness to slight pressure is of the highest service to the plant, for, as we have seen, if the delicate feet of a minute struggling insect press ever so lightly on the surfaces of 2 or 3 glands, the tentacles bearing these glands soon curl inwards and carry the insect with them to the center, causing, after a time, all the circumferential tentacles to embrace it. Nevertheless, the movements of the plant are not perfectly adapted to its requirements, for if a bit of dry moss, peat, or other rubbish is blown on to the disc, as often happens, The tentacles clasp it in a useless manner. They soon, however, discover their mistake and release such innutritious objects. It is also a remarkable fact that drops of water falling from a height, whether under the form of natural or artificial rain, do not cause the tentacles to move, yet the drops must strike the glands with considerable force, more especially after the secretion has been all washed away by heavy rain, and this often occurs. Page 36. Though the secretion is so viscid that it can be removed with difficulty merely by waving the leaves in water, if the falling drops of water are small, they adhere to the secretion, the weight of which must be increased in a much greater degree, as before remarked, than by the addition of minute particles of solid matter, yet the drops never cause the tentacles to become inflected. It would obviously have been a great evil to the plant, as in the case of occasional touches, if the tentacles were excited to bend by every shower of rain. But this evil has been avoided by the glands, either having become through habit insensible to the blows and prolonged pressure of drops of water, or to their having been originally rendered sensitive solely to the contact of solid bodies. We shall hereafter see that the filaments on the leaves of Dionea are likewise insensible to the impact of fluids, though exquisitely sensitive to momentary touches from any solid body. When the pedicel of a tentacle is cut off by a sharp pair of scissors quite close beneath the gland, the tentacle generally becomes inflected. I tried this experiment repeatedly, as I was much surprised at the fact for all other parts of the pedicels are insensible to any stimulus. These headless tentacles after a time re-expand. But I shall return to this subject. On the other hand, I occasionally succeeded in crushing a gland between a pair of pincers, but this caused no inflection. In this latter case, the tentacles seem paralyzed, as likewise follows from the action of too strong solutions of certain salts, and by too great heat, whilst weaker solutions of the same salts and a more gentle heat cause movement. We shall also see in future chapters that various other fluids, some page 37, vapors, and oxygen, after the plant has been for some time excluded from its action, all induce inflection, and this likewise results from an induced galvanic current. My son Francis, guided by the observations of Dr. Burden Sanderson on Dionea, finds that if 2 needles are inserted into the blade of a leaf of Drosera, the tentacles do not move, but that if similar needles in connection with the secondary coil of a Du Bois inductive apparatus are inserted, the tentacles curve inwards in the course of a few minutes. My son hopes soon to publish an account of his observations. Page 38. Chapter Roman 3, Aggregation of the protoplasm within the cells of the tentacles. Nature of the contents of the cells before aggregation. Various causes which excite aggregation. The process commences within the glands and travels down the tentacles. Description of the aggregated masses and of their spontaneous movements. Currents of protoplasm along the walls of the cells. Action of carbonate of ammonia. The granules in the protoplasm, which flows along the walls, coalesce with the central masses. Minuteness of the quantity of carbonate of ammonia, causing aggregation. Action of other salts of ammonia of other substances. Organic fluids. Roman 100. Of water of heat dissolution of the aggregated masses. Proximate causes of the aggregation of the protoplasm. Summary and concluding remarks. Supplementary observations on aggregation in the roots of plants. I will here interrupt my account of the movements of the leaves and describe the phenomenon of aggregation, to which subject I have already alluded. If the tentacles of a young yet fully matured leaf that has never been excited or become inflected be examined, the cells forming the pedicels are seen to be filled with homogeneous purple fluid. The walls are lined by a layer of colorless circulating protoplasm, but this can be seen with much greater distinctness after the process of aggregation has been partly effected than before. The purple fluid, which exudes from a crushed tentacle, is somewhat coherent and does not mingle with the surrounding water. It contains much flocculent or granular matter, but this matter may have been generated by the cells having been crushed. Some degree of aggregation having been thus almost instantly caused. Page 39. If a tentacle is examined some hours after the gland has been excited by repeated touches, or by an inorganic or organic particle placed on it, or by the absorption of certain fluids, it presents a wholly changed appearance. The cells, instead of being filled with homogeneous purple fluid, now contain variously shaped masses of purple matter, suspended in a colorless or almost colorless fluid, the change is so conspicuous that it is visible through a weak lens, and even sometimes by the naked eye. The tentacles now have a mottled appearance, so that one thus affected can be picked out with ease from all the others. The same result follows if the glands on the disc are irritated in any manner, so that the exterior tentacles become inflected, for their contents will then be found in an aggregated condition, although their glands have not as yet touched any object. But aggregation may occur independently of inflection, as we shall presently see. By whatever cause the process may have been excited, it commences within the glands, and then travels down the tentacles. It can be observed much more distinctly in the upper cells of the pedicels than within the glands, as these are somewhat opaque. Shortly after the tentacles have re-expanded, the little masses of protoplasm are all redissolved, and the purple fluid within the cells becomes as homogeneous and transparent as it was at first. The process of redissolution travels upwards from the bases of the tentacles to the glands, and therefore in a reverse direction to that of aggregation. Tentacles in an aggregated condition were shown to Prof. Huxley, Dr. Hooker, and Dr. Burden Sanderson, who observed the changes under the microscope and were much struck with the whole phenomenon. Page 40. The little masses of aggregated matter are of the most diversified shapes, often spherical or oval, sometimes much elongated or quite irregular, with thread or necklace-like or club-formed projections. They consist of thick, apparently viscid matter, which in the exterior tentacles is of a purplish, and in the short distal tentacles of a greenish color. These little masses incessantly change their forms and positions, being never at rest. A single mass will often separate into two, which afterwards reunite. Their movements are rather slow and resemble those of amoebae or of the white corpuscles of the blood. Fig. 7 Drosera rotundifolia diagram of the same cell of a tentacle, showing the various forms successively assumed by the aggregated masses of protoplasm. May therefore conclude that they consist of protoplasm. If their shapes are sketched at intervals of a few minutes, they are invariably seen to have undergone great changes of form, and the same cell has been observed for several hours. 8 Rude, though accurate sketches of the same cell, made at intervals of between 2 Roman 1000 or 3 Roman 1000, are here given. Fig. 7, and illustrates some of the simpler and commonest changes. The cell A, when first sketched, included two oval masses of purple protoplasm touching each other. These became separate as shown at B, and then reunited as at Roman 100. After the next interval, a very common appearance was presented. Page 41D, namely the formation of an extremely minute sphere at one end of an elongated mass. This rapidly increased in size as shown in E, and was then reabsorbed as at F, by which time another sphere had been formed at the opposite end. The cell above figured was from a tentacle of a dark red leaf, which had caught a small moth, and was examined under water. As I at first thought that the movements of the masses might be due to the absorption of water, I placed a fly on a leaf, and when after 18 HRS all the tentacles were well inflected, these were examined without being immersed in water. The Fig. 8 Drosera rotundifolia diagram of the same cell of a tentacle, showing the various forms successively assumed by the aggregated masses of protoplasm. Here represented. Fig. 8 was from this leaf being sketched 8 times in the course of 15 Roman 1000. These sketches exhibit some of the more remarkable changes which the protoplasm undergoes. At first there was at the base of the cell 1 a little mass on a short footstalk, and a larger mass near the upper end, and these seemed quite separate. Nevertheless, they may have been connected by a fine and invisible thread of protoplasm, for on 2 other occasions, whilst one mass was rapidly increasing and another in the same cell rapidly decreasing, I was able, by varying the light and using a high power, to detect a connecting thread of extreme tenuity, which evidently served as, page 42, the channel of communication between the two. On the other hand, such connecting threads are sometimes seen to break, and their extremities then quickly become club-headed. The other sketches in Fig. 8 show the forms successively assumed. Shortly after the purple fluid within the cells has become aggregated, the little masses float about in a colorless or almost colorless fluid, and the layer of white granular protoplasm, which flows along the walls, can now be seen much more distinctly. The stream flows at an irregular rate up one wall, and down the opposite one, generally at a slower rate across the narrow ends of the elongated cells. and so round and round, but the current sometimes ceases. The movement is often in waves, and their crests sometimes stretch almost across the whole width of the cell, and then sink down again. Small spheres of protoplasm, apparently quite free, are often driven by the current round. The cells and filaments attached to the central masses are swayed to and fro, as if struggling to escape altogether. One of these cells, with the ever-changing central masses, and with the layer of protoplasm flowing round the walls, presents a wonderful scene of vital activity. Many observations were made on the contents of the cells whilst undergoing the process of aggregation, but I shall detail only a few cases under different heads. A small portion of a leaf was cut off, placed under a high power, and the glands, very gently pressed under a compressor, in 15 Roman 1000 I, distinctly saw extremely minute spheres of protoplasm aggregating themselves in the purple fluid. These rapidly increased in size both within the cells of the glands and of the upper ends of the pedicels. Particles of glass, cork, and cinders were also placed on the glands of many tentacles. In 1 several of them were inflected, but after 1 35 Roman 1000, there was no aggregation. Other tentacles with these particles were examined after 8 HRS and page 43. Now all their cells had undergone aggregation, so had the cells of the exterior tentacles, which had become inflected through the irritation transmitted from the glands of the disc, on which the transported particles rested. This was likewise the case with the short tentacles round the margins of the disc, which had not as yet become inflected. This latter fact shows that the process of aggregation is independent of the inflection of the tentacles, of which indeed we have other and abundant evidence. Again, the exterior tentacles on 3 leaves were carefully examined and found to contain only homogeneous purple fluid. Little bits of thread were then placed on the glands of 3 of them, and after 22 HRS, the purple fluid in their cells, almost down to their bases, was aggregated into innumerable spherical, elongated, or filamentous masses of protoplasm. The bits of thread had been carried some time previously to the central disc, and this had caused all the other tentacles to become somewhat inflected, and their cells had likewise undergone aggregation, which, however, it should be observed, had not as yet extended down to their bases, but was confined to the cells close beneath the glands. Not only do repeated touches on the glands and the contact of minute particles cause aggregation, but if glands, without being themselves injured, are cut off from the summits of the pedicels, this induces a moderate amount of aggregation in the headless tentacles. After they have become inflected, on the other hand, if glands are suddenly crushed between pincers, as was tried in 6 cases, the tentacles seem paralyzed by so great a shock, for they neither become inflected nor exhibit any signs of aggregation. Carbonate of ammonia, of all the causes inducing aggregation, that which, as far as I have seen, acts the quickest and is the most powerful, is a solution of carbonate of ammonia. Whatever its strength may be, the glands are always affected first, and soon become quite opaque, so as to appear black. For instance, I placed a leaf in a few drops of a strong solution, namely of 1 part to 146 of water, or 3 GRS to 1 Rs, and observed it under a high power. All the glands began to. Judging from an account of Roman 1000 Haeckel's observations, which I have only just seen quoted in the Gardener's Chronicle, Oct. 10, 1874, he appears to have observed a similar phenomenon in the stamens of Berberis after they have been excited by a touch, and have moved, for he says, the contents of each individual cell are collected together in the center of the cavity, page 44. Darken in 10s seconds, and in 13s, were conspicuously darker. In 1 Roman 1000, extremely small spherical masses of protoplasm could be seen arising in the cells of the pedicels close beneath the glands, as well as in the cushions on which the long-headed marginal glands rest. In several cases, the process traveled down the pedicels for a length twice or thrice as great as that of the glands. In about 10 Roman 1000, it was interesting to observe the process momentarily arrested at each transverse partition between 2 cells, and then to see the transparent contents of the cell next below almost flashing into a cloudy mass in the lower part of the pedicels. The action proceeded slower, so that it took about 20 Roman 1000 before the cells, halfway down the long marginal and submarginal tentacles, became aggregated. We may infer that the carbonate of ammonia is absorbed by the glands not only from its action being so rapid, but from its effect being somewhat different from that of other salts, as the glands, when excited, secrete an acid belonging to the acetic series. The carbonate is probably at once converted into a salt of this series, and we shall presently see that the acetate of ammonia causes aggregation almost or quite as energetically as does the carbonate if a few drops of a solution of one part of the carbonate to 437 of water or 1 gr to 1 Rs be added to the purple fluid which exudes from crushed tentacles or to paper stained by being rubbed with them, the fluid and the paper are changed into a pale dirty green. Nevertheless, some purple color could still be detected after 1 hr 30 Roman 1000 within the glands of a leaf. Left in a solution of twice the above strength, viz. 2 grs to 1 Rs, and after 24 hrs, the cells of the pedicels close beneath the glands still contained spheres of protoplasm of a fine purple tint. These facts show that the ammonia had not entered as a carbonate, for otherwise the color would have been discharged. I have, however, sometimes observed, especially with the long-headed tentacles on the margins of very pale leaves, immersed in a solution, that the glands, as well as the upper cells of the pedicels, were discolored, and in these cases, I presume that the unchanged carbonate had been absorbed. The appearance above described of the aggregating process being arrested for a short time at each transverse partition impresses the mind with the idea of matter passing downwards from cell to cell, but as the cells one beneath the other undergo aggregation when inorganic and insoluble particles are placed on the glands, the process must be, at least in these cases, one of molecular change transmitted from the glands, page 45, independently of the absorption of any matter. So it may possibly be in the case of the carbonate of ammonia, as however the aggregation caused by this salt travels down the tentacles at a quicker rate than when insoluble particles are placed on the glands, it is probable that ammonia in some form is absorbed not only by the glands, but passes down the tentacles. Having examined a leaf in water and found the contents of the cells homogeneous, I placed it in a few drops of a solution of one part of the carbonate to 437 of water, and attended to the cells immediately beneath the glands, but did not use a very high power. No aggregation was visible in 3 Roman 1000, but after 15 Roman 1000 small spheres of protoplasm were formed, more especially beneath the long-headed marginal glands. The process, however, in this case, took place with unusual slowness. In 25 Roman 1000, conspicuous spherical masses were present in the cells of the pedicels for a length about equal to that of the glands, and in 3 hrs to that of a third or half of the whole tentacle. If tentacles with cells containing only very pale pink fluid and apparently but little protoplasm are placed in a few drops of a weak solution of one part of the carbonate to 4375 of water 1 gr to 10 Rs, and the highly transparent cells beneath the glands are carefully observed under a high power, these may be seen first to become slightly cloudy from the formation of numberless, only just perceptible granules, which rapidly grow larger either from coalescence or from attracting more protoplasm from the surrounding fluid. On one occasion, I chose a singularly pale leaf and gave it, whilst under the microscope, a single drop of a stronger solution of one part to 437 of water. In this case, the contents of the cells did not become cloudy, but after 10 Roman 1000 minute, irregular granules of protoplasm could be detected, which soon increased into irregular masses and globules of a greenish or very pale purple tint. But these never formed perfect spheres, though incessantly changing their shapes and positions. With moderately red leaves, the first effect of a solution of the carbonate, generally, is the formation of 2 or 3 or of several extremely minute purple spheres, which rapidly increase in size. To give an idea of the rate at which such spheres increase in size, I may mention that a rather pale purple leaf placed under a slip of glass was given a drop of a solution of 1 part to 292 of water, and in 13 Roman 1000, a few minute spheres of protoplasm were formed. One of these, after 2 hrs 30 Roman 1000, was about 2/3 of the diameter of the cell. After 4 HRS 25 Roman 1000 page 46, it nearly equaled the cell in diameter, and a second sphere, about half as large as the first together, with a few other minute ones, were formed after 6 HRS. The fluid in which these spheres floated was almost colorless after 8 HRS 35 Roman 1000, always reckoning from the time when the solution was first added, for new minute spheres had appeared next morning. After 22 HRS, there were, besides the 2 large spheres, 7 smaller ones floating in absolutely colorless fluid in which some flocculent greenish matter was suspended. At the commencement of the process of aggregation, more especially in dark red leaves, the contents of the cells often present a different appearance, as if the layer of protoplasm, primordial utricle, which lines the cells had separated itself and shrunk from the walls, an irregularly shaped purple bag, being thus formed other fluids besides a solution of the carbonate. For instance, an infusion of raw meat produced this same effect, but the appearance of the primordial utricle shrinking from the walls is certainly false, for before giving the solution I saw on several occasions that the walls were lined with colorless flowing protoplasm, and after the bag-like masses were formed, the protoplasm was still flowing along the walls in a conspicuous manner, even more so than before. It appeared, indeed, as if the stream of protoplasm was strengthened by the action of the carbonate, but it was impossible to ascertain whether this was really the case. The bag-like masses, when once formed, soon begin to glide slowly round the cells, sometimes sending out projections, which separate into little spheres. Other spheres appear in the fluid surrounding the bags, and these travel much more quickly. That the small spheres are separate is often shown by sometimes one and then another traveling in advance, and sometimes they revolve round each other. I have occasionally seen spheres of this kind proceeding up and down the same side of a cell, instead of round it. The bag-like masses, after a time, generally divide into 2 rounded or oval masses, and these undergo the changes shown in Figs 7 and 8. At other times, spheres appear within the bags, and these coalesce and separate in an endless cycle of change. After leaves have been left for several hours in a solution of the carbonate and complete aggregation has been effected, with other plants, I have often seen what appears to be a true shrinking of the primordial utricle from the walls of the cells, caused by a solution of carbonate of ammonia, as likewise follows from Mechanical Injuries, page 47. Stream of protoplasm on the walls of the cells ceases to be visible. I observed this fact repeatedly, but will give only one instance. A pale purple leaf was placed in a few drops of a solution of 1 part to 292 of water, and in 2 HRS, some fine purple spheres were formed in the upper cells of the pedicels, the stream of protoplasm round their walls being still quite distinct, but after an additional 4 HRS, during which time many more spheres were formed, the stream was no longer distinguishable on the most careful examination, and this no doubt was due to the contained granules having become united with the spheres, so that nothing was left by which the movement of the limpid protoplasm could be perceived, but minute-free spheres still traveled up and down the cells, showing that there was still a current. So it was next morning, after 22 HRS, by which time some new minute spheres had been formed. These oscillated from side to side and changed their positions, proving that the current had not ceased, though no stream of protoplasm was visible on another occasion. However, a stream was seen flowing round the cell walls of a vigorous dark-colored leaf, after it had been left for 24 HRS in a rather stronger solution, namely of 1 part of the carbonate to 218 of water. This leaf, therefore, was not much or at all injured by an immersion for this length of time in the above solution of 2 grains to the ounce, and on being afterwards left for 24 HRS in water, the aggregated masses in many of the cells were redissolved in the same manner as occurs with leaves in a state of nature when they re-expand after having caught insects. In a leaf which had been left for 22 HRS in a solution of 1 part of the carbonate to 292 of water, some spheres of protoplasm formed by the self-division of a bag-like mass were gently pressed beneath a covering glass, and then examined under a high power. They were now distinctly divided by well-defined radiating fissures, or were broken up into separate fragments with sharp edges, and they were solid to the center in the larger broken spheres. The central part was more opaque, darker colored, and less brittle than the exterior, the latter alone being in some cases penetrated by the fissures in many of the spheres. The line of separation between the outer and inner parts was tolerably well defined. The outer parts were of exactly the same very pale purple tint as that of the last formed smaller spheres, and these latter did not include any darker central core. From these several facts, we may conclude that when vigorous dark-colored leaves are subjected to the action of carbonate of page 48 ammonia, the fluid within the cells of the tentacles often aggregates exteriorly into coherent viscid matter, forming a kind of bag. Small spheres sometimes appear within this bag, and the whole generally soon divides into two or more spheres, which repeatedly coalesce and redivide after a longer or shorter time. The granules in the colorless layer of protoplasm, which flows round the walls, are drawn to and unite with the larger spheres or form small independent spheres, these latter being of a much paler color and more brittle than the first aggregated masses after the granules of protoplasm have been thus attracted. The layer of flowing protoplasm can no longer be distinguished, though a current of limpid fluid still flows round the walls. If a leaf is immersed in a very strong, almost concentrated solution of carbonate of ammonia, the glands are instantly blackened, and they secrete copiously, but no movement of the tentacles ensues. Two leaves thus treated became after 1 hr flaccid, and seemed killed. All the cells in their tentacles contained spheres of protoplasm, but these were small and discolored. Two other leaves were placed in a solution not quite so strong, and there was well-marked aggregation. In 30 Roman 1000 after 24 HRS, the spherical or more commonly oblong masses of protoplasm became opaque and granular instead of being as usual translucent, and in the lower cells there were only innumerable minute spherical granules. It was evident that the strength of the solution had interfered with the completion of the process, as we shall see likewise follows from too great heat. All the foregoing observations relate to the exterior tentacles, which are of a purple color, but the green pedicels of the short central tentacles are acted on by the carbonate, and by an infusion of raw meat in exactly the same manner, with the sole difference that the aggregated masses are of a greenish color, so that the process is in no way dependent on the color of the fluid within the cells. Finally, the most remarkable fact with respect to this salt is the extraordinary small amount which suffices to cause aggregation. Full details will be given in the 7th chapter, and here it will be enough to say that with a sensitive leaf, the absorption by a gland of 1,134,400 of a grain 482 mgr is enough to cause, in the course of 1 hour, well-marked aggregation in the cells immediately beneath the gland. The effects of certain other salts and fluids. 2 leaves were placed in a solution of 1 part of acetate of ammonia to about page 49, 146 of water, and were acted on quite as energetically, but perhaps not quite so quickly as by the carbonate. After 10 Roman 1000, the glands were black, and in the cells beneath them there were traces of aggregation, which after 15 Roman 1000 was well marked, extending down the tentacles for a length equal to that of the glands. After 2 HRS, the contents of almost all the cells in all the tentacles were broken up into masses of protoplasm. A leaf was immersed in a solution of 1 part of oxalate of ammonia to 146 of water, and after 24 Roman 1000 some, but not a conspicuous change, could be seen. Within the cells beneath the glands, after 47 Roman 1000, plenty of spherical masses of protoplasm were formed, and these extended down the tentacles for about the length of the glands. This salt, therefore, does not act so quickly as the carbonate. With respect to the citrate of ammonia, a leaf was placed in a little solution of the above strength, and there was not even a trace of aggregation in the cells beneath the glands until 56 Roman 1000 had elapsed, but it was well marked after 2 HRS 20 Roman 1000. On another occasion, a leaf was placed in a stronger solution of 1 part of the citrate to 109 of water 4 GRS to 1 Rs, and at the same time another leaf in a solution of the carbonate of the same strength. The glands of the latter were blackened in less than 2 Roman 1000, and after 1 HR 45 Roman 1000, the aggregated masses, which were spherical and very dark-colored, extended down all the tentacles for between half and 2/3 of their lengths, whereas in the leaf immersed in the citrate. The glands, after 30 Roman 1000, were of a dark red, and the aggregated masses in the cells beneath them pink, and elongated, after 1 HR 45 Roman 1000. These masses extended down for only about 1/5 or 1/4 of the length of the tentacles. 2 leaves were placed each in 10 minims of a solution of 1 part of nitrate of ammonia to 5250 of water 1 gr to 12 Rs, so that each leaf received 1576 of a grain 1124 mgr. This quantity caused all the tentacles to be inflected, but after 24 HRS there was only a trace of aggregation. One of these same leaves was then placed in a weak solution of the carbonate, and after 1 HR 45 Roman 1000, the tentacles for half their lengths showed an astonishing degree of aggregation. 2 other leaves were then placed in a much stronger solution of 1 part of the nitrate to 146 of water 3 GRS to 1 Rs. In one of these, there was no marked change after 3 HRS, but in the other, there was a trace of aggregation after 52 Roman 1000. And this was plainly marked after 1 HR 22 Roman 1000. But even after 2 HRS 12 Roman 1000, there was certainly not more aggregation than would have fall page 50 load from an immersion of from 5 Roman 1000 to 10 Roman 1000 in an equally strong solution of the carbonate. Lastly, a leaf was placed in 30 minims of a solution of 1 part of phosphate of ammonia to 43,750 of water 1 gr to 100 Rs, so that it received 11,600 of a grain 4,079 mgr. This soon caused the tentacles to be strongly inflected, and after 24 HRS, the contents of the cells were aggregated into oval and irregularly globular masses, with a conspicuous current of protoplasm flowing round the walls. But after so long an interval, aggregation would have ensued whatever had caused inflection. Only a few other salts besides those of ammonia were tried in relation to the process of aggregation. A leaf was placed in a solution of 1 part of chloride of sodium to 218 of water, and after 1 HR, the contents of the cells were aggregated into small, irregularly globular brownish masses. These, after 2 HRS, were almost disintegrated and pulpy. It was evident that the protoplasm had been injuriously affected, and soon afterwards some of the cells appeared quite empty. These effects differ altogether from those produced by the several salts of ammonia, as well as by various organic fluids, and by inorganic particles placed on the glands. A solution of the same strength of carbonate of soda and carbonate of potash acted in nearly the same manner as the chloride, and here again, after 2 HRS 30 Roman 1000, the outer cells of some of the glands had emptied themselves of their brown pulpy contents. We shall see in the 8th chapter that solutions of several salts of soda of half the above strength cause inflection, but do not injure the leaves. Weak solutions of sulfate of quinine, of nicotine, camphor poison, of the cobra Roman 100 soon induce well-marked aggregation, whereas certain other substances, for instance, a solution of curare, have no such tendency. Many acids, though much diluted, are poisonous, and though, as will be shown in the 8th chapter, they cause the tentacles to bend, they do not excite true aggregation. Thus, leaves were placed in a solution of 1 part of benzoic acid to 437 of water, and in 15 Roman 1000 the purple fluid within the cells had shrunk a little from the walls, yet, when carefully examined, after 1 HR 20 Roman 1000, there was no true aggregation, and after 24 HRS, the leaf was evidently dead. Other leaves in iodic acid diluted to the same degree showed, after 2 HRS 15 Roman 1000, the same shrunken appearance of the purple fluid within the cells, and these after 6 HRS 15 Roman 1000 were seen under a high power to be filled with excessively minute spheres of dull reddish protoplasm, page 51, which, by the next morning, after 24 HRS, had almost disappeared, the leaf being evidently dead, nor was there any true aggregation in leaves immersed in propionic acid of the same strength. But in this case, the protoplasm was collected in irregular masses towards the bases of the lower cells of the tentacles. A filtered infusion of raw meat induces strong aggregation, but not very quickly in one leaf, thus immersed. There was a little aggregation after 1 HR 20 Roman 1000, and in another after 1 HR 50 Roman 1000. With other leaves, a considerably longer time was required. For instance, one immersed for 5 HRS showed no aggregation, but was plainly acted on in 5 Roman 1000, when placed in a few drops of a solution of 1 part of carbonate of ammonia to 146 of water. Some leaves were left in the infusion for 24 HRS, and these became aggregated to a wonderful degree, so that the inflected tentacles presented to the naked eye plainly mottled appearance. The little masses of purple protoplasm were generally oval or beaded, and not nearly so often spherical as in the case of leaves subjected to carbonate of ammonia. They underwent incessant changes of form, and the current of colorless protoplasm round the walls was conspicuously plain. After an immersion of 25 HRS, raw meat is too powerful a stimulant, and even small bits generally injure and sometimes kill the leaves to which they are given. The aggregated masses of protoplasm become dingy or almost colorless, and present an unusual granular appearance, as is likewise the case with leaves which have been immersed in a very strong solution of carbonate of ammonia. A leaf placed in milk had the contents of its cells somewhat aggregated in 1 HR. 2 other leaves, one immersed in human saliva for 2 HRS 30 Roman 1000, and another in unboiled white of egg for 1 HR 30 Roman 1000, were not action on in this manner, though they undoubtedly would have been so had more time been allowed. These same 2 leaves, on being afterwards placed in a solution of carbonate of ammonia 3 GRS to 1 Rs, had their cells aggregated, the one in 10 Roman 1000, and the other in 5 Roman 1000. Several leaves were left for 4 HRS 30 Roman 1000 in a solution of 1 part of white sugar to 146 of water, and no aggregation ensued. On being placed in a solution of this same strength of carbonate of ammonia, they were acted on in 5 Roman 1000, as was likewise a leaf, which had been left for 1 HR 45 Roman 1000, in a moderately thick solution of gum arabic. Several other leaves were immersed for some hours in denser solutions of sugar, gum, and starch, and they had the contents of their cells greatly aggregated. This page 52 effect may be attributed to exosmose, for the leaves in the syrup became quite flaccid, and those in the gum and starch somewhat flaccid, with their tentacles twisted about in the most irregular manner, the longer ones like corkscrews. We shall hereafter see that solutions of these substances, when placed on the discs of leaves, do not incite inflection. Particles of soft sugar were added to the secretion round several glands, and were soon dissolved, causing a great increase of the secretion, no doubt by exosmose. And after 24 HRS, the cells showed a certain amount of aggregation, though the tentacles were not inflected. Glycerine causes, in a few minutes, well-pronounced aggregation, commencing as usual within the glands, and then traveling down the tentacles. And this, I presume, may be attributed to the strong attraction of this substance, for water immersion for several hours in water causes some degree of aggregation. 20 leaves were first carefully examined and re-examined after having been left immersed in distilled water for various periods. With the following results, it is rare to find even a trace of aggregation until 4 or 5, and generally not until several more hours have elapsed, when, however, a leaf becomes quickly inflected in water, as sometimes happens especially during very warm weather. Aggregation may occur in little over 1 HR. In all cases, leaves left in water for more than 24 HRS have their glands blackened, which shows that their contents are aggregated, and in the specimens which were carefully examined, there was fairly well-marked aggregation in the upper cells of the pedicels. These trials were made with cut ophalaves, and it occurred to me that this circumstance might influence the result, as the footstalks would not perhaps absorb water quickly enough to supply the glands as they continued to secrete. But this view was proved erroneous, for a plant with uninjured roots bearing 4 leaves was submerged in distilled water for 47 HRS, and the glands were blackened. Though the tentacles were very little inflected in one of these leaves, there was only a slight degree of aggregation in the tentacles. In the second, rather more the purple contents of the cells being a little separated from the walls in the third and fourth, which were pale leaves, the aggregation in the upper. Parts of the pedicels was well marked in these leaves, the little masses of protoplasm, many of which were oval, slowly changed their forms and positions, so that a submergence for 47 HRS had not killed the protoplasm in a previous trial with a submerged plant. The tentacles were not, in the least, inflected. Page 53. Heat induces aggregation. A leaf, with the cells of the tentacles containing only homogeneous fluid, was waved about for 1 Roman 1000 in water at 130 Far 544 cent, and was then examined under the microscope as quickly as possible, that is, in 2 Roman 1000 or 3 Roman 1000, and by this time the contents of the cells had undergone some degree of aggregation. A second leaf was waved for 2 Roman 1000 in water at 125 516 cent, and quickly examined, as before the tentacles were well inflected. The purple fluid in all the cells had shrunk a little from the walls, and contained many oval and elongated masses of protoplasm, with few minute spheres. A third leaf was left in water at 125 until it cooled, and when examined after 1 HR 45 Roman 1000, the inflected tentacles showed some aggregation, which became, after 3 HRS, more strongly marked, but did not subsequently increase. Lastly, a leaf was waved for 1 Roman 1000 in water at 124.88 cent, and then left for 1 HR 26 Roman 1000 in cold water. The tentacles were but little inflected, and there was only here and there a trace of aggregation. In all these and other trials with warm water, the protoplasm showed much less tendency to aggregate into spherical masses than when excited by carbonate of ammonia. Redissolution of the aggregated masses of protoplasm as soon as tentacles, which have clasped an insect or any inorganic object, or have been in any way excited, have fully re-expanded, the aggregated masses of protoplasm are redissolved and disappear, the cells being now refilled with homogeneous purple fluid, as they were before the tentacles were inflected. The process of redissolution, in all cases, commences at the bases of the tentacles, and proceeds up them towards the glands in old leaves, however, especially in those which have been several times in action. The protoplasm in the uppermost cells of the pedicels remains in a permanently more or less aggregated condition. In order to observe the process of redissolution, the following observations were made. A leaf was left for 24 HRS in a little solution of 1 part of carbonate of ammonia to 218 of water, and the protoplasm was as usual aggregated into numberless purple spheres, which were incessantly changing their forms. The leaf was then washed and placed in distilled water, and after 3 HRS 15 Roman 1000, some few of the spheres began to show by their less clearly defined edges signs of redissolution. After 9 HRS, many of them had become elongated, and the surrounding fluid in the cells was slightly more colored, showing plainly that redissolution had commenced. After 24 HRS, though many cells still contained spheres here and there, page 54 could be seen filled with purple fluid without a vestige of aggregated protoplasm, the whole having been redissolved. A leaf with aggregated masses, caused by its having been waved for 2 Roman 1000 in water at the temperature of 125 Far, was left in cold water, and after 11 HRS, the protoplasm showed traces of incipient redissolution. When again examined 3 days after its immersion in the warm water, there was a conspicuous difference, though the protoplasm was still somewhat aggregated. Another leaf, with the contents of all the cells strongly aggregated from the action of a weak solution of phosphate of ammonia, was left for between 3 and 4 days in a mixture known to be innocuous of 1 dram of alcohol to 8 drams of water, and when re-examined, every trace of aggregation had disappeared, the cells being now filled with homogeneous fluid. We have seen that leaves immersed for some hours in dense solutions of sugar, gum, and starch have the contents of their cells greatly aggregated and are rendered more or less flaccid, with the tentacles irregularly contorted. These leaves, after being left for 4 days in distilled water, became less flaccid, with their tentacles partially re-expanded, and the aggregated masses of protoplasm were partially redissolved a leaf with its tentacles closely clasped over a fly, and with the contents of the cells strongly aggregated, was placed in a little sherry wine. After 2 HRS, several of the tentacles had re-expanded, and the others could by a mere touch be pushed back into their properly expanded positions, and now all traces of aggregation had disappeared, the cells being filled with perfectly homogeneous pink fluid. The redissolution in these cases may, I presume, be attributed to endosmosis. On the proximate causes of the process of aggregation. As most of the stimulants, which cause the inflection of the tentacles, likewise induce aggregation in the contents of their cells, this latter process might be thought to be the direct result of inflection. But this is not the case if leaves are placed in rather strong solutions of carbonate of ammonia, for instance of 3 or 4, and even sometimes of only 2 grains to the ounce of water. Roman 1, E1 part to 109 or 146, or page 55, 218 of. Water. The tentacles are paralyzed and do not become inflected, yet they soon exhibit strongly marked aggregation. Moreover, the short central tentacles of a leaf, which has been immersed in a weak solution of any salt of ammonia or in any nitrogenous organic fluid, do not become in the least inflected. Nevertheless, they exhibit all the phenomena of aggregation. On the other hand, several acids cause strongly pronounced inflection, but no aggregation. It is an important fact that when an organic or inorganic object is placed on the glands of the disc, and the exterior tentacles are thus caused to bend inwards, not only is the secretion from the glands of the latter increased in quantity and rendered acid, but the contents of the cells of their pedicels become aggregated. The process always commences in the glands, although these have not as yet touched any object. Some force or influence must therefore be transmitted from the central glands to the exterior tentacles, first to near their bases, causing this part to bend, and next to the glands, causing them to secrete more copiously. After a short time, the glands thus indirectly excited transmit or reflect some influence down their own pedicels, inducing aggregation in cell beneath cell to their bases. It seems at first sight a probable view that aggregation is due to the glands being excited to secrete more copiously, so that sufficient fluid is not left in their cells, and in the cells of the pedicels to hold the protoplasm in solution. In favor of this view is the fact that aggregation follows the inflection of the tentacles, and during the movement the glands generally, or as I believe always, secrete more copiously than they did before. Again, during the re-expansion. Page 56 of the tentacles, the glands secrete less freely or quite cease to secrete, and the aggregated masses of protoplasm are then redissolved. Moreover, when leaves are immersed in dense vegetable solutions, or in glycerine, the fluid within the gland cells passes outwards and there is aggregation, and when the leaves are afterwards immersed in water or in an innocuous fluid of less specific gravity than water, the protoplasm is redissolved, and this no doubt is due to endosmosis. Opposed to this view that aggregation is caused by the outward passage of fluid from the cells are the following facts. There seems no close relation between the degree of increased secretion and that of aggregation. Thus, a particle of sugar added to the secretion round a gland causes a much greater increase of secretion and much less aggregation than does a particle of carbonate of ammonia given in the same manner. It does not appear probable that pure water would cause much exosmosis, and yet aggregation often follows from an immersion in water of between 16 HRS and 24 HRS, and always after from 24 HRS to 48 HRS. Still less probable is it that water at a temperature of from 125 to 130 far 516 to 544 cent should cause fluid to pass not only from the glands, but from all the cells of the tentacles down to their bases, so quickly that aggregation is induced within 2 Roman 1000 or 3 Roman 1000. Another strong argument against this view is that after complete aggregation, the spheres and oval masses of protoplasm float about in an abundant supply of thin colorless fluid, so that at least the latter stages of the process cannot be due to the want of fluid to hold the protoplasm in solution. There is still stronger evidence that aggregation is independent of secretion, for the papillae described in the first chapter, with which the page 57 leaves are studded, are not glandular and do not secrete, yet they rapidly absorb carbonate of ammonia or an infusion of raw meat, and their contents then quickly undergo aggregation. which afterwards spreads into the cells of the surrounding tissues, we shall hereafter see that the purple fluid within the sensitive filaments of Dionea, which do not secrete likewise, undergoes aggregation from the action of a weak solution of carbonate of ammonia. The process of aggregation is a vital one, by which I mean that the contents of the cells must be alive and uninjured to be thus affected, and they must be in an oxygenated condition for the transmission of the process at the proper rate. Some tentacles in a drop of water were strongly pressed beneath a slip of glass. Many of the cells were ruptured, and pulpy matter of purple color with granules of all sizes and shapes exuded, but hardly any of the cells were completely emptied. I then added a minute drop of a solution of 1 part of carbonate of ammonia to 109 of water, and after 1 hr, examined the specimens. Here and there a few cells both in the glands and in the pedicels had escaped being ruptured, and their contents were well aggregated into spheres which were constantly changing their forms and positions, and a current could still be seen flowing along the walls, so that the protoplasm was alive. On the other hand, the exuded matter, which was now almost colorless instead of being purple, did not exhibit a trace of aggregation, nor was there a trace in the many cells which were ruptured, but which had not been completely emptied of their contents. Though I looked carefully, no signs of a current could be seen within these ruptured cells. They had evidently been killed by the pressure, and the matter which they, page 58, still contained did not undergo aggregation any more than that which had exuded in these specimens. As I may add, the individuality of the life of each cell was well illustrated. A full account will be given in the next chapter of the effects of heat on the leaves, and I need here only state that leaves immersed for a short time in water at a temperature of 124 488 cent, which, as we have seen, does not immediately induce aggregation, were then placed in a few drops of a strong solution of 1 part of carbonate of ammonia to 109 of water, and became finely aggregated. On the other hand, leaves, after an immersion in water at 156-155 cent, on being placed in the same strong solution, did not undergo aggregation, the cells becoming filled with brownish pulpy or muddy matter, with leaves subjected to temperatures between these two extremes of 120 and 154 488 and 655 cent. There were gradations in the completeness of the process, the former temperature not preventing aggregation from the subsequent action of carbonate of ammonia, the latter quite stopping it. Thus leaves immersed in water heated to 135-144 cent, and then in the solution formed perfectly defined spheres, but these were decidedly smaller than in ordinary cases, with other leaves heated to 140-60 cent. The spheres were extremely small, yet well defined, but many of the cells contained, in addition, some brownish pulpy matter. In 2 cases of leaves heated to 145-627 cent, a few tentacles could be found, with some of their cells containing a few minute spheres, whilst the other cells and other whole tentacles included only the brownish disintegrated or pulpy matter. The fluid within the cells of the tentacles must be in an oxygenated condition in order that the force, or page 59, influence, which induces aggregation, should be transmitted at the proper rate from cell to cell. A plant with its roots in water was left for 45 Roman 1000 in a vessel containing 122 Rs of carbonic acid. A leaf from this plant, and for comparison, one from a fresh plant, were both immersed for 1 hr in a rather strong solution of carbonate of ammonia. They were then compared, and certainly there was much less aggregation in the leaf, which had been subjected to the carbonic acid, than in the other. Another plant was exposed in the same vessel for 2 hrs to carbonic acid, and one of its leaves was then placed in a solution of 1 part of the carbonate to 437 of water. The glands were instantly blackened, showing that they had absorbed and that their contents were aggregated, but in the cells close beneath the glands, there was no aggregation even after an interval of 3 hrs. After 4 hrs 15 Roman 1000, a few minute spheres of protoplasm were formed in these cells, but even after 5 hrs 30 Roman 1000, the aggregation did not extend down the pedicels for a length equal to that of the glands. After numberless trials with fresh leaves immersed in a solution of this strength, I have never seen the aggregating action transmitted at nearly so slow a rate. Another plant was left for 2 hrs in carbonic acid, but was then exposed for 20 Roman 1000 to the open air, during which time the leaves, being of a red color, would have absorbed some oxygen. One of them, as well as a fresh leaf for comparison, were now immersed in the same solution, as before the former were looked at repeatedly, and after an interval of 65 Roman 1000 a few spheres of protoplasm were first observed in the cells close beneath the glands, but only in 2 or 3 of the longer tentacles. After 3 HRS, the aggregation had traveled down the pedicels of a few of the tentacles, page 60, for a length equal to that of the glands. On the other hand, in the fresh leaf, similarly treated aggregation was plain in many of the tentacles after 15 Roman 1000. After 65 Roman 1000, it had extended down the pedicels for 4, 5 or more times the lengths of the glands, and after 3 HRS, the cells of all the tentacles were affected for 1/3 or 1/2 of their entire lengths. Hence, there can be no doubt that the exposure of leaves to carbonic acid either stops for a time the process of aggregation or checks the transmission of the proper influence when the glands are subsequently excited by carbonate of ammonia, and this substance acts more promptly and energetically than any other. It is known that the protoplasm of plants exhibits its spontaneous movements only as long as it is in an oxygenated condition, and so it is with the white corpuscles of the blood only as long as they receive oxygen from the red corpuscles. But the cases above given are somewhat different as they relate to the delay in the generation or aggregation of the masses of protoplasm by the exclusion of oxygen. Summary and concluding remarks: the process of aggregation is independent of the inflection of the tentacles and of increased secretion from the glands. It commences within the glands, whether these have been directly excited or indirectly, by a stimulus received from other glands. In both cases, the process is transmitted from cell to cell down the whole length of the tentacles, being arrested for a short time at each transverse partition, with pale-colored leaves the first change, which is perceptible, but only with respect to plants. Sachs, Trait, Debat, 3rd edit, 1874, p. 864, on blood corpuscles, see Quarterly Journal of Microscopical Science, April 1874, p. 185, page 61. Under a high power is the appearance of the finest granules in the fluid within the cells, making it slightly cloudy. These granules soon aggregate into small globular masses. I have seen a cloud of this kind appear in 10s after a drop of a solution of carbonate of ammonia had been given to a gland with dark red leaves. The first visible change often is the conversion of the outer layer of the fluid within the cells into bag-like masses. The aggregated masses, however they may have been developed incessantly, change their forms and positions. They are not filled with fluid, but are solid to their centers. Ultimately, the colorless granules in the protoplasm, which flows round the walls, coalesce with the central spheres or masses, but there is still a current of limpid fluid flowing within the cells. As soon as the tentacles fully re-expand, aggregated masses are redissolved, and the cells become filled with homogeneous purple fluid. As they were at first, the process of redissolution commences at the bases of the tentacles, thence proceeding upwards to the glands, and therefore in a reverse direction to that of aggregation. Aggregation is excited by the most diversified causes, the glands being several times touched by the pressure of particles of any kind, and as these are supported by the dense secretion, they can hardly press on the glands with the weight of a millionth of a grain, the tentacles being cut off close beneath. According to Hofmeister, as quoted by Sachs, Trait, Debat, 1874, p. 958, very slight pressure on the cell membrane arrests immediately the movements of the protoplasm, and even determines its separation from the walls, but the process of aggregation is a different phenomenon as it relates to the contents of the cells, and only secondarily to the layer of protoplasm, which flows along the walls, though no doubt the effects of pressure or of a touch on the outside must be transmitted through this layer, page 62. The glands absorbing various fluids or matter dissolved out of certain bodies be exosmos, and by a certain degree of heat. On the other hand, a temperature of about 150 Fahr 655 cent does not excite aggregation, nor does the sudden crushing of a gland. If a cell is ruptured, neither the exuded matter nor that which still remains within the cell undergoes aggregation, when carbonate of ammonia is added a very strong solution of this salt, and rather large bits of raw meat prevent the aggregated masses being well developed. From these facts, we may conclude that the protoplasmic fluid within a cell does not become aggregated unless it be in a living state, and only imperfectly if the cell has been injured. We have also seen that the fluid must be in an oxygenated state in order that the process of aggregation should travel from cell to cell at the proper rate. Various nitrogenous organic fluids and salts of ammonia induce aggregation, but in different degrees and at very different rates. Carbonate of ammonia is the most powerful of all known substances. The absorption of 1,134,400 of a grain 482 mg by a gland suffices to cause all the cells of the same tentacle to become aggregated. The first effect of the carbonate and of certain other salts of ammonia, as well as of some other fluids, is the darkening or blackening of the glands. This follows even from long immersion in cold distilled water. It apparently depends in chief part on the strong aggregation of their cell contents, which thus become opaque and do not reflect light. Some other fluids render the glands of a brighter red, while certain acids, though much diluted, the poison of the cobra snake, Roman 100, make the glands perfectly white and opaque, and this seems to depend on the coagulation of their contents. Without page 63 any aggregation, nevertheless, before being thus affected, they are able, at least in some cases, to excite aggregation in their own tentacles. That the central glands, if irritated, send centrifugally some influence to the exterior glands, causing them to send back a centripetal influence, inducing aggregation, is perhaps the most interesting fact given in this chapter. But the whole process of aggregation is in itself a striking phenomenon. Whenever the peripheral extremity of a nerve is touched or pressed, and a sensation is felt, it is believed that an invisible molecular change is sent from one end of the nerve to the other. But when a gland of Drosera is repeatedly touched or gently pressed, we can actually see a molecular change proceeding from the gland down the tentacle, though this change is probably of a very different nature from that in a nerve. Finally, as so many and such widely different causes excite aggregation, it would appear that the living matter within the gland cells is in so unstable a condition that almost any disturbance suffices to change its molecular nature, as in the case of certain chemical compounds, and this change in the glands, whether excited directly or indirectly by a stimulus received from other glands, is transmitted from cell to cell, causing granules of protoplasm either to be actually generated in the previously limpid fluid or to coalesce, and thus to become visible. Supplementary observations on the process of aggregation in the roots of plants. It will hereafter be seen that a weak solution of the carbonate of ammonia induces aggregation in the cells of the roots of Drosera, and this led me to make a few trials on the roots of other plants I dug up in the latter part of October. The first weed which I met with, viz. Euphorbia peplis, being care page 64 full, not to injure the roots. These were washed and placed in a little solution of 1 part of carbonate of ammonia to 146 of water. In less than 1 minute, I saw a cloud traveling from cell to cell up the roots with wonderful rapidity. After from 8 Roman 1000 to 9 Roman 1000, the fine granules which caused this cloudy appearance became aggregated towards the extremities of the roots into quadrangular masses of brown matter, and some of these soon changed their forms and became spherical. Some of the cells, however, remained unaffected. I repeated the experiment with another plant of the same species, but before I could get the specimen into focus, under the microscope clouds of granules and quadrangular masses of reddish and brown matter were formed, and had run far up all the roots. A fresh root was now left for 18 HRS in a dram of a solution of 1 part of the carbonate to 437 of water, so that it received 18 of a grain or 2 mg. Examined the cells of all the roots throughout their whole length, contained aggregated masses of reddish and brown matter. Before making these experiments, several roots were closely examined, and not a trace of the cloudy appearance or of the granular masses could be seen in any of them. Roots were also immersed for 35 Roman 1000 in a solution of 1 part of carbonate of potash to 218 of water, but this salt produced no effect. I may here add that thin slices of the stem of the euphorbia were placed in the same solution, and the cells which were green instantly became cloudy, whilst others which were before colorless were clouded with brown owing to the formation of numerous granules of this tint. I have also seen with various kinds of leaves left for some time in a solution of carbonate of ammonia that the grains of chlorophyll ran together and partially coalesced, and this seems to be a form of aggregation. Plants of duckweed lemna were left for between 30 Roman 1000 and 45 Roman 1000 in a solution of 1 part of this same salt to 146 of water, and 3 of their roots were then examined. In 2 of them, all the cells, which had previously contained only limpid fluid, now included little green spheres. After from 112 HR to 2 HRS, similar spheres appeared in the cells on the borders of the leaves, but whether the ammonia had traveled up the roots or had been directly absorbed by the leaves, I cannot say, as one species Lemna oryhiza produces no roots. The latter alternative is perhaps the most probable. After about 212 HRS, some of the little green spheres in the roots were broken up into small granules, which exhibited Brownian movements. Some duckweed was also left for 1 HR 30 Roman 1000 in a solution of 1 part of page 65 carbonate of potash to 218 of water, and no decided change could be perceived in the cells of the roots. But when these same roots were placed for 25 Roman 1000 in a solution of carbonate of ammonia of the same strength, little green spheres were formed. A green marine alga was left for some time in this same solution, but was very doubtfully affected. On the other hand, a red marine alga with finely pinnated fronds was strongly affected. The contents of the cells aggregated themselves into broken rings, still of a red color, which very slowly and slightly changed their shapes, and the central spaces within these rings became cloudy with red granular matter. The facts here given, whether they are new, I know not, indicate that interesting results would perhaps be gained by observing the action of various saline solutions and other fluids on the roots of plants, page 66. Chapter Roman 4, the effects of heat on the leaves. Nature of the experiments. Effects of boiling water. Warm water causes rapid inflection. Water at a higher temperature does not cause immediate inflection, but does not kill the leaves as shown by their subsequent re-expansion, and by the aggregation of the protoplasm. A still higher temperature kills the leaves and coagulates the albuminous contents of the glands. In my observations on Drosera rotundifolia, the leaves seem to be more quickly inflected over animal substances, and to remain inflected for a longer period during very warm than during cold. Whether I wished therefore to ascertain whether heat alone would induce inflection, and what temperature was the most efficient. Another interesting point presented itself, namely at what degree life was extinguished. For Drosera offers unusual facilities in this respect, not in the loss of the power of inflection, but in that of subsequent re-expansion, and more especially in the failure of the protoplasm to become aggregated when the leaves, after being heated, are immersed in a solution of carbonate of ammonia. When my experiments on the effects of heat were made, I was not aware that the subject had been carefully investigated by several observers. For instance, Sachs is convinced, Trait de Botanique, 1874, pp. 772-854, that the most different kinds of plants all perish if kept for 10 Roman 1000 in water at 45 to 46 cent, or 113 to 115 far. And he concludes that the protoplasm within their cells always coagulates if in a damp condition at a temperature of between 50 and 60 cent, or 122 to 140 far. Max Schulze and Kuhn, as quoted by Dr. Bastian in Contempt Review 1874, p. 528, found that the protoplasm of plant cells with which they experimented was always killed, and page 67, altered by a very brief exposure to a temperature of 118-12 far as a maximum, as my results are deduced from special phenomena, namely the subsequent aggregation of the protoplasm and the re-expansion of the tentacles. They seem to me worth giving. We shall find that Drosera resists heat somewhat better than most other plants. That there should be considerable differences in this respect is not surprising, considering that some low vegetable organisms grow in hot springs, cases of which have been collected by Prof. Wyman, American Journal of Science, Vol. Roman 44. Thus Dr. Hooker found confervae in water at 168 far Humboldt at 185 far, and disclose at 208 far page 67. My experiments were tried in the following manner. Leaves were cut off, and this does not in the least interfere with their powers. For instance, 3 cut-off leaves, with bits of meat placed on them, were kept in a damp atmosphere, and after 23 HRS closely embraced the meat, both with their tentacles and blades, and the protoplasm within their cells was well aggregated. 3 ounces of doubly distilled water was heated in a porcelain vessel, with a delicate thermometer having a long bulb obliquely suspended in it. The water was gradually raised to the required temperature by a spirit lamp moved about under the vessel, and in all cases the leaves were continually waved for some minutes close to the bulb. They were then placed in cold water or in a solution of carbonate of ammonia. In other cases, they were left in the water, which had been raised to a certain temperature, until it cooled again. In other cases, the leaves were suddenly plunged into water of a certain temperature and kept there for a specified time. Considering that the tentacles are extremely delicate and that their coats are very thin, it seems scarcely possible that the fluid contents of their cells should not have been heated to within a degree or 2 of the temperature of the surrounding water. Any further precautions would, I think, have been superfluous, as the leaves from age or constitutional causes differ slightly in their sensitiveness to heat. It will be convenient first briefly to describe the effects of immersion for 30 seconds in boiling water. The leaves are rendered flaccid with their tentacles bowed backwards, which, as we shall see in a future chapter, is probably due to their outer surfaces retaining their elasticity for a longer period than their inner surfaces retain the power of contraction. The purple fluid within the cells of the pedicels is rendered finely granular, but there is no true aggregation, nor does this follow page 68, when the leaves are subsequently placed in a solution of carbonate of ammonia. But the most remarkable change is that the glands become opaque and uniformly white, and this may be attributed to the coagulation of their albuminous contents. My first and preliminary experiment consisted in putting 7 leaves in the same vessel of water and warming it slowly up to the temperature of 110 far 433, a leaf being taken out as soon as the temperature rose to 8266, another at 85, another at 90, and so on. Each leaf, when taken out, was placed in water at the temperature of my room, and the tentacles of all soon became slightly, though irregularly, inflected. They were now removed from the cold water and kept in damp air with bits of meat placed on their discs. The leaf, which had been exposed to the temperature of 110°, became, in 15 Roman 1000, greatly inflected, and in 2 HRS, every single tentacle closely embraced the meat. So it was, but after rather longer intervals with the 6 other leaves. It appears, therefore, that the warm bath had increased their sensitiveness when excited by meat. I next observed the degree of inflection which leaves underwent within stated periods while still immersed in warm water, kept as nearly as possible at the same temperature. But I will here and elsewhere give only a few of the many trials made. A leaf was left for 10 Roman 1000 in water at 377 but no inflection occurred. A second leaf, however, treated in the same manner, had a few of its exterior tentacles very slightly inflected in 6 Roman 1000 and several irregularly, but not closely inflected, in 10 Roman 1000. A third leaf, kept in water at 105 to 106, 405 to 411 was very moderately inflected in 6 Roman 1000. A fourth leaf in water at 110, 433 was somewhat inflected in 4 Roman 1000, and considerably so in from 6 to 7 Roman 1003. Leaves were placed in water which was heated rather quickly, and by the time the temperature rose to 115,116, 461 to 4606, all 3 were inflected. I then removed the lamp, and in a few minutes every single tentacle was closely inflected. The protoplasm within the cells was not killed, for it was seen to be in distinct movement, and the leaves, having been left in cold water for 20 HRS, re-expanded. Another leaf was immersed in water at 100,037, which was raised to 124,188, and all the tentacles except the extreme marginal ones soon became closely inflected. The leaf was now placed in cold water, and in 7 HRS 30 Roman 1000 it had partly, and in 10 HRS fully re-expanded on the following morning. It was immersed in a weak solution of carbonate of page 69 ammonia, and the glands quickly became black with strongly marked aggregation, tentacles showing that the protoplasm was alive, and that the glands had not lost their power of absorption. Another leaf was placed in water at 110,433 cent, which was raised to 120,488 cent, and every tentacle accepting one was quickly and closely inflected. This leaf was now immersed in a few drops of a strong solution of carbonate of ammonia 1 part to 109 of water in 10 Roman 1000. All the glands became intensely black, and in 2 HRS, the protoplasm in the cells of the petacells was well aggregated. Another leaf was suddenly plunged, and as usual waved about in water at 120, and the tentacles became inflected in from 2 Roman 1000 to 3 Roman 1000, but only so as to stand at right angles to the disc. The leaf was now placed in the same solution, viz. 1 part of carbonate of ammonia to 109 of water, or 4 GRS to 1 Rs, which I will for the future designate as the strong solution. And when I looked at it again, after the interval of an hour the glands were blackened, and there was well-marked aggregation. After an additional interval of 4 HRS, the tentacles had become much more inflected. It deserves notice that a solution as strong as this never causes inflection in ordinary cases. Lastly, a leaf was suddenly placed in water at 125,516 cent and was left in it until the water cooled. The tentacles were rendered of a bright red and soon became inflected. The contents of the cells underwent some degree of aggregation, which in the course of 3 hours increased, but the masses of protoplasm did not become spherical, as almost always occurs with leaves immersed in a solution of carbonate of ammonia. We learn from these cases that a temperature of from 120 to 125,488 to 516 cent excites the tentacles into quick movement, but does not kill the leaves as shown either by their subsequent re-expansion or by the aggregation of the protoplasm. We shall now see that a temperature of 130,544 cent is too high to cause immediate inflection, yet does not kill the leaves. Experiment 1. A leaf was plunged, and as in all cases waved about for a few minutes in water at 130,544 cent, but there was no trace of inflection. It was then placed in cold water, and after an interval of 15 Roman 1000 very slow movement was page 70 distinctly seen in a small mass of protoplasm in one of the cells of a tentacle. After a few hours, all the tentacles and the blade became inflected. Experiment 2. Another leaf was plunged into water at 130,0 to 131,0, and as before there was no inflection. After being kept in cold water for an hour it was placed in the strong solution of ammonia, and in the course of 55 Roman 1000 the tentacles were considerably inflected. The glands, which before had been rendered of a brighter red, were now blackened. The protoplasm in the cells of the tentacles was distinctly aggregated, but the spheres were much smaller than those generated in unheated leaves when subjected to carbonate of ammonia. After an additional 2 HRS, all the tentacles, excepting 6 or 7, were closely inflected. Experiment 3. A similar experiment to the last, with exactly the same results. Experiment 4. A fine leaf was placed in water at 103,377 cent, which was then raised to 145,627 cent. Soon after immersion, there was, as might have been expected, strong inflection. The leaf was now removed and left in cold water, but from having been exposed to so high a temperature, it never re-expanded. Experiment 5. Leaf immersed at 130,544 cent, and the water raised to 145,627 cent. There was no immediate inflection. It was then placed in cold water, and after 1 HR 20 Roman 1000, some of the tentacles on one side became inflected. This leaf was now placed in the strong solution, and in 40 Roman 1000, all the submarginal tentacles were well inflected and the glands blackened. After an additional interval of 2 HRS 45 Roman 1000, all the tentacles except 8 or 10 were closely inflected, with their cells exhibiting a slight degree of aggregation. But the spheres of protoplasm were very small, and the cells of the exterior tentacles contained some pulpy or disintegrated brownish matter. Experiments 6 and 7, 2 leaves were plunged in water at 135,572 cent, which was raised to 145,627 cent. Neither became inflected. One of these, however, after having been left for 31 Roman 1000 in cold water, exhibited some slight inflection, which increased after an additional interval of 1 HR 45 Roman 1000 until. Sachs states Trait de Botanique, 1874 p. 855, that the movements of the protoplasm in the hairs of a cucurbita ceased after they were exposed for 1 Roman 1000 in water to a temperature of 47 to 48 cent, or 117 to 119, far page 71. All the tentacles except 16 or 17 were more or less inflected, but the leaf was so much injured that it never re-expanded. The other leaf, after having been left for half an hour in cold water, was put into the strong solution, but no inflection ensued. The glands, however, were blackened, and in some cells there was a little aggregation, the spheres of protoplasm being extremely small. In other cells, especially in the exterior tentacles, there was much greenish-brown pulpy matter. Experiment 8, a leaf was plunged and waved about for a few minutes in water at 140-60 cent, and was then left for half an hour in cold water, but there was no inflection. It was now placed in the strong solution, and after 2 HRS 30 Roman 1000, the inner submarginal tentacles were well inflected, with their glands blackened, and some imperfect aggregation in the cells of the pedicels. 3 or 4 of the glands were spotted with the white porcelain-like structure like that produced by boiling water. I have seen this result in no other instance after an immersion of only a few minutes in water at so low a temperature as 140, and in only 1 leaf out of 4 after a similar immersion at a temperature of 145. Far on the other hand, with 2 leaves, one placed in water at 145,627 cent, and the other in water at 140-60 cent, both being left therein until the water cooled. The glands of both became white and porcelain-like, so that the duration of the immersion is an important element in the result. Experiment 9, a leaf was placed in water at 140-60 cent, which was raised to 150-655 cent. There was no inflection. On the contrary, the outer tentacles were somewhat bowed backwards. The glands became like porcelain, but some of them were a little mottled with purple. The bases of the glands were often more affected than their summits. This leaf, having been left in the strong solution, did not undergo any inflection or aggregation. Experiment 10, a leaf was plunged in water at 150 to 150-12-655 cent. It became somewhat flaccid, with the outer tentacles slightly reflexed, and the inner ones a little bent inwards, but only towards their tips, and this latter fact shows that the movement was not one of true inflection, as the basal part alone normally bends. The tentacles were as usual rendered of a very bright red, with the glands almost white like porcelain, yet tinged with pink. The leaf having been placed in the strong solution, the cell contents of the tentacles became of a muddy brown with no trace of aggregation, page 72. Experiment 11, a leaf was immersed in water at 145,627 cent, which was raised to 156,688 cent. The tentacles became bright red and somewhat reflexed, with almost all the glands, like porcelain, those on the disc being still pinkish, those near the margin quite white. The leaf being placed as usual first in cold water, and then in the strong solution, the cells in the tentacles became of a muddy greenish-brown, with the protoplasm not aggregated. Nevertheless, 4 of the glands escaped being rendered like porcelain, and the pedicels of these glands were spirally curled like a French horn towards their upper ends, but this can by no means be considered as a case of true inflection. The protoplasm within the cells of the twisted portions was aggregated into distinct, though excessively minute purple spheres. This case shows clearly that the protoplasm, after having been exposed to a high temperature for a few minutes, is capable of aggregation when afterwards subjected to the action of carbonate of ammonia, unless the heat has been sufficient to cause coagulation. Concluding remarks: as the hair-like tentacles are extremely thin and have delicate walls, and as the leaves were waved about for some minutes close to the bulb of the thermometer, it seems scarcely possible that they should not have been raised very nearly to the temperature which the instrument indicated. From the 11 last observations we see the temperature of 130–544 never causes the immediate inflection of the tentacles, though a temperature from 120 to 125–488 to 516 quickly produces this effect. But the leaves are paralyzed only for a time by a temperature of 130, as afterwards, whether left in simple water or in a solution of carbonate of ammonia, they become inflected, and their protoplasm undergoes aggregation. This great difference in the effects of a higher and lower temperature may be compared with that from immersion in strong and weak solutions of the salts of ammonia, for the former do not excite movement, whereas the latter act energetically. A temporary suspension of the page 73 power of movement due to heat is called by Sachs heat rigidity, and this, in the case of the sensitive eplant mimosa, is induced by its exposure for a few minutes to humid air raised to 120,122 FAR of 49 to 50 cent. It deserves notice that the leaves of Drosera, after being immersed in water at 130 FAR, are excited into movement by a solution of the carbonate so strong that it would paralyze ordinary leaves and cause no inflection. The exposure of the leaves for a few minutes even to a temperature of 145 FAR 627 cent does not always kill them, as when afterwards left in cold water or in a strong solution of carbonate of ammonia, they generally, though not always, become inflected, and the protoplasm within their cells undergoes aggregation, though the spheres thus formed are extremely small, with many of the cells partly filled with brownish muddy matter. In 2 instances, when leaves were immersed in water at a lower temperature than 130–544 cent, which was then raised to 145–627 cent, they became, during the earlier period of immersion, inflected, but on being afterwards left in cold water, were incapable of re-expansion. Exposure for a few minutes to a temperature of 145°, sometimes causes some few of the more sensitive glands to be speckled with the porcelain-like appearance, and on one occasion this occurred at temperature of 140–60 cent. On another occasion, when a leaf was placed in water at this temperature of only 140°, and left therein till the water cooled, every gland became like porcelain. Exposure for a few minutes to a temperature of 150–655 cent generally produces this effect, yet many glands retain. Trait de Bott, 1874, p. 1034, page 74. Pinkish color, and many present a speckled appearance. This high temperature never causes true inflection. On the contrary, the tentacles commonly become reflexed, though to a less degree than when immersed in boiling water, and this apparently is due to their passive power of elasticity. After exposure to a temperature of 150 FAR, the protoplasm, if subsequently subjected to carbonate of ammonia, instead of undergoing aggregation, is converted into disintegrated or pulpy discolored matter. In short, the leaves are generally killed by this degree of heat, but owing to differences of age or constitution, they vary somewhat in this respect. In one anomalous case, 4 out of the many glands on a leaf, which had been immersed in water raised to 156–688 cent, escaped being rendered porcelainous, and the protoplasm in the cells close beneath these glands underwent some slight, though imperfect, degree of aggregation. Finally, it is a remarkable fact that the leaves of Drosera rotundifolia, which flourishes on bleak upland moors throughout Great Britain and exists hooker within the Arctic Circle, should be able to withstand for even a short time immersion in water heated to a temperature of 145. It may be worth adding that immersion in cold, as the opacity and porcelain-like appearance of the glands is probably due to the coagulation of the albumin. I may add, on the authority of Dr. Burden Sanderson, that albumin coagulates at about 155°, but in presence of acids, the temperature of coagulation is lower. The leaves of Drosera contain an acid, and perhaps a difference in the amount contained may account for the slight differences in the results above recorded. It appears that cold-blooded animals are, as might have been expected, far more sensitive to an increase of temperature than is Drosera. Thus, as I hear from Dr. Burden Sanderson, a frog begins to be distressed in water at a temperature of only 85 far at 95, the muscles become rigid, and the animal dies in a stiffened condition, page 75. Water does not cause any inflection. I suddenly placed 4 leaves taken from plants, which had been kept for several days at a high temperature generally, about 75 far 238, sent in water at 45-72 cent. But they were hardly at all affected, not so much as some other leaves from the same plants, which were at the same time immersed in water at 75, for these became, in a slight degree, inflected, page 76. Chapter Roman 5. The effects of non-nitrogenous and nitrogenous organic fluids on the leaves. Non-nitrogenous fluid solutions of gum, Arabic sugar, starch diluted, alcohol, olive oil infusion, and decoction, of T-nitrogenous fluids, milk, urine, liquid, albumin infusion, of raw meat, impure mucus, saliva solution, of isinglass difference. In the action of these 2 sets of fluids, decoction of green peas, decoction, and infusion of cabbage, decoction of grass leaves. When in 1860 I first observed Drosera, and was led to believe that the leaves absorbed nutritious matter from the insects which they captured, it seemed to me a good plan to make some preliminary trials with a few common fluids containing and not containing nitrogenous matter, and the results are worth giving. In all the following cases, a drop was allowed to fall from the same pointed instrument on the center of the leaf, and by repeated trials, one of these drops was ascertained to be on an average very nearly half a minim, or 1960, of a fluid ounce, or 295 Roman 1050. But these measurements obviously do not pretend to any strict accuracy. Moreover, the drops of the viscid fluids were plainly larger than those of water only. 1 leaf on the same plant was tried, and the plants were collected from 2 distant localities. The experiments were made during August and September. In judging of the effects, 1 caution is necessary. If a drop of any adhesive fluid is placed on an old or feeble leaf, the glands of which have ceased to secrete copiously, the drop sometimes dries up, especially if the plant, page 77, is kept in a room, and some of the central and submarginal tentacles are thus drawn together, giving to them the false appearance of having become inflected. This sometimes occurs with water, as it is rendered adhesive by mingling with the viscid secretion. Hence the only safe criterion, and to this alone I have trusted, is the bending inwards of the exterior tentacles, which have not been touched by the fluid, or at most only at their bases. In this case, the movement is wholly due to the central glands having been stimulated by the fluid, and transmitting a motor impulse to the exterior tentacles. The blade of the leaf likewise often curves inwards in the same manner, as when an insect or bit of meat is placed on the disc. This latter movement is never caused, as far as I have seen, by the mere drying up of an adhesive fluid, and the consequent drawing together of the tentacles. First, for the non-nitrogenous fluids, as a preliminary trial, drops of distilled water were placed on between 30 and 40 leaves, and no effect whatever was produced. Nevertheless, in some other and rare cases, a few tentacles became for a short time inflected, but this may have been caused by the glands having been accidentally touched in getting the leaves into a proper position. That water should produce no effect might have been anticipated, as otherwise the leaves would have been excited into movement by every shower of rain. Gum, Arabic solutions of 4 degrees of strength were made, 1 of 6 grains to the ounce of water, 1 part to 73, a second rather stronger, yet very thin, a third moderately thick, and a fourth so thick that it would only just drop from a pointed instrument. These were tried on 14 leaves, the drops being left on the discs from 24 HRS to 44 HRS, generally about page 78. 30 HRS inflection was never thus caused. It is necessary to try pure gum. Arabic for a friend tried a solution bought ready prepared, and this caused the tentacles to bend, but he afterwards ascertained that it contained much animal matter, probably glue. Sugar drops of a solution of white sugar of 3 strengths, the weakest, containing 1 part of sugar to 73 of water, were left on 14 leaves from 32 HRS to 48 HRS, but no effect was produced. Starchy mixture, about as thick as cream, was dropped on 6 leaves and left on them for 30 HRS, no effect being produced. I am surprised at this fact, as I believe that the starch of commerce generally contains a trace of gluten, and this nitrogenous substance causes inflection, as we shall see in the next chapter. Alcohol diluted 1 part of alcohol was added to 7 of water, and the usual drops were placed on the discs of 3 leaves. No inflection ensued in the course of 48 HRS. To ascertain whether these leaves had been at all injured, bits of meat were placed on them, and after 24 HRS, they were closely inflected. I also put drops of sherry wine on 3 other leaves. No inflection was caused, though 2 of them seemed somewhat injured. We shall hereafter see that cut-off leaves immersed in diluted alcohol of the above strength do not become inflected. Olive oil drops were placed on the discs of 11 leaves, and no effect was produced in from 24 HRS to 48 HRS. 4 of these leaves were then tested by bits of meat on their discs, and 3 of them were found after 24 HRS with all their tentacles and blades closely inflected. Whilst the 4th had only a few tentacles inflected, it will however be shown in a future place that cut-off leaves immersed in olive oil are powerfully affected. Infusion and decoction of tea. Drops of a strong infusion and decoction, as well as of a rather weak decoction of tea, were placed on 10 leaves, none of which became inflected. I afterwards tested 3 of them by adding bits of meat to the drops, which still remained on their discs, and when I examined them after 24 HRS, they were closely inflected. The chemical principle of tea, namely theine, was subsequently tried and produced no effect. The albuminous matter, which the leaves must originally have contained, no doubt, had been rendered insoluble by their having been completely dried. We thus see that, excluding the experiments with water, 61 leaves were tried with drops of the page 79 above named non-nitrogenous fluids, and the tentacles were not, in a single case, inflected. With respect to nitrogenous fluids, the first which came to hand, were tried. The experiments were made at the same time, and in exactly the same manner as the foregoing, as it was immediately evident that these fluids produced a great effect. I neglected in most cases to record how soon the tentacles became inflected, but this always occurred in less than 24 HRS, whilst the drops of non-nitrogenous fluids, which produced no effect, were observed in every case during a considerably longer period. Milk drops were placed on 16 leaves, and the tentacles of all, as well as the blades of several, soon became greatly inflected. The periods were recorded in only 3 cases, namely with leaves on which unusually small drops had been placed. Their tentacles were somewhat inflected in 45 Roman 1000, and after 7 HRS 45 Roman 1000, the blades of 2 were so much curved inwards that they formed little cups enclosing the drops. These leaves re-expanded on the 3rd day. On another occasion, the blade of a leaf was much inflected in 5 HRS after a drop of milk had been placed on it. Human urine drops were placed on 12 leaves, and the tentacles of all, with a single exception, became greatly inflected, owing, I presume, to differences in the chemical nature of the urine on different occasions. The time required for the movements of the tentacles varied much, but was always effected in under 24 HRS. In 2 instances, I recorded that all the exterior tentacles were completely inflected in 17 HRS, but not the blade of the leaf. In another case, the edges of a leaf, after 25 HRS 30 Roman 1000, became so strongly inflected that it was converted into a cup. The power of urine does not lie in the urea, which, as we shall hereafter see, is inoperative. Albumin fresh from a hen's egg placed on 7 leaves caused the tentacles of 6 of them to be well inflected. In one case, the edge of the leaf itself became much curled. In after 20 HRS, the one leaf which was unaffected remained so for 26 HRS and was then treated with a drop of milk, and this caused the tentacles to bend inwards in 12 HRS. Cold filtered infusion of raw meat. This was tried only on a single leaf, which had most of its outer tentacles, and the blade inflected in 19 HRS. During subsequent years, I repeatedly used this infusion to test leaves which had been experimented page 80 on with other substances, and it was found to act most energetically, but as no exact account of these trials was kept, they are not here introduced. Mucus, thick and thin mucus from the bronchial tubes placed on 3 leaves caused inflection. A leaf with thin mucus had its marginal tentacles and blades somewhat curved inward in 5 HRS 30 Roman 1000, and greatly so in 20 HRS, the action of this fluid, no doubt, is due either to the saliva or to some albuminous matter mingled with it, and not, as we shall see in the next chapter, to mucin or the chemical principle of mucus. Human saliva, when evaporated, yields from 1 to 1% of residue, and this yields 0% of ashes, so that the proportion of nitrogenous matter which saliva contains must be small. Nevertheless, drops placed on the discs of 8 leaves acted on them all in one case, all. The exterior tentacles, excepting 9, were inflected in 19 HRS 30 Roman 1000 in another case. A few became so in 2 HRS, and after 7 HRS 30 Roman 1000, all those situated near where the drop lay as well as the blade were acted on. Since making these trials, I have many scores of times just touched glands with the handle of my scalpel wetted with saliva to ascertain whether a leaf was in an active condition, for this was shown in the course of a few minutes by the bending inwards of the tentacles. The edible nest of the Chinese swallow is formed of matter secreted by the salivary glands. 2 grains were added to 1 ounce of distilled water, 1 part to 218, which was boiled for several minutes, but did not dissolve the whole. The usual-sized drops were placed on 3 leaves, and these in 1 HR 30 Roman 1000 were well, and in 2 HRS 15 Roman 1000 closely inflected. Isinglass drops of a solution about as thick as milk, and of a still thicker solution, were placed on 8 leaves, and the tentacles of all became inflected in one case. The exterior tentacles were well curved in after 6 HRS 30 Roman 1000, and the blade of the leaf to a partial extent after 24 HRS, as saliva acted so efficiently and yet contains so small a proportion of nitrogenous matter. I tried how small a quantity of isinglass would act. 1 part was dissolved in 218 parts of distilled water, and drops were placed on 4 leaves after 5 HRS. 2 of these were considerably, and 2 moderately inflected after 22 HRS. The former were greatly, and the latter much more inflected in the course of 48 HRS. Mucus from the air passages is said in Martial Outlines of Physiology Vol. Roman 2 p. 364 to contain some albumin. Muller's Elements of Physiology, Engtrans, Vol. Roman 1 p. 514, page 81. From the time when the drops were placed on the leaves, all 4 had almost re-expanded. They were then given little bits of meat, and these acted more powerfully than the solution. 1 part of isinglass was next dissolved in 437 of water. The fluid thus formed was so thin that it could not be distinguished from pure water. The usual-sized drops were placed on 7 leaves each, of which thus received 1960 of a grain 295 mg. 3 of them were observed for 41 HRS, but were in no way affected. The 4th and 5th had 2 or 3 of their exterior tentacles inflected after 18 HRS. The 6th had a few more, and the 7th had in addition the edge of the leaf just perceptibly curved inwards. The tentacles of the 4 latter leaves began to re-expand after an additional interval of only 8 HRS. Hence, the 1960 of a grain of isinglass is sufficient to affect very slightly the more sensitive, active leaves on one of the leaves which had not been acted on by the weak solution, and on another which had only 2 of its tentacles inflected. Drops of the solution as thick as milk were placed, and next morning, after an interval of 16 HRS, both were found with all their tentacles strongly inflected. Altogether, I experimented on 64 leaves with the above nitrogenous fluids, the 5 leaves tried only with the extremely weak solution of isinglass not being included, nor the numerous trials subsequently made, of which no exact account was kept. Of these 64 leaves, 63 had their tentacles, and often their blades well inflected. The one which failed was probably too old and torpid, but to obtain so large a proportion of successful cases, care must be taken to select young and active leaves. Leaves in this condition were chosen with equal care for the 61 trials, with non-nitrogenous fluids, water not included, and we have seen that not one of these was in the least affected. We may therefore safely conclude that in the 64 experiments with nitrogenous fluids, the inflection of the exterior tentacles was due to the absorption of page 82 nitrogenous matter, by the glands of the tentacles on the disc. Some of the leaves, which were not affected by the non-nitrogenous fluids, were as above stated immediately afterwards tested with bits of meat, and were thus proved to be in an active condition. But in addition to these trials, 23 of the leaves, with drops of gum syrup or starch still lying on their discs, which had produced no effect in the course of between 24 HRS and 48 HRS, were then tested with drops of milk urine or albumin. Of the 23 leaves thus treated, 17 had their tentacles, and in some cases their blades well inflected, but their powers were somewhat impaired, for the rate of movement was decidedly slower than when fresh leaves were treated with these same nitrogenous fluids. This impairment, as well as the insensibility of 6 of the leaves, may be attributed to injury from exosmos caused by the density of the fluids placed on their discs. The results of a few other experiments with nitrogenous fluids may be here conveniently given. Decoctions of some vegetables known to be rich in nitrogen were made, and these acted like animal fluids. Thus a few green peas were boiled for some time in distilled water, and the moderately thick decoction thus made was allowed to settle. Drops of the superincumbent fluid were placed on 4 leaves, and when these were looked at after 16 HRS, the tentacles and blades of all were found strongly inflected. I infer from a remark by Gerhardt that legumin is present in peas, in combination with an alkali forming an incoagulable solution, and this would mingle with boiling water. I may mention, in relation to the above and following experiments, that according to Skiff, certain forms of albumin. Watt's Dictionary of Chemistry, Vol. Roman 3 p. 568. Leçons sur la phase de la digestion, Tom IP 379, Tom Roman 2, pp 154, 166, on legumin, page 83. Exist, which are not coagulated by boiling water, but are converted into soluble peptones. On 3 occasions, chopped cabbage leaves were boiled in distilled water for 1 HR, or for 114 HR, and by decanting the decoction, after it had been allowed to rest, a pale dirty green fluid was obtained. The usual sized drops were placed on 13 leaves. Their tentacles and blades were inflected after 4 HRS to a quite extraordinary degree. Next day, the protoplasm within the cells of the tentacles was found aggregated in the most strongly marked manner. I also touched the viscid secretion round the glands of several tentacles with minute drops of the decoction on the head of a small pin, and they became well inflected in a few minutes. The fluid proving so powerful, 1 part was diluted with 3 of water, and drops were placed on the discs of 5 leaves, and these next morning were so much acted on that their blades were completely doubled over. We thus see that a decoction of cabbage leaves is nearly or quite as potent as an infusion of raw meat. About the same quantity of chopped cabbage leaves and of distilled water as in the last experiment were kept in a vessel for 20 HRS in a hot closet, but not heated to near the boiling point. Drops of this infusion were placed on 4 leaves. One of these, after 23 HRS, was much inflected. A second, slightly a third, had only the submarginal tentacles inflected, and the 4th was not at all affected. The power of this infusion is therefore very much less than that of the decoction, and it is clear that the immersion of cabbage leaves for an hour in water at the boiling temperature is much more efficient in extracting matter which excites Drosera than immersion during many hours in warm water. Perhaps the contents of the cells are protected, as Skiff remarks, with respect to legumin, by the walls being formed of cellulose, and that until these are ruptured by boiling water, but little of the contained albuminous matter is dissolved. We know from the strong odor of cooked cabbage leaves that boiling water produces some chemical change in them, and that they are thus rendered far more digestible and nutritious to man. It is therefore an interesting. The leaves of young plants before the heart is formed, such as were used by me, contain 2% of albuminous matter, and the outer leaves of mature plants 1%. Watt's Dictionary of Chemistry, Vol. Roman 1 p. 653, page 84. Fact that water at this temperature extracts matter from them, which excites Drosera to an extraordinary degree. Grasses contain far less nitrogenous matter than do peas or cabbages. The leaves and stalks of 3 common kinds were chopped and boiled for some time in distilled water. Drops of this decoction, after having stood for 24 HRS, were placed on 6 leaves and acted in a rather peculiar manner, of which other instances will be given in the 7th chapter on the salts of ammonia. After 2 HRS 30 Roman 1004 of the leaves had their blades greatly inflected, but not their exterior tentacles, and so it was with all 6 leaves. After 24 HRS 2 days afterwards, the blades, as well as the few submarginal tentacles, which had been inflected, all re-expanded, and much of the fluid on their discs was by this time absorbed. It appears that the decoction strongly excites the glands on the disc, causing the blade to be quickly and greatly inflected, but that the stimulus, differently from what occurs in ordinary cases, does not spread, or only in a feeble degree, to the exterior tentacles. I may here add that one part of the extract of belladonna procured from a druggist was dissolved in 437 of water, and drops were placed on 6 leaves. Next day, all 6 were somewhat inflected, and after 48 HRS were completely re-expanded. It was not the included atropine which produced this effect, for I subsequently ascertained that it is quite powerless. I also procured some extract of hyoscyamus from 3 shops, and made infusions of the same strength as before. Of these 3 infusions, only one acted on some of the leaves, which were tried, though druggists believe that all the albumin is precipitated in the preparation of these drugs. I cannot doubt that some is occasionally retained, and a trace would be sufficient to excite the more sensitive leaves of Drosera, page 85. Chapter Roman 6. The digestive power of the secretion of Drosera. The secretion, rendered acid by the direct and indirect excitement of the gland's nature, of the acid digestible substances albumin, its digestion, arrested by alkalis, recommences by the addition of an acid meat, fibrin, syntonin, areola, tissue cartilage, fibrocartilage, bone enamel, and dentine phosphate, of lime, fibrous basis, of bone, gelatin, chondrin, milk, casein, and cheese, gluten, legumin, pollen, globulin, hematin, indigestible substances, epidermic productions, fibroelastic tissue, mucin, pepsin, urea, chitin, cellulose, gun, cotton, chlorophyll, fat, and all starch. Action of the secretion on living seeds. Summary and concluding remarks. As we have seen that nitrogenous fluids act very differently on the leaves of Drosera from non-nitrogenous fluids, and as the leaves remain clasped for a much longer time over various organic bodies than over inorganic bodies, such as bits of glass cinder, wood, Roman 100, it becomes an interesting inquiry whether they can only absorb matter already in solution or render it soluble. That is, have the power of digestion. We shall immediately see that they certainly have this power, and that they act on albuminous compounds in exactly the same manner as does the gastric juice of mammals, the digested matter being afterwards absorbed. This fact, which will be clearly proved, is a wonderful one in the physiology of plants. I must here state that I have been aided throughout all my later experiments by many valuable suggestions and assistance given me with the greatest kindness by Dr. Burden Sanderson, page 86. It may be well to premise, for the sake of any reader who knows nothing about the digestion of albuminous compounds by animals, that this is effected by means of a ferment pepsin together with weak hydrochloric acid, though almost any acid will serve, yet neither pepsin nor an acid by itself has any such power. We have seen that, when the glands of the disc are excited by the contact of any object, especially of one containing nitrogenous matter, the outer tentacles, and often the blade, become inflected, the leaf being thus converted into a temporary cup or stomach. At the same time, the discal glands secrete more copiously, and the secretion becomes acid. Moreover, they transmit some influence to the glands of the exterior tentacles, causing them to pour forth a more copious secretion, which also becomes acid, or more acid than it was before. As this result is an important one, I will give the evidence. The secretion of many glands on 30 leaves, which had not been in any way excited, was tested with litmus paper, and the secretion of 22 of these leaves did not in the least affect the color, whereas that of 8 caused an exceedingly feeble and sometimes doubtful tinge of red. 2 other old leaves, however, which appeared to have been inflected several times, acted much more decidedly on the paper. Particles of clean glass were then placed on 5 of the leaves, cubes of albumin on 6, and bits of raw meat on 3, on none of which was the secretion at this time in the least acid. after an interval of 24 HRS, when almost all the tentacles on. It appears, however, according to Skiff and contrary to the opinion of some physiologists, that weak hydrochloric dissolves, though slowly, a very minute quantity of coagulated albumin. Skiff Phys de la digestion, Tom Roman 2, p. 25, page 87. These 14 leaves had become more or less inflected. I again tested the secretion-selecting glands, which had not as yet reached the center or touched any object, and it was now plainly acid. The degree of acidity of the secretion varied somewhat on the glands of the same leaf. On some leaves, a few tentacles did not, from some unknown cause, become inflected, as often happens, and in 5 instances their secretion was found not to be in the least acid, whilst the secretion of the adjoining and inflected tentacles on the same leaf was decidedly acid, with leaves excited by particles of glass placed on the central glands. The secretion which collects on the disc beneath them was much more strongly acid than that poured forth from the exterior tentacles, which were as yet only moderately inflected when bits of albumin, and this is naturally alkaline, or bits of meat, were placed on the disc. The secretion collected beneath them was likewise strongly acid, as raw meat moistened with water is slightly acid. I compared its action on litmus paper before it was placed on the leaves and afterwards when bathed in the secretion, and there could not be the least doubt that the latter was very much more acid. I have indeed tried hundreds of times the state of the secretion on the discs of leaves, which were inflected over various objects, and never failed to find it acid. We may therefore conclude that the secretion from unexcited leaves, though extremely viscid, is not acid or only slightly so, but that it becomes acid, or much more strongly so, after the tentacles have begun to bend over any inorganic or organic object, and still more strongly acid after the tentacles have remained for some time closely clasped over any object. I may here remind the reader that the secretion, page 88, appears to be to a certain extent antiseptic, as it checks the appearance of mold and infusoria, thus preventing for a time the discoloration and decay of such substances as the white of an egg. Cheese Roman 100. It therefore acts like the gastric juice of the higher animals, which is known to arrest putrefaction by destroying the microzymes. As I was anxious to learn what acid the secretion contained, 445 leaves were washed in distilled water given me by Prof. Frankland, but the secretion is so viscid that it is scarcely possible to scrape or wash off the whole. The conditions were also unfavorable, as it was late in the year, and the leaves were small. Prof. Frankland, with great kindness, undertook to test the fluid. Thus collected, the leaves were excited by clean particles of glass, placed on them 24 HRS. Previously, no doubt much more acid would have been secreted had the leaves been excited by animal matter, but this would have rendered the analysis more difficult. Prof. Frankland informs me that the fluid contained no trace of hydrochloric, sulfuric, tartaric, oxalic, or formic acids. This having been ascertained, the remainder of the fluid was evaporated nearly to dryness and acidified with sulfuric acid. It then evolved volatile acid vapor, which was condensed and digested with carbonate of silver. The weight of the silver salt thus produced was only 37 gr, much too small a quantity for the accurate determination of the molecular weight of the acid. The number obtained, however, corresponded nearly with that of propionic acid, and I believe that this, or a mixture of acetic and butyric acids, were present in the liquid. The acid doubtless belongs to the acetic or fatty series. Prof. Frankland, as well as his assistant, observed, and this is an important fact, that the fluid, when acidified with sulfuric acid, emitted a powerful odor like that of pepsin. The leaves, from which the secretion had been washed, were also sent to Prof. Frankland. They were macerated for some hours, then acidified with sulfuric acid and distilled, but no acid passed over. Therefore, the acid which fresh leaves contain, as shown by their discoloring litmus paper, when crushed, must be of a different nature from that present in the secretion, nor was any odor of pepsin emitted by them. Page 89. Although it has long been known that pepsin with acetic acid has the power of digesting albuminous compounds, it appeared advisable to ascertain whether acetic acid could be replaced without the loss of digestive power by the allied acids which are believed to occur in the secretion of Drosera, namely propionic, butyric, or valerianic Dr. Burden Sanderson, was so kind as to make for me the following experiments, the results of which are valuable independently of the present inquiry. Prof. Franklin supplied the acids. 1. The purpose of the following experiments was to determine the digestive activity of liquids containing pepsin when acidulated with certain volatile acids belonging to the acetic series in comparison with liquids acidulated with hydrochloric acid in proportion similar to that in which it exists in gastric juice. 2. It has been determined empirically that the best results are obtained in artificial digestion when a liquid containing 2 per thousand of hydrochloric acid gas by weight is used. This corresponds to about 6 cubic centimeters per liter of ordinary strong hydrochloric acid. The quantities of propionic, butyric, and valerianic acids, respectively, which are required to neutralize as much base as 6 cubic centimeters of HCl, are in grams: 4 of propionic acid, 4 of butyric acid, and 5 of valerianic acid. It was therefore judged expedient, in comparing the digestive powers of these acids with that of hydrochloric acid, to use them in these proportions. 3. 500 cub-cent of a liquid containing about 8 cub-cent of a glycerine extract of the mucous membrane of the stomach of a dog killed during digestion having been prepared, 10 cub-cent of it were evaporated and dried at 110. This quantity yielded 0 of residue. 4 of this liquid 4 quantities were taken which were severally acidulated with hydrochloric, propionic, butyric, and valerianic acids in the proportions above indicated. Each liquid was then placed in a tube which was allowed to float in a water bath containing a thermometer which indicated a temperature of 38 to 40 cent into each. A quantity of unboiled fibrin was introduced and the whole allowed to stand for 4 hours, the temperature being maintained during the whole time and care being taken that each contained throughout an excess of fibrin at the end of the period. Each liquid was filtered of the filtrate which, of course, contained as much of the fibrin as had been digested during the 4 hours. Page 90. 10 cub-cent were measured out and evaporated and dried at 110, as before. The residues were respectively: in the liquid containing hydrochloric acid, 0 propionic acid, 0 butyric acid, 0 valerianic acid, 0. Hence, deducting from each of these the above-mentioned residue left when the digestive liquid itself was evaporated, viz., we have: for propionic acid, 0 butyric acid, 0 valerianic acid, 0. As compared with 0 for hydrochloric acid, these several numbers, expressing the quantities of fibrin by weight digested in presence of equivalent quantities of the respective acids under identical conditions. The results of the experiment may be stated thus: if 100 represent the digestive power of a liquid containing pepsin, with the usual proportion of hydrochloric acid, 14, 35, and 30, will represent respectively the digestive powers of the 3 acids under investigation. 5. In a second experiment, in which the procedure was in every respect the same excepting that all the tubes were plunged into the same water bath, and the residues dried at 115 Roman 100, the results were as follows: quantity of fibrin dissolved in 4 hours by 10 cub-cent of the liquid. Propionic acid, 0 butyric acid, 0 valerianic acid, 0. The quantity digested by a similar liquid containing hydrochloric acid was 0. Hence, taking this as 100, the following numbers represent the relative quantities digested by the other acids: propionic acid, 16, butyric acid, 24, valerianic acid, 16. 6. A third experiment of the same kind gave page 90. Quantity of fibrin digested in 4 hours by 10 cub-cent of the liquid. Hydrochloric acid, 0 propionic acid, 0 butyric acid, 0 valerianic acid, 0. Comparing as before the 3 last numbers with the first taken as 100, the digestive power of propionic acid is represented by 16, that of butyric acid by 35, and that of valerianic by 17. The mean of these 3 sets of observations, hydrochloric acid being taken, as 100 gives for propionic acid 15, butyric acid 32, valerianic acid 21. 7. A further experiment was made to ascertain whether the digestive activity of butyric acid, which was selected as being apparently the most efficacious, was relatively greater at ordinary temperatures than at the temperature of the body. It was found that whereas 10 cub-cent of a liquid containing the ordinary proportion of hydrochloric acid digested 0 gram, a similar liquid prepared with butyric acid digested 0 gram of fibrin. Hence, taking the quantities digested with hydrochloric acid at the temperature of the body as 100, we have the digestive power of hydrochloric acid at the temperature of 16 to 18 cent, represented by 44, that of butyric acid at the same temperature being 15. We here see that at the lower of these two temperatures, hydrochloric acid with pepsin digests within the same time rather less than half the quantity of fibrin compared with what it digests at the higher temperature, and the power of butyric acid is reduced in the same proportion under similar conditions and temperatures. We have also seen that butyric acid, which is much more efficacious than propionic or valerianic acids, digests with pepsin at the higher temperature less than a third of the fibrin, which is digested at the same temperature by hydrochloric acid, page 92. I will now give in detail my experiments on the digestive power of the secretion of Drosera, dividing the substances tried into two series, namely those which are digested more or less completely, and those which are not digested. We shall presently see that all these substances are acted on by the gastric juice of the higher animals in the same manner. I beg leave to call attention to the experiments under the head albumin, showing that the secretion loses its power when neutralized by an alkali and recovers it when an acid is added. Substances which are completely or partially digested by the secretion of Drosera. Albumin. After having tried various substances, Dr. Burton Sanderson suggested to me the use of cubes of coagulated albumin or hard-boiled egg. I may premise that 5 cubes of the same size as those used in the following experiments were placed for the sake of comparison at the same time on wet moss close to the plants of Drosera. The weather was hot, and after 4 days some of the cubes were discolored and moldy with their angles a little rounded. They were not surrounded by a zone of transparent fluid. As in the case of those undergoing digestion, other cubes retained their angles and white color after 8 days. All were somewhat reduced in size, discolored with their angles much rounded. Nevertheless, in 4 out of the 5 specimens, the central parts were still white and opaque, so that their state differed widely, as we shall see from that of the cubes subjected to the action of the secretion. Experiment 1. Rather large cubes of albumin were first tried. The tentacles were well inflected in 24 HRS. After on page 93 additional day, the angles of the cubes were dissolved and rounded, but the cubes were too large, so that the leaves were injured, and after 7 days one died, and the others were dying. Albumin, which has been kept for 4 or 5 days, and which it may be presumed has begun to decay slightly, seems to act more quickly than freshly boiled eggs, as the latter were generally used. I often moistened them with a little saliva to make the tentacles close more quickly. Experiment 2. A cube of 110 of an inch Roman 1e, with each side 110 of an inch, or 2 Roman 2000 in length, was placed on a leaf, and after 50 HRS, it was converted into a sphere about 340 of an inch, 1 Roman 2000 in diameter, surrounded by perfectly transparent fluid. After 10 days, the leaf re-expanded, but there was still left on the disc a minute bit of albumin, now rendered transparent. More albumin had been given to this leaf than could be dissolved or digested. Experiment 3. 2 cubes of albumin of 120 of an inch, 1 Roman 2000, were placed on 2 leaves. After 46 HRS, every atom of one was dissolved, and most of the liquefied matter was absorbed, the fluid which remained being in this, as in all other cases, very acid and viscid. The other cube was acted on at a rather slower rate. Experiment 4. 2 cubes of albumin of the same size as the last were placed on 2 leaves and were converted in 50 HRS into 2 large drops of transparent fluid, but when these were removed from beneath the inflected tentacles and viewed by reflected light. Under the microscope, fine streaks of white opaque matter could be seen in the one and traces of similar streaks in the other. The drops were replaced on the leaves, which re-expanded after 10 days, and now nothing was left except a very little transparent acid fluid. Experiment 5. This experiment was slightly varied so that the albumin might be more quickly exposed to the action of the secretion. 2 cubes, each of about 140 of an inch 635 Roman 2000, were placed on the same leaf, and 2 similar cubes on another. In all my numerous experiments on the digestion of cubes of albumin, the angles and edges were invariably first rounded. Now Skiff states Lekens Phys de la Digestion vol Roman 2 page 149, that this is characteristic of the digestion of albumin by the gastric juice of animals. On the other hand, he remarks les dissolutions en chimie ont luceur tout la surface des corps en contact avec l'agent dissolvant, page 94. Leaf. These were examined after 21 HRS 30 Roman 1000, and all 4 were found rounded after 46 HRS. The 2 cubes on the one leaf were completely liquefied, the fluid being perfectly transparent on the other leaf. Some opaque white streaks could still be seen in the midst of the fluid. After 72 HRS, these streaks disappeared, but there was still a little viscid fluid left on the disc, whereas it was almost all absorbed on the first leaf. Both leaves were now beginning to re-expand. The best and almost sole test of the presence of some ferment analogous to pepsin in the secretion appeared to be to neutralize the acid of the secretion with an alkali, and to observe whether the process of digestion ceased, and then to add a little acid and observe whether the process recommenced. This was done, and as we shall see, with success, but it was necessary first to try 2 control experiments, namely, whether the addition of minute drops of water of the same size as those of the dissolved alkalis to be used would stop the process of digestion, and secondly, whether minute drops of weak hydrochloric acid of the same strength and size as those to be used would injure the leaves. The 2 following experiments were therefore tried. Experiment 6. Small cubes of albumin were put on 3 leaves, and minute drops of distilled water on the head of a pin were added 2 or 3 times daily. These did not in the least delay the process, for after 48 HRS, the cubes were completely dissolved on all 3 leaves. On the 3rd day, the leaves began to re-expand, and on the 4th day, all the fluid was absorbed. Experiment 7. Small cubes of albumin were put on 2 leaves, and minute drops of hydrochloric acid of the strength of 1 part to 437 of water were added 2 or 3 times. This did not in the least delay, but seemed rather to hasten the process of digestion, for every trace of the albumin disappeared in 24 HRS 30 Roman 1000. After 3 days, the leaves partially re-expanded, and by this time almost all the viscid fluid on their discs was absorbed. It is almost superfluous to state that page 95 cubes of albumin of the same size as those above used left for 7 days in a little hydrochloric acid of the above strength retained all their angles as perfect as ever. Experiment 8. Cubes of albumin of 120 of an inch or 2 Roman 2000 were placed on 5 leaves, and minute drops of a solution of 1 part of carbonate of soda to 437 of water were added at intervals to 3 of them, and drops of carbonate of potash of the same strength to the other 2. The drops were given on the head of a rather large pin, and I ascertained that each was equal to about 110 of a minimum 59 Roman 1050, so that each contained only 14,800 of a grain 135 mg of the alkali. This was not sufficient, for after 46 HRS, all 5 cubes were dissolved. Experiment 9. The last experiment was repeated on 4 leaves, with this difference that drops of the same solution of carbonate of soda were added rather often as the secretion became acid, so that it was much more effectually neutralized. And now, after 24 HRS, the angles of 3 of the cubes were not in the least rounded, those of the 4th being so in a very slight degree. Drops of extremely weak hydrochloric acid, viz. 1 part to 847 of water, were then added just enough to neutralize the alkali which was still present, and now digestion immediately recommenced, so that after 23 HRS 30 Roman 1003 of the cubes were completely dissolved, whilst the fourth was converted into a minute sphere surrounded by transparent fluid, and this sphere next day disappeared. Experiment 10 stronger solutions of carbonate of soda and of potash were next used, viz. 1 part to 109 of water, and as the same sized drops were given as before, each drop contained 11,200 of a grain 539 mg of either salt, 2 cubes of albumin each, about 140 of an inch, or 635 Roman 2000 were placed on the same leaf and 2 on another, each leaf received as soon as the secretion became slightly acid, and this occurred 4 times within 24 HRS drops either of the soda or potash, and the acid was thus effectually neutralized. The experiment now succeeded perfectly, for after 22 HRS, the angles of the cubes were as sharp as they were at first, and we know from experiment 5 that such small cubes would have been completely rounded within this time by the secretion in its natural state. Some of the fluid was now removed with blotting paper from the discs of the leaves, and minute drops of hydrochloric acid of the strength of the 1 part to 200 of water was added. Acid of this greater strength was used as the solutions of the alkalis were stronger. The page 96 process of digestion now commenced, so that within 48 HRS, from the time when the acid was given, the 4 cubes were not only completely dissolved, but much of the liquefied albumin was absorbed. Experiment 11 2 cubes of albumin 140 of an inch, or 635 Roman 2000, were placed on 2 leaves, and were treated with alkalis as in the last experiment, and with the same result, for after 22 HRS, they had their angles perfectly sharp, showing that the digestive process had been completely arrested. I then wished to ascertain what would be the effect of using stronger hydrochloric acid, so I added minute drops of the strength of 1%. This proved rather too strong, for, after 48 HRS, from the time when the acid was added, 1 cube was still almost perfect, and the other only very slightly rounded, and both were stained slightly pink. This latter fact shows that the leaves were injured, for during the normal process of digestion, the albumin is not thus colored, and we can thus understand why the cubes were not dissolved. From these experiments, we clearly see that the secretion has the power of dissolving albumin, and we further see that if an alkali is added, the process of digestion is stopped, but immediately recommences as soon as the alkali is neutralized by weak hydrochloric acid. Even if I had tried no other experiments than these, they would have almost sufficed to prove that the glands of Drosera secrete some ferment analogous to pepsin, which, in presence of an acid, gives to the secretion its power of dissolving albuminous compounds. Splinters of clean glass were scattered on a large number of leaves, and these became moderately inflected. They were cut off and divided into 3 lots. 2 of them, after being left for some time in a little distilled water, were strained, and some dis. Sachs remarks, trait Debat 1874 p. 774, that cells which are killed by freezing by too great heat, or by chemical agents, allow all their coloring matter to escape into the surrounding water. Page 97. Colored viscid slightly acid fluid was thus obtained. The 3rd lot was well soaked in a few drops of glycerine, which is well known to dissolve pepsin. Cubes of albumin 120 of an inch were now placed in the 3 fluids in watchglasses, some of which were kept for several days at about 90 Fahr 322 cent, and others at the temperature of my room, but none of the cubes were dissolved, the angles remaining as sharp as ever. This fact probably indicates that the ferment is not secreted until the glands are excited by the absorption of a minute quantity of already soluble animal matter, a conclusion which is supported by what we shall hereafter see with respect to Dionea. Dr. Hooker likewise found that although the fluid within the pitchers of Nepenthes possesses extraordinary power of digestion, yet, when removed from the pitchers before they have been excited and placed in a vessel, it has no such power, although it is already acid, and we can account for this fact only on the supposition that the proper ferment is not secreted until some exciting matter is absorbed. On 3 other occasions, 8 leaves were strongly excited with albumin moistened with saliva. They were then cut off and allowed to soak for several hours, or for a whole day, in a few drops of glycerine. Some of this extract was added to a little hydrochloric acid of various strengths, generally 1 to 400 of water, and minute cubes of albumin were placed in the mixture. In 2 of these trials, the cubes were not in the least acted on, but in the third. As a control experiment, bits of albumin were placed in the same glycerine with hydrochloric acid of the same strength, and the albumin, as might have been expected, was not in the least affected after 2 days, page 98. The experiment was successful, for in a vessel containing 2 cubes both were reduced in size in 3 HRS, and after 24 HRS mere streaks of undissolved albumin were left in a second vessel containing 2-minute ragged bits of albumin. Both were likewise reduced in size in 3 HRS, and after 24 HRS completely disappeared. I then added a little weak hydrochloric acid to both vessels and placed fresh cubes of albumin in them, but these were not acted on. This latter fact is intelligible according to the high authority of Skiff, who has demonstrated, as he believes, in opposition to the view held by some physiologists, that a certain small amount of pepsin is destroyed during the act of digestion, so that if my solution contained, as is probable, an extremely small amount of the ferment, this would have been consumed by the dissolution of the cubes of albumin first, given none being left. When the hydrochloric acid was added, the destruction of the ferment during the process of digestion, or its absorption after the albumin had been converted into a peptone, will also account for only 1 out of the 3 latter sets of experiments having been successful. Digestion of roast meat cubes of about 120 of an inch 1 Roman 2000 of moderately roasted meat were placed on 5 leaves, which became in 12 HRS closely inflected. After 48 HRS, I gently opened 1 leaf, and the meat now consisted of a minute central sphere, partially digested and surrounded by a thick envelope of transparent viscid fluid. The whole, without being much disturbed, was removed and placed under the microscope in the central part. The transverse striae on the muscular fibers were quite distinct, and it was Lecon's Phase de la Digestion, 1867, Tom Roman 2 pp. 114,126, page 99. Interesting to observe how gradually they disappeared when the same fiber was traced into the surrounding fluid. They disappeared by the striae being replaced by transverse lines formed of excessively minute dark points, which towards the exterior could be seen only under a very high power, and ultimately these points were lost. When I made these observations, I had not read Skiff's account of the digestion of meat by gastric juice, and I did not understand the meaning of the dark points, but this is explained in the following statement, and we further see how closely similar is the process of digestion by gastric juice and by the secretion of Drosera. On a dit le suc gastrique face perdre la fibre musculaire, says stries transversals ainsi enonci cette proposition pour donner lieu à une équivoque, car si, qui si peut si nest, que l'aspect extérieur de la striature, et non les elements anatomiques qui la composent en cette que les stries, qui donnent un aspect si caractéristique à la fibre musculaire, sont le résultat de la juxtaposition et du parallélisme des corpuscules élémentaires, places à distances égales dans l'intérieur des fibres contiguës, or des que le tissu connectif, qui relie entre elles les fibres élémentaires, vint à si gonfler et à si dissoudre, et que les fibres elles-mêmes si dissociant si parallélisme est détruite, et avec lui l'aspect le phénomène optique des stries, si après la désagrégation des fibres, on examine au microscope les fibres élémentaires, on distingue encore très nettement alors intérieur les corpuscules, et on continue à les voir de plus en plus pales, jusqu'au moment où les fibres elles-mêmes, si liquéfiantes et disparaissent dans le suc gastrique, si qui constitue la striature à proprement parler n'est donc pas détruite avant la liquefaction de la fibre charnue elle-même. In the viscid fluid surrounding the central sphere of undigested meat, there were globules of fat and little bits of fibroelastic tissue, neither of which were in Lecon's Phase de la Digestion, Tom Roman 2 p. 145, page 100. The least digested there were also little free parallelograms of yellowish, highly translucent matter. Skiff, in speaking of the digestion of meat, by gastric juice, alludes to such parallelograms and says, Le gonflement par lequel commence la digestion de la viande résulte de l'action du suc gastrique, aside sur le tissu connectif, qui s'y dissout d'abord et qui par sa liquéfaction désagrège les fibrilles cellesques qui s'y dissolvent ensuite, en grande partie mais avant de passer à l'état liquide elles tendent. Ainsi briser en petits fragments transversox les sarcus elements de Bowman, qui n'y sont autre chose, que les produits de cette division transversale des fibrilles élémentaires pouvant être préparés et isolés à l'aide du suc gastrique, pour vous qu'on n'attend pas jusqu'à la liquéfaction complete du muscle. After an interval of 72 HRS, from the time when the 5 cubes were placed on the leaves, I opened the 4 remaining ones. On 2 nothing could be seen but little masses of transparent viscid fluid, but when these were examined under a high-power fat globules, bits of fibroelastic tissue and some few parallelograms of sarcus matter could be distinguished, but not a vestige of transverse striae on the other 2 leaves. There were minute spheres of only partially digested meat in the center of much transparent fluid. Fibrin bits of fibrin were left in water during 4 days whilst the following experiments were tried, but they were not in the least acted on the fibrin, which I first used was not pure, and included dark particles. It had either not been well prepared or had subsequently undergone some change. Thin portions about 1/110 of an inch square were placed on several leaves, and though the fibrin was soon liquefied, the whole was never dissolved. Smaller particles were then placed on 4 leaves, and minute page 101 drops of hydrochloric acid 1 part to 437 of water were added. This seemed to hasten the process of digestion, for on 1 leaf all was liquefied and absorbed after 20 HRS, but on the 3 other leaves some undissolved residue was left after 48 HRS. It is remarkable that in all the above and following experiments, as well as when much larger bits of fibrin were used, the leaves were very little excited, and it was sometimes necessary to add a little saliva to induce complete inflection. The leaves, moreover, began to re-expand after only 48 HRS, whereas they would have remained inflected for a much longer time had insects' meat cartilage albumin Roman 100 been placed on them. I then tried some pure white fibrins sent me by Dr. Burden Sanderson. Experiment 1. 2 particles, barely 120 of an inch 1 Roman 2000 square, were placed on opposite sides of the same leaf. One of these did not excite the surrounding tentacles, and the gland on which it rested soon dried. The other particle caused a few of the short adjoining tentacles to be inflected, the more distant ones not being affected. After 24 HRS both were almost, and after 72 HRS completely dissolved. Experiment 2, the same experiment with the same result, only one of the 2 bits of fibrin exciting the short surrounding tentacles. This bit was so slowly acted on that after a day I pushed it on to some fresh glands. In 3 days from the time when it was first placed on the leaf, it was completely dissolved. Experiment 3, bits of fibrin of about the same size as before were placed on the discs of 2 leaves. These caused very little inflection in 23 HRS, but after 48 HRS, both were well clasped by the surrounding short tentacles, and after an additional 24 HRS were completely dissolved on the disc of one of these leaves. Much clear acid fluid was left. Experiment 4, similar bits of fibrin were placed on the discs of 2 leaves. As after 2 HRS, the glands seemed rather dry, they were freely moistened with saliva. This soon caused strong inflection, both of the tentacles and blades, with copious page 102 secretion from the glands. In 18 HRS, the fibrin was completely liquefied, but undigested atoms still floated in the liquid. These, however, disappeared in under 2 additional days. From these experiments, it is clear that the secretion completely dissolves pure fibrin. The rate of dissolution is rather slow, but this depends merely on this substance not exciting the leaves sufficiently so that only the immediately adjoining tentacles are inflected, and the supply of secretion is small. Syntonin, this substance extracted from muscle, was kindly prepared for me by Dr. Moore very differently from fibrin. It acts quickly and energetically. Small portions placed on the discs of 3 leaves cause their tentacles and blades to be strongly inflected within 8 HRS, but no further observations were made. It is probably due to the presence of this substance that raw meat is too powerful a stimulant, often injuring or even killing the leaves. Areolar tissue. Small portions of this tissue from a sheep were placed on the discs of 3 leaves. These became moderately well inflected in 24 HRS, but began to re-expand after 48 HRS, and were fully re-expanded in 72 HRS, always reckoning from the time when the bits were first given this substance. Therefore, like fibrin excites the leaves for only a short time, the residue left on the leaves after they were fully re-expanded was examined under a high power and found much altered, but owing to the presence of a quantity of elastic tissue which is never acted on, could hardly be said to be in a liquefied condition. Some areolar tissue free from elastic tissue was next procured from the visceral cavity of a toad, and moderately sized, as well as very small bits, were placed on 5 leaves after 24 HRS. 2 of the bits, page 103, were completely liquefied. 2 others were rendered transparent, but not quite liquefied, whilst the 5th was but little affected. Several glands on the 3 latter leaves were now moistened with a little saliva, which soon caused much inflection and secretion, with the result that in the course of 12 additional HRS, 1 leaf alone showed a remnant of undigested tissue on the discs of the 4 other leaves, to one of which a rather large bit had been given. Nothing was left, except some transparent viscid fluid. I may add that some of this tissue included points of black pigment, and these were not at all affected as a control experiment. Small portions of this tissue were left in water and on wet moss for the same length of time, and remained white and opaque. From these facts, it is clear that areolar tissue is easily and quickly digested by the secretion, but that it does not greatly excite the leaves. Cartilage. 3 cubes 120 of an inch, or 1 Roman 2000 of white, translucent, extremely tough cartilage, were cut from the end of a slightly roasted leg bone of a sheep. These were placed on 3 leaves borne by poor small plants in my greenhouse during November, and it seemed in the highest degree improbable that so hard a substance would be digested under such unfavorable circumstances. Nevertheless, after 48 HRS, the cubes were largely dissolved and converted into minute spheres. Surrounded by transparent, very acid fluid, 2 of these spheres were completely softened to their centers, whilst the third still contained a very small, irregularly shaped core of solid cartilage. Their surfaces were seen under the microscope to be curiously marked by prominent ridges, showing that the cartilage had been unequally corroded by the secretion. I need hardly, page 104, say that cubes of the same cartilage kept in water for the same length of time were not in the least affected. During a more favorable season, moderately sized bits of the skinned ear of a cat, which includes cartilage, areolar, and elastic tissue, were placed on 3 leaves. Some of the glands were touched with saliva, which caused prompt inflection. 2 of the leaves began to re-expand. After 3 days, and the third on the 5th day, the fluid residue left on their discs was now examined, and consisted in 1 case of perfectly transparent viscid matter. In the other 2 cases, it contained some elastic tissue and apparently remnants of half-digested areolar tissue. Fibrocartilage from between the vertebrae of the tail of a sheep, moderately sized and small bits, the latter about 120 of an inch, were placed on 9 leaves. Some of these were well, and some very little inflected. In the latter case, the bits were dragged over the discs, so that they were well bedaubed with the secretion, and many glands thus irritated. All the leaves re-expanded after only 2 days, so that they were but little excited by this substance. The bits were not liquefied, but were certainly in an altered condition, being swollen, much more transparent, and so tender as to disintegrate very easily. My son Francis prepared some artificial gastric juice, which was proved efficient by quickly dissolving fibrin and suspended portions of the fibrocartilage in it. These swelled and became hyaline, exactly like those exposed to the secretion of Drosera, but were not dissolved. This result surprised me much, as 2 physiologists were of opinion that fibrocartilage would be easily digested by gastric juice. I therefore asked Dr. Klein to examine the specimens, and page 105, he reports that the 2 which had been subjected to artificial gastric juice were in that state of digestion in which we find connective tissue, when treated with an acid vis swollen, more or less hyaline, the fibrillar bundles having become homogeneous and lost their fibrillar structure in the specimens which had been left on the leaves of Drosera, until they re-expanded parts were altered, though only slightly so, in the same manner as those subjected to the gastric juice, as they had become more transparent, almost hyaline, with the fibrillation of the bundles indistinct fibrocartilage, is therefore acted on in nearly the same manner by gastric juice and by the secretion of Drosera. Small smooth bits of the dried hiatal bone of a fowl moistened with saliva were placed on 2 leaves, and a similarly moistened splinter of an extremely hard broiled mutton chop bone on a 3rd leaf. These leaves soon became strongly inflected, and remained so for an unusual length of time, namely 1 leaf for 10, and the other 2. For 9 days, the bits of bone were surrounded all the time by acid secretion. When examined under a weak power, they were found quite softened, so that they were readily penetrated by a blunt needle torn into fibers or compressed. Dr. Klein was so kind as to make sections of both bones and examine them. He informs me that both presented the normal appearance of decalcified bone, with traces of the earthy salts, occasionally left. The corpuscles, with their processes, were very distinct in most parts, but in some parts, especially near the periphery of the hiatal bone, none could be seen. Other parts again appeared amorphous, with even the longitudinal striation of bone not distinguishable. This amorphous structure, page 106, as Dr. Klein thinks, may be the result either of the incipient digestion of the fibrous basis, or of all the animal matter having been removed, the corpuscles being thus rendered invisible. A hard, brittle, yellowish substance occupied the position of the medulla in the fragments of the hiatal bone. As the angles and little projections of the fibrous basis were not in the least rounded or corroded, 2 of the bits were placed on fresh leaves. These, by the next morning, were closely inflected, and remained soothed, 1 for 6, and the other for 7 days. Therefore, for not so long a time as on the first occasion, but for a much longer time than ever occurs with leaves inflected over inorganic, or even over many organic bodies, the secretion during the whole time colored litmus paper of a bright red. But this may have been due to the presence of the acid superphosphate of lime. When the leaves re-expanded, the angles and projections of the fibrous basis were as sharp as ever. I therefore concluded falsely, as we shall presently see, that the secretion cannot touch the fibrous basis of bone. The more probable explanation is that the acid was all consumed in decomposing the phosphate of lime, which still remained, so that none was left in a free state to act in conjunction with the ferment on the fibrous basis. Enamel and dentine. As the secretion decalcified ordinary bone, I determined to try whether it would act on enamel and dentine, but did not expect that it would succeed with so hard a substance as enamel. Dr. Klein gave me some thin transverse slices of the canine tooth of a dog, small angular fragments of which were placed on 4 leaves, and these were examined each succeeding day at the same hour. The results are, I think, worth giving in detail, page 107. Experiment 1. May 1st fragment placed on leaf, 3rd tentacles, but little inflected, so a little saliva was added. 6th, as the tentacles were not strongly inflected, the fragment was transferred to another leaf, which acted at first slowly, but by the 9th closely embraced it. On the 11th, this 2nd leaf began to re-expand. The fragment was manifestly softened, and Dr. Klein reports a great deal of enamel and the greater part of the dentine decalcified. Experiment 2. May 1st fragment placed on leaf, 2nd tentacles, fairly well inflected with much secretion on the disc, and remained so until the 7th, when the leaf re-expanded. The fragment was now transferred to a fresh leaf, which next day, 8th, was inflected in the strongest manner, and thus remained until the 11th, when it re-expanded. Dr. Klein reports a great deal of enamel and the greater part of the dentine decalcified. Experiment 3. May 1st fragment moistened with saliva and placed on a leaf, which remained well inflected until 5th, when it re-expanded. The enamel was not at all, and the dentine only slightly softened. The fragment was now transferred to a fresh leaf, which next morning, 6th, was strongly inflected, and remained so until the 11th. The enamel and dentine both now somewhat softened, and Dr. Klein reports less than half the enamel, but the greater part of the dentine decalcified. Experiment 4. May 1st, a minute and thin bit of dentine moistened with saliva was placed on a leaf, which was soon inflected and re-expanded. On the 5th, the dentine had become as flexible as thin paper. It was then transferred to a fresh leaf, which next morning 6th was strongly inflected, and reopened on the 10th. The decalcified dentine was now so tender that it was torn into shreds merely by the force of the re-expanding tentacles. From these experiments, it appears that enamel is attacked by the secretion with more difficulty than dentine, as might have been expected from its extreme hardness, and both with more difficulty than ordinary bone after the process of dissolution has once commenced. It is carried on with greater ease. This may be inferred from the leaves, to which the fragments were transferred, becoming, in all 4 cases, strongly inflected in the course of a single day, whereas the first set of leaves acted much less quickly, and page 108 energetically. The angles or projections of the fibrous basis of the enamel and dentine, except perhaps in no 4 which could not be well observed, were not in the least rounded, and Dr. Klein remarks that their microscopical structure was not altered, but this could not have been expected, as the decalcification was not complete in the 3 specimens which were carefully examined. Fibrous basis of boni at first concluded, as already stated, that the secretion could not digest this substance. I therefore asked Dr. Burden Sanderson to try bone enamel and dentine in artificial gastric juice, and he found that they were, after a considerable time, completely dissolved. Dr. Klein examined some of the small lamellae into which part of the skull of a cat became broken up after about a week's immersion in the fluid, and he found that towards the edges the matrix appeared rarefied, thus producing the appearance as if the canaliculi of the bone corpuscles had become larger. Otherwise, the corpuscles and their canaliculi were very distinct, so that with bone subjected to artificial gastric juice, complete decalcification precedes the dissolution of the fibrous basis. Dr. Burden Sanderson suggested to me that the failure of Drosera to digest the fibrous basis of bone enamel and dentine might be due to the acid being consumed in the decomposition of the earthy salts, so that there was none left for the work of digestion. Accordingly, my son thoroughly decalcified the bone of a sheep with weak hydrochloric acid, and 7-minute fragments of the fibrous basis were placed on so many leaves, 4 of the fragments being first damped with saliva to aid prompt inflection. All 7 leaves became inflected, but only very moderately. In the course of a day, page 109, they quickly began to re-expand, 5 of them on the 2nd day, and the other 2 on the 3rd day. On all 7 leaves, the fibrous tissue was converted into perfectly transparent viscid, more or less liquefied. Little masses in the middle, however, of one my son saw, under a high power, a few corpuscles with traces of fibrillation in the surrounding transparent matter. From these facts, it is clear that the leaves are very little excited by the fibrous basis of bone, but that the secretion easily and quickly liquefies it. If thoroughly decalcified, the glands, which had remained in contact for 2 or 3 days with the viscid masses, were not discolored, and apparently had absorbed little of the liquefied tissue or had been little affected by it. Phosphate of lime. As we have seen that the tentacles of the first set of leaves remained clasped for 9 or 10 days over minute fragments of bone, and the tentacles of the second set for 6 or 7 days over the same fragments, I was led to suppose that it was the phosphate of lime, and not any included animal matter, which caused such long-continued inflection. It is at least certain from what has just been shown that this cannot have been due to the presence of the fibrous basis, with enamel and dentine, the former of which contains only 4% of organic matter. The tentacles of 2 successive sets of leaves remained inflected altogether for 11 days. In order to test my belief in the potency of phosphate of lime, I procured some from Prof. Frankland, absolutely free of animal matter, and of any acid. A small quantity, moistened with water, was placed on the discs of 2 leaves. One of these was only slightly affected, the other remained closely inflected for 10 days, when a few of the tentacles began to, page 110, re-expand, the rest being much injured or killed. I repeated the experiment, but moistened the phosphate with saliva to ensure prompt inflection. 1 leaf remained inflected for 6 days. The little saliva used would not have acted for nearly so long a time, and then died. The other leaf tried to re-expand on the 6th day, but after 9 days failed to do so, and likewise died, although the quantity of phosphate given to the above 4 leaves was extremely small. Much was left in every case undissolved. A larger quantity, wetted with water, was next placed on the discs of 3 leaves, and these became most strongly inflected. In the course of 24 hrs, they never re-expanded. On the 4th day they looked sickly, and on the 6th were almost dead. Large drops of not very viscid fluid hung from their edges during the 6 days. This fluid was tested each day with litmus paper, but never colored it, and this circumstance I do not understand. As the superphosphate of lime is acid, I suppose that some superphosphate must have been formed by the acid of the secretion acting on the phosphate, but that it was all absorbed and injured the leaves. The large drops, which hung from their edges, being an abnormal and dropsical secretion. Anyhow, it is manifest that the phosphate of lime is a most powerful stimulant. Even small doses are more or less poisonous, probably on the same principle that raw meat and other nutritious substances given in excess kill the leaves. Hence the conclusion that the long-continued inflection of the tentacles over fragments of bone enamel and dentine is caused by the presence of phosphate of lime, and not of any included animal matter, is no doubt correct. Gelatine. I used pure gelatine in thin sheets, given page 111 me by Prof. Hoffman, for comparison. Squares of the same size as those placed on the leaves were left close by on wet moss. These soon swelled, but retained their angles for 3 days. After 5 days, they formed rounded, softened masses. But even on the 8th day, a trace of gelatine could still be detected. Other squares were immersed in water, and these, though much swollen, retained their angles for 6 days. Squares of 110 of an inch, 2 Roman 2000, just moistened with water, were placed on 2 leaves, and after 2 or 3 days nothing was left on them but some acid viscid fluid, which in this and other cases never showed any tendency to regelatinize, so that the secretion must act on the gelatine differently to what water does, and apparently in the same manner as gastric juice. 4 squares of the same size as before were then soaked for 3 days in water, and placed on large leaves. The gelatine was liquefied and rendered acid in 2 days, but did not excite much inflection. The leaves began to re-expand after 4 or 5 days, much viscid fluid being left on their discs, as if but little had been absorbed. One of these leaves, as soon as it re-expanded, caught a small fly, and after 24 hrs was closely inflected, showing how much more potent than gelatine is the animal matter absorbed from an insect. Some larger pieces of gelatine soaked for 5 days in water were next placed on 3 leaves, but these did not become much inflected until the 3rd day, nor was the gelatine completely liquefied until the 4th day. On this day, 1 leaf began to re-expand, the 2nd on the 5th, and 3rd on the 6th. These several facts. Dr. Lauder Brunton Handbook for the Phiz Laboratory, 1873, pp. 477-487, Skiff Lekens Phiz de la Digestion, 1867, p. 240, page 112. Prove that gelatine is far from acting energetically on Drosera. In the last chapter, it was shown that a solution of isinglass of commerce, as thick as milk or cream, induces strong inflection. I therefore wish to compare its action with that of pure gelatine. Solutions of 1 part of both substances to 218 of water were made, and half minim drops 296 Roman 1050 were placed on the discs of 8 leaves, so that each received 1480 of a grain, or 135 mg. The 4 with the isinglass were much more strongly inflected than the other 4. I conclude therefore that isinglass contains some, though perhaps very little, soluble albuminous matter. As soon as these 8 leaves re-expanded, they were given bits of roast meat, and in some hours all became greatly inflected again, showing how much more meat excites Drosera than does gelatine or isinglass. This is an interesting fact, as it is well known that gelatine by itself has little power of nourishing animals. Chondrin. This was sent me by Dr. Moore in a gelatinous state. Some was slowly dried, and a small chip was placed on a leaf, and a much larger chip on a second leaf. The first was liquefied in a day. The larger piece was much swollen and softened, but was not completely liquefied until the 3rd day. The undried jelly was next tried, and as a control experiment, small cubes were left in water for 4 days, and retained their angles. Cubes of the same size were placed on 2 leaves, and larger cubes on 2 other leaves. The tentacles and laminae of the latter were closely inflected after 22 hrs, but those of Dr. Lauder Brunton gives in the medical record January 1873 p. 36, an account of Voigt's view of the indirect part which gelatine plays in nutrition, page 113. 2 leaves with the smaller cubes, only to a moderate degree the jelly on all 4, was by this time liquefied and rendered very acid. The glands were blackened from the aggregation of their protoplasmic contents in 46 HRS. From the time when the jelly was given, the leaves had almost re-expanded, and completely so after 70 HRS. And now only a little slightly adhesive fluid was left unabsorbed on their discs. 1 part of chondrin jelly was dissolved in 218 parts of boiling water, and half-minim drops were given to 4 leaves, so that each received about 1480 of a grain 135 mg of the jelly, and of course much less of dry chondrin. This acted most powerfully for after. Only 3 HRS 30 Roman 1000, all 4 leaves were strongly inflected. 3 of them began to re-expand after 24 HRS, and in 48 HRS were completely open, but the 4th had only partially re-expanded. All the liquefied chondrin was by this time absorbed, hence a solution of chondrin seems to act far more quickly and energetically than pure gelatine or isinglass, but I am assured by good authorities that it is most difficult or impossible to know whether chondrin is pure, and if it contained any albuminous compound, this would have produced the above effects. Nevertheless, I have thought these facts worth giving, as there is so much doubt on the nutritious value of gelatine, and Dr. Lauder Brunton does not know of any experiments with respect to animals on the relative value of gelatine and chondrin. Milk. We have seen in the last chapter that milk acts most powerfully on the leaves, but whether this is due to the contained casein or albumin, I know not. Rather, large drops of milk excite so much secretion, which is very acid, that it sometimes trickles down, page 114, from the leaves, and this is likewise characteristic of chemically prepared casein. Minute drops of milk placed on leaves were coagulated in about 10 minutes. Skiff denies that the coagulation of milk by gastric juice is exclusively due to the acid which is present, but attributes it in part to the pepsin, and it seems doubtful whether with Drosera the coagulation can be wholly due to the acid, as the secretion does not commonly color litmus paper until the tentacles have become well inflected, whereas the coagulation commences, as we have seen, in about 10 minutes. Minute drops of skimmed milk were placed on the discs of 5 leaves. Large proportion of the coagulated matter or curd was dissolved in 6 HRS, and still more completely in 8 HRS. These leaves re-expanded after 2 days, and the viscid fluid left on their discs was then carefully scraped off and examined. It seemed at first sight as if all the casein had not been dissolved, for a little matter was left, which appeared of a whitish color by reflected light, but this matter, when examined under a high power, and when compared with a minute drop of skimmed milk coagulated by acetic acid, was seen to consist exclusively of oil globules more or less aggregated together with no trace of casein. As I was not familiar with the microscopical appearance of milk, I asked Dr. Lauder Brunton to examine the slides, and he tested the globules with ether, and found that they were dissolved. We may therefore conclude that the secretion quickly dissolves casein in the state in which it exists in milk. Chemically prepared casein, the substance which Lekens Roman 100 Tom Roman 2 page 151, page 115, is insoluble in water, is supposed by many chemists to differ from the casein of fresh milk. I procured some consisting of hard globules from Messrs. Hopkins and Williams, and tried many experiments with its small particles, and the powder, both in a dry state and moistened with water, caused the leaves on which they were placed to be inflected very slowly. Generally, not until 2 days had elapsed, other particles, wetted with weak hydrochloric acid, 1 part to 437 of water, acted in a single day, as did some casein freshly prepared for me by Dr. Moore. The tentacles commonly remained inflected for from 7 to 9 days, and during the whole of this time the secretion was strongly acid. Even on the 11th day, some secretion left on the disc of a fully re-expanded leaf was strongly acid. The acid seems to be secreted quickly, for in one case, the secretion from the discal glands, on which a little powdered casein had been strewed, colored litmus paper before any of the exterior tentacles were inflected. Small cubes of hard casein moistened with water were placed on 2 leaves. After 3 days, one cube had its angles a little rounded, and after 7 days, both consisted of rounded softened masses in the midst of much viscid and acid secretion. But it must not be inferred from this fact that the angles were dissolved, for cubes immersed in water were similarly acted on. After 9 days, these leaves began to re-expand. But in this and other cases, the casein did not appear, as far as could be judged by the eye, much, if at all, reduced in bulk. According to Hop-Saylor and Lubovin, casein consists of an albuminous with Dr. Lauder Brunton Handbook for Phys Lab p. 529, page 116. A non-albuminous substance, and the absorption of a very small quantity of the former would excite the leaves, and yet not decrease the casein to a perceptible degree. Skiff asserts, and this is an important fact for us, that la casein purifie des chemists est un corps presque complètement inattaquable par le suc gastrique, so that here we have another point of accordance between the secretion of Drosera and gastric juice, as both act so differently on the fresh casein of milk and on that prepared by chemists. A few trials were made with cheese cubes of 120 of an inch. 1 Roman 2000 were placed on 4 leaves, and these, after 1 or 2 days, became well inflected, their glands pouring forth much acid secretion. After 5 days, they began to re-expand, but one died, and some of the glands on the other leaves were injured. Judging by the eye, the softened and subsided masses of cheese left on the discs were very little or not at all reduced in bulk. We may, however, infer from the time during which the tentacles remained inflected, from the changed color of some of the glands and from the injury done to others, that matter had been absorbed from the cheese. Legumin I did not procure this substance in a separate state, but there can hardly be a doubt that it would be easily digested, judging from the powerful effect produced by drops of a decoction of green peas, as described in the last chapter. Thin slices of a dried pea, after being soaked in water, were placed on 2 leaves. These became somewhat inflected in the course of a single hour, and most strongly so in 21 HRS. They re-expanded after 3 or 4 days. Lekens Roman 100 Tom Roman 2 page 153 page 117. The slices were not liquefied, for the walls of the cells, composed of cellulose, are not in the least acted on by the secretion. Pollen, a little fresh pollen from the common pea, was placed on the discs of 5 leaves, which soon became closely inflected, and remained so for 2 or 3 days. The grains, being then removed and examined under the microscope, were found discolored, with the globules remarkably aggregated. Many had their contents much shrunk, and some were almost empty. In only a few cases were the pollen tubes omitted. There could be no doubt that the secretion had penetrated the outer coats of the grains and had partially digested their contents. So it must be with gastric juice of the insects which feed on pollen without masticating it. Drosera, in a state of nature, cannot fail to profit to a certain extent by this power of digesting pollen, as innumerable grains from the carices, grasses, rhumaces, fir trees, and other wind-fertilized plants, which commonly grow in the same neighborhood, will be inevitably caught by the viscid secretion surrounding the many glands. Gluten. This substance is composed of 2 albuminoids, one soluble, the other insoluble in alcohol. Some was prepared by merely washing wheaten flour in water. A provisional trial was made with rather large pieces placed on 2 leaves. These, after 21 HRS, were closely inflected and remained so for 4 days, when one was killed, and the other had its glands extremely blackened, but was not afterwards observed. Mr. O'Bennett found the undigested coats of the grains in the intestinal canal of pollinating Diptera. See Journal of Hort Soch of London, Vol. Roman 4 p. 158. Watt's Dict of Chemistry, Vol. Roman 2 p. 873 page 118. Smaller bits were placed on 2 leaves. These were only slightly inflected in 2 days, but afterwards became much more, so their secretion was not so strongly acid as that of leaves, excited by casein. The bits of gluten, after lying for 3 days on the leaves, were more transparent than other bits left for the same time in water. After 7 days, both leaves re-expanded, but the gluten seemed hardly at all reduced in bulk. The glands which had been in contact with it were extremely black. Still smaller bits of half-putrid gluten were now tried on 2 leaves. These were well inflected in 24 HRS and thoroughly in days, the glands in contact being much blackened. After 5 days 1 leaf began to re-expand, and after 8 days both were fully re-expanded, some gluten being still left on their discs. 4 little chips of dried gluten just dipped in water were next tried, and these acted rather differently from fresh gluten. 1 leaf was almost fully re-expanded in 3 days, and the other 3 leaves in 4 days. The chips were greatly softened, almost liquefied, but not nearly all dissolved. The glands, which had been in contact with them, instead of being much blackened, were of a very pale color, and many of them were evidently killed. In not one of these 10 cases was the whole of the gluten dissolved, even when very small bits were given. I therefore asked Dr. Burden Sanderson to try gluten in artificial digestive fluid of pepsin with hydrochloric acid, and this dissolved the whole. The gluten, however, was acted on much more slowly than fibrin. The proportion dissolved within 4 hours, being as 40 of gluten to 100 of fibrin. Gluten was also tried in 2 other digestive fluids, in which hydrochloric acid was replaced by propionic, page 119, and butyric acids, and it was completely dissolved by these fluids at the ordinary temperature of a room. Here, then, at last we have a case in which it appears that there exists an essential difference in digestive power between the secretion of Drosera and gastric juice, the difference being confined to the ferment, for as we have just seen, pepsin, in combination with acids of the acetic series, acts perfectly on gluten. I believe that the explanation lies simply in the fact that gluten is too powerful a stimulant, like raw meat or phosphate of lime, or even too large a piece of albumin, and that it injures or kills the glands before they have had time to pour forth a sufficient supply of the proper secretion. That some matter is absorbed from the gluten, we have clear evidence in the length of time during which the tentacles remain inflected, and in the greatly changed color of the glands. At the suggestion of Dr. Sanderson, some gluten was left for 15 HRS in weak hydrochloric acid, 2%, in order to remove the starch. It became colorless, more transparent, and swollen. Small portions were washed and placed on 5 leaves, which were soon closely inflected, but to my surprise re-expanded completely in 48 HRS. A mere vestige of gluten was left on 2 of the leaves, and not a vestige on the other 3, the viscid and acid secretion, which remained on the discs of the 3 latter leaves, was scraped off and examined by my son under a high power, but nothing could be seen except a little dirt and a good many starch grains, which had not been dissolved by the hydrochloric acid. Some of the glands were rather pale. We thus learn that gluten treated with weak hydrochloric acid is not so powerful or so enduring a page 120 stimulant as fresh gluten, and does not much injure the glands, and we further learn that it can be digested quickly and completely by the secretion. Globulin or crystalline, this substance was kindly prepared for me from the lens of the eye by Dr. Moore, and consisted of hard, colorless, transparent fragments. It is said that globulin ought to swell up in water and dissolve, for the most part forming a gummy liquid. But this did not occur with the above fragments, though kept in water for 4 days. Particles, some moistened with water, others with weak hydrochloric acid, others soaked in water for 1 or 2 days, were placed on 19 leaves. Most of these leaves, especially those with the long-soaked particles, became strongly inflected in a few hours. The greater number re-expanded after 3 or 4 days, but 3 of the leaves remained inflected during 1, 2, or 3 additional days. Hence some exciting matter must have been absorbed, but the fragments, though perhaps softened in a greater degree than those kept for the same time in water, retained all their angles as sharp as ever. As globulin is an albuminous substance, I was astonished at this result, and my object being to compare the action of the secretion with that of gastric juice, I asked Dr. Burden Sanderson to try some of the globulin used by me. He reports that it was subjected to a liquid containing 0% of hydrochloric acid and about 1% of glycerine extract of the stomach of a dog. It was then ascertained that this liquid was capable of digesting 1 of its weight of unboiled fibrin in 1 HR, whereas during the hour only 0 of the above globulin was dissolved. In both cases, an excess of the substance to be digested was subjected to the liquid. We thus see that within the same time less than 1/9 by weight of globulin than of fibrin was dissolved, and bearing in mind that pepsin with acids of the acetic series has only about 1/3 of the digestive power of pepsin with hydrochloric acid, it is not surprising that the fragments of Watt's Dictionary of Chemistry, Vol. Roman 2, p. 874. I may add that Dr. Sanderson prepared some fresh globulin by Schmidt's method, and of this zero was dissolved within the same time, namely 1 hour, so that it was far more soluble than that which I used, though less soluble than fibrin, of which, as we have seen, one was dissolved. I wish that I had tried on Drosera globulin, prepared by this method, page 121. Globulin were not corroded or rounded by the secretion of Drosera, though some soluble matter was certainly extracted from them and absorbed by the glands. Hematin, some dark red granules prepared from bullock's blood, were given me. These were found by Dr. Sanderson to be insoluble in water, acids, and alcohol, so that they were probably hematin together with other bodies derived from the blood particles, with little drops of water, were placed on 4 leaves, 3 of which were pretty closely inflected in 2 days, the 4th only moderately so. On the 3rd day the glands in contact with the hematin were blackened, and some of the tentacles seemed injured. After 5 days 2 leaves died, and the 3rd was dying. The 4th was beginning to re-expand, but many of its glands were blackened and injured. It is therefore clear that matter had been absorbed, which was either actually poisonous or of too stimulating a nature. The particles were much more softened than those kept for the same time in water, but judging by the eye, very little reduced in bulk. Dr. Sanderson tried this substance with artificial digestive fluid in the manner described under globulin, and found that whilst 1 of fibrin, only 0 of the hematin was dissolved in an hour, but the dissolution by the secretion of even a less amount would account for its action on Drosera. The residue left by the artificial digestive fluid at first yielded nothing more to it during several succeeding days. Substances which are not digested by the secretion. All the substances hitherto mentioned cause prolonged inflection of the tentacles, and are either completely or at least partially dissolved by the secretion. But there are many other substances, some of them containing nitrogen, which are not in the least acted on by the secretion, and do not induce inflection for a longer time than do inorganic and insoluble objects. These unexciting and indigestible substances are, as far as I have observed, epidermic productions, such as bits of human nails, balls of hair, the quills of feathers, fibroelastic tissue, mucin, pepsin, urea, chitin, chlorophyll, cellulose, gun cotton, fat, oil, and starch, page 122. To these may be added dissolved sugar and gum, diluted alcohol, and vegetable infusions, not containing albumin, for none of these, as shown in the last chapter, excite inflection. Now it is a remarkable fact, which affords additional and important evidence, that the ferment of Drosera is closely similar to or identical with pepsin, that none of these same substances are, as far as it is known, digested by the gastric juice of animals, though some of them are acted on by the other secretions of the alimentary canal. Nothing more need be said about some of the above enumerated substances, excepting that they were repeatedly tried on the leaves of Drosera, and were not in the least affected by the secretion. About the others, it will be advisable to give my experiments. Fibroelastic tissue. We have already seen that when little cubes of meat, Roman 100, were placed on leaves, the muscles, areolar tissue, and cartilage were completely dissolved, but the fibroelastic tissue, even the most delicate threads, were left without the least signs of having been attacked, and it is well known that this tissue cannot be digested by the gastric juice of animals. Mucin, as this substance contains about 7% of nitrogen, I expected that it would have excited the leaves greatly and been digested by the secretion, but in this I was mistaken from what is stated in chemical works. It appears extremely doubtful whether mucin can be prepared as a pure principle. That which I used, prepared by Dr. Moore, was dry, and hard particles moistened with water were placed on 4 leaves, but after 2 days there was only a trace of inflection in the immediately adjoining tentacles. These leaves were then tried with bits of meat, and all 4 soon became strongly inflected. Some of the dried mucin was then soaked in water for 2 days, and little cubes of the proper size were placed on 3 leaves. After 4 days, the tentacles See for instance, Skiff de la Digestion, 1867, Tom Roman 2p 38 page, 123. Round the margins of the discs were a little inflected, and the secretion collected on the disc was acid, but the exterior tentacles were not affected. One leaf began to re-expand on the 4th day, and all were fully re-expanded on the 6th. The glands which had been in contact with the mucin were a little darkened. We may therefore conclude that a small amount of some impurity of a moderately exciting nature had been absorbed, that the mucin employed by me did contain some soluble matter, was proved by Dr. Sanderson, who on subjecting it to artificial gastric juice, found that in 1 hr some was dissolved, but only in the proportion of 23 to 100 of fibrin. During the same time, the cubes, though perhaps rather softer than those left in water for the same time, retain their angles as sharp as ever. We may therefore infer that the mucin itself was not dissolved or digested, nor is it digested by the gastric juice of living animals, and according to Skiff, it is a layer of this substance which protects the coats of the stomach from being corroded during digestion. Pepsin. My experiments are hardly worth giving, as it is scarcely possible to prepare pepsin free from other albuminoids, but I was curious to ascertain, as far as that was possible, whether the ferment of the secretion of Drosera would act on the ferment of the gastric juice of animals. I first used the common pepsin sold for medicinal purposes, and afterwards some which was much purer, prepared for me by Dr. Moore. 5 leaves, to which a considerable quantity of the former was given, remained inflected for 5 days. 4 of them then died, apparently from too great stimulation. I then tried Dr. Moore's pepsin, making it into a paste with water, and placing such small particles on the discs of 5 leaves that all would have been quickly dissolved. Had it been meat or albumin, the leaves were soon inflected. 2 of them began to re-expand after only 20 hrs, and the other 3 were almost completely re-expanded after 44 hrs. Some of the glands which had been in contact with the particles of pepsin, or with the acid secretions surrounding them, were singularly pale, whereas others were singularly dark-colored. Some of the secretion was scraped off and examined under a high power, and it abounded with granules undistinguishable from those of pepsin left in water for the same length of time. We may therefore infer as highly probable, remembering what small quantities were given, that the ferment of Drosera does not act on or digest. Lecon's Phys de la Digestion, 1867, Tom Roman 2, p. 304 page, 124. Pepsin but absorbs from it some albuminous impurity, which induces inflection, and which in large quantity is highly injurious. Dr. Lauder Brunton, at my request, endeavored to ascertain whether pepsin with hydrochloric acid would digest pepsin, and as far as he could judge, it had no such power. Gastric juice therefore apparently agrees in this respect with the secretion of Drosera. Urea. It seemed to me an interesting inquiry whether this refuse of the living body, which contains much nitrogen, would, like so many other animal fluids and substances, be absorbed by the glands of Drosera and cause inflection. Half minim drops of a solution of 1 part to 437 of water were placed on the discs of 4 leaves, each drop containing the quantity usually employed by me, namely 1960 of a grain or 674 mg, but the leaves were hardly at all affected. They were then tested with bits of meat, and soon became closely inflected. I repeated the same experiment on 4 leaves, with some fresh urea prepared by Dr. Moore. After 2 days, there was no inflection. I then gave them another dose, but still there was no inflection. These leaves were afterwards tested with similarly sized drops of an infusion of raw meat, and in 6 hrs there was considerable inflection, which became excessive in 24 hrs. But the urea apparently was not quite pure, for when 4 leaves were immersed in 2 Dr. 7 Roman 1050 of the solution, so that all the glands, instead of merely those on the disc, were enabled to absorb any small amount of impurity in solution, there was considerable inflection after 24 hrs, certainly more than would have followed from a similar immersion in water. That the urea, which was not perfectly white, should have contained a sufficient quantity of albuminous matter, or of some salt of ammonia, to have caused the above effect, is far from surprising, for, as we shall see in the next chapter, astonishingly. Small doses of ammonia are highly efficient. We may therefore conclude that urea itself is not exciting or nutritious to Drosera, nor is it modified by the secretion so as to be rendered nutritious, for had this been the case, all the leaves with drops on their discs assuredly would have been well inflected. Dr. Lauder Brunton informs me that from experiments made at my request at St. Bartholomew's Hospital, it appears that urea is not acted on by artificial gastric juice, that is by pepsin with hydrochloric acid. Chitin, the chitinous coats of insects naturally captured by the leaves, do not appear in the least corroded small square pieces of the delicate wing, and of the elytron of a Staphylinus page 125 were placed on some leaves, and after these had re-expanded the pieces were carefully examined, their angles were as sharp as ever, and they did not differ in appearance from the other wing and elytron of the same insect, which had been left in water. The elytron, however, had evidently yielded some nutritious matter, for the leaf remained clasped over it for 4 days, whereas the leaves, with bits of the true wing, re-expanded on the second day. Anyone who will examine the excrement of insectivating animals will see how powerless their gastric juice is on chitin. Cellulose did not obtain this substance in a separate state, but tried angular bits of dry woodcork, sphagnum, moss, linen, and cotton thread. None of these bodies were in the least attacked by the secretion, and they caused only that moderate amount of inflection which is common to all inorganic objects. Gun cotton, which consists of cellulose with the hydrogen replaced by nitrogen, was tried with the same result. We have seen that a decoction of cabbage leaves excites the most powerful inflection. I therefore placed 2 little square bits of the blade of a cabbage leaf, and 4 little cubes cut from the midrib on 6 leaves of Drosera. These became well inflected in 12 HRS and remained so for between 2 and 4 days, the bits of cabbage being bathed all the time by acid secretion. This shows that some exciting matter, to which I shall presently refer, had been absorbed, but the angles of the squares and cubes remained as sharp as ever, proving that the framework of cellulose had not been attacked. Small square bits of spinach leaves were tried with the same result, the glands pouring forth a moderate supply of acid secretion, and the tentacles remaining inflected for 3 days. We have also seen that the delicate coats of pollen grains are not dissolved by the secretion. It is well known that the gastric juice of animals does not attack cellulose. Chlorophyll, this substance, was tried, as it contains nitrogen. Dr. Moore sent me some preserved in alcohol. It was dried, but soon deliquesced particles were placed on 4 leaves after 3 HRS. The secretion was acid. After 8 HRS, there was a good deal of inflection, which in 24 HRS became fairly well marked. After 4 days, 2 of the leaves began to open, and the other 2 were then almost fully re-expanded. It is therefore clear that this chlorophyll contained matter which excited the leaves to a moderate degree, but judging by the eye little or none was dissolved, so that in a pure state it would not probably have been attacked by the secretion. Dr. Sanderson tried that, which I page 126, used as well as some freshly prepared with artificial digestive liquid, and found that it was not digested. Dr. Lauder Brunton likewise tried some prepared by the process given in the British Pharmacopoeia, and exposed it for 5 days at the temperature of 37 cent to digestive liquid, but it was not diminished in bulk, though the fluid acquired a slightly brown color. It was also tried with the glycerine extract of pancreas, with a negative result, nor does chlorophyll seem affected by the intestinal secretions of various animals, judging by the color of their excrement. It must not be supposed from these facts that the grains of chlorophyll, as they exist in living plants, cannot be attacked by the secretion, for these grains consist of protoplasm merely colored by chlorophyll. My son Francis placed a thin slice of spinach leaf, moistened with saliva, on a leaf of Drosera, and other slices on damp cotton wool, all exposed to the same temperature. After 19 HRS, the slice on the leaf of Drosera was bathed in much secretion from the inflected tentacles, and was now examined under the microscope. No perfect grains of chlorophyll could be distinguished. Some were shrunken, of a yellowish-green color, and collected in the middle of the cells. Others were disintegrated and formed a yellowish mass likewise in the middle of the cells. On the other hand, in the slices surrounded by damp cotton wool. The grains of chlorophyll were green, and as perfect as ever. My son also placed some slices in artificial gastric juice, and these were acted on in nearly the same manner as by the secretion. We have seen that bits of fresh cabbage and spinach leaves cause the tentacles to be inflected, and the glands to pour forth much acid secretion, and there can be little doubt that it is the protoplasm forming the grains of chlorophyll, as well as that lining the walls of the cells, which excites the leaves. Fat and oil cubes of almost pure uncooked fat placed on several leaves did not have their angles in the least rounded. We have also seen that the oil globules in milk are not digested, nor does olive oil dropped on the discs of leaves cause any inflection. But when they are immersed in olive oil, they become strongly inflected. But to this subject, I shall have to recur. Oily substances are not digested by the gastric juice of animals. Starch, rather large bits of dry starch, caused well-marked inflection, and the leaves did not re-expand until the fourth day, but I have no doubt that this was due to the prolonged irritation of the glands. As the starch continued to absorb the secretion, the particles were not in the least reduced in size. Page 127, and we know that leaves immersed in an emulsion of starch are not at all affected. I need hardly say that starch is not digested by the gastric juice of animals. Action of the secretion on living seeds. The results of some experiments on living seeds, selected by hazard, may here be given, though they bear only indirectly on our present subject of digestion. 7 cabbage seeds of the previous year were placed on the same number of leaves. Some of these leaves were moderately, but the greater number only slightly inflected, and most of them re-expanded on the third day. One, however, remained clasped till the fourth, and another till the fifth day. These leaves, therefore, were excited somewhat more by the seeds than by inorganic objects of the same size. After they re-expanded, the seeds were placed under favorable conditions on damp sand, other seeds of the same lot being tried at the same time in the same manner, and found to germinate well. Of the 7 seeds which had been exposed to the secretion, only 3 germinated, and one of the 3 seedlings soon perished, the tip of its radical being from the first decayed, and the edges of its cotyledons of a dark brown color, so that altogether 5 out of the 7 seeds ultimately perished. Radish seeds, Raphanus sativus, of the previous year were placed on 3 leaves, which became moderately inflected and re-expanded on the third or fourth day. 2 of these seeds were transferred to damp sand, only 1 germinated, and that very slowly. This seedling had an extremely short, crooked, diseased radical with no absorbent hairs, and the cotyledons were oddly mottled with purple, with the edges blackened and partly withered. Cress seeds, Lepidum sativum, of the previous year were placed on 4 leaves. 2 of these next morning were moderately and 2 strongly inflected, and remained so for 4, 5, and even 6 days. Soon after these seeds were placed on the leaves and had become damp, they secreted in the usual manner a layer of tenacious mucus, and to ascertain whether it was the absorption of this substance by the glands which caused so much inflection, 2 seeds were put into water, and as much of the mucus as possible scraped off. They were then placed on leaves, which became very strongly inflected in the course of 3 hrs, and were still closely inflected on the third day, so that it evidently was not the mucus which excited. So page 128. Much inflection, on the contrary, this served to a certain extent as a protection to the seeds. 2 of the 6 seeds germinated while still lying on the leaves, but the seedlings, when transferred to damp sand, soon died. Of the other 4 seeds, only 1 germinated. 2 seeds of mustard, Sinapes nigra, 2 of celery, Apium graveolens, both of the previous year, 2 seeds well soaked of caraway, Carum carui, and 2 of wheat, did not excite the leaves more than inorganic objects often do. 5 seeds, hardly ripe, of a buttercup, Ranunculus, and 2 fresh seeds of anemone, Nemorosa, induced only a little more effect. On the other hand, 4 seeds, perhaps not quite ripe, of Carex sylvatica, caused the leaves on which they were placed to be very strongly inflected, and these only began to re-expand on the third day, 1 remaining inflected for 7 days. It follows from these few facts that different kinds of seeds excite the leaves in very different degrees. Whether this is solely due to the nature of their coats is not clear. In the case of the cress seeds, the partial removal of the layer of mucus hastened the inflection of the tentacles. Whenever the leaves remain inflected during several days over seeds, it is clear that they absorb some matter from them, that the secretion penetrates their coats, is also evident from the large proportion of cabbage, radish, and cress seeds which were killed, and from several of the seedlings being greatly injured. This injury to the seeds and seedlings may, however, be due solely to the acid of the secretion, and not to any process of digestion, for Mr. Traherne Magridge has shown that very weak acids of the acetic series are highly injurious to seeds. It never occurred to me to observe whether seeds are often blown on to the viscid leaves of plants growing in a state of nature, but this can hardly fail sometimes to occur, as we shall hereafter see in the case of Pinguicula. If so, Drosera will profit to a slight degree by absorbing matter from such seeds. A summary and concluding remarks on the digestive power of Drosera. When the glands on the disc are excited either by the absorption of nitrogenous matter or by mechanical irritation, their secretion increases in quantity and becomes acid. They likewise transmit, page 129, some influence to the glands of the exterior tentacles, causing them to secrete more copiously, and their secretion likewise becomes acid. With animals, according to Skiff, mechanical irritation excites the glands of the stomach to secrete an acid, but not pepsin. Now, I have every reason to believe, though the fact is not fully established, that although the glands of Drosera are continually secreting viscid fluid to replace that lost by evaporation, yet they do not secrete the ferment proper for digestion when mechanically irritated, but only after absorbing certain matter, probably of a nitrogenous nature. I infer that this is the case, as the secretion from a large number of leaves, which had been irritated by particles of glass placed on their discs, did not digest albumin, and more especially from the analogy of Dionea and Nepenthes. In like manner, the glands of the stomach of animals secrete pepsin, as Skiff asserts, only after they have absorbed certain soluble substances, which he designates as peptogenes. There is therefore a remarkable parallelism between the glands of Drosera and those of the stomach in the secretion of their proper acid and ferment. The secretion, as we have seen, completely dissolves albumin, muscle fibrin, areolar tissue, cartilage, the fibrous basis of bone, gelatin, chondrin, casein, in the state in which it exists in milk, and gluten, which has been subjected to weak hydrochloric acid, syntonin and legumin, excite the leaves so powerfully and quickly that there can hardly be a doubt that both would be dissolved by the secretion. The secretion. Phys de la Digestion, 1867, Tom Roman 2, pp. 188, 245, page 130. Failed to digest fresh gluten, apparently from its injuring the glands, though some was absorbed raw meat and less, in very small bits, and large pieces of albumin. Roman 100 likewise injure the leaves, which seem to suffer like animals from a surfeit. I know not whether the analogy is a real one, but it is worth notice that a decoction of cabbage leaves is far more exciting, probably nutritious to Drosera, than an infusion made with tepid water, and boiled cabbages are far more nutritious, at least to man, than the uncooked leaves. The most striking of all the cases, though not really more remarkable than many others, is the digestion of so hard and tough a substance as cartilage, the dissolution of pure phosphate of lime, of bone, dentine, and especially enamel. Seems wonderful, but it depends merely on the long-continued secretion of an acid, and this is secreted for a longer time under these circumstances than under any others. It was interesting to observe that as long as the acid was consumed in dissolving the phosphate of lime, no true digestion occurred, but that as soon as the bone was completely decalcified, the fibrous basis was attacked and liquefied with the greatest ease. The 12 substances above enumerated, which are completely dissolved by the secretion, are likewise dissolved by the gastric juice of the higher animals, and they are acted on in the same manner as shown by the rounding of the angles of albumin, and more especially by the manner in which the transverse striae of the fibers of muscle disappear. The secretion of Drosera and gastric juice were both able to dissolve some element or impurity out of the globulin and hematin employed by me. The secretion also dissolved something out of chemically, page 131, prepared casein, which is said to consist of 2 substances, and although Skiff asserts that casein in this state is not attacked by gastric juice, he might easily have overlooked a minute quantity of some albuminous matter which Drosera would detect and absorb again. Fibrocartilage, though not properly dissolved, is acted on in the same manner both by the secretion of Drosera and gastric juice. But this substance, as well as the so-called hematin used by me, ought perhaps to have been classed with indigestible substances. That gastric juice acts by means of its ferment pepsin solely in the presence of an acid is well established, and we have excellent evidence that a ferment is present in the secretion of Drosera, which likewise acts only in the presence of an acid, for we have seen that when the secretion is neutralized by minute drops of the solution of an alkali, the digestion of albumin is completely stopped, and that on the addition of a minute dose of hydrochloric acid, it immediately recommences. The 9 following substances, or classes of substances, namely epidermic productions, fibroelastic tissue, mucin, pepsin, urea, chitin, cellulose, guncotton, chlorophyll, starch, fat, and oil, are not acted on by the secretion of Drosera, nor are they, as far as is known, by the gastric juice of animals. Some soluble matter, however, was extracted from the mucin, pepsin, and chlorophyll used by me both by the secretion and by artificial gastric juice. The several substances which are completely dissolved by the secretion and which are afterwards absorbed by the glands affect the leaves rather differently. They induce inflection at very different page 132 rates and in very different degrees, and the tentacles remain inflected for very different periods of time. Quick inflection depends partly on the quantity of the substance given, so that many glands are simultaneously affected, partly on the facility with which it is penetrated and liquefied by the secretion, partly on its nature, but chiefly on the presence of exciting matter already in solution. Thus saliva, or a weak solution of raw meat, acts much more quickly than even a strong solution of gelatine. So again, leaves which have re-expanded after absorbing drops of a solution of pure gelatine or isinglass, the latter being the more powerful of the two, if given bits of meat, are inflected much more energetically and quickly than they were before, notwithstanding that some rest is generally requisite between 2 acts of inflection. We probably see the influence of texture in gelatine and globulin, when softened by having been soaked in water, acting more quickly than when merely wetted. It may be partly due to changed texture, and partly to changed chemical nature, that albumin, which had been kept for some time, and gluten, which had been subjected to weak hydrochloric acid, act more quickly than these substances in their fresh state. The length of time during which the tentacles remain inflected largely depends on the quantity of the substance given, partly on the facility with which it is penetrated or acted on by the secretion, and partly on its essential nature. The tentacles always remain inflected much longer over large bits or large drops than over small bits or drops. Texture probably plays a part in determining the extraordinary length of time during which the tentacles remain inflected. Page 133, over the hard grains of chemically prepared casein, but the tentacles remain inflected for an equally long time over finely powdered precipitated phosphate of lime phosphorus, in this latter case evidently being the attraction and animal matter. In the case of casein, the leaves remain long inflected over insects, but it is doubtful how far this is due to the protection afforded by their chitinous integuments, for animal matter is soon extracted from insects, probably by exosmosis from their bodies, into the dense surrounding secretion, as shown by the prompt inflection of the leaves. We see the influence of the nature of different substances in bits of meat, albumin, and fresh gluten, acting very differently from equal-sized bits of gelatine, areolar tissue, and the fibrous basis of bone. The former cause not only far more prompt and energetic, but more prolonged inflection than do the latter. Hence, we are, I think, justified in believing that gelatine, areolar tissue, and the fibrous basis of bone would be far less nutritious to Drosera than such substances as insects' meat, albumin. Roman 100. This is an interesting conclusion, as it is known that gelatine affords but little nutriment to animals, and so probably would areolar tissue, and the fibrous basis of bone. The chondrin which I used acted more powerfully than gelatine, but then I do not know that it was pure. It is a more remarkable fact that fibrin, which belongs to the great class of proteids, including albumin, in one of its subgroups does not excite the tentacles in a greater degree, or keep them inflected for a longer time than does gelatine or. See the classification adopted by Dr. Michael Foster in Watt's Dictionary of Chemistry Supplement, 1872, page 969, page 134. Areolar tissue, or the fibrous basis of bone. It is not known how long an animal would survive if fed on fibrin alone, but Dr. Sanderson has no doubt longer than on gelatine, and it would be hardly rash to predict, judging from the effects on Drosera, that albumin would be found more nutritious than fibrin. Globulin likewise belongs to the proteids, forming another subgroup, and this substance, though containing some matter which excited Drosera rather strongly, was hardly attacked by the secretion, and was very little or very slowly attacked by gastric juice. How far globulin would be nutritious to animals is not known. We thus see how differently the above specified several digestible substances act on Drosera, and we may infer as highly probable that they would in like manner be nutritious in very different degrees, both to Drosera and to animals. The glands of Drosera absorb matter from living seeds, which are injured or killed. By the secretion, they likewise absorb matter from pollen and from fresh leaves, and this is notoriously the case with the stomachs of vegetable-feeding animals. Drosera is properly an insectivorous plant, but as pollen cannot fail to be often blown on to the glands, as will occasionally the seeds and leaves of surrounding plants, Drosera is to a certain extent a vegetable feeder. Finally, the experiments recorded in this chapter show us that there is a remarkable accordance in the power of digestion between the gastric juice of animals with its pepsin and hydrochloric acid, and the secretion of Drosera with its ferment and acid belonging to the acetic series. We can therefore hardly doubt that the ferment in both cases is closely similar, page 135, if not identically the same. That a plant and an animal should pour forth the same or nearly the same complex secretion adapted for the same purpose of digestion is a new and wonderful fact in physiology, but I shall have to recur to this subject in the 15th chapter in my concluding remarks on the Drosera. See page 136. Chapter Roman 7, the effects of salts of ammonia. Manner of performing the experiments. Action of distilled water in comparison with the solution's carbonate of ammonia, absorbed by the roots. The vapor absorbed by the glands. Drops on the disc. Minute drops applied to separate glands. Leaves immersed in weak solutions. Minuteness of the doses which induce aggregation of the protoplasm. Nitrate of ammonia. Analogous experiments with phosphate of ammonia. Analogous experiments with other salts of ammonia. Summary and concluding remarks on the action of salts of ammonia. The chief object in this chapter is to show how powerfully the salts of ammonia act on the leaves of Drosera, and more especially to show what an extraordinarily small quantity suffices to excite inflection. I shall therefore be compelled to enter into full details. Doubly distilled water was always used, and for the more delicate experiments water, which had been prepared with the utmost possible care, was given me by Professor Frankland. The graduated measures were tested and found as accurate as such measures can be. The salts were carefully weighed, and in all the more delicate experiments by Borda's double method. But extreme accuracy would have been superfluous, as the leaves differ greatly in irritability, according to age, condition, and constitution. Even the tentacles on the same leaf differ in irritability to a marked degree. My experiments were tried in the following several ways. Firstly, drops which were ascertained by repeated trials to be on an average about half a minim, or the 1960 of a fluid, ounce 296 Roman 1050, were placed by the same pointed instrument on the page 137 discs of the leaves, and the inflection of the exterior rows of tentacles observed at successive intervals of time. It was first ascertained from between 30 and 40 trials that distilled water dropped in this manner produces no effect, except that sometimes, though rarely, 2 or 3 tentacles become inflected. In fact, all the many trials with solutions which were so weak as to produce no effect lead to the same result, that water is inefficient. Secondly, the head of a small pin fixed into a handle was dipped into the solution under trial. The small drop which adhered to it, and which was much too small to fall off, was cautiously placed by the aid of a lens in contact with the secretion surrounding the glands of 1, 2, 3, or 4 of the exterior tentacles of the same leaf. Great care was taken that the glands themselves should not be touched, I had supposed that the drops were of nearly the same size, but on trial this proved a great mistake. I first measured some water and removed 300 drops, touching the pin's head each time on blotting paper, and on again measuring the water, a drop was found to equal on an average about the 160 of a minim. Some water in a small vessel was weighed, and this is a more accurate method, and 300 drops removed as before, and on again weighing the water, a drop was found to equal, on an average, only the 189 of a minim. I repeated the operation, but endeavored this time, by taking the pin's head out of the water obliquely, and rather quickly to remove as large drops as possible, and the result showed that I had succeeded, for each drop, on an average, equaled 119 of a minim. I repeated the operation in exactly the same manner, and now the drops averaged 123 of a minim, bearing in mind that on these two latter occasions special pains were taken to remove as large drops as possible. We may safely conclude that the drops used in my experiments were at least equal to the 120 of a minim, or 29 Roman 1050. One of these drops could be applied to 3 or even 4 glands, and if the tentacles became inflected, some of the solution must have been absorbed by all, for drops of pure water applied in the same manner never produced any effect. I was able to hold the drop in steady contact with the secretion only for 10 to 15 seconds, and this was not time enough for the diffusion of all the salt in solution, as was evident from 3 or 4 tentacles treated successively with the same drop often becoming inflected. All the matter in solution was even then probably not exhausted. Thirdly, leaves cut off and immersed in a measured page 138 quantity of the solution. Under trial, the same number of leaves being immersed at the same time in the same quantity of the distilled water, which had been used in making the solution. The leaves in the two lots were compared at short intervals of time, up to 24 HRS, and sometimes to 48 HRS. They were immersed by being laid as gently as possible in numbered watchglasses, and 30 minims 1 Roman 1050 of the solution or of water was poured over each. Some solutions, for instance that of carbonate of ammonia, quickly discolor the glands, and as all on the same leaf were discolored simultaneously, they must all have absorbed some of the salt within the same short period of time. This was likewise shown by the simultaneous inflection of the several exterior rows of tentacles. If we had no such evidence as this, it might have been supposed that only the glands of the exterior and inflected tentacles had absorbed the salt, or that only those on the disc had absorbed it, and had then transmitted a motor impulse to the exterior tentacles. But in this latter case, the exterior tentacles would not have become inflected until some time had elapsed, instead of within half an hour or even within a few minutes, as usually occurred. All the glands on the same leaf are of nearly the same size, as may best be seen by cutting off a narrow transverse strip and laying it on its side. Hence their absorbing surfaces are nearly equal. The long-headed glands on the extreme margin must be accepted, as they are much longer than the others, but only the upper surface is capable of absorption. Besides the glands, both surfaces of the leaves and the pedicels of the tentacles bear numerous minute papillae, which absorb carbonate of ammonia, an infusion of raw meat, metallic salts, and probably many other substances. But the absorption of matter by these papillae never induces inflection. We must remember that the movement of each separate tentacle depends on its gland being excited, except when a motor impulse is transmitted from the glands of the disc, and then the movement, as just stated, does not take place until some little time has elapsed. I have made these remarks because they show us that when a leaf is immersed in a solution and the tentacles are inflected, we can judge with some accuracy how much of the salt each gland has absorbed. For instance, if a leaf bearing 212 glands be immersed in a measured quantity of a solution containing 110 of a grain of a salt, and all the exterior tentacles except 12 are inflected, we may feel sure that each of the 200 glands can on an average have absorbed at most 12,000 of a grain of the salt. I say at page 139, most, for the papillae will have absorbed some small amount, and so will perhaps the glands of the 12 excluded tentacles, which did not become inflected. The application of this principle leads to remarkable conclusions with respect to the minuteness of the doses causing inflection. On the action of distilled water in causing inflection. Although in all the more important experiments, the difference between the leaves simultaneously immersed in water and in the several solutions will be described, nevertheless, it may be well here to give a summary of the effects of water. The fact, moreover, of pure water acting on the glands deserves in itself some notice. Leaves, to the number of 141, were immersed in water at the same time with those in the solutions, and their state recorded at short intervals of time. 32 other leaves were separately observed in water, making altogether 173 experiments. Many scores of leaves were also immersed in water at other times, but no exact record of the effects produced was kept, yet these cursory observations support the conclusions arrived at in this chapter. A few of the long-headed tentacles, namely from 1 to about 6, were commonly inflected within half an hour after immersion, as were occasionally a few and rarely a considerable number of the exterior round-headed tentacles after an immersion of from 5 to 8 HRS. The short tentacles surrounding the outer parts of the disc generally become inflected so that their glands form a small dark ring on the disc, the exterior tentacles not partaking of this movement. Hence, excepting in a few cases hereafter to be specified, we can judge whether a solution produces any effect only by observing the exterior tentacles within the first 3 or 4 HRS after immersion. Now for a summary of the state of the 173 leaves after an immersion of 3 or 4 HRS in pure water. 1 leaf had almost all its tentacles inflected. 3 leaves had most of them subinflected, and 13 had on an average 36 tentacles inflected. Thus 17 leaves out of the 173 were acted on in a marked manner. 18 leaves had from 7 to 19 tentacles inflected, the average being 9 tentacles for each leaf. 4 to 4 leaves had from 1 to 6 tentacles inflected, generally the long-headed ones, so that altogether of the 173 leaves carefully observed, 17 were affected by the water in some degree, though commonly to a very slight degree, and 9 to 4 were not affected in the least degree. This page 140 amount of inflection is utterly insignificant, as we shall hereafter see, compared with that caused by very weak solutions of several salts of ammonia. Plants which have lived for some time in a rather high temperature are far more sensitive to the action of water than those grown out of doors or recently brought into a warm greenhouse. Thus, in the above 17 cases in which the immersed leaves had a considerable number of tentacles inflected, the plants had been kept during the winter in a very warm greenhouse, and they bore in the early spring remarkably fine leaves of a light red color. Had I then known that the sensitiveness of plants was thus increased, perhaps I should not have used the leaves for my experiments with the very weak solutions of phosphate of ammonia, but my experiments are not thus vitiated, as I invariably used leaves from the same plants for simultaneous immersion in water. It often happened that some leaves on the same plant and some tentacles on the same leaf were more sensitive than others, but why this should be so, I do not know. Fig. 9. Drosera rotundifolia, leaf enlarged, with all the tentacles closely inflected from immersion, in a solution of phosphate of ammonia 1 part to 87,500 of water. Besides the differences just indicated between the leaves immersed in water and in weak solutions of ammonia, the tentacles of the latter are in most cases much more closely inflected. The appearance of a leaf after immersion in a few drops of a solution of 1 grain of phosphate of ammonia to 200 Rs of water, Roman 1 E1, part to 87,500, is here reproduced. Such energetic inflection is never caused by water alone. With leaves in the weak solutions, the blade or lamina often becomes inflected, and this is so rare a circumstance with leaves in water that I have seen only 2 instances, and in both of these the inflection was very feeble. Again, with leaves in the weak solutions, the inflection of the tentacles and blade often goes on steadily, though slowly increasing during many hours, and page 141. This again is so rare a circumstance with leaves in water that I have seen only 3 instances of any such increase after the first 8 to 12 HRS. and in these three instances, the two outer rows of tentacles were not at all affected. Hence, there is sometimes a much greater difference between the leaves in water and in the weak solutions after from 8 HRS to 24 HRS than there was within the first 3 HRS, though as a general rule, it is best to trust to the difference observed within the shorter time. With respect to the period of the re-expansion of the leaves, when left immersed either in water or in the weak solutions, nothing could be more variable in both cases. The exterior tentacles not rarely begin to re-expand after an interval of only from 6 to 8 HRS, that is just about the time when the short tentacles round the borders of the disc become inflected. On the other hand, the tentacles sometimes remain inflected for a whole day or even 2 days, but as a general rule, they remain inflected for a longer period in very weak solutions than in water. In solutions which are not extremely weak, they never re-expand within nearly so short a period as 6 or 8 hours. From these statements, it might be thought difficult to distinguish between the effects of water and the weaker solutions, but in truth, there is not the slightest difficulty until excessively weak solutions are tried, and then the distinction, as might be expected, becomes very doubtful and at last disappears. But as in all except the simplest cases, the state of the leaves simultaneously immersed for an equal length of time in water and in the solutions will be described, the reader can judge for himself. Carbonate of ammonia. This salt, when absorbed by the roots, does not cause the tentacles to be inflected. A plant was so placed in a solution of 1 part of the carbonate to 146 of water that the young uninjured roots could be observed. The terminal cells, which were of a pink color, instantly became colorless, and their limpid contents cloudy like a mezzotinto engraving, so that some degree of aggregation was almost instantly caused. But no further change ensued, and the absorbent hairs were not visibly affected. The tentacles, page 142, did not bend. 2 other plants were placed with their roots surrounded by damp moss in half an ounce 14 Roman 1050 of a solution of 1 part of the carbonate to 218 of water, and were observed for 24 HRS, but not a single tentacle was inflected. In order to produce this effect, the carbonate must be absorbed by the glands. The vapor produces a powerful effect on the glands and induces inflection. 3 plants with their roots in bottles, so that the surrounding air could not have become very humid, were placed under a bell glass holding 122 fluid ounces together with 4 grains of carbonate of ammonia in a watch glass after an interval of 6 HRS 15 Roman 1000. The leaves appeared unaffected, but next morning, after 20 HRS, the blackened glands were secreting copiously, and most of the tentacles were strongly inflected. These plants soon died. 2 other plants were placed under the same bell glass together, with half a grain of the carbonate, the air being rendered as damp as possible, and in 2 HRS, most of the leaves were affected, many of the glands being blackened, and the tentacles inflected. But it is a curious fact that some of the closely adjoining tentacles on the same leaf, both on the disc and round the margins, were much, and some apparently not in the least affected. The plants were kept under the bell glass for 24 HRS, but no further change ensued. 1 healthy leaf was hardly at all affected, though other leaves on the same plant were much affected. On some leaves all the tentacles on one side, but not those on the opposite side, were inflected. I doubt whether this extremely unequal action can be explained by supposing that the more active glands absorb all the vapor as quickly as it is generated, so that none is left for the others, for we shall meet, with page 143, analogous cases, with air thoroughly permeated with the vapors of chloroform and ether. Minute particles of the carbonate were added to the secretions surrounding several glands. These instantly became black and secreted copiously, but except in 2 instances, when extremely minute particles were given, there was no inflection. This result is analogous to that which follows. From the immersion of leaves in a strong solution of 1 part of the carbonate to 109 or 146 or even 218 of water, for the leaves are then paralyzed, and no inflection ensues, though the glands are blackened, and the protoplasm in the cells of the tentacles undergoes strong aggregation. We will now turn to the effects of solutions of the carbonate. Half minims of a solution of 1 part to 437 of water were placed on the discs of 12 leaves, so that each received 1960 of a grain or 675 mg. 10 of these had their tentacles well inflected, the blades of some being also much curved inwards. In 2 cases, several of the exterior tentacles were inflected in 35 Roman 1000, but the movement was generally slower. These 10 leaves re-expanded in periods varying between 21 HRS and 45 HRS, but in one case not until 67 HRS had elapsed, so that they re-expanded much more quickly than leaves which have caught insects. The same size drops of a solution of 1 part to 875 of water were placed on the discs of 11 leaves. 6 remained quite unaffected, whilst 5 had from 3 to 6 or 8 of their exterior tentacles inflected, but this degree of movement can hardly be considered as trustworthy. Each of these leaves received 11,920 of a grain 337 mg distributed between the glands of the disc, but this was too small an amount to produce any decided effect on the exterior tentacles, the glands of which had not themselves received any of the salt. Minute drops on the head of a small pin of a solution of 1 part of the carbonate to 218 of water were next tried in the manner above described. A drop of this kind equals, on an average, 120 of a minim, and therefore contains 14,800 of a grain 135 mg of the carbonate. I touched with it the viscid secretion round 3 glands, so that each gland received only page 144, 114,400. A grain 445 mg. Nevertheless, in 2 trials, all the glands were plainly blackened. In one case, all 3 tentacles were well inflected, after an interval of 2 HRS 40 Roman 1000, and in another case, 2 of the 3 tentacles were inflected. I then tried drops of a weaker solution of 1 part to 292 of water on 24 glands, always touching the viscid secretion round 3 glands with the same little drop. Each gland, thus, received only the 119,200 of a grain 337 mg, yet some of them were a little darkened, but in no one instance were any of the tentacles inflected, though they were watched for 12 HRS, when a still weaker solution, viz. 1 part to 437 of water, was tried on 6 glands. No effect whatever was perceptible. We thus learn that the 114,400 of a grain 445 mg of carbonate of ammonia, if absorbed by a gland, suffices to induce inflection in the basal part of the same tentacle. But as already stated, I was able to hold with a steady hand the minute drops in contact with the secretion only for a few seconds, and if more time had been allowed for diffusion and absorption, a much weaker solution would certainly have acted. Some experiments were made by immersing cut-off leaves in solutions of different strengths. Thus 4 leaves were left for about 3 HRS each in a dram 3 Roman 1050 of a solution of 1 part of the carbonate to 5250 of water. 2 of these had almost every tentacle inflected. The third had about half the tentacles, and the fourth about 1/3 inflected, and all the glands were blackened. Another leaf was placed in the same quantity of a solution of 1 part to 7,000 of water, and in 1 HR 16 Roman 1000 every single tentacle was well inflected, and all the glands blackened. 6 leaves were immersed each in 30 minims 1 Roman 1050 of a solution of 1 part to 4375 of water, and the glands were all blackened in 31 Roman 1000. All 6 leaves exhibited some slight inflection, and 1 was strongly inflected. 4 leaves were then immersed in 30 minims of a solution of 1 part to 8750 of water, so that each leaf received the 1320 of a grain 2025 mg. Only 1 became strongly inflected, but all the glands on all the leaves were of so dark a red after 1 hour as almost to deserve to be called black, whereas this did not occur with the leaves, which were at the same time immersed in water, nor did water produce this effect on any other occasion in nearly so short a time as an hour. These cases of the simultaneous darkening or blackening of the glands from the action of weak solutions are important, as they show that all the glands absorbed the carbonate within the same time, which fact indeed there was not the least reason to doubt. So again, whenever all the page 145 tentacles become inflected within the same time, we have evidence as before remarked of simultaneous absorption, I did not count the number of glands on these 4 leaves, but as they were fine ones, and as we know that the average number of glands on 31 leaves was 192, we may safely assume that each bore on an average at least 170, and if so, each blackened gland could have absorbed only 154,400 of a grain 119 mg of the carbonate. A large number of trials had been previously made with solutions of 1 part of the nitrate and phosphate of ammonia to 43,750 of water Roman 1 E1 grain to 100 ounces, and these were found highly efficient. 14 leaves were therefore placed each in 30 minims of a solution of 1 part of the carbonate to the above quantity of water, so that each leaf received 11,600 of a grain 405 mg. The glands were not much darkened. 10 of the leaves were not affected, or only very slightly so. 4, however, were strongly affected, the first having all the tentacles, except 40 inflected in 47 Roman 1000 in 6, HRS 30 Roman 1000, all except 8, and after 4. HRS the blade itself, the second leaf, after 9 Roman 1000, had all its tentacles, except 9 inflected, after 6 HRS 30 Roman 1000, these 9 were subinflected, the blade having become much inflected in 4 HRS, the third leaf. After 1 HR 6 Roman 1000, had all but 40 tentacles inflected, the fourth, after 2 HRS 5 Roman 1000, had about half its tentacles, and after 4 HRS, all but 4 to 5 inflected leaves, which were immersed in water at the same time, were not at all affected, with the exception of 1, and this not until 8 HRS had elapsed. Hence, there can be no doubt that a highly sensitive leaf, if immersed in a solution, so that all the glands are enabled to absorb, is acted on by 11,600 of a grain of the carbonate, assuming that the leaf, which was a large one, and which had all its tentacles, excepting 8 inflected, bore 170 glands, each gland could have absorbed only 1,268,800 of a grain 24 mg, yet this sufficed to act on each of the 162 tentacles which were inflected. But as only 4 out of the above 14 leaves were plainly affected, this is nearly the minimum dose which is efficient. Aggregation of the protoplasm from the action of carbonate of ammonia. I have fully described in the 3rd chapter the remarkable effects of moderately strong doses of this salt in causing the aggregation of the protoplasm within the cells of the glands and tentacles, and here my object is merely to show what small doses suffice. A leaf was immersed in 20 minims 1 Roman 1050 of a solution of 1 part to 1750 of water, page 146, and another leaf in the same quantity of a solution of 1 part to 3062. In the former case, aggregation occurred in 4 Roman 1000, in the latter in 11 Roman 1000. A leaf was then immersed in 20 minims of a solution of 1 part to 4375 of water, so that it received 1240 of a grain 27 mg. In 5 Roman 1000, there was a slight change of color in the glands, and in 15 Roman 1000, small spheres of protoplasm were formed in the cells beneath the glands of all the tentacles. In these cases, there could not be a shadow of a doubt about the action of the solution. A solution was then made of 1 part to 5250 of water, and I experimented on 14 leaves, but will give only a few of the cases. 8 young leaves were selected and examined with care, and they showed no trace of aggregation. 4 of these were placed in a dram 3 Roman 1050 of distilled water, and 4 in a similar vessel, with a dram of the solution. After a time, the leaves were examined under a high power, being taken alternately from solution and the water. The first leaf was taken out of the solution after an immersion of 2 HRS 40 Roman 1000, and the last leaf out of the water after 3 HRS 50 Roman 1000, the examination lasting for 1 HR 40 Roman 1000. In the 4 leaves out of the water, there was no trace of aggregation, except in 1 specimen, in which a very few extremely minute spheres of protoplasm were present beneath some of the round glands. All the glands were translucent and red. The 4 leaves which had been immersed in the solution, besides being inflected, presented a widely different appearance, for the contents of the cells of every single tentacle on all 4 leaves were conspicuously aggregated the spheres and elongated masses of protoplasm, in many cases extending halfway down the tentacles. All the glands, both those of the central and exterior tentacles, were opaque and blackened, and this shows that all had absorbed some of the carbonate. These 4 leaves were very nearly the same size, and the glands were counted on one and found to be 167, this being the case, and the 4 leaves having been immersed in a dram of the solution. Each gland could have received on an average only 164,128 of a grain 1009 mg of the salt, and this quantity sufficed to induce, within a short time, conspicuous aggregation in the cells beneath all the glands. A vigorous but rather small red leaf was placed in 6 minims of the same solution, viz. 1 part to 5250 of water, so that it received 1960 of a grain 675 mg in 40 Roman 1000. The glands appeared rather darker, and in 1 hr from 4 to 6 spheres of protoplasm were formed in the cells beneath the glands of all the tentacles. I did not count the tentacles, but we may, page 147, safely assume that there were at least 140, and if so, each gland could have received only the 1,134,400 of a grain, or 48 mg. A weaker solution was then made of 1 part to 7,000 of water, and 4 leaves were immersed in it, but I will give only one case. A leaf was placed in 10 minims of this solution. After 1 hr 37 Roman 1000, the glands became somewhat darker, and the cells beneath all of them now contained many spheres of aggregated protoplasm. This leaf received 1768 of a grain and bore 166 glands. Each gland could therefore have received only 1,127,488 of a grain 507 mg of the carbonate. 2 other experiments are worth giving. A leaf was immersed for 4 HRS 15 Roman 1000 in distilled water, and there was no aggregation. It was then placed for 1 HR 15 Roman 1000 in a little solution of 1 part to 5250 of water, and this excited well-marked aggregation and inflection. Another leaf, after having been immersed for 21 HRS 15 Roman 1000 in distilled water, had its glands blackened, but there was no aggregation in the cells beneath them. It was then left in 6 minims of the same solution, and in 1 HR there was much aggregation in many of the tentacles. In 2 HRS, all the tentacles, 146 in number, were affected aggregation, extending down for a length equal to half or the whole of the glands. It is extremely improbable that these 2 leaves would have undergone aggregation if they had been left for a little longer in the water, namely for 1 HR and 1 HR 15 Roman 1000, during which time they were immersed in the solution, for the process of aggregation seems invariably to supervene slowly and very gradually in water. A summary of the results: with carbonate of ammonia, the roots absorbed the solution as shown by their changed color, and by the aggregation of the contents of their cells, the vapor is absorbed by the glands. These are blackened, and the tentacles are inflected. The glands of the disc, when excited by a half-minim drop, 296 Roman 1050, containing 1960 of a grain 675 mg, transmit a motor impulse to the exterior tentacles, causing them to bend inwards. A minute drop, containing 114,400 of a grain 445 mg, if held for a few seconds in contact with a gland, soon causes the tentacle bearing it to be inflected. If a leaf is left, page 148, immersed for a few hours in a solution, and a gland absorbs the 1,134,400 of a grain 48 mg, its color becomes darker, though not actually black, and the contents of the cells beneath the gland are plainly aggregated. Lastly, under the same circumstances, the absorption by a gland of the 1,268,800 of a grain 24 mg suffices to excite the tentacle bearing this gland into movement. Nitrate of ammonia. With the salt I attended only to the inflection of the leaves, for it is far less efficient than the carbonate in causing aggregation, although considerably more potent in causing inflection. I experimented with half-minims 296 Roman 1050 on the discs of 52 leaves, but will give only a few cases a solution of 1 part to 109 of water was too strong, causing little inflection, and after 24 HRS killing, or nearly killing 4 out of 6 leaves, which were thus tried, each of which received the 1240 of a grain, or 27 mg, a solution of 1 part to 218 of water acted most energetically, causing not only the tentacles of all the leaves, but the blades of some to be strongly inflected. 14 leaves were tried with drops of a solution of 1 part to 875 of water, so that the disc of each received the 11,920 of a grain. 337 mg of these leaves, 7 were very strongly acted on the edges, being generally inflected. 2 were moderately acted on, and 5 not at all. I subsequently tried 3 of these latter 5 leaves with urine, saliva, and mucus, but they were only slightly affected, and this proves that they were not in an active condition. I mention this fact to show how necessary it is to experiment on several leaves. 2 of the leaves, which were well inflected, re-expanded after 51 HRS. In the following experiment, I happened to select very sensitive leaves. Half-minims of a solution of 1 part to 1094 of water, Roman 1 E1 GR to 212 Rs, were placed on the discs of 9 leaves, so that each received the 12,400 of a grain. 27 mg. 3 of them had their tentacles strongly inflected, and their blades curled inwards. 5 were slightly and somewhat doubtfully affected, having from 3 to 8 of their exterior tentacles inflected. 1 leaf was not at all affected, yet was afterwards acted on by saliva. In 6 of these cases, a trace of action was perceptible in page 149, 7 HRS, but the full effect was not produced until from 24 HRS to 30 HRS had elapsed. 2 of the leaves, which were only slightly inflected, re-expanded after an additional interval of 19 HRS. Half-minims of a rather weaker solution, viz. of 1 part to 1312 of water, 1 GR to 3 Rs, were tried on 14 leaves, so that each received 12,880 of a grain. 225 mg, instead of, as in the last experiment, 12,400 of a grain. The blade of 1 was plainly inflected, as were 6 of the exterior tentacles. The blade of a second was slightly, and 2 of the exterior tentacles well inflected, all the other tentacles being curled in at right angles to the disc. 3 other leaves had from 5 to 8 tentacles inflected, 5 others only 2 or 3, and occasionally, though very rarely, drops of pure water caused this much action. The 4 remaining leaves were in no way affected, yet 3 of them, when subsequently tried with urine, became greatly inflected. In most of these cases, a slight effect was perceptible in from 6 HRS to 7 HRS, but the full effect was not produced until from 24 HRS to 30 HRS had elapsed. It is obvious that we have here reached very nearly the minimum amount, which distributed between the glands of the disc acts on the exterior tentacles, these having themselves not received any of the solution. In the next place, the viscid secretion round 3 of the exterior glands was touched with the same little drop, 120 of a minim, of a solution of 1 part to 437 of water, and after an interval of 2 HRS 50 Roman 1000, all 3 tentacles were well inflected. Each of these glands could have received only the 128,800 of a grain, or 225 mg. A little drop of the same size and strength was also applied to 4 other glands, and in 1 HR2 became inflected, whilst the other 2 never moved. We here see, as in the case of the half-minims placed on the discs, that the nitrate of ammonia is more potent in causing inflection than the carbonate. For minute drops of the latter salt of this strength produced no effect. I tried minute drops of a still weaker solution of the nitrate, viz. 1 part to 875 of water on 21 glands, but no effect whatever was produced, except perhaps in 1 instance. 6 to 3 leaves were immersed in solutions of various strengths, other leaves being immersed at the same time in the same pure water used in making the solutions. The results are so remarkable, though less so than with phosphate of ammonia, that I must describe the experiments in detail, but I will give only a few. In speaking of the successive periods when inflection occurred, I always reckon from the time of first immersion, page 150. Having made some preliminary trials as a guide 5 leaves were placed in the same little vessel in 30 minims of a solution of 1 part of the nitrate to 7875 of water 1 gr to 18 Rs, and this amount of fluid just sufficed to cover them. After 2 HRS 10 Roman 1003 of the leaves were considerably inflected, and the other 2 moderately, the glands of all became of so dark a red as almost to deserve to be called black. After 8 HRS 4 of the leaves had all their tentacles more or less inflected, whilst the 5th, which I then perceived to be an old leaf, had only 30 tentacles inflected next morning. After 23 HRS 40 Roman 1000, all the leaves were in the same state, excepting that the old leaf had a few more tentacles inflected. 5 leaves, which had been placed at the same time in water, were observed at the same intervals of time. After 2 HRS 10 Roman 1002 of them had 4 1 had 7 1 had 10 of the long-headed marginal tentacles, and the 5th had 4 round-headed tentacles inflected. After 8 HRS, there was no change in these leaves, and after 24 HRS, all the marginal tentacles had re-expanded, but in one leaf a dozen, and in a second leaf half a dozen submarginal tentacles had become inflected, as the glands of the 5 leaves in the solution were simultaneously darkened. No doubt, they had all absorbed a nearly equal amount of the salt, and as 1288 of a grain was given to the 5 leaves together, each got 11,440 of a grain 45 mg. I did not count the tentacles on these leaves, which were moderately fine ones, but as the average number on 31 leaves was 192, it would be safe to assume that each bore on an average at least 160. If so, each of the darkened glands could have received only 1,230,400 of a grain of the nitrate, and this caused the inflection of a great majority of the tentacles. This plan of immersing several leaves in the same vessel is a bad one, as it is impossible to feel sure that the more vigorous leaves do not rob the weaker ones of their share of the salt. The glands, moreover, must often touch one another or the sides of the vessel, and movement may have been thus excited, but the corresponding leaves in water, which were little inflected, though rather more so than commonly occurs, were exposed in an almost equal degree to these same sources of error. I will therefore give only one other experiment made in this manner, though many were tried, and all confirmed the foregoing and following results. 4 leaves were placed in 40 minims of a solution of 1 part to 10,500 of water, and assuming that they absorbed equally, each leaf received 11,152 of a grain 562 mg. After 1 HR 20 Roman 1000, many of the tentacles on all 4 leaves were somewhat inflected. After page 151.5, HRS 30 Roman 1002 leaves had all their tentacles inflected. A third leaf, all except the extreme marginals, which seemed old and torpid, and the 4th, a large number. After 21 HRS, every single tentacle on all 4 leaves was closely inflected. Of the 4 leaves placed at the same time in water, 1 had after 5 HRS 45 Roman 1005 marginal tentacles inflected, a second 10, a third 9 marginals and submarginals, and the 4th 12, chiefly submarginals, inflected. After 21 HRS, all these marginal tentacles re-expanded, but a few of the submarginals on 2 of the leaves remained slightly curved inwards. The contrast was wonderfully great between these 4 leaves in water and those in the solution, the latter having every one of their tentacles closely inflected, making the moderate assumption that each of these leaves bore 160 tentacles. Each gland could have absorbed only 1,184,320 of a grain 351 mg. This experiment was repeated on 3 leaves with the same relative amount of the solution, and after 6 HRS 15 Roman 1000, all the tentacles except 9 on all 3 leaves taken together were closely inflected. In this case, the tentacles on each leaf were counted and gave an average of 162 per leaf. The following experiments were tried during the summer of 1873 by placing the leaves each in a separate watchglass and pouring over it 30 minims 1 Roman 1050 of the solution, other leaves being treated in exactly the same manner with the doubly distilled water used in making the solutions. The trials above given were made several years before, and when I read over my notes, I could not believe in the results, so I resolved to begin again with moderately strong solutions. 6 leaves were first immersed, each in 30 minims of a solution of 1 part of the nitrate to 8750 of water 1 gr to 20 Rs, so that each received 1320 of a grain 2025 mg. Before 30 Roman 1000 had elapsed, 4 of these leaves were immensely, and 2 of them moderately inflected. The glands were rendered of a dark red. The 4 corresponding leaves in water were not at all affected until 6 HRS had elapsed, and then only the short tentacles on the borders of the disc, and their inflection, as previously explained, is never of any significance. 4 leaves were immersed each in 30 minims of a solution of 1 part to 17,500 of water 1 gr to 40 Rs, so that each received 1640 of a grain 101 mg, and in less than 45 Roman 1003 of them had all their tentacles, except from 4 to 10, inflected, the blade of one being inflected after 6 HRS, and the blade of a second after 21 HRS. The 4th leaf was not at all affected. The glands of none were darkened of the corresponding leaves. Page 152. In water, only 1 had any of its exterior tentacles, namely 5 inflected, after 6 HRS in one case, and after 21 HRS in 2 other cases. The short tentacles on the borders of the disc formed a ring in the usual manner. 4 leaves were immersed each in 30 minims of a solution of 1 part to 43,750 of water 1 gr to 100 Rs, so that each leaf got 11,600 of a grain 405 mg. Of these, 1 was much inflected in 8 Roman 1000, and after 2 HRS 7 Roman 1000 had all the tentacles except 13 inflected. The 2nd leaf, after 10 Roman 1000, had all except 3 inflected. The 3rd and 4th were hardly at all affected, scarcely more than the corresponding leaves in water of the latter. Only 1 was affected, this having 2 tentacles inflected with those on the outer parts of the disc, forming a ring in the usual manner in the leaf, which had all its tentacles, except 3 inflected in 10 Roman 1000 each gland, assuming that the leaf bore 160 tentacles, could have absorbed only 1,251,200 of a grain, or 258 mg. 4 leaves were separately immersed as before in a solution of 1 part to 131,250 of water 1 gr to 300 Rs, so that each received 14,800 of a grain, or 135 mg. After 50 Roman 1001 leaf had all its tentacles except 16, and after 8 HRS 20 Roman 1000 all but 14 inflected. The 2nd leaf, after 40 Roman 1000, had all but 20 inflected, and after 8 HRS 10 Roman 1000 began to re-expand. The 3rd, in 3 HRS, had about half its tentacles inflected, which began to re-expand. After 8 HRS 15 Roman 1000, the 4th leaf, after 3 HRS 7 Roman 1000, had only 29 tentacles more or less inflected. Thus 3 out of the 4 leaves were strongly acted on. It is clear that very sensitive leaves had been accidentally selected. The day, moreover, was hot. The 4 corresponding leaves in water were likewise acted on, rather more than is usual, for after 3 HRS, 1 had 9 tentacles, another 4, and another 2, and the 4th, none inflected with respect to the leaf, of which all the tentacles except 16 were inflected. After 50 Roman 1000 each gland, assuming that the leaf bore 160 tentacles, could have absorbed only 1,691,200 of a grain 937 mg, and this appears to be about the least quantity of the nitrate, which suffices to induce the inflection of a single tentacle. As negative results are important in confirming the foregoing positive ones, 8 leaves were immersed, as before, each in 30 minims of a solution of 1 part to 175,000 of water 1 gr to 400 Rs, so that each received only 16,400 of a grain 101 mg. This minute quantity produced a slight effect on only 4 of the 8 leaves. 1 had 56 tentacles inflected after 2 HRS 13 Roman 1000, a second 26 inflected or subinflected after page 153 38 Roman 1000, a third 18 inflected after 1 HR. 4th 10 inflected after 35 Roman 1000. The 4 other leaves were not in the least affected of the 8 corresponding leaves. In water 1 had after 2 HRS 10 Roman 1009 tentacles, and 4 others from 1 to 4 long-headed tentacles inflected, the remaining 3 being unaffected, hence the 16,400 of a grain given to a sensitive leaf during warm weather perhaps produces a slight effect, but we must bear in mind that occasionally water causes as great an amount of inflection as occurred in this last experiment. A summary of the results: with nitrate of ammonia, the glands of the disc, when excited by a half-minim drop 296 Roman 1050, containing 12,400 of a grain of the nitrate 27 mg, transmit a motor impulse to the exterior tentacles, causing them to bend inwards. A minute drop, containing 128,800 of a grain 225 mg, if held for a few seconds in contact with a gland, causes the tentacle bearing this gland to be inflected if a leaf is left immersed for a few hours, and sometimes for only a few minutes, in a solution of such strength that each gland can absorb only the 1,691,200 of a grain 937 mg. This small amount is enough to excite each tentacle into movement, and it becomes closely inflected. Phosphate of ammonia. This salt is more powerful than the nitrate, even in a greater degree than the nitrate is more powerful than the carbonate. This is shown by weaker solutions of the phosphate acting when dropped on the discs, or applied to the glands of the exterior tentacles, or when leaves are immersed. The difference in the power of these 3 salts, as tried in 3 different ways, supports the results presently to be page 154, given, which are so surprising that their credibility requires every kind of support. In 1872, I experimented on 12 immersed leaves, giving each only 10 minims of a solution. But this was a bad method, for so small a quantity hardly covered them. None of these experiments will therefore be given, though they indicate that excessively minute doses are efficient. When I read over my notes in 1873, I entirely disbelieved them, and determined to make another set of experiments with scrupulous care on the same plan as those made with the nitrate, namely, by placing leaves in watchglasses and pouring over each 30 minims of the solution under trial, treating at the same time, and in the same manner, other leaves, with the distilled water used in making the solutions. During 1873, 71 leaves were thus tried in solutions of various strengths, and the same number in water, notwithstanding the care taken, and the number of the trials made. When in the following year I looked merely at the results without reading over my observations, I again thought that there must have been some error, and 35 fresh trials were made with the weakest solution. But the results were as plainly marked as before. Altogether, 106 carefully selected leaves were tried both in water and in solutions of the phosphate. Hence, after the most anxious consideration, I can entertain no doubt of the substantial accuracy of my results. Before giving my experiments, it may be well to premise that crystallized phosphate of ammonia, such as I used, contains 35% of water of crystallization, so that in all the following trials, the efficient elements formed only 64% of the salt used. Extremely minute particles of the dry phosphate were placed, page 155, with the point of a needle on the secretions surrounding several glands. These poured forth much secretion, were blackened, and ultimately died. But the tentacles moved only slightly. The dose small, as it was evidently was too great, and the result was the same as with particles of the carbonate of ammonia. Half minims of a solution of 1 part to 437 of water were placed on the discs of 3 leaves, and acted most energetically, causing the tentacles of one to be inflected in 15 Roman 1000, and the blades of all 3 to be much curved inwards in 2 HRS 15 Roman 1000. Similar drops of a solution of 1 part to 1312 of water 1 gr to 3 Rs were then placed on the discs of 5 leaves, so that each received the 12,880 of a grain 225 mg. After 8 HRS, the tentacles of 4 of them were considerably inflected, and after 24 HRS, the blades of 3. After 48 HRS, all 5 were almost fully re-expanded. I may mention, with respect to one of these leaves, that a drop of water had been left during the previous 24 HRS on its disc, but produced no effect, and that this was hardly dry when the solution was added. Similar drops of a solution of 1 part to 1750 of water 1 gr to 4 Rs were next placed on the discs of 6 leaves, so that each received 13,840 of a grain 169 mg. After 8 HRS, 3 of them had many tentacles, and their blades inflected. 2 others had only a few tentacles slightly inflected, and the 6th was not at all affected. After 24 HRS, most of the leaves had a few more tentacles inflected, but one had begun to re-expand. We thus see that, with the more sensitive leaves, the 13,840 of a grain absorbed by the central glands is enough to make many of the exterior tentacles, and the blades bend, whereas the 11,920 of a grain of the carbonate, similarly given, produced no effect, and 12,880 of a grain of the nitrate was only just sufficient to produce a well-marked effect. A minute drop, about equal to 120 of a minim, of a solution of 1 part of the phosphate to 875 of water, was applied to the secretion on 3 glands, each of which, thus, received only 157,600 of a grain 112 mg, and all 3 tentacles became inflected. Similar drops of a solution of 1 part to 1312 of water 1 gr to 3 Rs were now tried on 3 leaves, a drop being applied to 4 glands on the same leaf. On the first leaf, 3 of the tentacles became slightly inflected in 6 Roman 1000 and re-expanded after 8 HRS. 45 Roman 1000 on the second, 2 tentacles became subinflected in 12 Roman 1000, and on the third, all 4 tentacles were decidedly inflected in 12 Roman 1000. They remained so for 8 HRS 30 Roman 1000, but by the next morning were fully re-expanded. Page 156. In this latter case, each gland could have received only the 1,115,200 or 563 mg of a grain. Lastly, similar drops of a solution of 1 part to 1750 of water 1 gr to 4 Rs were tried on 5 leaves, a drop being applied to 4 glands on the same leaf, the tentacles on 3 of these leaves were not in the least affected. On the 4th leaf, 2 became inflected, whilst on the 5th, which happened to be a very sensitive one, all 4 tentacles were plainly inflected in 6 HRS 15 m, but only 1 remained inflected after 24 HRS. I should, however, state that in this case, an unusually large drop adhered to the head of the pin. Each of these glands could have received very little more than 1,153,600 of a grain, or 423, but this small quantity sufficed to cause inflection. We must bear in mind that these drops were applied to the viscid secretion for only from 10 to 15 seconds, and we have good reason to believe that all the phosphate in the solution would not be diffused and absorbed in this time. We have seen, under the same circumstances, that the absorption by a gland of 119,200 of a grain of the carbonate and of 157,600 of a grain of the nitrate did not cause the tentacle bearing the gland in question to be inflected, so that here again the phosphate is much more powerful than the other 2 salts. We will now turn to the 106 experiments with immersed leaves. Having ascertained by repeated trials that moderately strong solutions were highly efficient, I commenced with 16 leaves, each placed in 30 minims of a solution of 1 part to 43,750 of water 1 gr to 100 Rs, so that each received 11,600 of a grain, or 4,058 mg of these leaves. 11 had nearly all or a great number of their tentacles inflected in 1 HR, and the 12th leaf in 3 HRS. One of the 11 had every single tentacle closely inflected in 50 Roman 1002 leaves out of the 16 were only moderately affected, yet more so than any of those simultaneously immersed in water, and the remaining 2, which were pale leaves, were hardly at all affected. Of the 16 corresponding leaves in water, 1 had 9 tentacles, another 6, and 2 others 2 tentacles inflected in the course of 5 HRS, so that the contrast in appearance between the 2 lots was extremely great. 18 leaves were immersed each in 30 minims of a solution of 1 part to 87,500 of water 1 gr to 200 Rs, so that each received 13,200 of a grain 202 mg. 14 of these were strongly inflected within 2 HRS, and some of them within 15 Roman 1003 out of the 18 were only slightly affected, having 21, 19 and 12 tentacles in page 157 flected, and 1 was not at all acted on by an accident. Only 15 instead of 18 leaves, were immersed at the same time in water. These were observed for 24 HRS. One had 6, another 4, and a third 2 of their outer tentacles inflected, the remainder being quite unaffected. The next experiment was tried under very favorable circumstances, for the day July 8 was very warm, and I happened to have unusually fine leaves. 5 were immersed as before in a solution of 1 part to 131,250 of water 1 gr to 300 Rs, so that each received 14,800 of a grain, or 135 mg, after an immersion of 25 Roman 1000 all. 5 leaves were much inflected. After 1 hr 25 Roman 1001 leaf had all but 8 tentacles inflected, the second all but 3, the third all but 5, the fourth all but 23, the fifth, on the other hand, never had more than 24 inflected. Of the corresponding 5 leaves in water, one had 7, a second 2, third 10, a fourth 1, and a fifth none inflected. Let it be observed what a contrast is presented between these latter leaves and those in the solution. I counted the glands on the second leaf in the solution, and the number was 217, assuming that the 3 tentacles which did not become inflected absorbed nothing. We find that each of the 214 remaining glands could have absorbed only 1 L027200 of a grain, or 631 mg. The third leaf bore 236 glands, and subtracting the 5, which did not become inflected, each of the remaining 231 glands could have absorbed only 11,108,800 of a grain, or 584 mg, and this amount sufficed to cause the tentacles to bend. 12 leaves were tried as before in a solution of 1 part to 175,000 of water 1 gr to 400 Rs, so that each leaf received 16,400 of a grain, 101 mg. My plants were not at the time in a good state, and many of the leaves were young and pale. Nevertheless, 2 of them had all their tentacles, except 3 or 4 closely inflected in under 1 hr. 7 were considerably affected, some within 1 hr, and others not until 3 HRS, 4 HRS, 30 Roman 1000, and 8 HRS had elapsed, and this slow action may be attributed to the leaves being young and pale. Of these 9 leaves, 4 had their blades well inflected, and a fifth slightly, so the 3 remaining leaves were not affected. With respect to the 12 corresponding leaves in water, not one had its blade inflected. After from 1 to 2 HRS, one had 13 of its outer tentacles inflected, a second 6, and 4 others either 1 or 2 inflected. After 8 HRS, the outer tentacles did not become more inflected, whereas this occurred with the leaves in the solution. I record in my notes that page 158, after the 8 HRS, it was impossible to compare the 2 lots and doubt for an instant the power of the solution. 2 of the above leaves in the solution had all their tentacles except 3 and 4 inflected within an hour. I counted their glands, and on the same principle as before, each gland on 1 leaf could have absorbed only 11,164,800, and on the other leaf only 11,472,000 of a grain of the phosphate. 20 leaves were immersed in the usual manner, each in 30 minims of a solution of 1 part to 218,750 of water 1 gr to 500 Rs. So many leaves were tried, because I was then under the false impression that it was incredible that any weaker solution could produce an effect. Each leaf received 18,000 of a grain, or 81 mg. The first 8 leaves, which I tried both in the solution and in water, were either young and pale or too old, and the weather was not hot. They were hardly at all affected. Nevertheless, it would be unfair to exclude them. I then waited until I got 8 pairs of fine leaves, and the weather was favorable. The temperature of the room where the leaves were immersed varying from 75 to 81, 238 to 272, sent in another trial with 4 pairs included in the above 20 pairs. The temperature in my room was rather low, about 60, 155 cent, but the plants had been kept for several days in a very warm greenhouse, and thus rendered extremely sensitive. Special precautions were taken for this set of experiments. A chemist weighed for me a grain in an excellent balance, and fresh water given me by Prof. Franklin was carefully measured. The leaves were selected from a large number of plants in the following manner. The 4 finest were immersed in water, and the next 4 finest in the solution, and so on till the 20 pairs were complete. The water specimens were thus a little favored, but they did not undergo more inflection than in the previous cases comparatively with those in the solution. Of the 20 leaves in the solution, 11 became inflected within 40 Roman 1008 of them plainly, and 3 rather doubtfully, but the latter had at least 20 of their outer tentacles inflected owing to the weakness of the solution. Inflection occurred, except in no one much more slowly than in the previous trials. The condition of the 11 leaves, which were considerably inflected, will now be given at stated intervals, always reckoning from the time of immersion. We will now turn to the 20 corresponding leaves in water. 9 had none of their outer tentacles inflected, 9 others had from 1 to 3 inflected, and these re-expanded after 8 HRS. The remaining 2 leaves were moderately affected, one having 6 tentacles inflected in 34 Roman 1000, the other 23 inflected in 2 HRS 12 Roman 1000, and both thus remained for 24 HRS. None of these leaves had their blades inflected, so that the contrast between the 20 leaves in water and the 20 in the solution was very great both within the first hour and after. From 8 to 12 HRS had elapsed. Of the leaves in the solution, the glands on leaf, no one which in 2 HRS had all its tentacles except 8 inflected, were counted and found to be 202. Subtracting the 8, each gland could have received only the 11,552,000 grain 411 mg of the phosphate leaf. No 9 had 213 tentacles, all of which, with the exception of 4, were inflected after 24 HRS, but none of them. Closely the blade was also inflected. Each gland could have received only the 11,672,000 of a grain, or 387 mg. Lastly leaf, no 11, which had after 24 HRS all its tentacles, except 1 closely inflected, as well as the blade, bore the unusually large number of 252 tentacles, and on the same principle as before each gland could have absorbed only the 12,008,000 of a grain, or 322 mg. With respect to the following experiments, I must premise that the leaves, both those placed in the solutions and in water, were taken from plants which had been kept in a very warm greenhouse during the winter. They were thus rendered extremely sensitive, as was shown by water exciting them much more than in the previous experiments. Before giving my observations, it may be well to remind the reader that, judging from 31 fine leaves, the average number of tentacles is 192, and that the outer or exterior ones, the movements of which are alone significant, are to the short ones on the disc, in the proportion of about 16 to 9. Page 161. 4 leaves were immersed as before, each in 30 minims of a solution of 1 part to 328,125 of water, 1 gr to 750 Rs each leaf, thus received 112,000 of a grain, 54 mg of the salt, and all 4 were greatly inflected. Of the 4 corresponding leaves in water, there can therefore be no doubt about the efficiency of the above solution, and it follows as before that each gland of no one could have absorbed only 12,412,000 of a grain, 268 mg, and of no 2 only 12,460,000 of a grain, 263 mg of the phosphate. 7 leaves were immersed each in 30 minims of a solution of 1 part to 437,500 of water, 1 gr to 1,000 Rs each leaf, thus received 116,000 of a grain, 405 mg. The day was warm, and the leaves were very fine, so that all circumstances were favorable. Now for the 7 corresponding leaves in water. Comparison of the leaves in the solution, especially of the first 5 or even 6 on the list, with those in the water after 1 HR or after 4 HRS, and in a still more marked degree after 7 HRS or 8 HRS, could not leave the least doubt that the solution had produced a great effect. This was shown not only by the vastly greater number of inflected tentacles, but by the degree or closeness of their inflection, and by that of their blades. Yet each gland on leaf no 1, which bore 255 glands, all of which excepting 5 were inflected in 30 Roman 1000, could not have received more than 1/4,000,000th of a grain. 162 mg of the salt again, each gland on leaf No. 3, which bore 233 glands, all of which except 9 were inflected in 2 HRS 30 Roman 1000, could have received at most only the 13,584,000 of a grain, or 181 mg. 4 leaves were immersed as before in a solution of 1 part to 656,250 of water 1 gr to 1,500 Rs. But on this occasion, I happened to select leaves which were very little sensitive, as on other occasions I chanced to select unusually sensitive leaves. The leaves were not more affected after 12 HRS than page 163, the 4 corresponding ones in water, but after 24 HRS they were slightly more inflected. Such evidence, however, is not at all trustworthy. 12 leaves were immersed each in 30 minims of a solution of 1 part to 1,312,500 of water 1 gr to 3,000 Rs, so that each leaf received 148,000 of a grain 135 mg. The leaves were not in very good condition. 4 of them were too old, and of a dark red color. 4 were too pale, yet one of these latter acted well. The 4 others, as far as could be told by the eye, seemed in excellent condition. The result was as follows. With respect to the 12 corresponding leaves in water, 1 had after 1 HR 35 Roman 1050 tentacles inflected, but after 11 HRS only 22 remained so, and these formed a group with the blade at this point slightly inflected. It appeared as if this leaf had been in some manner accidentally excited, for instance, by a particle of animal matter, which was dissolved by the water 2, after 1 HR 45 Roman 1032. Tentacles inflected, but after 5 HRS 30 Roman 1000 only 25 inflected, and these after 10 HRS all re-expanded. 3 after 1 HR 25 inflected, which after 10 HRS 20 Roman 1000 were all re-expanded. 4 and 5 after 1 HR 35 Roman 1006 and 7 tentacles inflected, which re-expanded after 11 HRS 6-7, and 8 from 1 to 3 inflected, which page 164 soon re-expanded. 9, 10, 11, and 12, none inflected though observed for 24 hours. Comparing the states of the 12 leaves in water with those in the solution, there could be no doubt that in the latter a larger number of tentacles were inflected, and these to a greater degree, but the evidence was by no means so clear as in the former experiments with stronger solutions. It deserves attention that the inflection of 4 of the leaves in the solution went on increasing during the first 6 HRS, and with some of them for a longer time, whereas in the water, the inflection of the 3 leaves, which were the most affected as well as of all the others, began to decrease during this same interval. It is also remarkable that the blades of 3 of the leaves in the solution were slightly inflected, and this is a most rare event with leaves in water, though it occurred to a slight extent in 1 which seemed to have been in some manner accidentally excited. All this shows that the solution produced some effect, though less, and at a much slower rate than in the previous cases. The small effect produced may, however, be accounted for in large part by the majority of the leaves having been in a poor condition. Of the leaves in the solution, no one bore 200 glands, and received 148,000 of a grain of the salt. Subtracting the 17 tentacles, which were not inflected, each gland could have absorbed only the 18,784,000 of a grain 738 mg. This amount caused the tentacle bearing each gland to be greatly inflected. The blade was also inflected. Lastly, 8 leaves were immersed each in 30 minims of a solution of 1 part of the phosphate to 21,875,000 of water 1 gr to 5,000 Rs each leaf, thus received 180,000 of a grain of the salt, or 81 mg. I took especial pains in selecting the finest leaves from the hothouse for immersion both in the solution and the water, and almost all proved extremely sensitive, beginning, as before, with those in the solution. Now for the 8 corresponding leaves in water. When the 2 lots of 8 leaves in the solution and in the water were compared after the lapse of 24 HRS, they undoubtedly differed much in appearance. The few tentacles on the leaves in water, which were inflected, had after this interval re-expanded with the exception of one leaf, and this presented the very unusual case of the blade being somewhat inflected though in a degree hardly approaching that of the two leaves in the solution of these latter leaves. No one had almost all its tentacles together, with its blade inflected, after an immersion of 2 HRS 30 Roman 1000 leaves. No 2 and 3 were affected at a much slower rate, but after from 24 HRS to 48 HRS, almost all their tentacles were closely inflected, and the blade of one quite doubled up, we must therefore admit. Incredible as the fact may at first appear that this extremely weak solution acted on the more sensitive leaves, each of which received only the 180,000 of a grain 80, 1 mg of the phosphate now leaf, no 3 or 178 tentacles, and subtracting the 3 which were not inflected, each gland could have absorbed only the 114 million of a grain or 463 mg leaf, no one which was strongly acted on. Within 2 HRS 30 Roman 1000, and had all its outer tentacles, except 13, inflected. Within 6 HRS 30 Roman 1000 bore 260 tentacles, and on the same principle as before each gland could have page 166, absorbed only 119,760,000 of a grain or 328 mg, and this excessively minute amount sufficed to cause all the tentacles bearing these glands to be greatly inflected. The blade was also inflected. A summary of the results: with phosphate of ammonia, the glands of the disc, when excited by a half-minim drop 296 Roman 1050, containing 13,840 of a grain 169 mg of this salt, transmit a motor impulse to the exterior tentacles, causing them to bend inwards. A minute drop, containing 1,153,600 of a grain 423 mg, if held for a few seconds in contact with a gland, causes the tentacle bearing this gland to be inflected. If a leaf is left immersed for a few hours, and sometimes for a shorter time, in a solution so weak that each gland can absorb only the 19,760,000 of a grain 328 mg, this is enough to excite the tentacle into movement, so that it becomes closely inflected, as does sometimes the blade. In the general summary to this chapter, a few remarks will be added showing that the efficiency of such extremely minute doses is not so incredible as it must at first appear. Sulfate of ammonia. The few trials made with this and the following 5 salts of ammonia were undertaken merely to ascertain whether they induced inflection. Half-minims of a solution of 1 part of the sulfate of ammonia to 437 of water were placed on the discs of 7 leaves, so that each received 1960 of a grain or 675 mg. After 1 hr, the tentacles of 5 of them, as well as the blade of 1, were strongly inflected. The leaves were not afterwards observed. Citrate of ammonia. Half-minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves. In 1 hr, the short outer tentacles round the discs were a little inflected, with the glands on the discs blackened. After 3 HRS 25 Roman 1001 leaf had its blade inflected, but none of the exterior tentacles. All 6 leaves remained in nearly the same state during the day. The submarginal tentacles, however, page 167, becoming more inflected. After 23 HRS, 3 of the leaves had their blades somewhat inflected, and the submarginal tentacles of all considerably inflected, but in none were the 2, 3, or 4 outer rows affected. I have rarely seen cases like this, except from the action of a decoction of grass. The glands on the discs of the above leaves, instead of being almost black, as after the first hour, were now, after 23 HRS, very pale. I next tried on 4 leaves half-minims of a weaker solution of 1 part to 1312 of water 1 gr to 3 Rs, so that each received 12,880 of a grain 225 mg. After 2 HRS 18 Roman 1000, the glands on the disc were very dark-colored. After 24 HRS, 2 of the leaves were slightly affected, the other 2 not at all. Acetate of ammonia. Half-minims of a solution of about 1 part to 109 of water were placed on the discs of 2 leaves, both of which were acted on in 5 HRS 30 Roman 1000, and after 23 HRS had every single tentacle closely inflected. Oxalate of ammonia half minims of a solution of 1 part to 218 of water were placed on 2 leaves, which after 7 HRS became moderately, and after 23 HRS strongly inflected 2 other leaves were tried with a weaker solution of 1 part to 437 of water. One was strongly inflected in 7 HRS, the other not, until 30 HRS had elapsed. Tartrate of ammonia half minims of a solution of 1 part to 437 of water were placed on the discs of 5 leaves in 31 Roman 1000. There was a trace of inflection in the exterior tentacles of some of the leaves, and this became more decided after 1 HR with all the leaves, but the tentacles were never closely inflected. After 8 HRS 30 Roman 1000, they began to re-expand next morning. After 23 HRS, all were fully re-expanded, excepting one which was still slightly inflected. The shortness of the period of inflection in this and the following case is remarkable. Chloride of ammonium half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves. A decided degree of inflection in the outer and submarginal tentacles was perceptible in 25 Roman 1000, and this increased during the next 3 or 4 hours, but never became strongly marked. After only 8 HRS 30 Roman 1000, the tentacles began to re-expand, and by the next morning after 24 HRS were fully re-expanded on 4 of the leaves, but still slightly inflected on 2. General summary and concluding remarks on the salts of ammonia. We have now seen that the 9-page 168 salts of ammonia, which were tried, all caused the inflection of the tentacles and often of the blade of the leaf. As far as can be ascertained from the superficial trials with the last 6 salts, the citrate is the least powerful, and the phosphate certainly by far the most. The tartrate and chloride are remarkable from the short duration of their action. The relative efficiency of the carbonate, nitrate, and phosphate is shown in the following table by the smallest amount, which suffices to cause the inflection of the tentacles. Column 1 solutions. How applied. Column 2 carbonate of ammonia. Column 3 nitrate of ammonia. Column 4 phosphate of ammonia. Placed on the glands of the disc, so as to act indirectly on the outer tentacles. 1960 of a grain, or 675 mg 12,400 of a grain, or 27 mg 13,840 of a grain, or 169 mg. Applied for a few seconds directly to the gland of an outer tentacle. 114,400 of a grain, or 445 mg 128,800 of a grain, or 25 mg grain. 1,153,600 of a grain, or 423 mg. Leaf immersed with time allowed for each gland to absorb all that it can. 1,268,800 of a grain, or 24 mg. 1,690,1,200 of a grain, or 937 mg. 119,760,000 of a grain, or 328 mg. Amount absorbed by a gland, which suffices to cause the aggregation of the protoplasm in the adjoining cells of the tentacles. 1,134,400 of a grain, or 48 mg. From the experiments tried in these 3 different ways, we see that the carbonate, which contains 23% of nitrogen, is less efficient than the nitrate, which contains 35%. The phosphate contains less nitrogen than either of these salts, namely only 21%, and yet is far more, page 169, efficient. Its power, no doubt, depending quite as much on the phosphorus as on the nitrogen which it contains. We may infer that this is the case from the energetic manner in which bits of bone and phosphate of lime affect the leaves. The inflection excited by the other salts of ammonia is probably due solely to their nitrogenin, the same principle that nitrogenous organic fluids act powerfully, whilst non-nitrogenous organic fluids are powerless. As such minute doses of the salts of ammonia affect the leaves, we may feel almost sure that Drosera absorbs and profits by the amount, though small, which is present in rainwater, in the same manner as other plants absorb these same salts by their roots. The smallness of the doses of the nitrate, and more especially of the phosphate of ammonia, which caused the tentacles of immersed leaves to be inflected, is perhaps the most remarkable fact recorded in this volume, when we see that much less than the millionth of a grain of the phosphate absorbed by a gland of one of the exterior tentacles causes it to bend. It may be thought that the effects of the solution on the glands of the disc have been overlooked, namely the transmission of a motor impulse from them to the exterior tentacles. No doubt the movements of the latter are thus aided, but the aid thus rendered must be insignificant, for we know that a drop containing as much as the 13,840 of a grain placed on the disc is only just able to cause the outer tentacles of a highly sensitive leaf to bend. It is, sir. It is scarcely possible to realize what a million means. The best illustration which I have met with is that given by Mr. Kroll, who says take a narrow strip of paper 83 ft 4 in length and stretch it along the wall of a large hall, then mark off at one end the 1/10 of an inch. This 1/10 will represent 100, and the entire strip a million, page 170. Tainly a most surprising fact that the 119,760,000 of a grain, or in round numbers the 1/20 millionth of a grain, 33 mg of the phosphate, should affect any plant or indeed any animal, and as this salt contains 35% of water of crystallization, the efficient elements are reduced to 130,555,126 of a grain, or in round numbers to 1/30 millionth of a grain, 216 mg. The solution, moreover, in these experiments was diluted in the proportion of 1 part of the salt to 2,187,500 of water, or 1 grain to 5,000 Rs. The reader will perhaps best realize this degree of dilution by remembering that 5,000 Rs would more than fill a 31-gallon cask, and that to this large body of water 1 grain of the salt was added only half a dram, or 30 minims of the solution being poured over a leaf. Yet this amount sufficed to cause the inflection of almost every tentacle and often of the blade of the leaf. I am well aware that this statement will at first appear incredible to almost everyone. Drosera is far from rivaling the power of the spectroscope, but it can detect, as shown by the movements of its leaves, a very much smaller quantity of the phosphate of ammonia than the most skillful chemist can of any substance. My results were for a long time incredible. When my first observations were made on the nitrate of ammonia 14 years ago, the powers of the spectroscope had not been discovered, and I felt all the greater interest in the then unrivaled powers of Drosera. Now, the spectroscope has altogether beaten Drosera, for according to Bunsen and Kirchhoff, probably less than 1,200,000,000 of a grain of sodium can be thus detected. See Balfour Stewart Treatise on Heat, 2nd edit, 1871, p. 228. With respect to ordinary chemical tests, I gather from Dr. Alfred Taylor's work on poisons that about 14,000 of a grain of arsenic, 14,400 of a grain of prussic acid, 11,400 of iodine, and 12,000 of tartarized antimony can be detected, but the power of detection depends much on the solutions under trial not being extremely weak. Page 171. Even to myself, and I anxiously sought for every source of error, the salt was in some cases weighed for me by a chemist in an excellent balance, and fresh water was measured many times with care. The observations were repeated during several years. 2 of my sons, who were as incredulous as myself, compared several lots of leaves simultaneously immersed in the weaker solutions and in water, and declared that there could be no doubt about the difference in their appearance. I hope that someone may hereafter be induced to repeat my experiments. In this case, he should select young and vigorous leaves, with the glands surrounded by abundant secretion. The leaves should be carefully cut off and laid gently in watchglasses, and a measured quantity of the solution and of water poured over each. The water used must be as absolutely pure as it can be made. It is to be especially observed that the experiments with the weaker solutions ought to be tried after several days of very warm, whether those with the weakest solutions should be made on plants which have been kept for a considerable time in a warm greenhouse or cool hothouse. But this is by no means necessary for trials with solutions of moderate strength. I beg the reader to observe that the sensitiveness or irritability of the tentacles was ascertained by three different methods: indirectly, by drops placed on the disc; directly, by drops applied to the glands of the outer tentacles; and by the immersion of whole leaves. And it was found by these three methods that the nitrate was more powerful than the carbonate, and the phosphate much more powerful than the nitrate, this result being intelligible from the difference in the amount of nitrogen in the first two salts, and from the presence of phosphorus in the third. It may aid the page 172 reader's faith to turn to the experiments with a solution of 1 grain of the phosphate to 1,000 Rs of water, and he will there find decisive evidence that the 1/4,000,000th of a grain is sufficient to cause the inflection of a single tentacle. There is therefore nothing very improbable in the fifth of this weight, or the 1/20,000,000th of a grain, acting on the tentacle of a highly sensitive leaf. Again, 2 of the leaves, in the solution of 1 grain to 3,000 Rs, and 3 of the leaves, in the solution of 1 grain to 5,000 Rs, were affected not only far more than the leaves tried at the same time in water, but incomparably more than any 5 leaves, which can be picked out of the 173 observed by me at different times in water. There is nothing remarkable in the mere fact of the 1/20,000,000th of a grain of the phosphate dissolved in above 2,000,000 times its weight of water being absorbed by a gland. All physiologists admit that the roots of plants absorb the salts of ammonia brought to them by the rain, and 14 gallons of rainwater contain a grain of ammonia, therefore only a little more than twice as much as in the weakest solution employed by me. The fact which appears truly wonderful is that the 1/20,000,000th of a grain of the phosphate of ammonia, including less than the 1/30,000,000th of efficient matter, when absorbed by a gland, should induce some change in it, which leads to a motor impulse being transmitted down the whole length of the tentacle, causing the basal part to bend, often through an angle of above 180 degrees. Astonishing as is this result, there is no sound reason. Miller's Elements of Chemistry, Part Roman 2, p. 107, 3rd edit, 1864, page 173. Why we should reject it as incredible. Prof. Donders of Utrecht informs me that, from experiments formerly made by him and Dr. de Ruyter, he inferred that less than the 1/1,000,000th of a grain of sulfate of atropine in an extremely diluted state, if applied directly to the iris of a dog, paralyses the muscles of this organ. But in fact, every time that we perceive an odor, we have evidence that infinitely smaller particles act on our nerves. When a dog stands a quarter of a mile to leeward of a deer or other animal, and perceives its presence, the odorous particles produce some change in the olfactory nerves, yet these particles must be infinitely smaller than those of the phosphate of ammonia, weighing the 1/20,000,000th of a grain. These nerves then transmit some influence to the brain of the dog, which leads to action on its part. With Drosera, the really marvelous fact is that a plant, without any specialized nervous system, should be affected by such minute particles, but we have no grounds for assuming that other tissues could not be rendered as exquisitely susceptible to impressions from without if this were beneficial to the organism, as is the nervous system of the higher animals. My son George Darwin has calculated for me the diameter of a sphere of phosphate of ammonia, specific gravity, one weighing the 1/20,000,000th of a grain, and finds it to be 11,644 of an inch. Now Dr. Klein informs me that the smallest micrococci, which are distinctly discernible under a power of 800 diameters, are estimated to be from 2 to 5 millimeter. That is from 150,800 to 1,127,000 of an inch in diameter. Therefore, an object between 131 and 177 of the size of a sphere of the phosphate of ammonia of the above weight can be seen under a high power, and no one supposes that odorous particles, such as those emitted from the deer in the above illustration, could be seen under any power of the microscope. Page 174. Chapter Roman 8, the effects of various other salts and acids on the leaves. Salts of sodium, potassium, and other alkaline, earthy, and metallic salts. Summary on the action of these salts. Various acids. Summary on their action. Having found that the salts of ammonia were so powerful, I was led to investigate the action of some other salts. It will be convenient first to give a list of the substances tried, including 49 salts and 2 metallic acids, divided into 2 columns showing those which cause inflection and those which do not do so or only doubtfully. My experiments were made by placing half-minim drops on the discs of leaves, or more commonly, by immersing them in the solutions, and sometimes by both methods. A summary of the results, with some concluding remarks, will then be given. The action of various acids will afterwards be described. Column 1 salts causing inflection, column 2 salts not causing inflection. Arranged in groups according to the chemical classification in Watt's Dictionary of Chemistry. Sodium carbonate, rapid inflection. Potassium carbonate, slowly poisonous. Sodium nitrate, rapid inflection. Potassium nitrate, somewhat poisonous. Sodium sulfate, moderately rapid inflection. Potassium sulfate. Sodium phosphate, very rapid inflection. Potassium phosphate. Sodium citrate, rapid inflection. Potassium citrate. Sodium oxalate, rapid inflection. Sodium chloride, moderately rapid inflection. Potassium chloride. Page 175. Column 1 salts causing inflection, column 2 salts not causing inflection. Arranged in groups according to the chemical classification in Watt's Dictionary of Chemistry. Sodium iodide, rather slow inflection. Potassium iodide, a slight and doubtful amount of inflection. Sodium bromide, moderately rapid inflection. Potassium bromide. Potassium oxalate, slow and doubtful inflection. Lithium nitrate, moderately rapid inflection. Lithium acetate. Cesium chloride, rather slow inflection. Rubidium chloride. Silver nitrate, rapid inflection, quick poison. Cadmium chloride, slow inflection. Calcium acetate. Mercury perchloride, rapid inflection, quick poison. Calcium nitrate. Magnesium acetate. Magnesium nitrate. Magnesium chloride. Magnesium sulfate. Barium acetate. Barium nitrate. Strontium acetate. Strontium nitrate. Zinc chloride. Aluminum chloride, slow and doubtful inflection. Aluminum nitrate, a trace of inflection. Gold chloride, rapid inflection, quick poison. Aluminum and potassium sulfate. Tin chloride, slow inflection, poisonous. Lead chloride. Antimony tartrate, slow inflection, probably poisonous. Arsenious acid, quick inflection, poisonous. Iron chloride, slow inflection, probably poisonous. Manganese chloride, chromic acid, quick inflection, highly poisonous. Copper chloride, rather slow inflection, poisonous. Cobalt chloride. Nickel chloride, rapid inflection, probably poisonous. Platinum chloride, rapid inflection, poisonous. Page 176. Sodium carbonate of pure, given me by Prof. Hoffman. Half-minims 296 Roman 1050 of a solution of 1 part to 218 of water 2 grs to 1 Rs were placed on the discs of 12 leaves. 7 of these became well inflected. 3 had only 2 or 3 of their outer tentacles inflected, and the remaining 2 were quite unaffected. But the dose, though only the 1480 of a grain 135 mg, was evidently too strong, for 3 of the 7 well-inflected leaves were killed. On the other hand, 1 of the 7, which had only a few tentacles inflected, re-expanded and seemed quite healthy after 48 HRS by employing a weaker solution, viz. 1 part to 437 of water, or 1 gr to 1 Rs. Doses of 1960 of a grain 675 mg were given to 6 leaves. Some of these were affected in 37 Roman 1000, and in 8 HRS, the outer tentacles of, as well as the blades of 2, were considerably inflected. After 23 HRS 15 Roman 1000, the tentacles had almost re-expanded, but the blades of the 2 were still just perceptibly curved inwards. After 48 HRS, all 6 leaves were fully re-expanded and appeared perfectly healthy. 3 leaves were immersed each in 30 minims of a solution of 1 part to 875 of water 1 gr to 2 Rs, so that each received 132 of a grain 2 mg. After 40 Roman 1000, the 3 were much affected, and after 6 HRS 45 Roman 1000, the tentacles of all, and the blade of 1 closely inflected. Sodium nitrate of pure half-minims of a solution of 1 part to 437 of water, containing 1960 of a grain 675 mg, were placed on the discs of 5 leaves. After 1 HR 25 Roman 1000, the tentacles of nearly all and the blade of 1 were somewhat inflected. The inflection continued to increase, and in 21 HRS 15 Roman 1000, the tentacles and the blades of 4 of them were greatly affected, and the blade of the 5th to a slight extent. After an additional 24 HRS, the 4 leaves still remained closely inflected, whilst the 5th was beginning to expand 4 days after the solution had been applied. 2 of the leaves had quite, and 1 had partially re-expanded, whilst the remaining 2 remained closely inflected and appeared injured. 3 leaves were immersed each in 30 minims of a solution of 1 part to 875 of water in 1 HR. There was great inflection, and after 8 HRS 15 Roman 1000, every tentacle and the blades of all 3 were most strongly inflected. Sodium sulfate of half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves after 5 HRS 30 Roman 1000. The tentacles of 3 of them with the blade of 1 were considerably, and those of the other 3 slightly inflected after 21 HRS. The inflection had a little decreased, page 177, and in 45 HRS, the leaves were fully expanded, appearing quite healthy. 3 leaves were immersed each in 30 minims of a solution of 1 part of the sulfate to 875 of water after 1 HR 30 Roman 1000. There was some inflection which increased so much that in 8 HRS 10 Roman 1000, all the tentacles and the blades of all 3 leaves were closely inflected. Sodium phosphate of half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves. The solution acted with extraordinary rapidity, for in 8 Roman 1000, the outer tentacles on several of the leaves were much incurved. After 6 HRS, the tentacles of all 6 leaves and the blades of 2 were closely inflected. This state of things continued for 24 HRS, excepting that the blade of a 3rd leaf became incurved. After 48 HRS, all the leaves re-expanded. It is clear that 1960 of a grain of phosphate of soda has great power in causing inflection. Sodium citrate of half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves, but these were not observed until 22 HRS had elapsed. The submarginal tentacles of 5 of them and the blades of 4 were then found inflected, but the outer rows of tentacles were not affected. 1 leaf, which appeared older than the others, was very little affected in any way. After 46 HRS, 4 of the leaves were almost re-expanded, including their blades. 3 leaves were also immersed each in 30 minims of a solution of 1 part of the citrate to 875 of water. They were much acted on in 25 Roman 1000. And after 6 HRS 35 Roman 1000, almost all the tentacles, including those of the outer rows, were inflected, but not the blades. Sodium oxalate of half minims of a solution of 1 part to 437 of water were placed on the discs of 7 leaves after 5 HRS 30 Roman 1000. The tentacles of all, and the blades of most of them, were much affected in 22 HRS. Besides the inflection of the tentacles, the blades of all 7 leaves were so much doubled over that their tips and bases almost touched. On no other occasion have I seen the blades so strongly affected. 3 leaves were also immersed each in 30 minims of a solution of 1 part to 875 of water. After 30 Roman 1000, there was much inflection, and after 6 HRS 35 Roman 1000, the blades of 2 and the tentacles of all were closely inflected. Sodium chloride of best culinary salt half minims of a solution of 1 part to 218 of water were placed on the discs, page 178, of 4 leaves. 2 apparently were not at all affected in 48 HRS. The 3rd had its tentacles slightly inflected, whilst the 4th had almost all its tentacles inflected in 24 HRS, and these did not begin to re-expand until the 4th day, and were not perfectly expanded on the 7th day. I presume that this leaf was injured by the salt half minims of a weaker solution of 1 part to 437 of water were then dropped on the discs of 6 leaves, so that each received 1960 of a grain in 1 HR 33 Roman 1000. There was slight inflection, and after 5 HRS 30 Roman 1000, the tentacles of all 6 leaves were considerably, but not closely inflected. After 23 HRS 15 Roman 1000, all had completely re-expanded and did not appear in the least injured. 3 leaves were immersed each in 30 minims of a solution of 1 part to 875 of water, so that each received 132 of a grain or 2 mg after 1 hr. There was much inflection after 8 hrs 30 Roman 1000. All the tentacles and the blades of all 3 were closely inflected. 4 other leaves were also immersed in the solution, each receiving the same amount of salt as before, viz. 132 of a grain. They all soon became inflected. After 48 hrs, they began to re-expand and appeared quite uninjured, though the solution was sufficiently strong to taste saline. Sodium iodide of half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves. After 24 hrs, 4 of them had their blades, and many tentacles inflected. The other 2 had only their submarginal tentacles inflected, the outer ones in most of the leaves being but little affected. After 46 hrs, the leaves had nearly re-expanded. 3 leaves were also immersed each in 30 minims of a solution of 1 part to 875 of water. After 6 hrs 30 Roman 1000, almost all the tentacles and the blade of 1 leaf were closely inflected. Sodium bromide of half minims of a solution of 1 part to 437 of water were placed on 6 leaves. After 7 hrs, there was some inflection. After 22 hrs, 3 of the leaves had their blades, and most of their tentacles inflected. The 4th leaf was very slightly, and the 5th and 6th hardly at all affected. 3 leaves were also immersed each in 30 minims of a solution of 1 part to 875 of water. After 40 Roman 1000, there was some inflection. After 4 hrs, the tentacles of all 3 leaves and the blades of 2 were inflected. These leaves were then placed in water, and after 17 hrs 30 Roman 1000, 2 of them were almost completely, and the 3rd partially re-expanded, so that apparently they were not injured. Page 179. Potassium carbonate of pure half minims of a solution of 1 part to 437 of water were placed on 6 leaves. No effect was produced in 24 hrs, but after 48 hrs, some of the leaves had their tentacles, and one the blade considerably inflected. This, however, seemed the result of their being injured, for on the 3rd day after the solution was given, 3 of the leaves were dead, and 1 was very unhealthy. The other 2 were recovering, but with several of their tentacles apparently injured, and these remained permanently inflected. It is evident that the 1960 of a grain of this salt acts as a poison. 3 leaves were also immersed each in 30 minims of a solution of 1 part to 875 of water, though only for 9 hrs, and very differently from what occurs with the salts of soda, no inflection ensued. Potassium nitrate of half minims of a strong solution of 1 part to 109 of water, 4 grs to 1 rs, were placed on the discs of 4 leaves. 2 were much injured, but no inflection ensued. 8 leaves were treated in the same manner, with drops of a weaker solution of 1 part to 218 of water. After 50 hrs, there was no inflection, but 2 of the leaves seemed injured. 5 of these leaves were subsequently tested with drops of milk and a solution of gelatine on their discs, and only 1 became inflected, so that the solution of the nitrate of the above strength, acting for 50 hrs, apparently had injured or paralyzed the leaves. 6 leaves were then treated in the same manner with a still weaker solution of 1 part to 437 of water, and these after 48 hrs were in no way affected, with the exception of perhaps a single leaf. 3 leaves were next immersed for 25 hrs each in 30 minims of a solution of 1 part to 875 of water, and this produced no apparent effect. They were then put into a solution of 1 part of carbonate of ammonia to 218 of water. The glands were immediately blackened, and after 1 hr there was some inflection, and the protoplasmic contents of the cells became plainly aggregated. This shows that the leaves had not been much injured by their immersion for 25 hrs in the nitrate. Potassium sulfate of half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves. After 20 hrs 30 Roman 1000, no effect was produced. After an additional 24 hrs 3 remained quite unaffected. 2 seemed injured, and the 6th seemed almost dead, with its tentacles inflected nevertheless. After 2 additional days, all 6 leaves recovered the immersion of 3 leaves for 24 HRS each in 30 minims of page 180. A solution of 1 part to 875 of water produced no apparent effect. They were then treated with the same solution of carbonate of ammonia, with the same result, as in the case of the nitrate of potash. Potassium phosphate of half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves, which were observed during 3 days, but no effect was produced. The partial drying up of the fluid on the disc slightly drew together the tentacles on it. As often occurs in experiments of this kind, the leaves on the 3rd day appeared quite healthy. Potassium citrate of half minims of a solution of 1 part to 437 of water left on the discs of 6 leaves for 3 days, and the immersion of 3 leaves for 9 HRS each in 30 minims of a solution of 1 part to 875 of water did not produce the least effect. Potassium oxalate of half minims were placed on different occasions on the discs of 17 leaves, and the results perplexed me much, as they still do. Inflection supervened very slowly after 24 HRS. 4 leaves out of the 17 were well inflected together, with the blades of 2/6 were slightly affected, and 7 not at all. 3 leaves of 1 lot were observed for 5 days, and all died, but in another lot of 6, all excepting 1 looked healthy. After 4 days, 3 leaves were immersed during 9 HRS each in 30 minims of a solution of 1 part to 875 of water, and were not in the least affected, but they ought to have been observed for a longer time. Potassium chloride of neither half minims of a solution of 1 part to 437 of water left on the discs of 6 leaves for 3 days, nor the immersion of 3 leaves during 25 HRS in 30 minims of a solution of 1 part to 875 of water produced the least effect. The immersed leaves were then treated with carbonate of ammonia as described under nitrate of potash, and with the same result. Potassium iodide of half minims of a solution of 1 part to 437 of water were placed on the discs of 7 leaves in 30 Roman 1001 leaf. Had the blade inflected, after some hours, 3 leaves had most of their submarginal tentacles moderately inflected, the remaining 3 being very slightly affected. Hardly any of these leaves had their outer tentacles inflected. After 21 HRS, all re-expanded excepting 2, which still had a few submarginal tentacles. Inflected 3 leaves were next, page 181, immersed for 8 HRS 40 Roman 1000 each in 30 minims of a solution of 1 part to 875 of water, and were not in the least affected. I do not know what to conclude from this conflicting evidence, but it is clear that the iodide of potassium does not generally produce any marked effect. Potassium bromide of half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves. After 22 HRS, 1 had its blade and many tentacles inflected, but I suspect that an insect might have alighted on it and then escaped. The 5 other leaves were in no way affected. I tested 3 of these leaves with bits of meat, and after 24 HRS, they became splendidly inflected. 3 leaves were also immersed for 21 HRS in 30 minims of a solution of 1 part to 875 of water, but they were not at all affected, excepting that the glands looked rather pale. Lithium acetate of 4 leaves were immersed together in a vessel containing 120 minims of a solution of 1 part to 437 of water, so that each received, if the leaves absorbed equally, 116 of a grain after 24 HRS. There was no inflection. I then added, for the sake of testing the leaves, some strong solution, viz. 1 gr to 20 Rs, or 1 part to 8750 of water of phosphate of ammonia, and all 4 became in 30 Roman 1000 closely inflected. Lithium nitrate of 4 leaves were immersed, as in the last case, in 120 minims of a solution of 1 part to 437 of water. After 1H 30 Roman 1000, all 4 were a little, and after 24 HRS greatly inflected. I then diluted the solution with some water, but they still remained somewhat inflected on the 3rd day. Cesium chloride of 4 leaves were immersed as above in 120 minims of a solution of 1 part to 437 of water after 1 HR5 Roman 1000. The glands were darkened after 4 HRS 20 Roman 1000. There was a trace of inflection after 6 HRS 40 Roman 1002. Leaves were greatly but not closely, and the other two considerably inflected after 22 HRS. The inflection was extremely great, and 2 had their blades inflected. I then transferred the leaves into water, and in 46 HRS from their first immersion they were almost re-expanded. Rubidium chloride of 4 leaves, which were immersed as above in 120 minims of a solution of 1 part to 437 of water, were not acted on in 22 HRS. I then added some of the strong solution 1 gr to 20 Rs of phosphate of ammonia, and in 30 Roman 1000 all were immensely inflected. Silver nitrate of 3 leaves were immersed in 90 page 182 minims of a solution of 1 part to 437 of water, so that each received as before 116 of a grain after 5 Roman 1000 slight inflection, and after 11 Roman 1000 very strong inflection, the glands becoming excessively black after 40 Roman 1000. All the tentacles were closely inflected after 6 HRS. The leaves were taken out of the solution, washed, and placed in water, but next morning they were evidently dead. Calcium acetate of 4 leaves were immersed in 120 minims of a solution of 1 part to 437 of water after 24 HRS. None of the tentacles were inflected, excepting a few where the blade joined the petiole, and this may have been caused by the absorption of the salt by the cut-off end of the petiole. I then added some of the solution 1 gr to 20 Rs of phosphate of ammonia, but this, to my surprise, excited only slight inflection even after 24 HRS. Hence, it would appear that the acetate had rendered the leaves torpid. Calcium nitrate of 4 leaves were immersed in 120 minims of a solution of 1 part to 437 of water, but were not affected in 24 HRS. I then added some of the solution of phosphate of ammonia 1 gr to 20 Rs, but this caused only very slight inflection after 24 HRS. A fresh leaf was next put into a mixed solution of the above strengths of the nitrate of calcium and phosphate of ammonia, and it became closely inflected in between 5 Roman 1000 and 10 Roman 1000 half minims of a solution of 1 part of the nitrate of calcium to 218 of water were dropped on the discs of 3 leaves, but produced no effect. Magnesium acetate nitrate and chloride of 4 leaves were immersed in 120 minims of solutions of 1 part to 437 of water of each of these 3 salts. After 6 HRS, there was no inflection, but after 22 HRS, one of the leaves in the acetate was rather more inflected than generally occurs from an immersion for this length of time in water. Some of the solution 1 gr to 20 Rs of phosphate of ammonia was then added to the 3 solutions. The leaves in the acetate mixed with the phosphate underwent some inflection, and this was well pronounced. After 24 HRS, those in the mixed nitrate were decidedly inflected in 4 HRS 30 Roman 1000, but the degree of inflection did not afterwards much increase, whereas the 4 leaves in the mixed chloride were greatly inflected in a few minutes, and after 4 HRS had almost every tentacle closely inflected. We thus see that the acetate and nitrate of magnesium injure the leaves, or at least prevent the subsequent action of phosphate of ammonia, whereas the chloride has no such tendency. Page 183. Magnesium sulfate of half minims of a solution of 1 part to 218 of water were placed on the discs of 10 leaves and produced no effect. Barium acetate of 4 leaves were immersed in 120 minims of a solution of 1 part to 437 of water, and after 22 HRS there was no inflection, but the glands were blackened. The leaves were then placed in a solution 1 gr to 20 Rs of phosphate of ammonia, which caused, after 26 HRS, only a little inflection in 2 of the leaves. Barium nitrate of 4 leaves were immersed in 120 minims of a solution of 1 part to 437 of water, and after 22 HRS, there was no more than that slight degree of inflection which often follows from an immersion of this length. In pure water, I then added some of the same solution of phosphate of ammonia, and after 30 Roman 1001 leaf was greatly inflected, 2 others moderately, and the 4th not at all. The leaves remained in this state for 24 HRS. Strontium acetate of 4 leaves immersed in 120 minims of a solution of 1 part to 437 of water were not affected in 22 HRS. They were then placed in some of the same solution of phosphate of ammonia, and in 25 Roman 1002 of them were greatly inflected. After 8 HRS, the 3rd leaf was considerably inflected, and the 4th exhibited a trace of inflection. They were in the same state next morning. Strontium nitrate of 5 leaves were immersed in 120 minims of a solution of 1 part to 437 of water. After 22 HRS, there was some slight inflection, but not more than sometimes occurs with leaves in water. They were then placed in the same solution of phosphate of ammonia. After 8 HRS, 3 of them were moderately inflected, as were all 5 after 24 HRS, but not one was closely inflected. It appears that the nitrate of strontium renders the leaves half torpid. Cadmium chloride of 3 leaves were immersed in 90 minims of a solution of 1 part to 437 of water. After 5 HRS, 20 Roman 1000 slight inflection occurred which increased during the next 3 hours. After 24 HRS, all 3 leaves had their tentacles well inflected and remained so for an additional 24 HRS, glands not discolored. Mercury perchlorate of 3 leaves were immersed in 90 minims of a solution of 1 part to 437 of water. After 22 Roman 1000, there was some slight inflection, which in 48 Roman 1000 became well pronounced. The glands were now blackened. After 5 HRS, 35 Roman 1000, all the tentacles closely inflected. After 24 HRS, still page 184, inflected and discolored. The leaves were then removed and left for 2 days in water, but they never re-expanded, being evidently dead. Zinc chloride of 3 leaves immersed in 90 minims of a solution of 1 part to 437 of water were not affected in 25 HRS. 30 Roman 1000 aluminum chloride of 4 leaves were immersed in 120 minims of a solution of 1 part to 437 of water. After 7 HRS, 45 Roman 1000 no inflection. After 24 HRS, 1 leaf, rather closely, the 2nd moderately, and 4th hardly at all inflected. The evidence is doubtful, but I think some power in slowly causing inflection must be attributed to this salt. These leaves were then placed in the solution 1 gr to 20 Rs of phosphate of ammonia, and after 7 HRS, 30 Roman 1000, the 3 which had been but little affected by the chloride became rather closely inflected. Aluminum nitrate of 4 leaves were immersed in 120 minims of a solution of 1 part to 437 of water. After 7 HRS, 45 Roman 1000, there was only a trace of inflection. After 24 HRS, 1 leaf was moderately inflected. The evidence is here again doubtful, as in the case of the chloride of aluminum. The leaves were then transferred to the same solution as before of phosphate of ammonia. This produced hardly any effect in 7 HRS, 30 Roman 1000. But after 25 HRS, 1 leaf was pretty closely inflected. The 3 others very slightly, perhaps not more so than from water. Aluminum and potassium sulfate of common alum half-minims of a solution of the usual strength were placed on the discs of 9 leaves, but produced no effect. Gold chloride of 7 leaves were immersed in so much of a solution of 1 part to 437 of water that each received 30 minims, containing 116 of a grain or 4 mg of the chloride. There was some inflection in 8 Roman 1000, which became extreme in 45 Roman 1000. In 3 HRS, the surrounding fluid was colored purple, and the glands were blackened. After 6 HRS, the leaves were transferred to water. Next morning, they were found discolored and evidently killed. The secretion decomposes the chloride very readily, the glands themselves becoming coated with the thinnest layer of metallic gold, and particles float about on the surface of the surrounding fluid. Lead chloride of 3 leaves were immersed in 90 minims of a solution of 1 part to 437 of water after 23 HRS. There was not a trace of inflection, the glands were not blackened, and the leaves did not appear. Injured, they were then page 185, referred to the solution 1 gr to 20 Rs of phosphate of ammonia, and after 24 HRS 2 of them were somewhat, the third very little inflected, and they thus remained for another 24 HRS. Tin chloride of 4 leaves were immersed in 120 minims of a solution of about 1 part, all not being dissolved, to 437 of water, after 4 HRS no effect, after 6 HRS 30 Roman 1000, all 4 leaves had their submarginal tentacles inflected after 22 HRS every single tentacle, and the blades were closely inflected. The surrounding fluid was now colored pink. The leaves were washed and transferred to water, but next morning were evidently dead. This chloride is a deadly poison, but acts slowly. Antimony tartrate of 3 leaves were immersed in 90 minims of a solution of 1 part to 437 of water. After 8 HRS 30 Roman 1000, there was slight inflection. After 24 HRS 2 of the leaves were closely, and the third moderately inflected glands, not much darkened. The leaves were washed and placed in water, but they remained in the same state for 48 additional hours. This salt is probably poisonous, but acts slowly. Arsenious acid solution of 1 part to 437 of water. 3 leaves were immersed in 90 minims in 25 Roman 1000. Considerable inflection. In 1H, great inflection, glands not discolored. After 6 HRS, the leaves were transferred to water. Next morning, they looked fresh, but after 4 days were pale-colored, had not re-expanded, and were evidently dead. Iron chloride of 3 leaves were immersed in 90 minims of a solution of 1 part to 437 of water. In 8 HRS, no inflection, but after 24 HRS, considerable inflection, glands, blackened, fluid colored yellow, with floating flocculent particles of oxide of iron. The leaves were then placed in water. After 48 HRS, they had re-expanded a very little, but I think were killed, glands excessively black. Chromic acid 1 part to 437 of water. 3 leaves were immersed in 90 minims in 30 Roman 1000 some, and in 1 HR, considerable inflection. After 2 HRS, all the tentacles closely inflected, with the glands discolored, placed in water. Next day, leaves quite discolored and evidently killed. Manganese chloride of 3 leaves immersed in 90 minims of a solution of 1 part to 437 of water. After 22 HRS, no more inflection than often occurs in water, glands not blackened. The leaves were then placed in the usual solution of phosphate of ammonia, but no inflection was caused even after 48 HRS. Copper chloride of 3 leaves immersed in 90 minims, page 186, of a solution of 1 part to 437 of water. After 2 HRS, some inflection. After 3 HRS, 45 Roman 1000 tentacles closely inflected, with the glands blackened. After 22 HRS, still closely inflected, and the leaves flaccid, placed in pure water. Next day, evidently dead, a rapid poison. Nickel chloride of 3 leaves immersed in 90 minims of a solution of 1 part to 437 of water. In 25 Roman 1000, considerable inflection, and in 3 HRS, all the tentacles closely inflected. After 22 HRS, still closely inflected, most of the glands, but not all blackened. The leaves were then placed in water. After 24 HRS, remained inflected, were somewhat discolored, with the glands and tentacles dingy red, probably killed. Cobalt chloride of 3 leaves immersed in 90 minims of a solution of 1 part to 437 of water. After 23 HRS, there was not a trace of inflection, and the glands were not more blackened than often occurs after an equally long immersion in water. Platinum chloride of 3 leaves immersed in 90 minims of a solution of 1 part to 437 of water. In 6 Roman 1000, some inflection, which became immense. After 48 Roman 1000, after 3 HRS, the glands were rather pale. After 24 HRS, all the tentacles still closely inflected, glands colorless, remained in same state for 4 days, leaves evidently killed. Concluding remarks on the action of the foregoing salts of the 51 salts and metallic acids, which were tried 25, caused the tentacles to be inflected, and 26 had no such effect, two rather doubtful cases occurring in each series in the table at the head of this discussion. The salts are arranged according to their chemical affinities, but their action on Drosera does not seem to be thus governed. The nature of the base is far more important as far as can be judged from the few experiments here given than that of the acid, and this is the conclusion at which physiologists have arrived with respect to animals. We see this fact illustrated in all the 9 salts of soda causing inflection, and in not being poisonous, except when given in large doses, whereas 7 of page 187, the corresponding salts of potash, do not cause inflection. And some of them are poisonous. 2 of them, however, viz. The oxalate and iodide of potash, slowly induced a slight and rather doubtful amount of inflection. This difference between the 2 series is interesting, as Dr. Burden Sanderson informs me that sodium salts may be introduced in large doses into the circulation of mammals without any injurious effects, while small doses of potassium salts cause death by suddenly arresting the movements of the heart. An excellent instance of the different action of the 2 series is presented by the phosphate of soda quickly causing vigorous inflection, whilst phosphate of potash is quite inefficient. The great power of the former is probably due to the presence of phosphorus, as in the cases of phosphate of lime and of ammonia. Hence, we may infer that Drosera cannot obtain phosphorus from the phosphate of potash. This is remarkable, as I hear from Dr. Burden Sanderson that phosphate of potash is certainly decomposed within the bodies of animals. Most of the salts of soda act very rapidly, the iodide acting slowest. The oxalate, nitrate, and citrate seem to have a special tendency to cause the blade of the leaf to be inflected. The glands of the disc, after absorbing the citrate, transmit hardly any motor impulse to the outer tentacles, and in this character the citrate of soda resembles the citrate of ammonia or a decoction of grassolaves, these 3 fluids all acting chiefly on the blade. It seems opposed to the rule of the preponderant influence of the base that the nitrate of lithium causes moderately rapid inflection, whereas the acetate causes none. But this metal is closely allied to sodium, page 188, and potassium, which acts so differently. Therefore, we might expect that its action would be intermediate. We see also that cesium causes inflection, and rubidium does not, and these 2 metals are allied to sodium and potassium. Most of the earthy salts are inoperative. 2 salts of calcium, 4 of magnesium, 2 of barium, and 2 of strontium did not cause any inflection, and thus follow the rule of the preponderant power of the base. Of 3 salts of aluminum, 1 did not act, a second showed a trace of action, and the third acted slowly and doubtfully, so that their effects are nearly alike. Of the salts and acids of ordinary metals, 17 were tried, and only 4, namely those of zinc, lead, manganese, and cobalt, failed to cause inflection. The salts of cadmium, tin, antimony, and iron act slowly, and the 3 latter seem more or less poisonous. The salts of silver, mercury, gold, copper, nickel, and platinum, chromic, and arsenious acids cause great inflection with extreme quickness and are deadly poisons. It is surprising, judging from animals, that lead and barium should not be poisonous. Most of the poisonous salts make the glands black, but chloride of platinum made them very pale. I shall have occasion in the next chapter to add a few remarks on the different effects of phosphate of ammonia on leaves previously immersed in various solutions. Acids. I will first give, as in the case of the salts, a list of the 24 acids which were tried, divided into 2 series according as they cause or do not cause. Miller's Elements of Chemistry, 3rd edit, pp. 337-448, page 189. Inflection. After describing the experiments, a few concluding remarks will be added. Acids much diluted, which cause inflection. Nitric, strong inflection, poisonous. 2 hydrochloric, moderate and slow inflection, not poisonous. 3 hydriodic, strong inflection, poisonous. 4 iodic, strong inflection, poisonous. 5 sulfuric, strong inflection, somewhat poisonous. 6 phosphoric, strong inflection, poisonous. 7 boracic, moderate and rather slow inflection, not poisonous. 8 formic, very slight inflection, not poisonous 9 acetic strong and rapid inflection, poisonous 10 propionic strong, but not very rapid. Inflection poisonous 11 oleic quick inflection, very poisonous 12 carbolic very slow inflection poisonous 13 lactic slow, and moderate inflection poisonous 14 oxalic moderately quick inflection, very poisonous 15 malic very slow, but considerable inflection, not poisonous 16 benzoic rapid inflection, very poisonous 17 succinic moderately quick inflection, moderately poisonous 18 hippuric, rather slow inflection, poisonous 19 hydrocyanic, rather rapid inflection, very poisonous. Acids diluted to the same degree, which do not cause inflection. Gallic not poisonous 2 tannic, not poisonous 3 tartaric, not poisonous 4 citric, not poisonous 5 uric not poisonous. Nitric acid 4 leaves were placed each in 30 minims of 1 part, by weight of the acid, to 437 of water, so that each received 116 of a grain or 4 mg. This strength was chosen for this, and most of the following experiments, as it is the same page 190 as that of most of the foregoing saline solutions. In 2 HRS 30 Roman 1000, some of the leaves were considerably, and in 6 HRS 30 Roman 1000, all were immensely inflected. As were their blades, the surrounding fluid was slightly colored pink, which always shows that the leaves have been injured. They were then left in water for 3 days, but they remained inflected and were evidently killed. Most of the glands had become colorless. 2 leaves were then immersed each in 30 minims of 1 part to 1000 of water in a few hours. There was some inflection, and after 24 HRS, both leaves had almost all their tentacles and blades inflected. They were left in water for 3 days, and 1 partially re-expanded and recovered 2 leaves were next immersed each in 30 minims of 1 part to 2000 of water. This produced very little effect, except that most of the tentacles close to the summit of the petiole were inflected, as if the acid had been absorbed by the cut-off end. Hydrochloric acid 1 part to 437 of water 4 leaves were immersed as before each in 30 minims. After 6 HRS, only 1 leaf was considerably inflected. After 8 HRS 15 Roman 1001 had its tentacles and blade well inflected. The other 3 were moderately inflected, and the blade of 1 slightly. The surrounding fluid was not colored at all pink. After 25 HRS, 3 of these 4 leaves began to re-expand, but their glands were of a pink instead of a red color. After 2 more days, they fully re-expanded, but the 4th leaf remained inflected and seemed much injured or killed, with its glands white. 4 leaves were then treated each with 30 minims of 1 part to 875 of water. After 21 HRS, they were moderately inflected, and on being transferred to water fully re-expanded in 2 days and seemed quite healthy. Hydriodic acid 1 to 437 of water 3 leaves were immersed as before each in 30 minims. After 45 Roman 1000, the glands were discolored, and the surrounding fluid became pinkish, but there was no inflection. After 5 HRS, all the tentacles were closely inflected, and an immense amount of mucus was secreted, so that the fluid could be drawn out into long ropes. The leaves were then placed in water but never re-expanded, and were evidently killed. 4 leaves were next immersed in 1 part to 875 of water. The action was now slower, but after 22 HRS, all 4 leaves were closely inflected, and were affected in other respects. As above described, these leaves did not re-expand though left for 4 days in water. This acid acts far more powerfully than hydrochloric, and is poisonous. Iodic acid 1 to 437 of water 3 leaves were immersed page 191 each in 30 minims. After 3 HRS strong inflection, after 4 HRS glands dark brown, after 8 HRS 30 Roman 1000 close inflection, and the leaves had become flaccid surrounding fluid not colored pink. These leaves were then placed in water, and next day were evidently. Sulfuric acid 1 to 437 of water 4 leaves were immersed each in 30 minims. After 4 HRS great inflection, after 6 HRS surrounding fluid just tinged pink, they were then placed in water, and after 46 HRS 2 of them were still closely inflected, 2 beginning to re-expand, many of the glands colorless. This acid is not so poisonous as hydriodic or iodic acids. Phosphoric acid 1 to 437 of water 3 leaves were immersed together in 90 minims after 5 HRS 30 Roman 1000 some inflection, and some glands colorless after 8 HRS, all the tentacles closely inflected, and many glands colorless surrounding fluid pink, left in water for 2 days and a half, remained in the same state, and appeared dead. Boracic acid 1 to 437 of water 4 leaves were immersed together in 120 minims after 6 HRS very slight inflection, after 8 HRS 15 Roman 1002 were considerably inflected, the other 2 slightly, after 24 HRS 1 leaf was rather closely inflected, the second less closely. The third and fourth moderately. The leaves were washed and put into water after 24 HRS. They were almost fully re-expanded and looked healthy. This acid agrees closely with hydrochloric acid of the same strength in its power of causing inflection and in not being poisonous. Formic acid 4 leaves were immersed together in 120 minims of 1 part to 437 of water after 40 Roman 1000 slight, and after 6 HRS 30 Roman 1000 very moderate inflection, after 22 HRS only a little more inflection than often occurs in water. 2 of the leaves were then washed and placed in a solution 1 gr to 20 Rs of phosphate of ammonia. After 24 HRS, they were considerably inflected, with the contents of their cells aggregated, showing that the phosphate had acted, though not to the full and ordinary degree. Acetic acid 4 leaves were immersed together in 120 minims of 1 part to 437 of water in 1 HR 20 Roman 1000. The tentacles of all 4 and the blades of 2 were greatly inflected. After 8 HRS, the leaves had become flaccid, but still remained closely inflected, the surrounding fluid being colored pink. They were then washed and placed in water. Next morning, they were still inflected and of a very dark red color, but with their glands colorless. After another day, they were dingy-colored. In page 192, evidently dead, this acid is far more powerful than formic, and is highly poisonous. Half-minim drops of a stronger mixture, viz. 1 part by measure to 320 of water, were placed on the discs of 5 leaves, none of the exterior tentacles. Only those on the borders of the disc, which actually absorbed the acid, became inflected. Probably the dose was too strong and paralyzed the leaves, for drops of a weaker mixture caused much inflection. Nevertheless, the leaves all died after 2 days. Propionic acid 3 leaves were immersed in 90 minims of a mixture of 1 part to 437 of water in 1 HR 50 Roman 1000. There was no inflection, but after 3 HRS 40 Roman 1001 leaf was greatly inflected, and the other 2 slightly. The inflection continued to increase, so that in 8 HRS all 3 leaves were closely inflected. Next morning, after 20 HRS, most of the glands were very pale, but some few were almost black. No mucus had been secreted, and the surrounding fluid was only just perceptibly tinted of a pale pink. After 46 HRS, the leaves became slightly flaccid and were evidently killed, as was afterwards proved to be the case. By keeping them in water, the protoplasm in the closely inflected tentacles was not in the least aggregated, but towards their bases it was collected in little brownish masses at the bottoms of the cells. This protoplasm was dead, for on leaving the leaf, in a solution of carbonate of ammonia, no aggregation ensued. Propionic acid is highly poisonous to Drosera, like its ally acetic acid, but induces inflection at a much slower rate. Oleic acid given me by Prof. Franklin. 3 leaves were immersed in this acid. Some inflection was almost immediately caused, which increased slightly, but then ceased, and the leaves seemed killed. Next morning they were rather shriveled, and many of the glands had fallen off the tentacles. Drops of this acid were placed on the discs of 4 leaves in 40 Roman 1000. All the tentacles were greatly inflected, excepting the extreme marginal ones, and many of these after 3 HRS became inflected. I was led to try this acid, from supposing that it was present, which does not seem to be the case in olive oil, the action of which is anomalous. Thus drops of this oil placed on the disc do not cause the outer tentacles to be inflected. Yet when minute drops were added to the secretions surrounding the glands of the outer tentacles, these were occasionally, but by no means always inflected 2 leaves were also immersed in this oil and there. See articles on glycerine and oleic acid in Watt's Dict of Chemistry, page 193. There's no inflection for about 12 HRS, but after 23 HRS, almost all the tentacles were inflected. 3 leaves were likewise immersed in unboiled linseed oil, and soon became somewhat, and in 3 HRS, greatly inflected. After 1 HR, the secretion round the glands was colored pink. I infer from this latter fact that the power of linseed oil to cause inflection cannot be attributed to the albumin, which it is said to contain. Carbolic acid. 2 leaves were immersed in 60 minims of a solution of 1 gr to 437 of water in 7 HRS. 1 was slightly, and in 24 HRS both were closely inflected with a surprising amount of mucus. Secreted. These leaves were washed and left for 2 days in water. They remained inflected. Most of their glands became pale, and they seemed dead. This acid is poisonous, but does not act nearly so rapidly or powerfully as might have been expected from its known destructive power on the lowest organisms. Half minims of the same solution were placed on the discs of 3 leaves. After 24 HRS, no inflection of the outer tentacles ensued, and when bits of meat were given them, they became fairly well inflected again. Half minims of a stronger solution of 1 part to 218 of water were placed on the discs of 3 leaves. No inflection of the outer tentacles ensued. Bits of meat were then given as before. 1 leaf alone became well inflected, the discal glands of the other 2 appearing much injured and dry. We thus see that the glands of the discs, after absorbing this acid, rarely transmit any motor impulse to the outer tentacles, though these, when their own glands absorb the acid, are strongly acted on. Lactic acid. 3 leaves were immersed in 90 minims of 1 part to 437 of water. After 48 Roman 1000, there was no inflection, but the surrounding fluid was colored pink. After 8 HRS 30 Roman 1001 leaf alone was a little inflected, and almost all the glands on all 3 leaves were of a very pale color. The leaves were then washed and placed in a solution 1 gr to 20 Rs of phosphate of ammonia. After about 16 HRS, there was only a trace of inflection. They were left in the phosphate for 48 HRS and remained in the same state with almost all their glands discolored. The protoplasm within the cells was not aggregated, except in a very few tentacles, the glands of which were not much discolored. I believe, therefore, that almost all the glands and tentacles had been killed by the acid so suddenly that hardly any inflection was caused. 4 leaves were next immersed in 120 minims of a weaker solution of 1 part to 875 of water. After 2 HRS 30 Roman 1000, the surrounding fluid was quite pink. The glands were pale, but page 194, there was no inflection. After 7 HRS 30 Roman 1002 of the leaves showed some inflection, and the glands were almost white. After 21 HRS 2 of the leaves were considerably inflected, and a third slightly. Most of the glands were white, the others dark red. After 45 HRS, 1 leaf had almost every tentacle inflected. A second, a large number, the third and fourth, very few, almost all. The glands were white, excepting those on the discs of 2 of the leaves, and many of these were very dark red. The leaves appeared dead, hence lactic acid acts in a very peculiar manner, causing inflection at an extraordinarily slow rate, and being highly poisonous. Immersion in even weaker solutions, viz. of 1 part to 1312 and 1750 of water, apparently killed the leaves. The tentacles, after a time, being bowed backwards, and rendered the glands white, but caused no inflection. Galactanic, tartaric, and citric acids. 1 part to 437 of water. 3 or 4 leaves were immersed each in 30 minims of these 4 solutions, so that each leaf received 116 of a grain or 4 mg. No inflection was caused in 24 HRS, and the leaves did not appear at all injured. Those which had been in the tannic and tartaric acids were placed in a solution 1 gr to 20 Rs of phosphate of ammonia, but no inflection ensued in 24 HRS. On the other hand, the 4 leaves which had been in the citric acid, when treated with the phosphate, became decidedly inflected in 50 Roman 1000, and strongly inflected after 5 HRS, and so remained for the next 24 HRS. Malic acid 3 leaves were immersed in 90 minims of a solution of 1 part to 437 of water. No inflection was caused in 8 HRS 20 Roman 1000, but after 24 HRS 2 of them were considerably, and the third slightly inflected more so than could be accounted for by the action of water. No great amount of mucus was secreted. They were then placed in water, and after 2 days partially re-expanded. Hence, this acid is not poisonous. Oxalic acid 3 leaves were immersed in 90 minims of a solution of 1 gr to 437 of water. After 2 HRS 10 Roman 1000, there was much inflection. Glands pale, the surrounding fluid of a dark pink color. After 8 HRS excessive inflection, the leaves were then placed in water. After about 16 HRS, the tentacles were of a very dark red color, like those of the leaves in acetic acid. After 24 additional hours, the 3 leaves were dead, and their glands colorless. Benzoic acid 5 leaves were immersed each in 30 minims of a solution of 1 gr to 437 of water. This solution was so weak that it only just tasted acid, yet, as we shall see, was highly poisonous to Drosera. After 52 Roman 1000, the submarginal page 195. Tentacles were somewhat inflected, and all the glands very pale-colored. The surrounding fluid was colored pink. On one occasion, the fluid became pink in the course of only 12 Roman 1000, and the glands as white as if the leaf had been dipped in boiling water. After 4 HRS, much inflection, but none of the tentacles were closely inflected, owing, as I believe, to their having been paralyzed before they had time to complete their movement. An extraordinary quantity of mucus was secreted. Some of the leaves were left in the solution. Others, after an immersion of 6 HRS 30 Roman 1000, were placed in water. Next morning, both lots were quite dead, the leaves in the solution being flaccid, those in the water now colored yellow of a pale brown tint, and their glands white. Succinic acid 3 leaves were immersed in 90 minims of a solution of 1 gr to 437 of water. After 4 HRS 15 Roman 1000, considerable, and after 23 HRS great inflection, many of the glands pale fluid-colored pink. The leaves were then washed and placed in water. After 2 days, there was some re-expansion, but many of the glands were still white. This acid is not nearly so poisonous as oxalic or benzoic. Uric acid 3 leaves were immersed in 180 minims of a solution of 1 gr to 875 of warm water, but all the acid was not dissolved, so that each received nearly 116 of a grain. After 25 Roman 1000, there was some slight inflection, but this never increased. After 9 HRS, the glands were not discolored, nor was the solution colored pink. Nevertheless, much mucus was secreted. The leaves were then placed in water, and by next morning fully re-expanded. I doubt whether this acid really causes inflection, for the slight movement which at first occurred may have been due to the presence of a trace of albuminous matter, but it produces some effect as shown by the secretion of so much mucus. Hippuric acid 4 leaves were immersed in 120 minims of a solution of 1 gr to 437 of water. After 2 HRS, the fluid was colored pink, glands pale, but no inflection. After 6 HRS, some inflection. After 9 HRS, all 4 leaves greatly inflected. Much mucus secreted, all the glands very pale. The leaves were then left in water for 2 days. They remained closely inflected, with their glands colorless, and I do not doubt were killed. Hydrocyanic acid 4 leaves were immersed each in 30 minims of 1 part to 437 of water in 2 HRS 45 Roman 1000. All the tentacles were considerably inflected, with many of the glands pale. After 3 HRS 45 Roman 1000, all strongly inflected, and the surrounding fluid colored pink. After 6 HRS, all closely inflected. After page 196, an immersion of 8 HRS 20 Roman 1000, the leaves were washed and placed in water. Next morning, after about 16 HRS, they were still inflected and discolored. On the succeeding day, they were evidently dead. 2 leaves were immersed in a stronger mixture of 1 part to 50 of water in 1 HR 15 Roman 1000. The glands became as white as porcelain, as if they had been dipped in boiling water. Very few of the tentacles were inflected, but after 4 HRS almost all were inflected. These leaves were then placed in water, and next morning, were evidently dead half-minim drops of the same strength, viz. 1 part to 50 of water, were next placed on the discs of 5 leaves. After 21 HRS, all the outer tentacles were inflected, and the leaves appeared much injured. I likewise touched the secretion round a large number of glands, with minute drops, about 120 of a minim, or 296 Roman 1050 of Scheele's, mixture 6%. The glands first became bright red, and after 3 HRS 15 Roman 1000, about 2/3 of the tentacles bearing these glands were inflected, and remained so for the 2 succeeding days when they appeared. Concluding remarks on the action of acids. It is evident that acids have a strong tendency to cause the inflection of the tentacles, for out of the 24 acids tried 19, thus acted either rapidly and energetically or slowly. And slightly, this fact is remarkable, as the juices of many plants contain more acid judging by the taste than the solutions employed in my experiments. From the powerful effects of so many acids on Drosera, we are led to infer that those naturally contained in the tissues of this plant, as well as of others, must play some important part in their economy. Of the 5 cases in which acids did not cause the tentacles to be inflected, one is doubtful, for uric acid did act slightly, and caused a copious secretion of mucus. Mere sourness to the taste is no. According to Roman 1000 Fournier de la Fecundation dans les Phanerogames, 1863, p. 61, drops of acetic hydrocyanic and sulfuric acid cause the stamens of berberis instantly to close, though drops of water have no such power, which latter statement I can confirm. Page 197. Criterion of the power of an acid on Drosera, as citric and tartaric acids are very sour, yet do not excite inflection. It is remarkable how acids differ in their power. Thus hydrochloric acid acts far less powerfully than hydriodic and many other acids of the same strength, and is not poisonous. This is an interesting fact, as hydrochloric acid plays so important a part in the digestive process of animals. Formic acid induces very slight inflection and is not poisonous, whereas its ally acetic acid acts rapidly and powerfully, and is poisonous. Malic acid acts slightly, whereas citric and tartaric acids produce no effect. Lactic acid is poisonous, and is remarkable from inducing inflection only after a considerable interval of time. Nothing surprised me more than that a solution of benzoic acid, so weak as to be hardly acidulous to the taste, should act with great rapidity and be highly poisonous, for I am informed that it produces no marked effect on the animal economy. It may be seen by looking down the list at the head of this discussion that most of the acids are poisonous, often highly so diluted. Acids are known to induce negative osmose, and the poisonous action of so many acids on Drosera is perhaps connected with this power, for we have seen that the fluids in which they were immersed often became pink, and the glands pale-colored or white. Many of the poisonous acids, such as hydriodic, benzoic, hippuric, and carbolic, but I neglected to record all the cases, caused the secretion of an extraordinary amount of mucus, so that long ropes of this matter hung from the leaves when they were lifted out of the solutions. Other acids, such as hydrochloric and malic, have no such 10. Miller's Elements of Chemistry, Part 1, p. 87, page 198. Densely in these 2 latter cases, the surrounding fluid was not colored pink, and the leaves were not poisoned. On the other hand, propionic acid, which is poisonous, does not cause much mucus to be secreted, yet the surrounding fluid became slightly pink. Lastly, as in the case of saline solutions, leaves, after being immersed in certain acids, were soon acted on by phosphate of ammonia. On the other hand, they were not thus affected after immersion in certain other acids. To this subject, however, I shall have to recur. Page 199. Chapter Roman 9, the effects of certain alkaloid poisons, other substances and vapors. Strychnine salts of quinine sulfate of does not soon arrest the movement of the protoplasm. Other salts of quinine, digitaline, nicotine, atropine, veratrine, colchicine, thane, curare, morphia, hyoscyamus. Poison of the cobra apparently accelerates the movements of the protoplasm. Camphor, a powerful stimulant, its vapor narcotic. Certain essential oils excite movement. Glycerine, water, and certain solutions retard or prevent the subsequent action of phosphate of ammonia. Alcohol, innocuous, its vapor narcotic and poisonous. Chloroform, sulfuric, and nitric ether, their stimulant, poisonous, and narcotic paracarbonic, acid narcotic, not quickly poisonous concluding remarks. As in the last chapter, I will first give my experiments and then a brief summary of the results with some concluding remarks. Acetate of strychnine half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves so that each received 1960 of a grain or 675 mg in 2 HRS 30 Roman 1000. The outer tentacles on some of them were inflected, but in an irregular manner, sometimes only on one side of the leaf. The next morning, after 22 HRS 30 Roman 1000, the inflection had not increased. The glands on the central disc were blackened and had ceased secreting. After an additional 24 HRS, all the central glands seemed dead. But the inflected tentacles had re-expanded and appeared quite healthy. Hence, the poisonous action of strychnine seems confined to the glands, which have absorbed it. Nevertheless, these glands transmit a motor impulse to the exterior tentacles. Minute drops, about 120 of a minim of the same solution applied to the glands of the outer tentacles, occasionally caused them to bend. The poison does not seem to act quickly, for having applied to several glands similar drops of a rather stronger solution of 1 part to 292 of water, this did not prevent the tentacles bending when their glands, page 200, were excited after an interval of 1/4 to 3/4 of an hour by being rubbed or given bits of meat. Similar drops of a solution of 1 part to 218 of water 2 GRS to 1 Rs quickly blackened the glands. Some few tentacles thus treated moved, whilst others did not. The latter, however, on being subsequently moistened with saliva or given bits of meat, became incurved, though with extreme slowness, and this shows that they had been injured. Stronger solutions, but the strength was not ascertained, sometimes arrested all power of movement very quickly. Thus bits of meat were placed on the glands of several exterior tentacles, and as soon as they began to move, minute drops of the strong solution were added. They continued for a short time to go on bending, and then suddenly stood still. Other tentacles on the same leaves with meat on their glands, but not wetted with the strychnine, continued to bend, and soon reached the center of the leaf. Citrate of strychnine half minims of a solution of 1 part to 437 of water were placed on the discs of 6 leaves. After 24 HRS, the outer tentacles showed only a trace of inflection. Bits of meat were then placed on 3 of these leaves, but in 24 HRS only slight and irregular inflection occurred, proving that the leaves had been greatly injured. 2 of the leaves, to which meat had not been given, had their discal glands dry and much injured. Minute drops of a strong solution of 1 part to 109 of water 4 GRS to 1 Rs were added to the secretion round several glands, but did not produce nearly so plain an effect as the drops of a much weaker solution of the acetate. Particles of the dry citrate were placed on 6 glands. 2 of these moved some way towards the center, and then stood still, being no doubt killed. 3 others curved much farther inwards, and were then fixed. 1 alone reached the center. 5 leaves were immersed each in 30 minims of a solution of 1 part to 437 of water, so that each received 116 of a grain. After about 1 HR, some of the outer tentacles became inflected, and the glands were oddly mottled with black and white. These glands, in from 4 HRS to 5 HRS, became whitish and opaque, and the protoplasm in the cells of the tentacles was well aggregated. By this time, 2 of the leaves were greatly inflected, but the 3 others not much more inflected than they were before. Nevertheless, 2 fresh leaves, after an immersion respectively for 2 HRS and 4 HRS in the solution, were not killed, for on being left for 1 HR 30 Roman 1000 in a solution of 1 part of carbonate of ammonia to 218 of water, their tentacles became more inflected, and there was much aggregation. The glands, page 200, 1 of 2 other leaves, after an immersion for 2 HRS in a stronger solution of 1 part of the citrate to 218 of water, became of an opaque pale pink color, which before long disappeared, leaving them white. 1 of these 2 leaves had its blade and tentacles greatly inflected, the other hardly at all, but the protoplasm in the cells of both was aggregated down to the bases of the tentacles, with the spherical masses in the cells close beneath the glands blackened after 24 HRS. One of these leaves was colorless and evidently dead. Sulfate of quinine, some of this salt, was added to water, which is said to dissolve 11,000 part of its weight. 5 leaves were immersed each in 30 minims of this solution, which tasted bitter in less than 1 HR. Some of them had a few tentacles inflected in 3 HRS. Most of the glands became whitish, others dark-colored, and many oddly mottled. After 6 HRS, 2 of the leaves had a good many tentacles inflected, but this very moderate degree of inflection never increased. One of the leaves was taken out of the solution after 4 HRS and placed in water. By the next morning, some few of the inflected tentacles had re-expanded, showing that they were not dead, but the glands were still much discolored. Another leaf, not included in the above lot, after an immersion of 3 HRS 15 Roman 1000, was carefully examined. The protoplasm in the cells of the outer tentacles and of the short green ones on the disc had become strongly aggregated down to their bases, and I distinctly saw that the little masses changed their positions and shapes rather rapidly, some coalescing and again separating. I was surprised at this fact, because quinine is said to arrest all movement in the white corpuscles of the blood. But as according to Bintz, this is due to their being no longer supplied with oxygen by the red corpuscles. Any such arrestment of movement could not be expected in Drosera. That the glands had absorbed some of the salt was evident from their change of color, but I at first thought that the solution might not have traveled down the cells of the tentacles, where the protoplasm was seen in active movement. This view, however, I have no doubt, is erroneous for a leaf, which had been immersed for 3 HRS in the quinine. Solution was then placed in a little solution of 1 part of carbonate of ammonia to 218 of water, and in 30 Roman 1000 the glands and the upper cells of the tentacles became intensely black, with the protoplasm presenting a very unusual appearance for it. Quarterly Journal of Microscopical Science, April 1874, p. 180, 5 page 202. Had become aggregated into reticulated dinghy-colored masses having rounded and angular interspaces. As I have never seen this effect produced by the carbonate of ammonia alone, it must be attributed to the previous action of the quinine. These reticulated masses were watched for some time, but did not change their forms, so that the protoplasm no doubt had been killed by the combined action of the 2 salts, though exposed to them for only a short time. Another leaf, after an immersion for 24 HRS in the quinine solution, became somewhat flaccid, and the protoplasm in all the cells was aggregated. Many of the aggregated masses were discolored and presented a granular appearance. They were spherical or elongated, or still more commonly consisted of little curved chains of small globules. None of these masses exhibited the least movement, and no doubt were all dead. Half minims of the solution were placed on the discs of 6 leaves. After 23 HRS, 1 had all its tentacles, 2 had a few, and the others none inflected, so that the discal glands, when irritated by this salt, do not transmit any strong motor impulse to the outer tentacles. After 48 HRS, the glands on the discs of all 6 leaves were evidently much injured or quite killed. It is clear that this salt is highly poisonous. Acetate of quinine, 4 leaves were immersed each in 30 minims of a solution of 1 part to 437 of water. The solution was tested with litmus paper and was not acid. After only 10 Roman 1000, all 4 leaves were greatly, and after 6 HRS, immensely inflected. They were then left in water for 60 HRS, but never re-expanded. The glands were white, and the leaves evidently dead. This salt is far more efficient than the sulfate in causing inflection, and like that salt is highly poisonous. Nitrate of quinine, 4 leaves were immersed each in 30 minims of a solution of 1 part to 437 of water. After 6 HRS, there was hardly a trace of inflection. After 22 HRS, 3 of the leaves were moderately, and the 4th slightly inflected, so that this salt induces, though rather slowly, well-marked inflection. These leaves, on being left in water for 48 HRS, almost. Bintz found several years ago, as stated in the Journal of Anatomy and Phys, November 1872, p. 195, that quinine is an energetic poison to low vegetable and animal organisms, even 1 part, added to 4,000 parts of blood, arrests the movements of the white corpuscles which become rounded and granular in the tentacles of Drosera, the aggregated masses of protoplasm, which appeared killed by the quinine likewise, presented a granular appearance. A similar appearance is caused by very hot water, page 203. Completely re-expanded, but the glands were much discolored, hence this salt is not poisonous in any high degree. The different action of the 3 foregoing salts of quinine is singular. Digitaline half-minims of a solution of 1 part to 437 of water were placed on the discs of 5 leaves in 3 HRS 45 Roman 1000. Some of them had their tentacles, and 1 had its blade moderately inflected. After 8 HRS, 3 of them were well inflected. The 4th had only a few tentacles inflected, and the 5th, an old leaf, was not at all affected. They remained in nearly the same state for 2 days, but the glands on their discs became pale on the 3rd day. The leaves appeared much injured. Nevertheless, when bits of meat were placed on 2 of them, the outer tentacles became inflected. A minute drop, about 120 of a minim of the solution, was applied to 3 glands, and after 6 HRS, all 3 tentacles were inflected, but next day had nearly re-expanded, so that this very small dose of 128,800 of a grain 225 mg acts on a tentacle, but is not poisonous. It appears from these several facts that digitaline causes inflection and poisons the glands, which absorb a moderately large amount. Nicotine, the secretion round several glands, was touched with a minute drop of the pure fluid, and the glands were instantly blackened, the tentacles becoming inflected. In a few minutes 2 leaves were immersed in a weak solution of 2 drops to 1 Rs or 437 grains of water. When examined, after 3 HRS 20 Roman 1000, only 21 tentacles on 1 leaf were closely inflected, and 6 on the other slightly so, but all the glands were blackened or very dark-colored, with the protoplasm in all the cells of all the tentacles much aggregated and dark-colored. The leaves were not quite killed, for on being placed in a little solution of carbonate of ammonia 2 GRS to 1 Rs, a few more tentacles became inflected, the remainder not being acted on during the next 24 HRS. Half-minims of a stronger solution, 2 drops to 12 Rs of water, were placed on the discs of 6 leaves, and in 30 Roman 1000, all those tentacles became inflected, the glands of which had actually touched the solution as shown by their blackness. But hardly any motor influence was transmitted to the outer tentacles. After 22 HRS, most of the glands on the discs appeared dead, but this could not have been the case, as when bits of meat were placed on 3 of them, some few of the outer tentacles were inflected in 24 HRS. Hence nicotine has a great tendency to blacken the glands and to induce aggregation, page 204 of the protoplasm, but except when pure has very moderate power of inducing inflection, and still less power of causing a motor influence to be transmitted from the discal glands to the outer tentacles, it is moderately poisonous. Atropine grain was added to 437 grains of water, but was not all dissolved. Another grain was added to 437 grains of a mixture of 1 part of alcohol to 7 parts of water, and a third solution was made by adding 1 part of valerianate of atropine to 437 of water. Half-minims of these 3 solutions were placed in each case on the discs of 6 leaves, but no effect whatever was produced, excepting that the glands on the discs to which the valerianate was given were slightly discolored. The 6 leaves on which drops of the solution of atropine in diluted alcohol had been left for 21 HRS were given bits of meat, and all became in 24 HRS fairly well inflected, so that atropine does not excite movement and is not poisonous. I also tried in the same manner the alkaloid sold as daturine, which is believed not to differ from atropine, and it produced no effect. 3 of the leaves on which drops of this latter solution had been left for 24 HRS were likewise given bits of meat, and they had in the course of 24 HRS a good many of their submarginal tentacles inflected. Veratrine colchicine thene solutions were made of these 3 alkaloids by adding 1 part to 437 of water. Half-minims were placed in each case on the discs of at least 6 leaves, but no inflection was caused, except perhaps a very slight amount, by the thene halfminims of a strong infusion of tea. Likewise produced, as formerly stated, no effect. I also tried similar drops of an infusion of 1 part of the extract of colchicum sold by druggists to 218 of water, and the leaves were observed for 48 HRS without any effect being produced. The 7 leaves on which drops of veratrine had been left for 26 HRS were given bits of meat, and after 21 HRS were well inflected. These 3 alkaloids are therefore quite innocuous. Curare, 1 part of this famous poison, was added to 218 of water, and 3 leaves were immersed in 90 minims of the filtered solution in 3 HRS 30 Roman 1000. Some of the tentacles were a little inflected, as was the blade of 1 after 4 HRS, after 7 HRS. Glands were wonderfully blackened, showing that matter of some kind had been absorbed in 9 HRS. 2 of the leaves had most of their tentacles subinflected, but the inflection did not increase. In the course of 24 HRS, 1 of these leaves, after being immersed for 9 HRS in the solution, was placed in water, and by next morning had largely re-expanded. Page 205. The other 2, after their immersion for 24 HRS, were likewise placed in water, and in 24 HRS were considerably re-expanded, though their glands were as black as ever. Halfminims were placed on the discs of 6 leaves, and no inflection ensued. But after 3 days, the glands on the discs appeared rather dry, yet to my surprise were not blackened. On another occasion, drops were placed on the discs of 6 leaves, and a considerable amount of inflection was soon caused, but as I had not filtered the solution, floating particles may have acted on the glands. After 24 HRS, bits of meat were placed on the discs of 3 of these leaves, and next day they became strongly inflected, as I at first thought that the poison might not have been dissolved in pure water. 1 grain was added to 437 grains of a mixture of 1 part of alcohol to 7 of water, and halfminims were placed on the discs of 6 leaves. These were not at all affected, and when after a day bits of meat were given them, they were slightly inflected in 5 HRS, and closely after 24 HRS. It follows from these several facts that a solution of curare induces a very moderate degree of inflection, and this may perhaps be due to the presence of a minute quantity of albumin. It certainly is not poisonous. The protoplasm in 1 of the leaves, which had been immersed for 24 HRS, and which had become slightly inflected, had undergone a very slight amount of aggregation. Not more than often ensues from an immersion of this length of time in water. Acetate of morphii tried a great number of experiments with this substance, but with no certain result. A considerable number of leaves were immersed from between 2 HRS and 6 HRS in a solution of 1 part to 218 of water, and did not become inflected, nor were they poisoned, for when they were washed and placed in weak solutions of phosphate and carbonate of ammonia, they soon became strongly inflected with the protoplasm in the cells well aggregated. If, however, whilst the leaves were immersed in the morphia, phosphate of ammonia was added, inflection did not rapidly ensue. Minute drops of the solution were applied in the usual manner to the secretion round between 30 and 40 glands, and when after an interval of 6 bits of meat, a little saliva or particles of glass were placed on them, the movement of the tentacles was greatly retarded, but on other occasions no such retardation occurred. Drops of water, similarly applied, never have any retarding power. Minute drops of a solution of sugar of the same strength, 1 part to 218 of water, sometimes retarded the subsequent action of meat and of particles of glass, and page 206, sometimes did not do so. At one time I felt convinced that morphia acted as a narcotic on Drosera, but after having found in what a singular manner immersion in certain non-poisonous salts and acids prevents the subsequent action of phosphate of ammonia, whereas other solutions have no such power, my first conviction seems very doubtful. Extract of hyoscyamus. Several leaves were placed each in 30 minims of an infusion of 3 GRS of the extract sold by druggists to 1 Rs of water. 1 of them, after being immersed for 5 HRS 15 Roman 1000, was not inflected, and was then put into a solution 1 gr to 1 Rs of carbonate of ammonia after 2 HRS 40 Roman 1000. It was found considerably inflected, and the glands much blackened 4 of the leaves. After being immersed for 2 HRS 14 Roman 1000, were placed in 120 minims of a solution 1 gr to 20 Rs of phosphate of ammonia. They had already become slightly inflected from the hyoscyamus, probably owing to the presence of some albuminous matter, as formerly explained, but the inflection immediately increased, and after 1 HR was strongly pronounced, so that hyoscyamus does not act as a narcotic or poison. Poison from the fang of a living adder minute drops were placed on the glands of many tentacles. These were quickly inflected, just as if saliva had been given them next morning. After 17 HRS 30 Roman 1000, all were beginning to re-expand, and they appeared uninjured. Poison from the cobra. Dr. Farah, well known from his investigations on the poison of this deadly snake, was so kind as to give me some in a dried state. It is an albuminous substance, and is believed to replace the tyoline of saliva. A minute drop, about 120 of a minim of a solution of 1 part to 437 of water, was applied to the secretion round 4 glands, so that each received only about 138,400. A grain 16 mg. The operation was repeated on 4 other glands, and in 15 Roman 1000 several of the 8 tentacles became well inflected, and all of them in 2 HRS. Next morning, after 24 HRS, they were still inflected, and the glands of a very pale pink color. After an additional 24 HRS, they were nearly re-expanded, and completely so on the succeeding day, but most of the glands remained almost white. Half minims of the same solution were placed on the discs of 3 leaves, so that each received 1960 of a grain 675 mg. Dr. Farah, the Thanatophilia of India, 1872 p. 150 page 207. 4 HRS 15 Roman 1000, the outer tentacles were much inflected, and after 6 HRS 30 Roman 1000, those on 2 of the leaves were closely inflected, and the blade of 1, the 3rd leaf, was only moderately affected. The leaves remained in the same state during the next day, but after 48 HRS re-expanded. 3 leaves were now immersed each in 30 minims of the solution, so that each received 116 of a grain or 4 mg in 6 Roman 1000. There was some inflection which steadily increased, so that after 2 HRS 30 Roman 1000, all 3 leaves were closely inflected. The glands were at first somewhat darkened, then rendered pale, and the protoplasm within the cells of the tentacles was partially aggregated. The little masses of protoplasm were examined after 3 HRS and again after 7 HRS, and on no other occasion have I seen them undergoing such rapid changes of form. After 8 HRS 30 Roman 1000, the glands had become quite white. They had not secreted any great quantity of mucus. The leaves were now placed in water, and after 40 HRS re-expanded, showing that they were not much or at all injured during their immersion in water. The protoplasm within the cells of the tentacles was occasionally examined and always found in strong movement. 2 leaves were next immersed each in 30 minims of a much stronger solution of 1 part to 109 of water, so that each received 14 of a grain or 16 mg. After 1 HR 45 Roman 1000, the submarginal tentacles were strongly inflected, with the glands somewhat pale. After 3 HRS 30 Roman 1000, both leaves had all their tentacles closely inflected, and the glands white. Hence the weaker solution, as in so many other cases, induced more rapid inflection than the stronger one, but the glands were sooner rendered white by the latter. After an immersion of 24 HRS, some of the tentacles were examined, and the protoplasm, still of a fine purple color, was found aggregated into chains of small globular masses. These changed their shapes with remarkable quickness. After an immersion of 48 HRS, they were again examined, and their movements were so plain that they could easily be seen under a weak power. The leaves were now placed in water, and after 24 HRS Roman 1 E72 HRS, from their first immersion, the little masses of protoplasm, which had become of a dingy purple, were still in strong movement, changing their shapes, coalescing, and again separating. In 8 HRS, after these 2 leaves had been placed in water Roman 1e in 56 HRS, after their immersion in the solution, they began to re-expand, and by the next morning were more expanded. After an additional day Roman 1e on the 4th day, after their immersion in the solution, they were largely, but not quite fully, page 208, expanded. The tentacles were now examined, and the aggregated masses were almost wholly redissolved, the cells being filled with homogeneous purple fluid, with the exception here and there of a single globular mass. We thus see how completely the protoplasm had escaped all injury from the poison. As the glands were soon rendered quite white, it occurred to me that their texture might have been modified in such a manner as to prevent the poison passing into the cells beneath, and consequently, that the protoplasm within these cells had not been at all affected accordingly. I placed another leaf, which had been immersed for 48 HRS in the poison and afterwards for 24 HRS in water in a little solution of 1 part of carbonate of ammonia to 218 of water in 30 Roman 1000. The protoplasm in the cells beneath the glands became darker, and in the course of 24 HRS, the tentacles were filled down to their bases with dark-colored spherical masses. Hence, the glands had not lost their power of absorption as far as the carbonate of ammonia is concerned. From these facts, it is manifest that the poison of the cobra, though so deadly to animals, is not at all poisonous to Drosera, yet it causes strong and rapid inflection of the tentacles, and soon discharges all color from the glands. It seems even to act as a stimulant to the protoplasm, for after considerable experience in observing the movements of this substance in Drosera, I have never seen it on any other occasion in so active a state. I was therefore anxious to learn how this poison affected animal protoplasm, and Dr. Farah was so kind as to make some observations for me, which he has since published. Ciliated epithelium from the mouth of a frog was placed in a solution of 3 gram to 4 cubic Roman 900 of water, others being placed at the same time in pure water for comparison. The movements of the cilia in the solution seemed at first increased, but soon languished, and after between 15 and 20 minutes ceased, whilst those in the water were still acting vigorously. The white corpuscles of the blood of a frog, and the cilia on 2 infusorial animals, Paramecium and Volvox, were similarly affected by the poison. Dr. Farah also found that the muscle of a frog lost its irritability after an immersion of 20 Roman 1000 in the solution, not then responding to a strong electrical current. On the other hand, the movements of the cilia on the mantle of an unio were not always arrested, even when left for a consider. Proceedings of Royal Society, Feb. 18, 1875, page 209. Able time in a very strong solution. On the whole, it seems that the poison of the cobra acts far more injuriously on the protoplasm of the higher animals than on that of Drosera. There is one other point which may be noticed. I have occasionally observed that the drops of secretion round the glands were rendered somewhat turbid by certain solutions, and more especially by some acids, a film being formed on the surfaces of the drops, but I never saw this effect produced in so conspicuous a manner as by the cobra poison. When the stronger solution was employed, the drops appeared in 10 Roman 1000, like little white rounded clouds. After 48 HRS, the secretion was changed into threads and sheets of a membranous substance, including minute granules of various sizes. Camphor. Some scraped camphor was left for a day in a bottle with distilled water, and then filtered. A solution thus made is said to contain 11,000 of its weight of camphor. It smelt and tasted of this substance. 10 leaves were immersed in this solution. After 15 Roman 1005 of them were well inflected too, showing a first trace of movement in 11 Roman 1000 and 12 Roman 1000. The 6th leaf did not begin to move until 15 Roman 1000 had elapsed, but was fairly well inflected in 17 Roman 1000 and quite closed in 24 Roman 1000. The 7th began to move in 17 Roman 1000 and was completely shut in 26 Roman 1000. The 8th, 9th, and 10th leaves were old and of a very dark red color, and these were not inflected after an immersion of 24 HRS, so that in making experiments with camphor, it is necessary to avoid such leaves. Some of these leaves, on being left in the solution for 4 HRS, became of a rather dingy pink color and secreted much mucus, although their tentacles were closely inflected. The protoplasm within the cells was not at all aggregated on another occasion. However, after a longer immersion of 24 HRS, there was well-marked aggregation, a solution made by adding 2 drops of camphorated spirits to an ounce of water did not act on 1 leaf, whereas 30 minims added to an ounce of water acted on 2 leaves immersed together. Roman 1000 Vogel has shown that the flowers of various plants do not wither so soon when their stems are placed in a solution of camphor as when in water, and that if already slightly withered they recover more quickly. The germination of certain seeds is also accelerated by the solution, so that camphor acts as a stimulant, and it is the only known stimulant for plants. Gardner's Chronicle, 1874, p. 671. Nearly similar observations were made in 1798 by B. S. Barton, page 210. Wished therefore to ascertain whether camphor would render the leaves of Drosera more sensitive to mechanical irritation than they naturally are. 6 leaves were left in distilled water for 5 Roman 1000 or 6 Roman 1000, and then gently brushed twice or thrice whilst still under water with a soft camel hair brush, but no movement ensued. 9 leaves, which had been immersed in the above solution of camphor for the times stated in the following table, were next brushed only once with the same brush, and in the same manner as before. The results are given in the table. My first trials were made by brushing the leaves while still immersed in the solution, but it occurred to me that the viscid secretion round the glands would thus be removed, and the camphor might act more effectually on them. All the following trials, therefore, each leaf was taken out of the solution, waved for about 15s in water, then placed in fresh water and brushed, so that the brushing would not allow the freer access of the camphor, but this treatment made no difference in the results. Column 1 Number of leaves. Column 2 Length of immersion in the solution of camphor. Column 3 Length of time between the act of brushing and the inflection of the tentacles. Column 4 Length of time between the immersion of the leaves in the solution and the first sign of the inflection of the tentacles. 1.5 Roman 1003 Roman 1000 Considerable inflection. 4 Roman 1000 All the tentacles, except 3 or 4, inflected. 8 Roman 1005 Roman 1006 Roman 1000 First sign of inflection. 11 Roman 1005 Roman 1006 Roman 1000 S slight inflection. 7 Roman 1000 S plain inflection. 11 Roman 1000 S. 4 Roman 1000 S 2 Roman 1000 S a trace of inflection. 3 Roman 1000 plain. 4 Roman 1000 strongly marked. 7 Roman 1000 4 Roman 1000 2 Roman 1000 S a trace of inflection. 3 Roman 1000 plain inflection. 6 Roman 1000 S. 6 4 Roman 1002 Roman 1000 S decided inflection. 3 Roman 1000 S strongly marked. 6 Roman 1000 S. 7 4 Roman 1002 Roman 1000 S slight inflection. 3 Roman 1000 plain. 4 Roman 1000 well marked. 6 Roman 1000 S. 8 3 Roman 1002 Roman 1000 trace of inflection. 3 Roman 1000 considerable. 6 Roman 1000 strong inflection. 5 Roman 1003 Roman 1002 Roman 1000 trace of inflection. 3 Roman 1000 considerable. 6 Roman 1000 strong inflection. 5 Roman 1000 Other leaves were left in the solution without being brushed. One of these first showed a trace of inflection after 11 Roman 1000 a second. After 12 Roman 1005 were not inflected until 15 Roman 1000 had. Page 211 elapsed, and 2 not until a few minutes later. On the other hand, it will be seen in the right-hand column of the table that most of the leaves subjected to the solution and which were brushed became inflected in a much shorter time. The movement of the tentacles of some of these leaves was so rapid that it could be plainly seen through a very weak lens. 2 or 3 other experiments are worth giving. A large old leaf, after being immersed for 10 Roman 1000 in the solution, did not appear likely to be soon inflected, so I brushed it, and in 2 Roman 1000 it began to move, and in 3 Roman 1000 was completely shut. Another leaf, after an immersion of 15 Roman 1000, showed no signs of inflection. So was brushed, and in 4 Roman 1000 was grandly inflected. A third leaf, after an immersion of 17 Roman 1000 likewise, showed no signs of inflection. It was then brushed, but did not move for 1 HR, so that here was a failure. It was again brushed. And now in 9 Roman 1000 a few tentacles became inflected. The failure, therefore, was not complete. We may conclude that a small dose of camphor in solution is a powerful stimulant to Drosera. It not only soon excites the tentacles to bend, but apparently renders the glands sensitive to a touch which by itself does not cause any movement, or it may be that a slight mechanical irritation, not enough to cause any inflection, yet gives some tendency to movement, and thus reinforces the action of the camphor. This latter view would have appeared to me the more probable one had it not been shown by Roman 1000 Vogel that camphor is a stimulant in other ways to various plants and seeds. 2 plants bearing 4 or 5 leaves, and with their roots in a little cup of water, were exposed to the vapor of some bits of camphor about as large as a filbert nut under a vessel, holding 10 fluid hours. After 10 HRS no inflection ensued, but the glands appeared to be secreting more copiously. The leaves were in a narcotized condition, for on bits of meat being placed on 2 of them, there was no inflection. In 3 HRS 15 Roman 1000, and even after 13 HRS 15 Roman 1000, only a few of the outer tentacles were slightly inflected, but this degree of movement shows that the leaves had not been killed by an exposure during 10 HRS to the vapor of camphor. Oil of caraway water is said to dissolve about 1,000th part of its weight. Of this oil, a drop was added to an ounce of water, and the bottle occasionally shaken during a day, but many minute globules remained undissolved. 5 leaves were immersed in this mixture. In from 4 Roman 1000 to 5 Roman 1000, there was some inflection, which became moderately pronounced in 2 or page 212. 3 additional minutes after 14 Roman 1000, all 5 leaves were well, and some of them closely inflected. After 6 HRS, the glands were white, and much mucus had been secreted. The leaves were now flaccid, of a peculiar dull red color, and evidently dead. One of the leaves, after an immersion of 4 Roman 1000, was brushed like the leaves in the camphor, but this produced no effect. A plant with its roots in water was exposed under a 10 Rs vessel to the vapor of this oil, and in 1 HR 20 Roman 1001 leaf showed a trace of inflection. After 5 HRS 20 Roman 1000, the cover was taken off, and the leaves examined. One had all its tentacles closely inflected, the second about half in the same state, and the third all subinflected. The plant was left in the open air for 42 HRS, but not a single tentacle expanded. All the glands appeared dead, except here, and there one which was still secreting. It is evident that this oil is highly exciting and poisonous to Drosera. Oil of cloves. A mixture was made in the same manner as in the last case, and 3 leaves were immersed in it. After 30 Roman 1000, there was only a trace of inflection, which never increased. After 1 HR 30 Roman 1000, the glands were pale, and after 6 HRS white. No doubt the leaves were much injured or killed. Turpentine. Small drops placed on the discs of some leaves killed them, as did likewise drops of creosote. A plant was left for 15 Roman 1000 under a 12 Rs vessel, with its inner surface wetted with 12 drops of turpentine, but no movement of the tentacles ensued. After 24 HRS, the plant was dead. Glycerine half-minims were placed on the discs of 3 leaves. In 2 HRS, some of the outer tentacles were irregularly inflected, and in 19 HRS, the leaves were flaccid and apparently dead. The glands which had touched the glycerine were colorless. Minute drops, about 120 of a minim, were applied to the glands of several tentacles, and in a few minutes these moved and soon reached the center. Similar drops of a mixture of 4 drops to 1 Rs of water were likewise applied to several glands, but only a few of the tentacles moved, and these very slowly. And slightly half-minims of this same mixture, placed on the discs of some leaves, caused to my surprise no inflection. In the course of 48 HRS, bits of meat were then given them, and next day they were well inflected, notwithstanding that some of the discal glands had been rendered almost colorless 2 leaves were immersed in the same mixture, but only for 4 HRS they were not inflected, and on being afterwards left for 2 HRS 30 Roman 1000 in a solution 1 gr to 1 Rs of carbonate of ammonia, their glands were blackened, their tentacles inflected, and the protoplasm within their cells aggregated. It appears page 213 from these facts that a mixture of 4 drops of glycerine to an ounce of water is not poisonous and excites very little inflection, but that pure glycerine is poisonous, and if applied in very minute quantities to the glands of the outer tentacles, causes their inflection. The effects of immersion in water and in various solutions on the subsequent action of phosphate and carbonate of ammonia have seen in the 3rd and 7th chapters that immersion in distilled water causes after a time some degree of aggregation of the protoplasm and a moderate amount of inflection, especially in the case of plants which have been kept at a rather high temperature. Water does not excite a copious secretion of mucus. We have here to consider the effects of immersion in various fluids on the subsequent action of salts of ammonia and other stimulants. 4 leaves, which had been left for 24 HRS in water, were given bits of meat but did not clasp them. 10 leaves, after a similar immersion, were left for 24 HRS in a powerful solution 1 gr to 20 Rs of phosphate of ammonia, and only 1 showed even a trace of inflection. 3 of these leaves, on being left for an additional day in the solution, still remained quite unaffected, when however some of these leaves which had been first immersed in water for 24 HRS, and then in the phosphate for 24 HRS, were placed in a solution of carbonate of ammonia 1 part to 218 of water, the protoplasm in the cells of the tentacles became in a few hours strongly aggregated, showing that this salt had been absorbed and taken effect. A short immersion in water for 20 Roman 1000 did not retard the subsequent action of the phosphate or of splinters of glass placed on the glands, but in 2 instances an immersion for 50 Roman 1000 prevented any effect from a solution of camphor. Several leaves, which had been left for 20 Roman 1000 in a solution of 1 part of white sugar to 218 of water, were placed in the phosphate solution, the action of which was delayed, whereas a mixed solution of sugar and the phosphate did not in the least interfere with the effects of the latter. 3 leaves, after being immersed for 20 Roman 1000 in the sugar solution, were placed in a solution of carbonate of ammonia 1 part to 218 of water in 2 Roman 1000 or 3 Roman 1000. The glands were blackened, and after 7 Roman 1000, the tentacles were considerably inflected, so that the solution of sugar, though it delayed the action of the phosphate, did not delay that of the carbonate. Immersion in a similar solution of gum arabic for 20 Roman 1000 had no retarding action on the phosphate. 3 leaves were left for 20 Roman 1000 in a mixture of 1 part of alcohol to 7 parts of water, page 214, and then placed in the phosphate solution in 2 HRS 15 Roman 1000. There was a trace of inflection in 1 leaf, and in 5 HRS 30 Roman 1000 a second was slightly affected. The inflection subsequently increased, though slowly. Hence diluted alcohol, which, as we shall see, is hardly at all poisonous, plainly retards the subsequent action of the phosphate. It was shown in the last chapter that leaves which did not become inflected by nearly a day's immersion in solutions of various salts and acids behaved very differently from one another when subsequently placed in the phosphate solution. I here give a table summing up the results. Column 1, name of the salts and acids in solution. Column 2, period of immersion of the leaves in solutions of 1 part to 437 of water. Column 3, effects produced on the leaves by their subsequent immersion for stated periods in a solution of 1 part of phosphate of ammonia to 8750 of water, or 1 gr to 20 Rs. Rubidium chloride 22 HRS after 30 Roman 1000 strong inflection of the tentacles. Potassium carbonate 20 Roman 1000 scarcely any inflection until 5 HRS had elapsed. Calcium acetate 24 HRS after 24 HRS very slight inflection. Calcium nitrate 24 HRS doo-doo. Magnesium acetate 22 HRS some slight inflection, which became well pronounced in 24 HRS. Magnesium nitrate 22 HRS, after 4 HRS 30 Roman 1000 a fair amount of inflection, which never increased. Magnesium chloride 22 HRS, after a few minutes great inflection, after 4 HRS, all 4 leaves, with almost every tentacle closely inflected. Barium acetate 22 HRS, after 24 HRS2, leaves out of 4 slightly inflected. Barium nitrate 22 HRS, after 30 Roman 1001, leaf greatly, and 2 others moderately inflected they remained, thus, for 24 HRS. Strontium acetate 22 HRS, after 25 Roman 1002, leaves greatly inflected, after 8 HRS, a third leaf moderately, and the fourth very slightly inflected, all 4 thus remained for 24 HRS. Strontium nitrate 22 HRS, after 8 HRS3, leaves out of 5 moderately inflected, after 24 HRS, all 5 in this state, but not 1 closely inflected. Aluminum chloride 24 HRS3 leaves, which had either been slightly or not at all affected by the chloride, became, after 7 HRS 30 Roman 1000, rather closely inflected, page 215. Column 1 name of the salts and acids in solution, column 2 period of immersion of the leaves, in solutions of 1 part to 437 of water, column 3, effects produced on the leaves by their subsequent immersion for stated periods, in a solution of 1 part of phosphate of ammonia to 8750 of water, or 1 gr to 20 Rs. Aluminum nitrate 24 HRS, after 25 HRS slight and doubtful effect. Lead chloride 23 HRS, after 24 HRS2 leaves, somewhat inflected the third very little, and thus remained. Manganese chloride 22 HRS, after 48 HRS, not the least inflection. Lactic acid 48 HRS, after 24 HRS, a trace of inflection in a few tentacles, the glands of which had not been killed by the acid. Tannic acid 24 HRS, after 24 HRS, no inflection. Tartaric acid 24 HRS doo-doo. Citric acid 24 HRS, after 50 Roman 1000 tentacles decidedly inflected, and after 5 HRS, strongly inflected, so remained for the next 24 HRS. Formic acid 22 HRS, not observed, until 24 HRS had elapsed, tentacles considerably inflected, and protoplasm aggregated. In a large majority of these 20 cases, a varying degree of inflection was slowly caused by the phosphate. In 4 cases, however, the inflection was rapid, occurring in less than half an hour, or at most, in 50 Roman 1000, in 3 cases, the phosphate did not produce the least effect. Now what are we to infer from these facts? We know from 10 trials that immersion in distilled water for 24 HRS prevents the subsequent action of the phosphate solution. It would therefore appear as if the solutions of chloride of manganese, tannic, and tartaric acids, which are not poisonous, acted exactly like water for the phosphate, produced no effect on the leaves, which had been previously immersed in these 3 solutions. The majority of the other solutions behaved to a certain extent like water, for the phosphate produced, after a considerable interval of time, only a slight effect. On the other hand, the leaves which had been immersed in the solutions of the chloride of rubidium and magnesium of acetate of strontium nitrate of barium and citric acid were quickly acted on by the phosphate. Now was water absorbed from these 5 weak solutions, and yet owing to the presence of the salts, did not prevent the subsequent action of the phosphate. Or page 216, may we not suppose that the interstices of the walls of the glands were blocked up with the molecules of these 5 substances, so that they were rendered impermeable to water, for had water entered, we know from the 10 trials that the phosphate would not afterwards have produced any effect. It further appears that the molecules of the carbonate of ammonia can quickly pass into glands, which from having been immersed for 20 Roman 1000 in a weak solution of sugar, either absorb the phosphate very slowly or are acted on by it very slowly. On the other hand, glands, however they may have been treated, seem easily to permit the subsequent entrance of the molecules of carbonate of ammonia. Thus leaves, which had been immersed in a solution of 1 part to 437 of water of nitrate of potassium, for 48 Hr soft sulfate of potassium, for 24 Hr sand of the chloride of potassium, for 25 Hr son being placed in a solution of 1 part of carbonate of ammonia to 218 of water, had their glands immediately blackened, and after 1 Hr their tentacles somewhat inflected, and the protoplasm aggregated, but it would be an endless task to endeavor to ascertain the wonderfully diversified effects of various solutions on Drosera. Alcohol 1 part to 7 of water. It has already been shown that half minims of this strength placed on the discs of leaves do not cause any inflection, and that when 2 days afterwards the leaves were given bits of meat, they became strongly inflected. 4 leaves were immersed in this mixture, and 2 of them after 30 Roman 1000 were brushed with a camel hair brush like the leaves in the solution of camphor, but this produced no effect. See Dr. Roman 1000 Traub's curious experiments on the production of artificial cells and on their permeability to various salts, described in his papers Experiment zur Theorie der Zellenbildung und Endosmos Breslau 1866 and Experiment zur physikalischen Erklärung der Bildung der Zellhaut ihres Wachstums durch Interzeption Breslau 1874. These researches perhaps explain my results. Dr. Traub commonly employed as a membrane the precipitate formed when tannic acid comes into contact with a solution of gelatine by allowing a precipitation of sulfate of barium to take place at the same time. The membrane becomes infiltrated with this salt, and in consequence of the intercalation of molecules of sulfate of barium among those of the gelatine precipitate, the molecular interstices in the membrane are made smaller. In this altered condition, the membrane no longer allows the passage through it of either sulfate of ammonia or nitrate of barium, though it retains its permeability for water and chloride of ammonia, page 217. Nor did these 4 leaves, on being left for 24 HRS in the diluted alcohol, undergo any inflection. They were then removed, one being placed in an infusion of raw meat, and bits of meat on the discs of the other 3, with their stalks in water. Next day, one seemed a little injured, whilst 2 others showed merely a trace of inflection. We must, however, bear in mind that immersion for 24 HRS in water prevents leaves from clasping meat, hence alcohol of the above strength is not poisonous, nor does it stimulate the leaves like camphor does. The vapor of alcohol acts differently. A plant having 3 good leaves was left for 25 Roman 1000 under a receiver holding 19 Rs with 60 minims of alcohol. In a watchglass no movement ensued, but some few of the glands were blackened and shriveled, whilst many became quite pale. These were scattered over all the leaves in the most irregular manner, reminding me of the manner in which the glands were affected by the vapor of carbonate of ammonia. Immediately on the removal of the receiver, particles of raw meat were placed on many of the glands, those which retained their proper color being chiefly selected, but not a single tentacle was inflected during the next 4 HRS. After the first 2 HRS, the glands on all the tentacles began to dry, and next morning, after 22 HRS, all 3 leaves appeared almost dead, with their glands dry, the tentacles on 1 leaf alone being partially inflected. A second plant was left for only 5 Roman 1000 with some alcohol in a watchglass under a 12 Rs receiver, and particles of meat were then placed on the glands of several tentacles. After 10 Roman 1000, some of them began to curve inwards, and after 55 Roman 1000, nearly all were considerably inflected, but a few did not move. Some anesthetic effect is here probable, but by no means certain. A third plant was also left for 5 Roman 1000 under the same small vessel, with its whole inner surface wetted with about a dozen drops of alcohol. Particles of meat were now placed on the glands of several tentacles, some of which first began to move in 25 Roman 1000. After 40 Roman 1000, most of them were somewhat inflected, and after 1 HR 10 Roman 1000, almost all were considerably inflected from their slow rate of movement. There can be no doubt that the glands of these tentacles had been rendered insensible for a time by exposure during 5 Roman 1000 to the vapor of alcohol. Vapor of chloroform. The action of this vapor on Drosera is very variable depending, I suppose, on the constitution or age of the plant, or on some unknown condition. It sometimes causes the tentacles to move with extraordinary rapidity, and sometimes produces no such effect. The glands are sometimes, page 218, rendered for a time insensible to the action of raw meat, but sometimes are not thus affected, or in a very slight degree. A plant recovers from a small dose, but is easily killed by a larger one. A plant was left for 30 Roman 1000 under a bell glass holding 19 fluid Rs, 539 Roman 1050, with 8 drops of chloroform, and before the cover was removed, most of the tentacles became much inflected, though they did not reach the center. After the cover was removed, bits of meat were placed on the glands of several of the somewhat incurved tentacles. These glands were found much blackened after 6 HRS 30 Roman 1000, but no further movement ensued. After 24 HRS, the leaves appeared almost dead. A smaller bell glass holding 12 fluid Rs 340 Roman 1050 was now employed, and a plant was left for 90 S under it, with only 2 drops of chloroform. Immediately on the removal of the glass, all the tentacles curved inwards so as to stand perpendicularly up, and some of them could actually be seen moving with extraordinary quickness by little starts and therefore in an unnatural manner, but they never reached the center. After 22 HRS, they fully re-expanded, and on meat being placed on their glands, or when roughly touched by a needle, they promptly became inflected, so that these leaves had not been in the least injured. Another plant was placed under the same small bell glass with 3 drops of chloroform, and before 2 minutes had elapsed, the tentacles began to curl inwards with rapid little jerks. The glass was then removed, and in the course of 2 or 3 additional minutes, almost every tentacle reached the center. On several other occasions, the vapor did not excite any movement of this kind. There seems also to be great variability in the degree and manner in which chloroform renders the glands insensible to the subsequent action of meat in the plant last referred to, which had been exposed for 2 Roman 1000 to 3 drops of chloroform. Some few tentacles curved up only to a perpendicular position, and particles of meat were placed on their glands. This caused them in 5 Roman 1000 to begin moving, but they moved so slowly that they did not reach the center until 1 HR 30 Roman 1000 had elapsed. Another plant was similarly exposed, that is for 2 Roman 1000 to 3 drops of chloroform, and on particles of meat being placed on the glands of several tentacles which had curved up into a perpendicular position. One of these began to bend in 8 Roman 1000, but afterwards moved very slowly, whilst none of the other tentacles, page 219, moved for the next 40 Roman 1000. Nevertheless, in 1 HR 45 Roman 1000, from the time when the bits of meat had been given, all the tentacles reached the center. In this case, some slight anesthetic effect apparently had been produced. On the following day, the plant had perfectly recovered. Another plant bearing 2 leaves was exposed for 2 Roman 1000 under the 19 Rs vessel to 2 drops of chloroform. It was then taken out and examined again, exposed for 2 Roman 1000 to 2 drops, taken out and re-exposed for 3 Roman 1000 to 3 drops, so that altogether it was exposed alternately to the air, and during 7 Roman 1000 to the vapor of 7 drops of chloroform. Bits of meat were now placed on 13 glands. On the 2 leaves, on one of these leaves, a single tentacle first began moving in 40 Roman 1000, and 2 others in 54 Roman 1000 on the second leaf. Some tentacles first moved in 1 HR 11 Roman 1000. After 2 HRS, many tentacles on both leaves were inflected, but none had reached the center within this time. In this case, there could not be the least doubt that the chloroform had exerted an anesthetic influence on the leaves. On the other hand, another plant was exposed under the same vessel for a much longer time, viz. 20 Roman 1000 to twice as much chloroform. Bits of meat were then placed on the glands of many tentacles, and all of them, with a single exception, reached the center in from 13 Roman 1000 to 14 Roman 1000. In this case, little or no anesthetic effect had been produced, and how to reconcile these discordant results I know not. Vapor of sulfuric ether plant was exposed for 30 Roman 1000 to 30 minims of this ether in a vessel holding 19 Rs, and bits of raw meat were afterwards placed on many glands which had become pale-colored, but none of the tentacles moved after 6 HRS 30 Roman 1000. The leaves appeared sickly, and the discal glands were almost dry by the next morning. Many of the tentacles were dead, as were all those on which meat had been placed, showing that matter had been absorbed from the meat, which had increased the evil effects of the vapor. After 4 days the plant itself died. Another plant was exposed in the same vessel for 15 Roman 1000 to 40 minims. One young small and tender leaf had all its tentacles inflected, and seemed much injured. Bits of raw meat were placed on several glands on 2 other and older leaves. These glands became dry after 6 HRS and seemed injured. The tentacles never moved, excepting one, which was ultimately a little inflected. The glands of the other tentacles continued to secrete and appeared uninjured, but the whole plant, after 3 days, became very sickly. Page 220. In the 2 foregoing experiments, the doses were evidently too large and poisonous. With weaker doses, the anesthetic effect was variable. As in the case of chloroform, a plant was exposed for 5 Roman 1000 to 10 drops under a 12 Rs vessel, and bits of meat were then placed on many glands. None of the tentacles thus treated began to move in a decided manner until 40 Roman 1000 had elapsed, but then some of them moved very quickly, so that 2 reached the center after an additional interval of only 10 Roman 1000 in 2 HRS 12 Roman 1000 from the time when the meat was given. All the tentacles reached the center. Another plant with 2 leaves was exposed in the same vessel for 5 Roman 1000 to a rather larger dose of ether, and bits of meat were placed on several glands. In this case, 1 tentacle on each leaf began to bend in 5 Roman 1000, and after 12 Roman 1002 tentacles on 1 leaf and 1 on the second leaf reached the center in 30 Roman 1000 after the meat had been given. All the tentacles, both those with and without meat, were closely inflected, so that the ether apparently had stimulated these leaves, causing all the tentacles to bend. Vapor of nitric ether. This vapor seems more injurious than that of sulfuric ether. A plant was exposed for 5 Roman 1000 in a 12 Rs vessel to 8 drops in a watchglass, and I distinctly saw a few tentacles curling inwards. Before the glass was removed, immediately afterwards bits of meat were placed on 3 glands. But no movement ensued in the course of 18 Roman 1000. The same plant was placed again under the same vessel for 16 Roman 1000, with 10 drops of the ether. None of the tentacles moved, and next morning those with the meat were still in the same position. After 48 HRS, 1 leaf seemed healthy, but the others were much injured. Another plant having 2 good leaves was exposed for 6 Roman 1000 under a 19 Rs vessel to the vapor from 10 minims of the ether, and bits of meat were then placed on the glands of many tentacles on both leaves. After 36 Roman 1000, several of them on 1 leaf became inflected, and after 1 HR, almost all the tentacles, those with and without meat, nearly reached the center. On the other leaf, the glands began to dry in 1 HR 40 Roman 1000, and after several hours, not a single tentacle was inflected, but by the next morning, after 21 HRS, many were inflected, though they seemed much injured in this and the previous experiment. It is doubtful, owing to the injury which the leaves had suffered, whether any anesthetic effect had been produced. A third plant having 2 good leaves was exposed for only 4 Roman 1000 in the 19 Rs vessel to the vapor from 6 drops. Bits of meat were then placed on the glands of 7 tentacles on the page 221 same leaf. A single tentacle moved after 1 HR 23 Roman 1000. After 2 HRS 3 Roman 1000, several were inflected, and after 3 HRS 3 Roman 1000, all the 7 tentacles with meat were well inflected. From the slowness of these movements, it is clear that this leaf had been rendered insensible for a time to the action of the meat. A second leaf was rather differently affected. Bits of meat were placed on the glands of 5 tentacles, 3 of which were slightly inflected. in 28 Roman 1000, after one HR 21 Roman 1001 reached the center, but the other two were still only slightly inflected. After 3 HRS, they were much more inflected, but even after 5 HRS 16 Roman 1000, all 5 had not reached the center. Although some of the tentacles began to move moderately soon, they afterwards moved with extreme slowness. By next morning, after 20 HRS, most of the tentacles on both leaves were closely inflected, but not quite regularly. After 48 HRS, neither leaf appeared injured, though the tentacles were still inflected. After 72 HRS, one was almost dead, whilst the other was re-expanding and recovering. Carbonic acid. A plant was placed under a 122 Rs Bell glass filled with this gas and standing over water, but I did not make sufficient allowance for the absorption of the gas by the water, so that towards the latter part of the experiment some air was drawn in. After an exposure of 2 HRS, the plant was removed, and bits of raw meat placed on the glands of 3 leaves. One of these leaves hung a little down, and was at first partly and soon afterwards completely covered by the water which rose within the vessel. As the gas was absorbed on this latter leaf, the tentacles to which meat had been given became well inflected in 2 Roman 1000 s, that is at about the normal rate, so that until I remembered that the leaf had been protected from the gas, and might perhaps have absorbed oxygen from the water, which was continually drawn inwards, I falsely concluded that the carbonic acid had produced no effect on the other 2 leaves. The tentacles with meat behaved very differently from those on the first leaf. 2 of them first began to move slightly in 1 HR 50 Roman 1000, always reckoning from the time when the meat was placed on the glands, were plainly inflected in 2 HRS 22 Roman 1000, and in 3 HRS 22 Roman 1000 reached the center. 3 other tentacles did not begin to move until 2 HRS 20 Roman 1000 had elapsed, but reached the center at about the same time with the others, viz. in 3 HRS 22 Roman 1000. This experiment was repeated several times with nearly the same results, excepting that the interval before the tentacles began to move varied a little. I will give only one other case. Page 222. A plant was exposed in the same vessel to the gas for 45 Roman 1000, and bits of meat were then placed on 4 glands, but the tentacles did not move for 1 HR 40 Roman 1000. After 2 HRS 30 Roman 1000, all 4 were well inflected, and after 3 HRS reached the center. The following singular phenomenon sometimes, but by no means always, occurred. A plant was immersed for 2 HRS, and bits of meat were then placed on several glands. In the course of 13 Roman 1000, all the submarginal tentacles on one leaf became considerably inflected, those with the meat not in the least degree more than the others on a second leaf, which was rather old. The tentacles with meat, as well as a few others, were moderately inflected on a third leaf. All the tentacles were closely inflected, though meat had not been placed on any of the glands. This movement, I presume, may be attributed to excitement from the absorption of oxygen. The last mentioned leaf, to which no meat had been given, was fully re-expanded after 24 HRS, whereas the 2 other leaves had all their tentacles closely inflected over the bits of meat, which by this time had been carried to their centers. Thus these 3 leaves had perfectly recovered from the effects of the gas in the course of 24 HRS. On another occasion, some fine plants, after having been left for 2 HRS in the gas, were immediately given bits of meat in the usual manner, and on their exposure to the air most of their tentacles became, in 12 Roman 1000, curved into a vertical or subvertical position, but in an extremely irregular manner. Some only on one side of the leaf, and some on the other. They remained in this position for some time, the tentacles with the bits of meat not having at first moved more quickly or farther inwards than the others without meat. But after 2 HRS 20 Roman 1000, the former began to move and steadily went on bending until they reached the center. Next morning, after 22 HRS, all the tentacles on these leaves were closely clasped over the meat, which had been carried to their centers, whilst the vertical and subvertical tentacles on the other leaves, to which no meat had been given, had fully re-expanded. Judging, however, from the subsequent action of a weak solution of carbonate of ammonia on one of these latter leaves, it had not perfectly recovered its excitability and power of movement in 22 HRS, but another leaf, after an additional 24 HRS, had completely recovered, judging from the manner in which it clasped a fly placed on its disc. I will give only one other experiment. After the exposure of a plant for 2 HRS to the gas, one of its leaves was immersed in a rather strong solution of carbonate of ammonia together with page 223, a fresh leaf from another plant. The latter had most of its tentacles strongly inflected within 30 Roman 1000, whereas the leaf which had been exposed to the carbonic acid remained for 24 HRS in the solution without undergoing any inflection, with the exception of 2 tentacles. This leaf had been almost completely paralyzed and was not able to recover its sensibility whilst still in the solution, which, from having been made with distilled water, probably contained little oxygen. Concluding remarks on the effects of the foregoing agents: as the glands, when excited, transmit some influence to the surrounding tentacles, causing them to bend, and their glands to pour forth an increased amount of modified secretion, I was anxious to ascertain whether the leaves included any element having the nature of nerve tissue, which, though not continuous, served as the channel of transmission. This led me to try the several alkaloids and other substances which are known to exert a powerful influence on the nervous system of animals. I was at first encouraged in my trials by finding that strychnine, digitaline, and nicotine, which all act on the nervous system, were poisonous to Drosera and caused a certain amount of inflection. Hydrocyanic acid, again, which is so deadly a poison to animals, caused rapid movement of the tentacles, but as several innocuous acids, though much diluted, such as benzoic acetic Roman 100, as well as some essential oils, are extremely poisonous to Drosera and quickly cause strong inflection. It seems probable that strychnine, nicotine, digitaline, and hydrocyanic acid excite inflection by acting on elements in no way analogous to the nervous cells of animals. If elements of this latter nature had been present in the leaves, it might have been expected that morphia, hyoscyamus, atropine, veratrine, colchicine, curare, and diluted alcohol would have produced some marked effect, whereas, page 224, these substances are not poisonous and have no power, or only a very slight one, of inducing inflection. It should, however, be observed that curare, colchicine, and veratrine are muscle poisons that act on nerves having some special relation with the muscles, and therefore could not be expected to act on Drosera. The poison of the cobra is most deadly to animals by paralyzing their nervous centers, yet is not in the least so to Drosera, though quickly causing strong inflection. Notwithstanding the foregoing facts, which show how widely different is the effect of certain substances on the health or life of animals and of Drosera, yet there exists a certain degree of parallelism in the action of certain other substances. We have seen that this holds good in a striking manner with the salts of sodium and potassium, again various metallic salts, and acids, namely those of silver, mercury, gold, tin, arsenic, chromium, copper, and platina, most or all of which are highly poisonous to animals, are equally so to Drosera. But it is a singular fact that the chloride of lead and 2 salts of barium were not poisonous to this plant. It is an equally strange fact that though acetic and propionic acids are highly poisonous, their ally formic acid is not so, and that whilst certain vegetable acids, namely oxalic, benzoic, Roman 100, are poisonous in a high degree, gallic, tannic, tartaric, and malic, all diluted to an equal degree, are not so. Malic acid induces inflection, whilst the 3 other just named vegetable acids have no such power, but a pharmacopoeia would be requisite to describe the diversified effects of various substances on Drosera. Dr. Farah, The Thanatophytia of India, 1872, p. 4. Seeing that acetic, hydrocyanic, and chromic acids, acetate of strychnine, and vapor of ether, are poisonous to Drosera, page 225, it is remarkable that Dr. Ransom, Philosoph, Transact, 1867, p. 480, who used much stronger solutions of these substances than I did, states that the rhythmic contractility of the yolk of the ova of the pike is not materially influenced by any of the poisons used, which did not act chemically, with the exception of chloroform and carbonic acid. I find it stated by several writers that curare has no influence on sarcode or protoplasm, and we have seen that though curare excites some degree of inflection, it causes very little aggregation of the protoplasm, page 226. Of the alkaloids and their salts, which were tried several, had not the least power of inducing inflection. Others, which were certainly absorbed as shown by the changed color of the glands, had but a very moderate power of this kind. Others again, such as the acetate of quinine and digitaline, caused strong inflection. The several substances mentioned in this chapter affect the color of the glands very differently. These often become dark at first, and then very pale or white, as was conspicuously the case with glands subjected to the poison of the cobra and citrate of strychnine. In other cases, they are from the first rendered white, as with leaves placed in hot water, and several acids, and this, I presume, is the result of the coagulation of the albumin on the same leaf. Some glands become white, and others dark-colored, as occurred with leaves, in a solution of the sulfate of quinine, and in the vapor of alcohol. Prolonged immersion in nicotine curare, and even water blackens the glands, and this, I believe, is due to the aggregation of the protoplasm within their cells, yet curare caused very little aggregation in the cells of the tentacles, whereas nicotine and sulfate of quinine induced strongly marked aggregation down their bases. The aggregated masses in leaves, which had been immersed for 3 HRS 15 Roman 1000 in a saturated solution of sulfate of quinine, exhibited incessant, page 226, changes of form, but after 24 HRS were motionless, the leaf being flaccid and apparently dead. On the other hand, with leaves subjected for 48 HRS to a strong solution of the poison of the cobra, the protoplasmic masses were unusually active, whilst, with the higher animals, the vibratile cilia and white corpuscles of the blood seemed to be quickly paralyzed by this substance. With the salts of alkalis and earths, the nature of the base, and not that of the acid, determines their physiological action on Drosera, as is likewise the case with animals, but this rule hardly applies to the salts of quinine and strychnine, for the acetate of quinine causes much more inflection than the sulfate, and both are poisonous, whereas the nitrate of quinine is not poisonous and induces inflection at a much slower rate than the acetate. The action of the citrate of strychnine is also somewhat different from that of the sulfate. Leaves which have been immersed for 24 HRS in water, and for only 20 Roman 1000 in diluted alcohol or in a weak solution of sugar, are afterwards acted on very slowly or not at all by the phosphate of ammonia, though they are quickly acted on by the carbonate immersion for 20 Roman 1000 in a solution of gum arabic, has no such inhibitory power. The solutions of certain salts and acids affect the leaves with respect to the subsequent action of the phosphate exactly like water, whilst others allow the phosphate afterwards to act quickly and energetically. In this latter case, the interstices of the cell walls may have been blocked up by the molecules of the salts first given in solution, so that water could not afterwards enter, though the molecules of the phosphate could do so, and those of the carbonate still more easily. Page 227. The action of camphor dissolved in water is remarkable, for it not only soon induces inflection, but apparently renders the glands extremely sensitive to mechanical irritation, for if they are brushed with a soft brush after being immersed in the solution for a short time, the tentacles begin to bend in about 2 Roman 1000. It may, however, be that the brushing, though not a sufficient stimulus by itself, tends to excite movement merely by reinforcing the direct action of the camphor. The vapor of camphor, on the other hand, serves as a narcotic. Some essential oils, both in solution and in vapor, cause rapid inflection. Others have no such power. Those which I tried were all poisonous. Diluted alcohol 1 part to 7 of water is not poisonous, does not induce inflection, nor increase the sensitiveness of the glands to mechanical irritation. The vapor acts as a narcotic or anesthetic, and long exposure to it kills the leaves. The vapors of chloroform sulfuric and nitric ether act in a singularly variable manner on different leaves, and on the several tentacles of the same leaf. This, I suppose, is owing to differences in the age or constitution of the leaves, and to whether certain tentacles have lately been in action that these vapors are absorbed by the glands is shown by their changed color. But as other plants not furnished with glands are affected by these vapors, it is probable that they are likewise absorbed by the stomata of Drosera. They sometimes excite extraordinarily rapid inflection, but this is not an invariable result. If allowed to act for even a moderately long time, they kill the leaves, whilst a small dose, acting for only a short time, serves as a narcotic or anesthetic in this case. The tentacles, whether or not they have page 228, become inflected, are not excited to further movement by bits of meat placed on the glands. Until some considerable time has elapsed, it is generally believed that with animals and plants, these vapors act by arresting oxidation. Exposure to carbonic acid for 2-HRS, and in one case for only 45 Roman 1000 likewise, rendered the glands insensible for a time to the powerful stimulus of raw meat. The leaves, however, recovered their full powers, and did not seem in the least injured on being left in the air for 24 or 48 HRS. We have seen in the 3rd chapter that the process of aggregation in leaves subjected for 2 hours to this gas, and then immersed in a solution of the carbonate of ammonia, is much retarded, so that a considerable time elapses before the protoplasm in the lower cells of the tentacles becomes aggregated, in some cases soon after the leaves were removed from the gas and brought into the air. The tentacles moved spontaneously, this being due, I presume, to the excitement from the access of oxygen. These inflected tentacles, however, could not be excited for some time afterwards to any further movement by their glands being stimulated with other irritable plants. It is known that the exclusion of oxygen prevents their moving and arrests the movements of the protoplasm within their cells, but this arrest is a different phenomenon from the retardation of the process of aggregation, just alluded to. Whether this latter fact ought to be attributed to the direct action of the carbonic acid or to the exclusion of oxygen, I know not. Sachs Trait Debat, 1874 pp. 846, 1037 page 229. Chapter Roman 10, on the sensitiveness of the leaves and on the lines of transmission of the motor impulse. Glands and summits of the tentacles alone sensitive transmission of the motor impulse down the pedicels of the tentacles and across the blade of the leaf aggregation of the protoplasm, a reflex action first discharge of the motor impulse sudden direction of the movements of the tentacles motor impulse, transmitted through the cellular tissue mechanism of the movement's nature of the motor impulse re-expansion of the tentacles. We have seen in the previous chapters that many widely different stimulants, mechanical and chemical, excite the movement of the tentacles as well as of the blade of the leaf. And we must now consider, firstly, what are the points which are irritable or sensitive, and secondly, how the motor impulse is transmitted from one point to another. The glands are almost exclusively the seat of irritability, yet this irritability must extend for a very short distance below them, for when they were cut off with a sharp pair of scissors, without being themselves touched, the tentacles often became inflected. These headless tentacles frequently re-expanded, and when afterwards drops of the 2 most powerful known stimulants were placed on the cut-off ends, no effect was produced. Nevertheless, these headless tentacles are capable of subsequent inflection, if excited by an impulse sent from the disc. Succeeded on several occasions in crushing glands between fine pincers, but this did not excite any movement, nor did raw meat and salts of ammonia. When placed on such crushed glands, page 230, it is probable that they were killed so instantly that they were not able to transmit any motor impulse, for in 6 observed cases, in 2 of which, however, the gland was quite pinched off, the protoplasm within the cells of the tentacles did not become aggregated, whereas in some adjoining tentacles, which were inflected from having been roughly touched by the pincers, it was well aggregated. In like manner, the protoplasm does not become aggregated when a leaf is instantly killed by being dipped into boiling water. On the other hand, in several cases, in which tentacles became inflected after their glands had been cut off with sharp scissors, a distinct though moderate degree of aggregation supervened. The pedicels of the tentacles were roughly and repeatedly rubbed. Raw meat or other exciting substances were placed on them, both on the upper surface near the base and elsewhere, but no distinct movement ensued. Some bits of meat, after being left for a considerable time on the pedicels, were pushed upwards so as just to touch the glands, and in a minute the tentacles began to bend. I believe that the blade of the leaf is not sensitive to any stimulant. I drove the point of a lancet through the blades of several leaves, and a needle 3 or 4 times through 19 leaves. In the former case, no movement ensued, but about a dozen of the leaves, which were repeatedly pricked, had a few tentacles irregularly inflected, as however their backs had to be supported during the operation. Some of the outer glands, as well as those on the disc, may have been touched, and this perhaps sufficed to cause the slight degree of movement observed. Nitschke says. Bot Zeitung, 1860p, 230, 4 page 231. That cutting and pricking the leaf does not excite movement. The petiole of the leaf is quite insensible. The backs of the leaves bear numerous minute papillae, which do not secrete, but have the power of absorption. These papillae are, I believe, rudiments of formerly existing tentacles together with their glands. Many experiments were made to ascertain whether the backs of the leaves could be irritated in any way, 37 leaves being thus tried. Some were rubbed for a long time with a blunt needle and drops of milk and other exciting fluids, raw meat, crushed flies, and various substances placed on others. These substances were apt soon to become dry, showing that no secretion had been excited. Hence I moistened them with saliva solutions of ammonia, weak hydrochloric acid, and frequently with the secretion from the glands of other leaves. I also kept some leaves on the backs of which exciting objects had been placed under a damp bellglass. But with all my care, I never saw any true movement. I was led to make so many trials, because contrary to my previous experience, Nitschke states that after affixing objects to the backs of leaves by the aid of the viscid secretion, he repeatedly saw the tentacles, and in one instance the blade become reflexed. This movement, if a true one, would be most anomalous, for it implies that the tentacles receive a motor impulse from an unnatural source, and have the power of bending in a direction exactly the reverse of that which is habitual to them, this power not being of the least use to the plant, as insects cannot adhere to the smooth backs of the leaves. I have said that no effect was produced in the above. Bot Zeitung, 1860p, 430, 7 page 232. Cases, but this is not strictly true, for in 3 instances a little syrup was added to the bits of raw meat on the backs of leaves in order to keep them damp for a time, and after 36 HRS there was a trace of reflection in the tentacles of one leaf, and certainly in the blade of another. After 12 additional hours the glands began to dry, and all 3 leaves seemed much injured. 4 leaves were then placed under a bellglass with their footstalks in water with drops of syrup on their backs, but without any meat. 2 of these leaves after a day had a few tentacles reflexed. The drops had now increased considerably in size from having imbibed moisture, so as to trickle down the backs of the tentacles and footstalks. On the 2nd day 1 leaf had its blade much reflexed. On the 3rd day the tentacles of 2 were much reflexed, as well as the blades of all 4 to a greater or less degree. The upper side of 1 leaf, instead of being as at first slightly concave, now presented a strong convexity upwards. Even on the 5th day the leaves did not appear dead now, as sugar does not in the least excite Drosera. We may safely attribute the reflection of the blades and tentacles of the above leaves to exosmos from the cells which were in contact with the syrup, and their consequent contraction when drops of syrup are placed on the leaves of plants with their roots still in damp earth. No inflection ensues, for the roots no doubt pump up water as quickly as it is lost by exosmos, but if cut-off leaves are immersed in syrup or in any dense fluid, the tentacles are greatly, though irregularly inflected, some of them assuming the shape of corkscrews, and the leaves soon become flaccid. If they are now immersed in a fluid of low specific gravity, the tentacles re-expand. From these page 233 facts, we may conclude that drops of syrup placed on the backs of leaves do not act by exciting a motor impulse, which is transmitted to the tentacles, but that they cause reflection by inducing exosmos. Dr. Nitschke used the secretion for sticking insects to the backs of the leaves, and I suppose that he used a large quantity, which from being dense probably caused exosmos. Perhaps he experimented on cut-off leaves or on plants with their roots not supplied with enough water. As far, therefore, as our present knowledge serves, we may conclude that the glands, together with the immediately underlying cells of the tentacles, are the exclusive seats of that irritability or sensitiveness with which the leaves are endowed. The degree to which a gland is excited can be measured only by the number of the surrounding tentacles which are inflected, and by the amount and rate of their movement equally. Vigorous leaves exposed to the same temperature, and this is an important condition, are excited in different degrees. Under the following circumstances, a minute quantity of a weak solution produces no effect. Add more or give a rather stronger solution, and the tentacles bend, touch a gland once or twice, and no movement follows. Touch it 3 or 4 times, and the tentacle becomes inflected, but the nature of the substance which is given is a very important element. If equal-sized particles of glass, which acts only mechanically of gelatine and raw meat, are placed on the discs of several leaves, the meat causes far more rapid, energetic, and widely extended movement than the 2 former substances. The number of glands which are excited also makes a great difference in the result. Place a bit of meat on 1 or 2 of the discal, page 234 glands, and only a few of the immediately surrounding short tentacles are inflected. Place it on several glands, and many more are acted on. Place it on 30 or 40, and all the tentacles, including the extreme marginal ones, become closely inflected. We thus see that the impulses proceeding from a number of glands strengthen one another, spread farther, and act on a larger number of tentacles than the impulse from any single gland. Transmission of the motor impulse. In every case, the impulse from a gland has to travel for at least a short distance to the basal part of the tentacle, the upper part, and the gland itself being merely carried by the inflection of the lower part. The impulse is thus always transmitted down nearly the whole length of the pedicel. When the central glands are stimulated and the extreme marginal tentacles become inflected, the impulse is transmitted across half the diameter of the disc. And when the glands on one side of the disc are stimulated, the impulse is transmitted across nearly the whole width of the disc. A gland transmits its motor impulse far more easily and quickly down its own tentacle to the bending place than across the disc to neighboring tentacles. Thus a minute dose of a very weak solution of ammonia, if given to one of the glands of the exterior tentacles, causes it to bend and reach the center, whereas a large drop of the same solution given to a score of glands on the disc will not cause, through their combined influence, the least inflection of the exterior tentacles. Again, when a bit of meat is placed on the gland of an exterior tentacle, I have seen movement in 10 seconds and repeatedly within a minute, but a much larger bit placed on several glands on the disc does not cause, page 235, the exterior tentacles to bend until half an hour or even several hours have elapsed. The motor impulse spreads gradually on all sides from one or more excited glands, so that the tentacles which stand nearest are always first affected. Hence, when the glands in the center of the disc are excited, the extreme marginal tentacles are the last inflected, but the glands on different parts of the leaf transmit their motor power in a somewhat different manner. If a bit of meat be placed on the long-headed gland of a marginal tentacle, it quickly transmits an impulse to its own bending portion, but never, as far as I have observed, to the adjoining tentacles, for these are not affected until the meat has been carried to the central glands, which then radiate forth their conjoint impulse on all sides. On 4 occasions leaves were prepared by removing some days previously all the glands from the center, so that these could not be excited by the bits of meat brought to them by the inflection of the marginal tentacles. And now these marginal tentacles re-expanded after a time, without any other tentacle being affected. Other leaves were similarly prepared, and bits of meat were placed on the glands of 2 tentacles in the 3rd row from the outside, and on the glands of 2 tentacles in the 5th row. In these 4 cases, the impulse was sent in the first place laterally, that is, in the same concentric row of tentacles, and then towards the center, but not centrifugally, or towards the exterior tentacles. In one of these cases, only a single tentacle on each side of the one with meat was affected. In the 3 other cases, from half a dozen to a dozen tentacles, both laterally and towards the center, were well inflected or subinflected. Lastly, in page 23610, other experiments, minute bits of meat were placed on a single gland or on 2 glands in the center of the disc, in order that no other glands should touch the meat through the inflection of the closely adjoining short tentacles. About half a dozen glands had been previously removed round the selected ones. On 8 of these leaves, from 16 to 25 of the short surrounding tentacles were inflected in the course of 1 or 2 days, so that the motor impulse radiating from one or two of the discal glands is able to produce this much effect. The tentacles which had been removed are included in the above numbers, for from standing so close they would certainly have been affected on the two remaining leaves. Almost all the short tentacles on the disc were inflected with a more powerful stimulus than meat, namely a little phosphate of lime moistened with saliva. I have seen the inflection spread still farther from a single gland thus treated, but even in this case, the 3 or 4 outer rows of tentacles were not affected. From these experiments it appears that the impulse from a single gland on the disc acts on a greater number of tentacles than that from a gland of one of the exterior elongated tentacles, and this probably follows at least in part from the impulse having to travel a very short distance down the pedicels of the central tentacles, so that it is able to spread to a considerable distance all round. Whilst examining these leaves, I was struck with the fact that in 6, perhaps 7 of them, the tentacles were much more inflected at the distal and proximal ends of the leaf Roman 1e towards the apex and base than on either side, and yet the tentacles on the sides stood as near to the gland where the bit of meat lay as did those at the two ends. It thus appeared, as page 237, if the motor impulse was transmitted from the center across the disc more readily in a longitudinal than in a transverse direction. And as this appeared a new and interesting fact in the physiology of plants, 35 fresh experiments were made to test its truth. Bits of meat were placed on a single gland or on a few glands on the right or left side of the discs of 18 leaves, other bits of the same size being placed on the distal or proximal ends of 17 other leaves. Now, if the motor impulse were transmitted with equal force or at an equal rate through the blade in all directions, a bit of meat placed at one side or at one end of the disc ought to affect equally all the tentacles situated at an equal distance from it. But this certainly is not the case. Before giving the general results, it may be well to describe 3 or 4 rather unusual cases. 1. A minute fragment of a fly was placed on one side of the disc, and after 32 Roman 1007 of the outer tentacles near the fragment were inflected. After 10 HRS, several more became so, and after 23 HRS, a still greater number, and now the blade of the leaf on this side was bent inwards, so as to stand up at right angles to the other side. Neither the blade of the leaf nor a single tentacle on the opposite side was affected. The line of separation between the two halves extending from the footstalk to the apex. The leaf remained in this state for 3 days, and on the 4th day began to re-expand, not a single tentacle having been inflected on the opposite side. Now for the general results of the 18 leaves on which bits of meat were placed on the right or left sides of the disc. 8 had a vast number of tentacles inflected on the same side, and in 4 of them the blade itself on this side was likewise inflected, whereas not a single tentacle nor the blade was affected on the opposite side. These leaves presented a very curious appearance, as if only the inflected side was active and the other paralyzed. In the remaining 10 cases, a few tentacles became inflected beyond the medial line on the side opposite to that where the meat lay, but in some of these cases only at the proximal or distal ends of the leaves. The inflection on the opposite side always occurred considerably after that on the same side, and in one instance not until the 4th day. We have also seen page 239, with no 5, that bits of meat had to be added thrice before all the short tentacles on the opposite side of the disc were inflected. The result was widely different when bits of meat were placed in a medial line at the distal or proximal ends of the disc. In 3 of the 17 experiments thus made, owing either to the state of the leaf or to the smallness of the bit of meat, only the immediately adjoining tentacles were affected, but in the other 14 cases, the tentacles at the opposite end of the leaf were inflected, though these were as distant from where the meat lay as were those on one side of the disc from the meat on the opposite side. In some of the present cases, the tentacles on the sides were not at all affected, or in a less degree, or after a longer interval of time than those at the opposite end. One set of experiments is worth giving in fuller detail. Cubes of meat, not quite so small as those usually employed, were placed on one side of the discs of 4 leaves, and cubes of the same size at the proximal or distal end of 4 other leaves. Now, when these 2 sets of leaves were compared after an interval of 24 HRS, they presented a striking difference. Those having the cubes on one side were very slightly affected on the opposite side, whereas those with the cubes at either end had almost every tentacle at the opposite end, even the marginal ones closely inflected, after 48 HRS. The contrast in the state of the two sets was still great, yet those with the meat on one side now had their discal and submarginal tentacles on the opposite side, somewhat inflected, this being due to the large size of the cubes. Finally, we may conclude from these 35 experiments, not to mention the 6 or 7 previous ones, that the motor impulse is transmitted from any single gland, page 240, or small group of glands through the blade, to the other tentacles more readily and effectually in a longitudinal than in a transverse direction. As long as the glands remain excited, and this may last for many days, even for 11, as when in contact with phosphate of lime, they continue to transmit a motor impulse to the basal and bending parts of their own pedicels, for otherwise they would re-expand, the great difference in the length of time during which tentacles remain inflected over inorganic objects and over objects of the same size containing soluble nitrogenous matter proves the same fact, but the intensity of the impulse transmitted from an excited gland, which has begun to pour forth its acid secretion, and is at the same time absorbing, seems to be very small compared with that which it transmits when first excited. Thus, when moderately large bits of meat were placed on one side of the disc, and the discal and submarginal tentacles on the opposite side became inflected, so that their glands at last touched the meat and absorbed matter from it, they did not transmit any motor influence to the exterior rows of tentacles on the same side, for these never became inflected. If, however, meat had been placed on the glands of these same tentacles before they had begun to secrete copiously and to absorb, they undoubtedly would have affected the exterior rows. Nevertheless, when I gave some phosphate of lime, which is a most powerful stimulant to several submarginal tentacles already considerably inflected, but not yet in contact with some phosphate previously placed on two glands in the center of the disc, the exterior tentacles on the same side were acted on. When a gland is first excited, the motor impulse is discharged within a few seconds, as we know from the page 241 bending of the tentacle, and it appears to be discharged at first with much greater force than afterwards. Thus, in the case above given of a small fly naturally caught by a few glands on one side of a leaf, an impulse was slowly transmitted from them across the whole breadth of the leaf, causing the opposite tentacles to be temporarily inflected, but the glands which remained in contact with the insect, though they continued for several days to send an impulse down their own pedicels to the bending place, did not prevent the tentacles on the opposite side from quickly re-expanding, so that the motor discharge must at first have been more powerful than afterwards. When an object of any kind is placed on the disc, and the surrounding tentacles are inflected, their glands secrete more copiously, and the secretion becomes acid, so that some influence is sent to them from the discal glands. This change in the nature and amount of the secretion cannot depend on the bending of the tentacles, as the glands of the short central tentacles secrete acid when an object is placed on them, though they do not themselves bend. Therefore, I inferred that the glands of the disc sent some influence up the surrounding tentacles to their glands, and that these reflected back a motor impulse to their basal parts. But this view was soon proved erroneous. It was found by many trials that tentacles with their glands closely cut off by sharp scissors often become inflected, and again re-expand, still appearing healthy. One, which was observed, continued healthy for 10 days after the operation. I therefore cut the glands off 25 tentacles at different times, and on different leaves, and 17 of these soon became inflected, and afterwards re-expanded. The re-expansion commenced in about page 242 8 HRS or 9 HRS, and was completed in from 22 HRS to 30 HRS from the time of inflection. After an interval of a day or two, raw meat with saliva was placed on the discs of these 17 leaves. When observed next day, 7 of the headless tentacles were inflected over the meat as closely as the uninjured ones on the same leaves, and an 8th headless tentacle became inflected after 3 additional days. The meat was removed from one of these leaves, and the surface washed with a little stream of water, and after 3 days the headless tentacle re-expanded for the second time. These tentacles without glands were, however, in a different state from those provided with glands, and which had absorbed matter from the meat. For the protoplasm within the cells of the former had undergone far less aggregation from these experiments with headless tentacles. It is certain that the glands do not, as far as the motor impulse is concerned, act in a reflex manner like the nerve ganglia of animals. But there is another action, namely that of aggregation, which in certain cases may be called reflex, and it is the only known instance in the vegetable kingdom. We should bear in mind that the process does not depend on the previous bending of the tentacles, as we clearly see when leaves are immersed in certain strong solutions, nor does it depend on increased secretion from the glands, and this is shown by several facts, more especially by the papillae, which do not secrete yet undergoing aggregation if given carbonate of ammonia or an infusion of raw meat. When a gland is directly stimulated in any way, as by the pressure of a minute particle of glass, the protoplasm within the cells of the gland first becomes aggregated, then that in the cells immediately beneath the gland, and so lower and lower down the tentacles to their bases, page 243. That is, if the stimulus has been sufficient and not injurious. Now, when the glands of the disc are excited, the exterior tentacles are affected in exactly the same manner. The aggregation always commences in their glands, though these have not been directly excited, but have only received some influence from the disc, as shown by their increased acid secretion. The protoplasm within the cells immediately beneath the glands are next affected, and so downwards from cell to the bases of the tentacles. This process apparently deserves to be called a reflex action. In the same manner as when a sensory nerve is irritated and carries an impression to a ganglion, which sends back some influence to a muscle or gland, causing movement or increased secretion. But the action in the two cases is probably of a widely different nature after the protoplasm in a tentacle has been aggregated. Its redissolution always begins in the lower part and slowly travels up the pedicel to the gland, so that the protoplasm last aggregated is first redissolved. This probably depends merely on the protoplasm being less and less aggregated lower and lower down in the tentacles, as can be seen plainly when the excitement has been slight. As soon, therefore, as the aggregating action altogether ceases, redissolution naturally commences in the less strongly aggregated matter in the lowest part of the tentacle, and is there first completed. Direction of the inflected tentacles: when a particle of any kind is placed on the gland of one of the outer tentacles, this invariably moves towards the center of the leaf, and so it is with all the tentacles of a leaf immersed in any exciting fluid. The glands of the exterior tentacles then form a ring round the middle part of the disc, as shown in a previous figure, Fig. 4, page 244 p10. The short tentacles within this ring still retain their vertical position, as they likewise do when a large object is placed on their glands, or when an insect is caught by them. In this latter case, we can see that the inflection of the short central tentacles would be useless, as their glands are already in contact with their prey. Fig. 10, Drosera rotundifolia, leaf enlarged, with the tentacles inflected over a bit of meat placed on one side of the disc. The result is very different when a single gland on one side of the disc is excited, or a few in a group. These send an impulse to the surrounding tentacles, which do not now bend towards the center of the leaf, but to the point of excitement. We owe this capital observation to Nitschke, and since reading his paper a few years ago, I have repeatedly verified it. If a minute bit of meat be placed by the aid of a needle on a single gland, or on 3 or 4 together, halfway between the center and the circumference of the disc, the directed movement of the surrounding tentacles is well exhibited. An accurate drawing of a leaf with meat in this position is here reproduced, Fig. 10, and we see the tentacles, including some of the exterior ones, accurately directed to the point where the meat lay, but a much better. Bot-Zeitung, 1860p, 240, page 245. Plan is to place a particle of the phosphate of lime, moistened with saliva, on a single gland on one side of the disc of a large leaf, and another particle on a single gland on the opposite side. In 4 such trials, the excitement was not sufficient to affect the outer tentacles, but all those near the 2 points were directed to them. so that two wheels were formed on the disc of the same leaf, the pedicels of the tentacles forming the spokes, and the glands united. Mass over the phosphate representing the axils, the precision with which each tentacle pointed to the particle was wonderful, so that in some cases I could detect no deviation from perfect accuracy. Thus, although the short tentacles in the middle of the disc do not bend when their glands are excited in a direct manner, yet if they receive a motor impulse from a point on one side, they direct themselves to the point equally well with the tentacles on the borders of the disc. In these experiments, some of the short tentacles on the disc, which would have been directed to the center, had the leaf been immersed in an exciting fluid, were now inflected in an exactly opposite direction, viz. Towards the circumference. These tentacles, therefore, had deviated as much as 180° from the direction which they would have assumed if their own glands had been stimulated, and which may be considered as the normal one between this, the greatest possible, and no deviation from the normal direction. Every degree could be observed in the tentacles on these several leaves, notwithstanding the precision with which the tentacles generally were directed. Those near the circumference of one leaf were not accurately directed towards some phosphate of lime at a rather distant point on the opposite side of the disc. It appeared as if the motor page 246 impulse, in passing transversely across nearly the whole width of the disc, had departed somewhat from a true course. This accords with what we have already seen of the impulse traveling less readily in a transverse than in a longitudinal direction. In some other cases, the exterior tentacles did not seem capable of such accurate movement as the shorter and more central ones. Nothing could be more striking than the appearance of the above four leaves, each with their tentacles pointing truly to the two little masses of the phosphate on their discs. We might imagine that we were looking at a lowly organized animal seizing prey with its arms in the case of Drosera. The explanation of this accurate power of movement no doubt lies in the motor impulse radiating in all directions, and whichever side of a tentacle it first strikes, that side contracts, and the tentacle consequently bends towards the point of excitement. The pedicels of the tentacles are flattened or elliptic in section near the bases of the short central tentacles. The flattened or broad face is formed of about 5 longitudinal rows of cells in the outer tentacles of the disc. It consists of about 6 or 7 rows, and in the extreme marginal tentacles of above a dozen rows, as the flattened bases are thus formed of only a few rows of cells. The precision of the movements of the tentacles is the more remarkable, for when the motor impulse strikes the base of a tentacle in a very oblique direction relatively to its broad face, scarcely more than 1 or 2 cells towards one end can be effected at first, and the contraction of these cells must draw the whole tentacle into the proper direction. It is perhaps owing to the exterior pedicels being much flattened that they do not bend quite so accurately to the point of excitement as the page 247 more central ones. The properly directed movement of the tentacles is not an unique case in the vegetable kingdom, for the tendrils of many plants curve towards the side which is touched, but the case of Drosera is far more interesting, as here the tentacles are not directly excited, but receive an impulse from a distant point. Nevertheless, they bend accurately towards this point. Figure 11, Drosera rotundifolia diagram, showing the distribution of the vascular tissue in a small leaf. On the nature of the tissues through which the motor impulse is transmitted, it will be necessary first to describe briefly the course of the main fibrovascular bundles. These are shown in the accompanying sketch. Fig. 11 of a small leaf. Little vessels from the neighboring bundles enter all the many tentacles with which the surface is studded. But these are not here represented. The central trunk, which runs up the footstalk, bifurcates near the center of the leaf, each branch bifurcating. And again, according to the size of the leaf, this central trunk sends off low down on each side a delicate branch, which may be called the sublateral branch. There is also on each side a main lateral branch or bundle which bifurcates in the same manner as the others. Bifurcation does not imply that any single vessel divides, but that a bundle, page 248, divides into two by looking to either side of the leaf. It will be seen that a branch from the great central bifurcation inosculates with a branch from the lateral bundle, and that there is a smaller inosculation between the two chief branches of the lateral bundle. The course of the vessels is very complex at the larger inosculation, and here vessels retaining the same diameter are often formed by the union of the bluntly pointed ends of two vessels. But whether these points open into each other by their attached surfaces, I do not know. By means of the two inosculations, all the vessels on the same side of the leaf are brought into some sort of connection near the circumference of the larger leaves. The bifurcating branches also come into close union, and then separate again, forming a continuous zigzag line of vessels round the whole circumference. But the union of the vessels in this zigzag line seems to be much less intimate than at the main inosculation. It should be added that the course of the vessels differs somewhat in different leaves, and even on opposite sides of the same leaf, but the main inosculation is always present. Now, in my first experiments with bits of meat placed on one side of the disc, it so happened that not a single tentacle was inflected on the opposite side, and when I saw that the vessels on the same side were all connected together by the two inosculations, whilst not a vessel passed over to the opposite side, it seemed probable that the motor impulse was conducted exclusively along them. In order to test this view, I divided transversely with the point of a lancet the central trunks of 4 leaves just beneath the main bifurcation, and 2 days afterwards placed rather large bits of raw meat, page 249, a most powerful stimulant near the center of the disc, above the incision that is a little towards the apex, with the following results. 1. This leaf proved rather torpid after 4 HRS 40 Roman 1000, in all cases reckoning from the time when the meat was given. The tentacles at the distal end were a little inflected, but nowhere else. They remained so for 3 days, and re-expanded on the 4th day. The leaf was then dissected, and the trunk, as well as the 2 sublateral branches, were found divided. The appearance presented by the leaves 2 and 3 was very curious, and might be aptly compared with that of a man with his backbone broken and lower extremities paralyzed, excepting that the line between the 2 halves was here transverse instead of longitudinal. These leaves were in the same state as some of those in the former experiments, with bits of meat placed on one side of the disc, the case of leaf 4. Roman 1000. Ziegler made similar experiments by cutting the spiral vessels of Drosera intermediacamps rendus 1874 p. 1417, but arrived at conclusions widely different from mine, page 250. Proves that the spiral vessels of the central trunk may be divided, and yet the motor impulse be transmitted from the distal to the basal end, and this led me at first to suppose that the motor force was sent through the closely surrounding fibrous tissue, and that if one half of this tissue was left undivided, it sufficed for complete transmission. But opposed to this conclusion is the fact that no vessels pass directly from one side of the leaf to the other, and yet, as we have seen, if a rather large bit of meat is placed on one side, the motor impulse is sent, though slowly and imperfectly, in a transverse direction across the whole breadth of the leaf. Nor can this latter fact be accounted for by supposing that the transmission is effected through the 2 inosculations or through the circumferential zigzag line of union, for had this been the case, the exterior tentacles on the opposite side of the disc would have been affected before the more central ones, which never occurred. We have also seen that the extreme marginal tentacles appear to have no power to transmit an impulse to the adjoining tentacles, yet the little bundle of vessels which enters each marginal tentacle sends off a minute branch to those on both sides, and this I have not observed in any other tentacles, so that the marginal ones are more closely connected together by spiral vessels than are the others, and yet have much less power of communicating a motor impulse to one another. But besides these several facts and arguments, we have conclusive evidence that the motor impulse is not sent at least exclusively through the spiral vessels, or through the tissue immediately surrounding them. We know that if a bit of meat is placed on a gland, the immediately adjoining ones having been removed on any part of the disc, all the short page 251 rounding tentacles bend almost simultaneously with great precision towards it. Now there are tentacles on the disc, for instance near the extremities of the sublateral bundles, Fig. 11, which are supplied with vessels that do not come into contact with the branches that enter the surrounding tentacles, except by a very long and extremely circuitous course. Nevertheless, if a bit of meat is placed on the gland of a tentacle of this kind, all the surrounding ones are inflected towards it with great precision. It is of course possible that an impulse might be sent through a long and circuitous course, but it is obviously impossible that the direction of the movement could be thus communicated, so that all the surrounding tentacles should bend precisely to the point of excitement. The impulse, no doubt, is transmitted in straight radiating lines from the excited gland to the surrounding tentacles. It cannot therefore be sent along the fibrovascular bundles, the effect of cutting the central vessels in the above cases in preventing the transmission of the motor impulse from the distal to the basal end of a leaf may be attributed to a considerable space of the cellular tissue having been divided. We shall hereafter see, when we treat of Dionea, that this same conclusion, namely, that the motor impulse is not transmitted by the fibrovascular bundles, is plainly confirmed. And Prof. Cohn has come to the same conclusion with respect to Aldra van der Both, members of the Droz race. As the motor impulse is not transmitted along the vessels, there remains for its passage only the cellular tissue, and the structure of this tissue explains to a certain extent how it travels so quickly down the long exterior tentacles and much more slowly across the blade of the leaf. We shall also see why it crosses page 252, the blade more quickly in a longitudinal than in a transverse direction, though with time it can pass in any direction. We know that the same stimulus causes movement of the tentacles and aggregation of the protoplasm, and that both influences originate in and proceed from the glands within the same brief space of time. It seems therefore probable that the motor impulse consists of the first commencement of a molecular change in the protoplasm, which when well developed is plainly visible, and has been designated aggregation. But to this subject I shall return. We further know that in the transmission of the aggregating process the chief delay is caused by the passage of the transverse cell walls, for as the aggregation travels down the tentacles, the contents of each successive cell seem almost to flash into a cloudy mass. We may therefore infer that the motor impulse is in like manner delayed chiefly by passing through the cell walls. The greater celerity with which the impulse is transmitted down the long exterior tentacles than across the disc may be largely attributed to its being closely confined within the narrow pedicel, instead of radiating forth on all sides as on the disc. But besides this confinement, the exterior cells of the tentacles are fully twice as long as those of the disc, so that only half the number of transverse partitions have to be traversed in a given length of a tentacle, compared with an equal space on the disc, and there would be in the same proportion less retardation of the impulse. Moreover, in sections of the exterior tentacles given by Dr. Warming, the parenchymatous Wetenskapelig Metallulcer de la Soche, die Hist Nat de Copenhagen Nose, 10-12-1872, woodcuts Roman 4 and Roman 5, page 253. Cells are shown to be still more elongated, and these would form the most direct line of communication from the gland to the bending place of the tentacle. If the impulse travels down the exterior cells, it would have to cross from between 20 to 30 transverse partitions, but rather fewer if down the inner parenchymatous tissue. In either case, it is remarkable that the impulse is able to pass through so many partitions down nearly the whole length of the pedicel, and to act on the bending place in 10 seconds, why the impulse, after having passed so quickly down one of the extreme marginal tentacles, about 120 of an inch in length, should never, as far as I have seen, affect the adjoining tentacles. I do not understand it, may be in part accounted for by much energy being expended in the rapidity of the transmission. Most of the cells of the disc, both the superficial ones and the larger cells which form the 5 or 6 underlying layers, are about 4 times as long as broad. They are arranged almost longitudinally, radiating from the footstock. The motor impulse, therefore, when transmitted across the disc, has to cross nearly 4 times as many cell walls as when transmitted in a longitudinal direction, and would consequently be much delayed in the former case. The cells of the disc converge towards the bases of the tentacles, and are thus fitted to convey the motor impulse to them from all sides. On the whole, the arrangement and shape of the cells, both those of the disc and tentacles, throw much light on the rate and manner of diffusion of the motor impulse. But why the impulse proceeding from the glands of the exterior rows of tentacles tends to travel laterally and towards the center of the leaf, but not centrifugally, is by no means clear. Page 254. Mechanism of the movements and nature of the motor impulse. Whatever may be the means of movement, the exterior tentacles, considering their delicacy, are inflected with much force, a bristle held, so that a length of 1 inch projected from a handle yielded when I tried to lift with it an inflected tentacle, which was somewhat thinner than the bristle. The amount or extent also of the movement is great fully expanded tentacles in becoming inflected sweep through an angle of 180°, and if they are beforehand reflexed, as often occurs, the angle is considerably greater. It is probably the superficial cells at the bending place, which chiefly or exclusively contract, for the interior cells have very delicate walls, and are so few in number that they could hardly cause a tentacle to bend with precision to a definite point. Though I carefully looked, I could never detect any wrinkling of the surface at the bending place, even in the case of a tentacle abnormally curved into a complete circle under circumstances hereafter to be mentioned. All the cells are not acted on, though the motor impulse passes through them. When the gland of one of the long exterior tentacles is excited, the upper cells are not in the least affected. About halfway down, there is a slight bending, but the chief movement is confined to a short space near the base, and no part of the inner tentacles bends, except the basal portion. With respect to the blade of the leaf, the motor impulse may be transmitted through many cells from the center to the circumference without their being in the least affected, or they may be strongly acted on, and the blade greatly inflected. In the latter case, the movement seems to depend partly on the strength of the stimulus, and partly on page 255, its nature, as when leaves are immersed in certain fluids. The power of movement, which various plants possess when irritated, has been attributed by high authorities to the rapid passage of fluid out of certain cells, which from their previous state of tension immediately contract. Whether or not this is the primary cause of such movements, fluid must pass out of closed cells when they contract or are pressed together in one direction, unless they at the same time expand in some other direction. For instance, fluid can be seen to ooze from the surface of any young and vigorous shoot. If slowly bent into a semicircle in the case of Drosera, there is certainly much movement of the fluid throughout the tentacles whilst they are undergoing inflection. Many leaves can be found in which the purple fluid within the cells is of an equally dark tint on the upper and lower sides of the tentacles, extending also downwards on both sides to equally near their bases. If the tentacles of such a leaf are excited into movement, it will generally be found after some hours that the cells on the concave side are much paler than they were before, or are quite colorless, those on the convex side having become much darker in 2 instances after particles of hair had been placed on glands, and when in the course of 1 Roman 1000 the tentacles were incurved halfway towards the center of the leaf. This change of color in the 2 sides was conspicuously plain in another case. After a bit of meat had been placed on a gland, the purple color was observed at intervals to be slowly traveling from the upper to the lower part down the convex side of Sachs Trait de Bot 3rd edit 1874, p. 1038. This view was, I believe, first suggested by Lamarck. Sachs Ibid p. 919, page 256. The bending tentacle, but it does not follow from these observations that the cells on the convex side become filled with more fluid during the act of inflection than they contained before, for fluid may all the time be passing into the disc or into the glands, which then secrete freely. The bending of the tentacles when leaves are immersed in a dense fluid, and their subsequent re-expansion in a less dense fluid, show that the passage of fluid from or into the cells can cause movements like the natural ones, but the inflection thus caused is often irregular, the exterior tentacles being sometimes spirally curved. Other unnatural movements are likewise caused by the application of dense fluids, as in the case of drops of syrup placed on the backs of leaves and tentacles. Such movements may be compared with the contortions which many vegetable tissues undergo when subjected to exosmose. It is therefore doubtful whether they throw any light on the natural movements. If we admit that the outward passage of fluid is the cause of the bending of the tentacles, we must suppose that the cells before the act of inflection are in a high state of tension, and that they are elastic to an extraordinary degree, for otherwise their contraction could not cause the tentacles often to sweep through an angle of above 180°. Prof. Cohn, in his interesting paper on the movements of the stamens of certain compositae, states that these organs, when dead, are as elastic as threads of india rubber, and are then only half as long as they were when alive. He believes that the living protoplasm. Abhan der Schles Gessel, Fr. Vattel Coulter, 1861 Heft Roman 1. An excellent abstract of this paper is given in the Annals and Mag of Nat Hist 3rd Series, 1863, Vol Roman 11 pp. 180, 8197, page 257. Within their cells is ordinarily in a state of expansion, but is paralyzed by irritation or may be said to suffer temporary death, the elasticity of the cell walls then coming into play and causing the contraction of the stamens. Now the cells on the upper or concave side of the bending part of the tentacles of Drosera do not appear to be in a state of tension nor to be highly elastic, for when a leaf is suddenly killed or dies slowly, it is not the upper, but the lower sides of the tentacles which contract from elasticity. We may therefore conclude that their movements cannot be accounted for by the inherent elasticity of certain cells opposed as long as they are alive, and not irritated by the expanded state of their contents. A somewhat different view has been advanced by other physiologists, namely, that the protoplasm, when irritated, contracts like the soft sarcode of the muscles of animals in Drosera. The fluid within the cells of the tentacles at the bending place appears under the microscope thin and homogeneous, and after aggregation consists of small soft masses of matter undergoing incessant changes of form and floating in almost colorless fluid. These masses are completely redissolved when the tentacles re-expand. Now, it seems scarcely possible that such matter should have any direct mechanical power, but if through some molecular change it were to occupy less space than it did before, no doubt the cell walls would close up and contract, but in this case it might be expected that the walls would exhibit wrinkles, and none could ever be seen. Moreover, the contents of all the cells seem to be of exactly the same nature both before and after aggregation, and yet only a few of the basal cells contract, the rest of the tentacle remaining straight. A third view maintained by some physiologists, page 258, though rejected by most others, is that the whole cell, including the walls, actively contracts if the walls are composed solely of non-nitrogenous cellulose. This view is highly improbable, but it can hardly be doubted that they must be permeated by protid matter at least whilst they are growing, nor does there seem any inherent improbability in the cell walls of Drosera contracting, considering their high state of organization, as shown in the case of the glands, by their power of absorption and secretion, and by being exquisitely sensitive so as to be affected by the pressure of the most minute particles. The cell walls of the petacells also allow various impulses to pass through them, inducing movement, increased secretion, and aggregation. On the whole, the belief that the walls of certain cells contract some of their contained fluid being at the same time forced outwards perhaps accords best with the observed facts. If this view is rejected, the next most probable one is that the fluid contents of the cells shrink owing to a change in their molecular state, with the consequent closing in of the walls. Anyhow, the movement can hardly be attributed to the elasticity of the walls together with a previous state of tension. With respect to the nature of the motor impulse, which is transmitted from the glands down the petacells and across the disc, it seems not improbable that it is closely allied to that influence which causes the protoplasm within the cells of the glands and tentacles to aggregate. We have seen that both forces originate in and proceed from the glands within a few seconds of the same time, and are excited by the same causes. The aggregation of the protoplasm lasts almost as long as the tentacles remain inflected, even though this be for more than a week. But the page 259 protoplasm is redissolved at the bending place shortly before the tentacles re-expand, showing that the exciting cause of the aggregating process has then quite ceased. Exposure to carbonic acid causes both the latter process and the motor impulse to travel very slowly down the tentacles. We know that the aggregating process is delayed in passing through the cell walls, and we have good reason to believe that this holds good with the motor impulse, for we can thus understand the different rates of its transmission in a longitudinal and transverse line across the disc, under a high power, the first sign of aggregation is the appearance of a cloud and soon afterwards of extremely fine granules in the homogeneous purple fluid within the cells, and this apparently is due to the union of molecules of protoplasm. Now, it does not seem an improbable view that the same tendency, namely, for the molecules to approach each other, should be communicated to the inner surfaces of the cell walls, which are in contact with the protoplasm. And if so, their molecules would approach each other, and the cell wall would contract. To this view, it may with truth be objected that when leaves are immersed in various strong solutions, or are subjected to a heat of above 134–544, aggregation ensues, but there is no movement again. Various acids and some other fluids cause rapid movement, but no aggregation, or only of an abnormal nature, or only after a long interval of time. But as most of these fluids are more or less injurious, they may check or prevent the aggregating process by injuring or killing the protoplasm. There is another and more important difference in the two processes. When the glands on the disc are excited, they transmit some influence up the surrounding page 260 tentacles, which acts on the cells at the bending place, but does not induce aggregation until it has reached the glands. These then send back some other influence, causing the protoplasm to aggregate first in the upper and then in the lower cells. The re-expansion of the tentacles. This movement is always slow and gradual. When the center of the leaf is excited, or a leaf is immersed in a proper solution, all the tentacles bend directly towards the center and afterwards directly back from it. But when the point of excitement is on one side of the disc, the surrounding tentacles bend towards it, and therefore obliquely with respect to their normal direction. When they afterwards re-expand, they bend obliquely back so as to recover their original positions. The tentacles farthest from an excited point, wherever that may be, are the last and the least affected, and probably in consequence of this, they are the first to re-expand. The bent portion of a closely inflected tentacle is in a state of active contraction, as shown by the following experiment. Meat was placed on a leaf, and after the tentacles were closely inflected and had quite ceased to move, narrow strips of the disc, with a few of the outer tentacles attached to it, were cut off and laid on one side under the microscope. After several failures, I succeeded in cutting off the convex surface of the bent portion. Tentacle movement immediately recommenced, and the already greatly bent portion went on bending until it formed a perfect circle, the straight distal portion of the tentacle passing on one side of the strip. The convex surface must therefore have previously been in a state of tension sufficient to counterbalance that of the concave surface, which when free curled into a complete ring. The tentacles of an expanded and unexcited leaf, page 261, are moderately rigid and elastic. If bent by a needle, the upper end yields more easily than the basal and thicker part, which alone is capable of becoming inflected. The rigidity of this basal part seems due to the tension of the outer surface balancing a state of active and persistent contraction of the cells of the inner surface. I believe that this is the case because when a leaf is dipped into boiling water, the tentacles suddenly become reflexed, and this apparently indicates that the tension of the outer surface is mechanical, whilst that of the inner surface is vital and is instantly destroyed by the boiling water. We can thus also understand why the tentacles, as they grow old and feeble, slowly become much reflexed. If a leaf with its tentacles closely inflected is dipped into boiling water, these rise up a little, but by no means fully re-expand. This may be owing to the heat quickly destroying the tension and elasticity of the cells of the convex surface, but I can hardly believe that their tension at any one time would suffice to carry back the tentacles to their original position, often through an angle of above 180°. It is more probable that fluid, which we know travels along the tentacles during the act of inflection, is slowly reattracted into the cells of the convex surface, their tension being thus gradually and continually increased. A recapitulation of the chief facts and discussions in this chapter will be given at the close of the next chapter, page 262. Chapter Roman 11, Recapitulation of the chief observations on Drosera rotundifolia. As summaries have been given to most of the chapters, it will be sufficient here to recapitulate as briefly as I can the chief points in the first chapter, a preliminary sketch was given of the structure of the leaves and of the manner in which they capture insects. This is affected by drops of extremely viscid fluid surrounding the glands and by the inward movement of the tentacles as the plants gain most of their nutriment. By this means, their roots are very poorly developed, and they often grow in places where hardly any other plant except mosses can exist. The glands have the power of absorption besides that of secretion. They are extremely sensitive to various stimulants, namely repeated touches, the pressure of minute particles, the absorption of animal matter, and of various fluids, heat, and galvanic action. A tentacle with a bit of raw meat on the gland has been seen to begin bending in 10s, to be strongly incurved in 5 Roman 1000, and to reach the center of the leaf in half an hour. The blade of the leaf often becomes so much inflected that it forms a cup enclosing any object placed on it. A gland, when excited, not only sends some influence down its own tentacle, causing it to bend, but likewise to the surrounding tentacles, which become incurved, so that the bending place can be acted on by an impulse received from opposite directions. Page 263, namely from the gland on the summit of the same tentacle, and from one or more glands of the neighboring tentacles. Tentacles, when inflected, re-expand after a time, and during this process, the glands secrete less copiously or become dry. As soon as they begin to secrete again, the tentacles are ready to react, and this may be repeated at least 3, probably many more times. It was shown in the second chapter that animal substances placed on the discs cause much more prompt and energetic inflection than do inorganic bodies of the same size or mere mechanical irritation. But there is a still more marked difference in the greater length of time during which the tentacles remain inflected over bodies yielding soluble and nutritious matter than over those which do not yield such matter. Extremely minute particles of glass cinders, hair thread, precipitated chalk, Roman 100, when placed on the glands of the outer tentacles, cause them to bend. A particle, unless it sinks through the secretion and actually touches the surface of the gland with some one point, does not produce any effect. A little bit of thin human hair, 81,000 of an inch, 203 Roman 2000 in length, and weighing only 178,740 of a grain, 822 mg, though largely supported by the dense secretion, suffices to induce movement. It is not probable that the pressure in this case could have amounted to that from the millionth of a grain. Even smaller particles cause a slight movement, as could be seen through a lens. Larger particles than those of which the measurements have been given cause no sensation when placed on the tongue, one of the most sensitive parts of the human body. Movement ensues if a gland is momentarily touched 3 or 4 times, but if touched only once or twice, page 264, though with considerable force, and with a hard object, the tentacle does not bend. The plant is thus saved from much useless movement, as during a high wind, the glands can hardly escape being occasionally brushed by the leaves of surrounding plants. Though insensible to a single touch, they are exquisitely sensitive, as just stated, to the slightest pressure, if prolonged for a few seconds, and this capacity is manifestly of service to the plant in capturing small insects. Even gnats, if they rest on the glands with their delicate feet, are quickly and securely embraced. The glands are insensible to the weight and repeated blows of drops of heavy rain, and the plants are thus likewise saved from much useless movement. The description of the movements of the tentacles was interrupted in the 3rd chapter for the sake of describing the process of aggregation. This process always commences in the cells of the glands, the contents of which first become cloudy, and this has been observed within 10s. After a gland has been excited, granules, just resolvable under a very high power, soon appear, sometimes within a minute, in the cells beneath the glands, and these then aggregate into minute spheres. The process afterwards travels down the tentacles, being arrested for a short time at each transverse partition. The small spheres coalesce into larger spheres, or into oval club-headed thread or necklace-like or otherwise shaped masses of protoplasm, which, suspended in almost colorless fluid, exhibit incessant spontaneous changes of form. These frequently coalesce and again separate. If a gland has been powerfully excited, all the cells down to the base of the tentacle are affected. In cells, especially if filled with dark red fluid, the first step in the page 265 process often is the formation of a dark red bag-like mass of protoplasm, which afterwards divides and undergoes the usual repeated changes of form before any aggregation has been excited. A sheet of colorless protoplasm, including granules, the primordial utricle of mole, flows round the walls of the cells, and this becomes more distinct after the contents have been partially aggregated into spheres or bag-like masses. But after a time, the granules are drawn towards the central masses and unite with them, and then the circulating sheet can no longer be distinguished, but there is still a current of transparent fluid within the cells. Aggregation is excited by almost all the stimulants, which induce movement, such as the glands being touched 2 or 3 times, the pressure of minute inorganic particles, the absorption of various fluids, even long immersion in distilled water, exosmosis, and heat of the many stimulants. Tried carbonate of ammonia is the most energetic and acts the quickest. A dose of 1,134,400 of a grain 48 mg given to a single gland suffices to cause in 1 hour well-marked aggregation in the upper cells of the tentacle. The process goes on only as long as the protoplasm is in a living, vigorous, and oxygenated condition. The result is in all respects exactly the same whether a gland has been excited directly or has received an influence from other and distant glands, but there is one important difference. When the central glands are irritated, they transmit centrifugally an influence up the pedicels of the exterior tentacles to their glands, but the actual process of aggregation travels centripetally from the glands of the exterior tentacles down their pedicels. The exciting influence, therefore, which is transmitted from page 266, one part of the leaf to another, must be different from that which actually induces aggregation. The process does not depend on the glands secreting more copiously than they did before, and is independent of the inflection of the tentacles. It continues as long as the tentacles remain inflected, and as soon as these are fully re-expanded, the little masses of protoplasm are all redissolved, the cells becoming filled with homogeneous purple fluid, as they were before the leaf was excited. As the process of aggregation can be excited by a few touches or by the pressure of insoluble particles, it is evidently independent of the absorption of any matter, and must be of a molecular nature even when caused by the absorption of the carbonate or other salts of ammonia or an infusion of meat. The process seems to be of exactly the same nature. The protoplasmic fluid must therefore be in a singularly unstable condition to be acted on by such slight and varied causes. Physiologists believe that when a nerve is touched and it transmits an influence to other parts of the nervous system, a molecular change is induced in it, though not visible to us. Therefore, it is a very interesting spectacle to watch the effects on the cells of a gland of the pressure of a bit of hair weighing only 178,700 of grain and largely supported by the dense secretion. For this excessively slight pressure soon causes a visible change in the protoplasm, which change is transmitted down the whole length of the tentacle, giving it at last a mottled appearance distinguishable even by the naked eye. In the 4th chapter, it was shown that leaves placed for a short time in water at a temperature of 110°F become somewhat inflected. They are thus also rendered more sensitive to the action, page 267, of meat than they were before if exposed to a temperature of between 115 and 125,461,516. They are quickly inflected, and their protoplasm undergoes aggregation when afterwards placed in cold water. They re-expand. Exposed to 130,544 cent, no inflection immediately occurs, but the leaves are only temporarily paralyzed, for on being left in cold water, they often become inflected and afterwards re-expand in one leaf. Thus treated, I distinctly saw the protoplasm in movement in other leaves treated in the same manner, and then immersed in a solution of carbonate of ammonia. Strong aggregation ensued. Leaves placed in cold water after an exposure to so high a temperature as 145,627 cent sometimes become slightly, though slowly, inflected, and afterwards have the contents of their cells strongly aggregated by carbonate of ammonia. But the duration of the immersion is an important element, for if left in water at 145/627 cent, or only at 140/60 cent. Until it becomes cool, they are killed, and the contents of the glands are rendered white and opaque. This latter result seems to be due to the coagulation of the albumin, and was almost always caused by even a short exposure to 150/65 cent, but different leaves, and even the separate cells in the same tentacle differ considerably in their power of resisting heat. Unless the heat has been sufficient to coagulate, the albumin carbonate of ammonia subsequently induces aggregation. In the 5th chapter, the results of placing drops of various nitrogenous and non-nitrogenous organic fluids on the discs of leaves were given, and it was shown that they detect with almost unerring certainty the presence of nitrogen. A decoction of green peas or of fresh cabbage leaves acts almost as powerfully as an infusion of raw meat, whereas an infusion of cabbage, page 268, leaves made by keeping them for a long time in merely warm water is far less efficient. A decoction of grass leaves is less powerful than one of green peas or cabbage leaves. These results led me to inquire whether Drosera possessed the power of dissolving solid animal matter, the experiments proving that the leaves are capable of true digestion, and that the glands absorb the digested matter are given in detail in the 6th chapter. These are perhaps the most interesting of all my observations on Drosera, as no such power was before distinctly known to exist in the vegetable kingdom. It is likewise an interesting fact that the glands of the disc, when irritated, should transmit some influence to the glands of the exterior tentacles, causing them to secrete more copiously, and the secretion to become acid, as if they had been directly excited by an object placed on them. The gastric juice of animals contains, as is well known, an acid and a ferment, both of which are indispensable for digestion, and so it is with the secretion of Drosera. When the stomach of an animal is mechanically irritated, it secretes an acid, and when particles of glass or other such objects were placed on the glands of Drosera, the secretion and that of the surrounding and untouched glands was increased in quantity and became acid. But according to Skiff, the stomach of an animal does not secrete its proper ferment pepsin until certain substances, which he calls peptogenes, are absorbed, and it appears from my experiments that some matter must be absorbed by the glands of Drosera before they secrete their proper ferment. That the secretion does contain a ferment, which acts only in the presence of an acid on solid animal matter, was clearly proved by adding minute doses of page 269, an alkali which entirely arrested the process of digestion, this immediately recommencing as soon as the alkali was neutralized by a little weak hydrochloric acid. From trials made with a large number of substances, it was found that those which the secretion of Drosera dissolves completely or partially, or not at all, are acted on in exactly the same manner by gastric juice. We may therefore conclude that the ferment of Drosera is closely analogous to or identical with the pepsin of animals. The substances which are digested by Drosera act on the leaves very differently. Some cause much more energetic and rapid inflection of the tentacles, and keep them inflected for a much longer time than do others. We are thus led to believe that the former are more nutritious than the latter, as is known to be the case with some of these same substances when given to animals. For instance, meat in comparison with gelatine, as cartilage is so tough a substance, and is so little acted on by water, its prompt dissolution by the secretion of Drosera and subsequent absorption is perhaps one of the most striking cases, but it is not really more remarkable than the digestion of meat, which is dissolved by this secretion in the same manner, and by the same stages as by gastric juice. The secretion dissolves bone and even the enamel of teeth, but this is simply due to the large quantity of acid secreted, owing apparently to the desire of the plant for phosphorus. In the case of bone, the ferment does not come into play until all the phosphate of lime has been decomposed and free acid is present, and then the fibrous basis is quickly dissolved. Lastly, the secretion attacks and dissolves matter out of living seeds, which it sometimes kills or injures, as shown by the diseased state, page 270 of the seedlings. It also absorbs matter from pollen and from fragments of leaves. The seventh chapter was devoted to the action of the salts of ammonia. These all cause the tentacles, and often the blade of the leaf, to be inflected, and the protoplasm to be aggregated. They act with very different power, the citrate being the least powerful, and the phosphate, owing no doubt to the presence of phosphorus and nitrogen, by far the most powerful. But the relative efficiency of only 3 salts of ammonia was carefully determined, namely the carbonate, nitrate, and phosphate. The experiments were made by placing half-minims 296 Roman 1050 of solutions of different strengths on the discs of the leaf, applying a minute drop, about the 120 of a minim, or 296 Roman 1050, for a few seconds to 3 or 4 gland sand, by the immersion of whole leaves in a measured quantity. In relation to these experiments, it was necessary first to ascertain the effects of distilled water, and it was found, as described in detail, that the more sensitive leaves are affected by it, but only in a slight degree. A solution of the carbonate is absorbed by the roots and induces aggregation in their cells, but does not affect the leaves. The vapor is absorbed by the glands and causes inflection as well as aggregation. A drop of a solution containing 1960 of a grain 675 mg is the least quantity which, when placed on the glands of the disc, excites the exterior tentacles to bend inwards, but a minute drop containing 114,400 of a grain 445 mg, if applied for a few seconds to the secretions surrounding a gland, causes the inflection of the same tentacle. When a highly sensitive leaf is immersed in a solution, and there is ample time for absorption, the 1,268,800 of a grain, page 271.24 mg, is sufficient to excite a single tentacle into movement. The nitrate of ammonia induces aggregation of the protoplasm much less quickly than the carbonate, but is more potent in causing inflection. A drop containing 12,400 of a grain 27 mg placed on the disc acts powerfully on all the exterior tentacles, which have not themselves received any of the solution, whereas a drop with 12,800 of a grain caused only a few of these tentacles to bend, but affected rather more plainly the blade. A minute drop applied as before, and containing 128,800 of a grain 25 mg, caused the tentacle bearing this gland to bend by the immersion of whole leaves. It was proved that the absorption by a single gland of 1,691,200 of a grain 937 mg was sufficient to set the same tentacle into movement. The phosphate of ammonia is much more powerful than the nitrate. A drop containing 13,840 of a grain 169 mg placed on the disc of a sensitive leaf causes most of the exterior tentacles to be inflected as well as the blade of the leaf. A minute drop containing 1,153,600 of a grain 423 mg applied for a few seconds to a gland acts, as shown by the movement of the tentacle, when a leaf is immersed in 30 minims 1 Roman 1050 of a solution of 1 part by weight of the salt to 21,875,000 of water. The absorption by a gland of only the 119,760,000 of a grain 328 mg, that is about the 1/20 of a grain, is sufficient to cause the tentacle bearing this gland to bend to the center of the leaf. In this experiment, owing to the presence of the water of crystallization, less than the 1/30 of a grain of the efficient elements could have been absorbed. There is nothing remarkable in such minute quantities being absorbed by the glands. Page 272. For all physiologists admit that the salts of ammonia, which must be brought in still smaller quantity by a single shower of rain to the roots, are absorbed by them, nor is it surprising that Drosera should be enabled to profit by the absorption of these salts, for yeast and other low fungoid forms flourish in solutions of ammonia if the other necessary elements are present. But it is an astonishing fact, on which I will not here again enlarge, that so inconceivably minute a quantity as the 1/20 of a grain of phosphate of ammonia should induce some change in a gland of Drosera sufficient to cause a motor impulse to be sent down the whole length of the tentacle, this impulse exciting movement, often through an angle of above 180°. I know not whether to be most astonished at this fact, or that the pressure of a minute bit of hair supported by the dense secretion should quickly cause conspicuous movement. Moreover, this extreme sensitiveness, exceeding that of the most delicate part of the human body, as well as the power of transmitting various impulses from one part of the leaf to another, have been acquired without the intervention of any nervous system. As few plants are at present known to possess glands specially adapted for absorption, it seemed worthwhile to try the effects on Drosera of various other salts, besides those of ammonia, and of various acids. Their action, as described in the 8th chapter, does not correspond at all strictly with their chemical affinities, as inferred from the classification commonly followed. The nature of the base is far more influential than that of the acid, and this is known to hold good with animals. For instance, 9 salts of sodium all caused well-marked inflection, and none of them were poisonous in small doses, whereas 7 of the 9 core page 273 sponding salts of potassium produced no effect too, causing slight inflection, small doses. Moreover, of some of the latter salts were poisonous. The salts of sodium and potassium, when injected into the veins of animals, likewise differ widely in their action. The so-called earthy salts produce hardly any effect on Drosera. On the other hand, most of the metallic salts cause rapid and strong inflection and are highly poisonous, but there are some odd exceptions to this rule. Thus, chloride of lead and zinc, as well as 2 salts of barium, did not cause inflection and were not poisonous. Most of the acids which were tried, though much diluted 1 part to 437 of water, and given in small doses, acted powerfully on Drosera. 19 out of the 24, causing the tentacles to be more or less inflected. Most of them, even the organic acids, are poisonous, often highly so, and this is remarkable, as the juices of so many plants contain acids. Benzoic acid, which is innocuous to animals, seems to be as poisonous to Drosera as hydrocyanic. On the other hand, hydrochloric acid is not poisonous either to animals or to Drosera, and induces only a moderate amount of inflection. Many acids excite the glands to secrete an extraordinary quantity of mucus, and the protoplasm within their cells seems to be often killed, as may be inferred from the surrounding fluid soon becoming pink. It is strange that allied acids act very differently. Formic acid induces very slight inflection and is not poisonous, whereas acetic acid of the same strength acts most powerfully and is poisonous. Lactic acid is also poisonous, but causes inflection only after a considerable lapse of time. Malic acid acts slightly, whereas citric and tartaric acids produce no effect. Page 274. In the 9th chapter, the effects of the absorption of various alkaloids and certain other substances were described. Although some of these are poisonous, yet as several which act powerfully on the nervous system of animals produce no effect on Drosera, we may infer that the extreme sensibility of the glands and their power of transmitting an influence to other parts of the leaf, causing movement or modified secretion or aggregation, does not depend on the presence of a diffused element allied to nerve tissue. One of the most remarkable facts is that long immersion in the poison of the cobra snake does not in the least check, but rather stimulates the spontaneous movements of the protoplasm. In the cells of the tentacles, solutions of various salts and acids behave very differently in delaying or in quite arresting the subsequent action of a solution of phosphate of ammonia. Camphor dissolved in water acts as a stimulant, as do small doses of certain essential oils, for they cause rapid and strong inflection. Alcohol is not a stimulant. The vapors of camphor, alcohol, chloroform, sulfuric, and nitric ether are poisonous in moderately large doses, but in small doses serve as narcotics or anesthetics, greatly delaying the subsequent action of meat. But some of these vapors also act as stimulants, exciting rapid, almost spasmodic movements in the tentacles. Carbonic acid is likewise a narcotic and retards the aggregation of the protoplasm. When carbonate of ammonia is subsequently given the first access of air to plants which have been immersed in this gas, sometimes acts as a stimulant and induces movement. But as before remarked, a special pharmacopoeia would be necessary to describe the diversified effects of various substances on the leaves of Drosera. In the 10th chapter, it was shown that the sensitive page 275-ness of the leaves appears to be wholly confined to the glands, and to the immediately underlying cells, it was further shown that the motor impulse and other forces or influences proceeding from the glands, when excited, pass through the cellular tissue and not along the fibrovascular bundles. A gland sends its motor impulse with great rapidity down the pedicel of the same tentacle to the basal part, which alone bends the impulse, then passing onwards spreads on all sides to the surrounding tentacles, first affecting those which stand nearest, and then those farther off, but by being thus spread out, and from the cells of the disc not being so much elongated as those of the tentacles, it loses force and here travels much more slowly than down the pedicels, owing also to the direction and form of the cells. It passes with greater ease and celerity in a longitudinal than in a transverse line across the disc. The impulse proceeding from the glands of the extreme marginal tentacles does not seem to have force enough to affect the adjoining tentacles, and this may be in part due to their length. The impulse from the glands of the next few inner rows spreads chiefly to the tentacles on each side and towards the center of the leaf, but that proceeding from the glands of the shorter tentacles on the disc radiates almost equally on all sides. When a gland is strongly excited by the quantity or quality of the substance placed on it, the motor impulse travels farther than from one slightly excited, and if several glands are simultaneously excited, the impulses from all unite and spread still farther. As soon as a gland is excited, it discharges an impulse which extends to a considerable distance, but afterwards, whilst the gland is secreting and absorbing the impulse, suffices only to keep the same tentacle, page 276, inflected, though the inflection may last for many days. If the bending place of a tentacle receives an impulse from its own gland, the movement is always towards the center of the leaf, and so it is with all the tentacles when their glands are excited by immersion in a proper fluid. The short ones in the middle part of the disc must be excepted, as these do not bend at all when thus excited. On the other hand, when the motor impulse comes from one side of the disc, the surrounding tentacles, including the short ones in the middle of the disc, all bend with precision towards the point of excitement, wherever this may be seated. This is in every way a remarkable phenomenon, for the leaf falsely appears as if endowed with the senses of an animal. It is all the more remarkable, as when the motor impulse strikes the base of a tentacle obliquely, with respect to its flattened surface, the contraction of the cells must be confined to one, two, or a very few rows at one end, and different sides of the surrounding tentacles must be acted on in order that all should bend with precision to the point of excitement. The motor impulse, as it spreads from one or more glands across the disc, enters the bases of the surrounding tentacles and immediately acts on the bending place. It does not, in the first place, proceed up the tentacles to the glands, exciting them to reflect back an impulse to their bases. Nevertheless, some influence is sent up to the glands, as their secretion is soon increased and rendered acid, and then the glands, being thus excited, send back some other influence, not dependent on increased secretion, nor on the inflection of the tentacles, causing the protoplasm to aggregate in cell beneath cell. This may, page 277, be called a reflex action, though probably very different from that proceeding from the nerve ganglion of an animal, and it is the only known case of reflex action in the vegetable kingdom. About the mechanism of the movements and the nature of the motor impulse, we know very little. During the act of inflection, fluid certainly travels from one part to another of the tentacles, but the hypothesis which agrees best with the observed facts is that the motor impulse is allied in nature to the aggregating process, and that this causes the molecules of the cell walls to approach each other in the same manner as do the molecules of the protoplasm within the cells, so that the cell walls contract. But some strong objections may be urged against this view. The re-expansion of the tentacles is largely due to the elasticity of their outer cells, which comes into play as soon as those on the inner side cease contracting with prepotent force. But we have reason to suspect that fluid is continually and slowly attracted into the outer cells during the act of re-expansion, thus increasing their tension. I have now given a brief recapitulation of the chief points observed by me with respect to the structure, movements, constitution, and habits of Drosera rotundifolia, and we see how little has been made out in comparison with what remains unexplained and unknown, page 278. Chapter Roman 12, on the structure and movements of some other species of Drosera. Drosera anglica, Drosera intermedia, Drosera capensis, Drosera spathulata, Drosera filiformis, Drosera binata. Concluding remarks. I examined 6 other species of Drosera, some of them inhabitants of distant countries, chiefly for the sake of ascertaining whether they caught insects. This seemed the more necessary, as the leaves of some of the species differ to an extraordinary degree in shape from the rounded ones of Drosera rotundifolia. In functional powers, however, they differ very little. Drosera anglica Hudson. The leaves of this species, which was sent to me from Ireland, are much elongated and gradually widen from the footstalk to the bluntly pointed apex. They stand almost erect, and their blades sometimes exceed 1 inch in length, whilst their breadth is only the 15th of an inch. The glands of all the tentacles have the same structure, so that the extreme marginal ones do not differ from the others, as in the case of Drosera rotundifolia, when they are irritated by being roughly touched, or by the pressure of minute inorganic particles, or by contact with animal matter, or by the absorption of carbonate of ammonia. The tentacles become inflected, the basal portion being the chief seat of movement, cutting or pricking. The blade of the leaf did not excite any movement. They frequently capture insects, and the glands of the inflected tentacles pour forth much acid secretion. Bits of roast meat were placed on some glands, and the tentacles began to move in 1 Roman 1000, or. Mrs. Treat has given an excellent account in the American Naturalist, December 1873, p. 705, of Drosera longifolia, which is a synonym in part of Drosera anglica, of Drosera rotundifolia, and filiformis, page 279. 1 Roman 1000 s, and in 1 Hr 10 Roman 1000, reached the center 2 bits of boiled cork, 1 of boiled thread, and 2 of coal cinders taken from the fire, were placed by the aid of an instrument, which had been immersed in boiling water on 5 glands, these superfluous precautions having been taken on account of Roman 1000. Ziegler's statements. 1 of the particles of cinder caused some inflection in 8 HRS 45 Roman 1000, as did after 23 HRS the other particle of cinder, the bit of thread, and both bits of cork. 3 glands were touched half a dozen times with a needle. 1 of the tentacles became well inflected in 17 Roman 1000, and re-expanded after 24 HRS. The 2 others never moved. The homogeneous fluid within the cells of the tentacles undergoes aggregation after these have become inflected, especially if given a solution of carbonate of ammonia. And I observed the usual movements in the masses of protoplasm. In 1 case, aggregation ensued in 1 Hr 10 Roman 1000, after a tentacle had carried a bit of meat to the center. From these facts, it is clear that the tentacles of Drosera anglica behave like those of Drosera rotundifolia. If an insect is placed on the central glands, or has been naturally caught there, the apex of the leaf curls inwards. For instance, dead flies were placed on 3 leaves near their bases, and after 24 HRS, the previously straight apices were curled completely over so as to embrace and conceal the flies. They had therefore moved through an angle of 180 O. After 3 days, the apex of 1 leaf together with the tentacles began to re-expand, but as far as I have seen, and I made many trials, the sides of the leaf are never inflected, and this is the one functional difference between this species and Drosera rotundifolia. Drosera intermedia Hain. This species is quite as common in some parts of England as Drosera rotundifolia. It differs from Drosera anglica as far as the leaves are concerned only in their smaller size, and in their tips being generally a little reflexed. They capture a large number of insects. The tentacles are excited into movement by all the causes above specified, and aggregation ensues with movement of the protoplasmic masses. I have seen through a lens a tentacle beginning to bend in less than a minute after a particle of raw meat had been placed on the gland. The apex of the leaf curls over an exciting object. As in the case of Drosera anglica, acid secretion is copiously poured over captured insects. A leaf which had embraced a fly with all its tentacles re-expanded after nearly 3 days. Drosera capensis, This species, a native of the Cape of Good Hope, was sent to me by Dr. Hooker. The leaves are elongated, slightly concave, along the middle and taper towards the apex, page 280, which is bluntly pointed and reflexed. They rise from an almost woody axis, and their greatest peculiarity consists in their foliaceous green footstalks, which are almost as broad and even longer than the gland-bearing blade. This species, therefore, probably draws more nourishment from the air and less from captured insects than the other species of the genus. Nevertheless, the tentacles are crowded together on the disc and are extremely numerous, those on the margins being much longer than the central ones. All the glands have the same form. Their secretion is extremely viscid and acid. The specimen which I examined had only just recovered from a weak state of health. This may account for the tentacles moving very slowly when particles of meat were placed on the glands, and perhaps for my never succeeding in causing any movement by repeatedly touching them with a needle. But with all the species of the genus, this latter stimulus is the least effective of any. Particles of glass, cork, and coal cinders were placed on the glands of 6 tentacles, and one alone moved after an interval of 2 HRS 30 Roman 1000. Nevertheless, 2 glands were extremely sensitive to very small doses of the nitrate of ammonia, namely to about 120 of minimum of a solution, 1 part to 5250 of water, containing only 1,115,200 of a grain. 562 mg of the salt fragments of flies were placed on 2 leaves near their tips, which became incurved in 15 HRS. A fly was also placed in the middle of the leaf. In a few hours, the tentacles on each side embraced it, and in 8 HRS the whole leaf directly beneath the fly was a little bent transversely. By the next morning, after 23 HRS, the leaf was curled so completely over that the apex rested on the upper end of the footstalk. In no case did the sides of the leaves become inflected. A crushed fly was placed on the foliaceous footstalk, but produced no effect. Drosera spathulata, sent to me by Dr. Hooker, made only a few observations on this Australian species, which has long, narrow leaves, gradually widening towards their tips. The glands of the extreme marginal tentacles are elongated and differ from the others. As in the case of Drosera rotundifolia, a fly was placed on a leaf. In 18 HRS, it was embraced by the adjoining tentacles. Gumwater, dropped on several leaves, produced no effect. A fragment of a leaf was immersed in a few drops of a solution of 1 part of carbonate of ammonia to 146 of water. All the glands were instantly blackened. The process of aggregation could be seen traveling rapidly down the cells of the tentacles, and the granules of protoplasm soon united into spheres and variously shaped masses, which displayed the usual move. Page 281 meant half a minim of a solution of 1 part of nitrate of ammonia to 146 of water was next placed on the center of a leaf. After 6 HRS, some marginal tentacles on both sides were inflected, and after 9 HRS, they met in the center. The lateral edges of the leaf also became incurved, so that it formed a half-cylinder, but the apex of the leaf in none of my few trials was inflected. The above dose of the nitrate, viz. 1320 of a grain or 202 mg, was too powerful, for in the course of 23 HRS, the leaf died. Drosera filiformis, this North American species, grows in such abundance in parts of New Jersey as almost to cover the ground. It catches, according to Mrs. Treat, an extraordinary number of small and large insects, even great flies of the genus Acilus moths, and butterflies. The specimen which I examined, sent me by Dr. Hooker, had thread-like leaves from 6 to 12 inches in length, with the upper surface convex and the lower flat, and slightly channeled the whole convex surface. Down to the root sphere, there is no distinct footstalkies, covered with short gland-bearing tentacles, those on the margins being the longest, and reflexed bits of meat placed on the glands of some tentacles caused them to be slightly inflected. In 20 Roman 1000, but the plant was not in a vigorous state. After 6 HRS, they moved through an angle of 90°, and in 24 HRS reached the center. The surrounding tentacles, by this time, began to curve inwards. Ultimately, a large drop of extremely viscid, slightly acid secretion was poured over the meat from the united glands. Several other glands were touched with a little saliva, and the tentacles became incurved in under 1 HR and re-expanded after 18 HRS particles of glass cork. Cinders, thread, and gold leaf were placed on numerous glands on 2 leaves. In about 1 HR, 4 tentacles became curved, and 4 others, after an additional interval of 2 HRS 30 Roman 1000, I never once succeeded in causing any movement by repeatedly touching the glands with a needle, and Mrs. Treat made similar trials for me with no success. Small flies were placed on several leaves near their tips, but the thread-like blade became only on one occasion very slightly bent directly beneath the insect. Perhaps this indicates that the blades of vigorous plants would bend over captured insects, and Dr. Canby informs me that this is the case, but the movement cannot be strongly pronounced, as it was not observed by Mrs. Treat. Drosera binata, or dichotoma, I am much indebted to Lady. American Naturalist, December 1873, page 705, page 282. Dorothy Neville, for a fine plant of this almost gigantic Australian species, which differs in some interesting points from those previously described in this specimen. The rush-like footstalks of the leaves were 20 inches in length. The blade bifurcates at its junction with the footstalk and twice or thrice afterwards curling about in an irregular manner. It is narrow, being only 320 of an inch in breadth. One blade was 7/12 inches long, so that the entire leaf, including the footstalk, was above 27 inches in length. Both surfaces are slightly hollowed out. The upper surface is covered with tentacles arranged in alternate rows, those in the middle being short, and crowded together, those towards the margins longer, even twice or thrice as long as the blade is broad. The glands of the exterior tentacles are of a much darker red than those of the central ones. The pedicels of all are green. The apex of the blade is attenuated and bears very long tentacles. Mr. Copeland informs me that the leaves of a plant, which he kept for some years, were generally covered with captured insects before they withered. The leaves do not differ in essential points of structure or of function from those of the previously described species. Bits of meat or a little saliva placed on the glands of the exterior tentacles caused well-marked movement in 3 Roman 1000, and particles of glass acted in 4 Roman 1000, the tentacles, with the latter particles re-expanded after 22 HRS. A piece of leaf immersed in a few drops of a solution of 1 part of carbonate of ammonia to 437 of water had all the glands blackened and all the tentacles inflected in 5 Roman 1000. A bit of raw meat placed on several glands in the medial furrow was well clasped in 2 HRS 10 Roman 1000 by the marginal tentacles on both sides. Bits of roast meat and small flies did not act quite so quickly, and albumin and fibrin still less quickly. One of the bits of meat excited so much secretion, which is always acid, that it flowed some way down the medial furrow, causing the inflection of the tentacles on both sides as far as it extended. Particles of glass placed on the glands in the medial furrow did not stimulate them sufficiently for any motor impulse to be sent to the outer tentacles. In no case was the blade of the leaf, even the attenuated apex, at all inflected. On both the upper and lower surface of the blade, there are numerous minute, almost sessile glands consisting of 4, 8, or 12 cells on the lower surface. They are pale purple on the upper, greenish. Nearly similar organs occur on the footstalks, but they are smaller and often in a shriveled condition. The minute glands on the blade can absorb rapidly. Thus a piece of leaf was immersed in a solution of 1 part of carbonate, page 283, of ammonia to 218 of water, 1 gr to 2 Rs, and in 5 Roman 1000 they were all so much darkened as to be almost black. With their contents aggregated, they do not, as far as I could observe, secrete spontaneously, but in between 2 and 3 HRS, after a leaf had been rubbed with a bit of raw meat moistened with saliva, they seemed to be secreting freely, and this conclusion was afterwards supported by other appearances. They are therefore homologous with the sessile glands hereafter to be described on the leaves of Dionea and Drosophilum. In this latter genus, they are associated, as in the present case, with glands which secrete spontaneously, that is, without being excited. Drosera binata presents another and more remarkable peculiarity, namely the presence of a few tentacles on the backs of the leaves near their margins. These are perfect in structure spiral vessels run up their pedicels. Their glands are surrounded by drops of viscid secretion, and they have the power of absorbing. This latter fact was shown by the glands immediately becoming black, and the protoplasm aggregated when a leaf was placed in a little solution of 1 part of carbonate of ammonia to 437 of water. These dorsal tentacles are short, not being nearly so long as the marginal ones on the upper surface. Some of them are so short as almost to graduate into the minute sessile glands. Their presence, number, and size vary on different leaves, and they are arranged rather irregularly on the back of one leaf. I counted as many as 21 along one side. These dorsal tentacles differ in one important respect from those on the upper surface, namely in not possessing any power of movement in whatever manner they may be stimulated. Thus, portions of 4 leaves were placed at different times in solutions of carbonate of ammonia, 1 part to 437 or 218 of water, and all the tentacles on the upper surface soon became closely inflected, but the dorsal ones did not move, though the leaves were left in the solution for many hours, and though their glands from their blackened color had obviously absorbed some of the salt. Rather, young leaves should be selected for such trials, for the dorsal tentacles, as they grow old and begin to wither, often spontaneously incline towards the middle of the leaf. If these tentacles had possessed the power of movement, they would not have been thus rendered more serviceable to the plant, for they are not long enough to bend round the margin of the leaf, so as to reach an insect caught on the upper surface, nor would it have been of any use if these tentacles could have page 284 moved towards the middle of the lower surface, for there are no viscid glands there by which insects can be caught. Although they have no power of movement, they are probably of some use by absorbing animal matter from any minute insect which may be caught by them, and by absorbing ammonia from the rainwater. But their varying presence and size and their irregular position indicate that they are not of much service, and that they are tending towards abortion. In a future chapter, we shall see that Drosophilum, with its elongated leaves, probably represents the condition of an early progenitor of the genus Drosera, and none of the tentacles of Drosophilum, neither those on the upper nor lower surface of the leaves, are capable of movement. When excited, though they capture numerous insects, which serve as nutriment. Therefore, it seems that Drosera binata has retained remnants of certain ancestral characters, namely, a few motionless tentacles on the backs of the leaves, and fairly well-developed sessile glands which have been lost by most or all of the other species of the genus. Concluding remarks from what we have now seen, there can be little doubt that most or probably all the species of Drosera are adapted for catching insects by nearly the same means. Besides the 2 Australian species above described, it is said that 2 other species from this country, namely Drosera pallida and Drosera sulphurea, close their leaves upon insects with great rapidity, and the same phenomenon is manifested by an Indian species, Roman 500 lunata, and by several of those of the Cape of Good Hope, especially by Roman 500 trinovis, another Australian species Drosera heterophylla, made by Lindley into a distinct genus Sondera, is remarkable from its peculiarly shaped leaves, but I know nothing of its power of catching insects, for I have seen only dried specimens. The leaves form minute flattened cups, with the footstalks attached not to one margin, but to the bottom. Gardner's Chronicle, 1874, p. 209, page 285. Inner surface and the edges of the cups are studded with tentacles, which include fibrovascular bundles rather different from those seen by me in any other species, for some of the vessels are barred and punctured. Instead of being spiral, the glands secrete copiously, judging from the quantity of dried secretion adhering to them, page 286. Chapter Roman 13, Dionea, Marcipula. Structure of the leaves. Sensitiveness of the filaments. Rapid movement of the lobes caused by irritation of the filaments glands. Their power of secretion. Slow movement caused by the absorption of animal matter. Evidence of absorption from the aggregated condition of the glands. Digestive power of the secretion. Action of chloroform ether and hydrocyanic acid. The manner in which insects are captured. Use of the marginal spike skines of insects captured. The transmission of the motor impulse and mechanism of the movements. Re-expansion of the lobes. This plant, commonly called Venus flytrap, from the rapidity and force of its movements, is one of the most wonderful in the world. It is a member of the small family of the Droseraceae and is found only in the eastern part of North Carolina. Growing in damp situations, the roots are small, those of a moderately fine plant, which I examined consisted of 2 branches, about 1 inch in length, springing from a bulbous enlargement. They probably serve, as in the case of Drosera, solely for the absorption of water. For a gardener who has been very successful in the cultivation of this plant, grows it like an epiphytic orchid in well-drained damp moss, without any soil. The form of the bilobed leaf, with its foliaceous footstalk, is shown in the accompanying drawing, Fig. 12. Dr. Hooker, in his address to the British Association at Belfast 1874, has given so full an historical account of the observations which have been published on the habits of this plant that it would be superfluous on my part to repeat them. Gardener's Chronicle, 1874, p. 464, page 287. 2 lobes stand at rather less than a right angle to each other. 3-minute pointed processes or filaments placed triangularly project from the upper surfaces of both. But I have seen 2 leaves with 4 filaments on each side and another with only 2. These filaments are remarkable from their extreme sensitiveness to a touch, as shown not by their own movement, but by that of the lobes. The margins of the leaf are prolonged into sharp, rigid projections, which I will call spikes, into each of which a bundle. Fig. 12. Dionea marcipula leaf, viewed laterally in its expanded state. Of spiral vessels, enters. The spikes stand in such a position that when the lobes close they interlock, like the teeth of a rat trap. The midrib of the leaf on the lower side is strongly developed and prominent. The upper surface of the leaf is thickly covered, excepting towards the margins, with minute glands of a reddish or purplish color, the rest of the leaf being green. There are no glands on the spikes or on the foliaceous footstalk. The glands are formed of, from page 288, 20 to 30 polygonal cells filled with purple fluid. Their upper surface is convex. They stand on very short pedicels, into which spiral vessels do not enter, in which respect they differ from the tentacles of Drosera. They secrete, but only when excited by the absorption of certain matters. And they have the power of absorption. Minute projections, formed of 8 divergent arms of a reddish-brown or orange color, and appearing under the microscope like elegant little flowers, are scattered in considerable numbers over the footstalk, the backs of the leaves, and the spikes, with a few on the upper surface of the lobes. These octafid projections are no doubt homologous with the papillae on the leaves of Drosera rotundifolia. There are also a few very minute simple pointed hairs, about 712,148 Roman 2000, of an inch in length, on the backs of the leaves. The sensitive filaments are formed of several rows of elongated cells filled with purplish fluid. They are a little above the 120 of an inch in length, are thin and delicate, and taper to a point. I examined the bases of several making sections of them, but no trace of the entrance of any vessel could be seen. The apex is sometimes bifid or even trifid, owing to a slight separation between the terminal pointed cells. Towards the base, there is constriction formed of broader cells, beneath which there is an articulation supported on an enlarged base, consisting of differently shaped polygonal cells. As the filaments project at right angles to the surface of the leaf, they would have been liable to be broken whenever the lobes closed together, had it not been for the articulation, which allows them to bend flat down. These filaments, from their tips to their bases, are exquisitely sensitive to a momentary touch. It is scarcely page 289 possible to touch them ever so lightly or quickly with any hard object, without causing the lobes to close. A piece of very delicate human hair, 12 inches in length, held dangling over a filament and swayed to and fro so as to touch it, did not excite any movement. But when a rather thick cotton thread of the same length was similarly swayed, the lobes closed. Pinches of fine wheaten flour dropped from a height produced no effect. The above-mentioned hair was then fixed into a handle and cut off so that 1 inch projected, this length being sufficiently rigid to support itself in a nearly horizontal line. The extremity was then brought by a slow movement laterally into contact with the tip of a filament, and the leaf instantly closed. On another occasion, 2 or 3 touches of the same kind were necessary, before any movement ensued. When we consider how flexible a fine hair is, we may form some idea how slight must be the touch given by the extremity of a piece 1 inch in length, moved slowly. Although these filaments are so sensitive to a momentary and delicate touch, they are far less sensitive than the glands of Drosera to prolonged pressure. Several times I succeeded in placing on the tip of a filament, by the aid of a needle, moved with extreme slowness, bits of rather thick human hair, and these did not excite movement, although they were more than 10 times as long as those which caused the tentacles of Drosera to bend. And although in this latter case they were largely supported by the dense secretion, on the other hand, the glands of Drosera may be struck with a needle or any hard object once, twice, or even thrice, with considerable force, and no movement ensues. This singular difference in the nature of the sensitiveness of the filaments of Dionea and of page 290, the glands of Drosera, evidently stands in relation to the habits of the two plants. If a minute insect alights with its delicate feet on the glands of Drosera, it is caught by the viscid secretion. The slight though prolonged pressure gives notice of the presence of prey, which is secured by the slow bending of the tentacles. On the other hand, the sensitive filaments of Dionea are not viscid, and the capture of insects can be assured only by their sensitiveness to a momentary touch, followed by the rapid closure of the lobes. As just stated, the filaments are not glandular and do not secrete, nor have they the power of absorption, as may be inferred from drops of a solution of carbonate of ammonia 1 part to 146 of water, placed on 2 filaments, not producing any effect on the contents of their cells, nor causing the lobes to close. When, however, a small portion of a leaf with an attached filament was cut off and immersed in the same solution, the fluid within the basal cells became almost instantly aggregated into purplish or colorless, irregularly shaped masses of matter. The process of aggregation gradually traveled up the filaments from cell to their extremities, that is, in a reverse course to what occurs in the tentacles of Drosera when their glands have been excited. Several other filaments were cut off close to their bases and left for 1 Roman 1000 in a weaker solution of 1 part of the carbonate to 218 of water, and this caused aggregation in all the cells, commencing as before at the bases of the filaments. Long immersion of the filaments in distilled water likewise causes aggregation, nor is it rare to find the contents of a few of the terminal cells in a spontaneously aggregated condition. The aggregated page 291 masses undergo incessant slow changes of form, uniting and again separating, and some of them apparently revolve round their own axes. A current of colorless granular protoplasm could also be seen traveling round the walls of the cells. This current ceases to be visible as soon as the contents are well aggregated, but it probably still continues, though no longer visible, owing to all the granules in the flowing layer having become united with the central masses. In all these respects, the filaments of Dionea behave exactly like the tentacles of Drosera. Notwithstanding this similarity, there is one remarkable difference. The tentacles of Drosera, after their glands have been repeatedly touched, or a particle of any kind has been placed on them, become inflected and strongly aggregated. No such effect is produced by touching the filaments of Dionea. I compared, after an hour or two, some which had been touched, and some which had not, and others after 25 hours, and there was no difference in the contents of the cells. The leaves were kept open all the time by clips, so that the filaments were not pressed against the opposite lobe. Drops of water or a thin broken stream falling from a height on the filaments did not cause the blades to close, though these filaments were afterwards proved to be highly sensitive, no doubt. As in the case of Drosera, the plant is indifferent to the heaviest shower of rain. Drops of a solution of ½ ounce of sugar to a fluid ounce of water were repeatedly allowed to fall from a height on the filaments, but produced no effect unless they adhered to them. Again, I blew many times through a fine-pointed tube with my utmost force against the filaments, without any effect, such blowing being received. Page 292, with as much indifference as, no doubt, is a heavy gale of wind. We thus see that the sensitiveness of the filaments is of a specialized nature, being related to a momentary touch rather than to prolonged pressure. and the touch must not be from fluids such as air or water, but from some solid object. Although drops of water and of a moderately strong solution of sugar falling on the filaments does not excite them, yet the immersion of a leaf in pure water sometimes caused the lobes to close. One leaf was left immersed for 1 Roman 1000, and 3 other leaves for some minutes in water at temperatures varying between 59 and 65-15 to 183 cent without any effect. One, however, of these 4 leaves, on being gently withdrawn from the water, closed rather quickly. The 3 other leaves were proved to be in good condition as they closed when their filaments were touched. Nevertheless, 2 fresh leaves, on being dipped into water at 75 and 62, 238 and 169 cent, instantly closed. These were then placed with their footstalks in water, and after 23 HRS partially re-expanded on touching their filaments. One of them closed. This latter leaf, after an additional 24 HRS, again re-expanded, and now, on the filaments of both leaves being touched, both closed. We thus see that a short immersion in water does not at all injure the leaves, but sometimes excites the lobes to close. The movement in the above cases was evidently not caused by the temperature of the water. It has been shown that long immersion causes the purple fluid within the cells of the sensitive filaments to become aggregated, and the tentacles of Drosera are acted on in the same manner by long immersion, often being somewhat inflected in both cases. The result is probably due to a slight degree of exosmos, page 293. I am confirmed in this belief by the effects of immersing a leaf of Dionea in a moderately strong solution of sugar, the leaf having been previously left for 1 Roman 1000 in water, without any effect, for now the lobes closed rather quickly, the tips of the marginal spikes crossing in 2 Roman 1000 s, and the leaf being completely shut in 3 Roman 1003 leaves, were then immersed in a solution of half an ounce of sugar to a fluid ounce of water, and all 3 leaves closed quickly, as I was doubtful whether this was due to the cells on the upper surface of the lobes, or to the sensitive filaments being acted on by exosmos. One leaf was first tried by pouring a little of the same solution in the furrow between the lobes over the midrib, which is the chief seat of movement. It was left there for some time, but no movement ensued. The whole upper surface of leaf was then painted, except close round the bases of the sensitive filaments, which I could not do without risk of touching them with the same solution, but no effect was produced, so that the cells on the upper surface are not thus affected. But when after many trials I succeeded in getting a drop of the solution to cling to one of the filaments, the leaf quickly closed. Hence, we may, I think, conclude that the solution causes fluid to pass out of the delicate cells of the filaments by exosmos, and that this sets up some molecular change in their contents analogous to that which must be produced by a touch. The immersion of leaves in a solution of sugar affects them for a much longer time than does an immersion in water or a touch on the filaments, for in these latter cases, the lobes begin to re-expand in less than a day. On the other hand, of the 3 leaves, which were immersed for a short time in the solution, and were then washed by means of a syringe, inserted page 294, between the lobes, one re-expanded after 2 days, a second after 7 days, and the third after 9 days, the leaf, which closed owing to a drop of the solution having adhered to one of the filaments, opened after 2 days. I was surprised to find on 2 occasions that the heat from the rays of the sun concentrated by a lens on the bases of several filaments, so that they were scorched and discolored, did not cause any movement, though the leaves were active, as they closed, though rather slowly. When a filament on the opposite side was touched on a 3rd trial, a fresh leaf closed after a time, though very slowly, the rate not being increased by one of the filaments, which had not been injured. Being touched after a day, these 3 leaves opened, and were fairly sensitive. When the uninjured filaments were touched, the sudden immersion of a leaf into boiling water does not cause it to close. Judging from the analogy of Drosera, the heat in these several cases was too great and too suddenly applied. The surface of the blade is very slightly sensitive. It may be freely and roughly handled without any movement being caused. A leaf was scratched rather hard with a needle, but did not close. But when the triangular space between the 3 filaments on another leaf was similarly scratched, the lobes closed. They always closed when the blade or midrib was deeply pricked or cut. Inorganic bodies even of large size, such as bits of stone glass Roman 100, or organic bodies, not containing soluble nitrogenous matter, such as bits of wood cork moss or bodies containing soluble nitrogenous matter, if perfectly dry, such as bits of meat albumin gelatine Roman 100 may be long left, and many were tried on the lobes, and no movement is excited. The result, however, is widely different, as we page 295 shall presently see, if nitrogenous organic bodies, which are at all damp, are left on the lobes, for these then close by a slow and gradual movement, very different from that caused by touching one of the sensitive filaments. The footstalk is not in the least sensitive. A pin may be driven through it, or it may be cut off, and no movement follows. The upper surface of the lobes, as already stated, is thickly covered with small purplish, almost sessile glands. These have the power both of secretion and absorption, but unlike those of Drosera, they do not secrete. Until excited by the absorption of nitrogenous matter, no other excitement, as far as I have seen, produces this effect. Objects such as bits of wood cork, moss, paper, stone, or glass may be left for a length of time on the surface of a leaf, and it remains quite dry, nor does it make any difference if the lobes close over such objects. For instance, some little balls of blotting paper were placed on a leaf, and a filament was touched, and when after 24 HRS the lobes began to reopen, the balls were removed by the aid of thin pincers, and were found perfectly dry. On the other hand, if a bit of damp meat or a crushed fly is placed on the surface of an expanded leaf, the glands, after a time, secrete freely. In one such case, there was a little secretion directly beneath the meat in 4 HRS, and after an additional 3 HRS, there was a considerable quantity both under and close round it. In another case, after 3 HRS 40 Roman 1000, the bit of meat was quite wet, but none of the glands secreted excepting those which actually touched the meat, or the secretion containing dissolved animal matter. If, however, the lobes are made to close over a bit of meat or an insect, the result is different, for the glands over the whole surface of the leaf now secrete copiously. Page 296. As in this case, the glands on both sides are pressed against the meat or insect, the secretion from the first is twice as great as when a bit of meat is laid on the surface of one lobe, and as the two lobes come into almost close contact, the secretion containing dissolved animal matter spreads by capillary attraction, causing fresh glands on both sides to begin secreting in a continually widening circle. The secretion is almost colorless, slightly mucilaginous, and judging by the manner in which it colored litmus paper more strongly acid than that of Drosera, it is so copious that on one occasion, when a leaf was cut open, on which a small cube of albumin had been placed 45 HRS, before drops rolled off the leaf. On another occasion, in which a leaf with an enclosed bit of roast meat spontaneously opened after 8 days, there was so much secretion in the furrow over the midrib that it trickled down. A large crushed fly tipula was placed on a leaf from which a small portion at the base of one lobe had previously been cut away, so that an opening was left, and through this the secretion continued to run down the footstalk during 9 days. That is for as long a time as it was observed. By forcing up one of the lobes, I was able to see some distance between them, and all the glands within sight were secreting freely. We have seen that inorganic and non-nitrogenous objects placed on the leaves do not excite any movement, but nitrogenous bodies, if in the least degree damp, cause, after several hours, the lobes to close slowly. Thus bits of quite dry meat and gelatine were placed at opposite ends of the same leaf, and in the course of 24 HRS excited neither secretion nor movement. They were then dipped in water, their surfaces dried on blotting paper, and replaced on the same page 297 leaf. The plant being now covered with a belglass, after 24 HRS, the damp meat had excited some acid secretion, and the lobes at this end of the leaf were almost shut. At the other end, where the damp gelatine lay, the leaf was still quite open, nor had any secretion been excited, so that, as with Drosera, gelatine is not nearly so exciting a substance as meat. The secretion beneath the meat was tested by pushing a strip of litmus paper under it, the filaments not being touched, and this slight stimulus caused the leaf to shut. On the 11th day, it reopened, but the end, where the gelatine lay, expanded several hours before the opposite end with the meat. A second bit of roast meat, which appeared dry though it had not been purposely dried, was left for 24 HRS on a leaf caused neither movement nor secretion. The plant in its pot was now covered with a belglass, and the meat absorbed some moisture from the air. This sufficed to excite acid secretion, and by the next morning the leaf was closely shut. A third bit of meat dried so as to be quite brittle was placed on a leaf under a belglass, and this also became in 24 HRS slightly damp, and excited some acid secretion, but no movement. A rather large piece of perfectly dry albumin was left at one end of a leaf for 24 HRS without any effect. It was then soaked for a few minutes in water rolled about on blotting paper and replaced on the leaf in 9 HRS. Some slightly acid secretion was excited, and in 24 HRS this end of the leaf was partially closed. The bit of albumin, which was now surrounded by much secretion, was gently removed, and although no filament was touched, the lobes closed. In this and the previous case it appears that the absorption of animal matter by the glands renders page 298 the surface of the leaf much more sensitive to a touch than it is in its ordinary state. And this is a curious fact. 2 days afterwards the end of the leaf, where nothing had been placed, began to open, and on the third day was much more open than the opposite end, where the albumin had lain. Lastly, large drops of a solution of 1 part of carbonate of ammonia to 146 of water were placed on some leaves, but no immediate movement ensued. I did not then know of the slow movement caused by animal matter, otherwise I should have observed the leaves for a longer time, and they would probably have been found closed, though the solution, judging from Drosera, was perhaps too strong. From the foregoing cases, it is certain that bits of meat and albumin, if at all damp, excite not only the glands to secrete, but the lobes to close. This movement is widely different from the rapid closure caused by one of the filaments being touched. We shall see its importance when we treat of the manner in which insects are captured. There is a great contrast between Drosera and Dionea in the effects produced by mechanical irritation on the one hand, and the absorption of animal matter on the other. Particles of glass placed on the glands of the exterior tentacles of Drosera excite movement within nearly the same time as do particles of meat, the latter being rather the most efficient. But when the glands of the disc have bits of meat given them, they transmit a motor impulse to the exterior tentacles much more quickly than do these glands when bearing inorganic particles, or when irritated by repeated touches. On the other hand, with Dionea, touching the filaments excites incomparably quicker movement than the absorption of animal matter by the glands. Nevertheless, in page 299, certain cases, this latter stimulus is the more powerful of the two. On 3 occasions leaves were found which from some cause were torpid, so that their lobes closed only slightly. However much their filaments were irritated, but on inserting crushed insects between the lobes, they became in a day closely shut. The facts just given plainly show that the glands have the power of absorption, for otherwise it is impossible that the leaves should be so differently affected by non-nitrogenous and nitrogenous bodies. And between these latter, in a dry and damp condition, it is surprising how slightly damp a bit of meat or albumin need be in order to excite secretion and afterwards slow movement, and equally surprising how minute a quantity of animal matter, when absorbed, suffices to produce these two effects. It seems hardly credible, and yet it is certainly a fact, that a bit of hard-boiled white of egg, first thoroughly dried, then soaked for some minutes in water and rolled on blotting paper, should yield in a few hours enough animal matter to the glands to cause them to secrete, and afterwards the lobes to close. That the glands have the power of absorption is likewise shown by the very different lengths of time, as we shall presently see, during which the lobes remain closed over insects and other bodies yielding soluble nitrogenous matter, and over such as do not yield any. But there is direct evidence of absorption in the condition of the glands, which have remained for some time in contact with animal matter. Thus bits of meat and crushed insects were several times placed on glands, and these were compared after some hours with other glands from distant parts of the same leaf. The latter showed not a trace of aggregation, whereas those which had been in contact with the animal matter were page 300. Well aggregated aggregation may be seen to occur very quickly if a piece of a leaf is immersed in a weak solution of carbonate of ammonia. Again, small cubes of albumin and gelatine were left for 8 days on a leaf, which was then cut open. The whole surface was bathed with acid secretion, and every cell in the many glands which were examined had its contents aggregated in a beautiful manner into dark or pale purple or colorless globular masses of protoplasm. These underwent incessant slow changes of forms, sometimes separating from one another, and then reuniting exactly as in the cells of Drosera. Boiling water makes the contents of the gland cells white and opaque, but not so purely white and porcelain-like as in the case of Drosera. How living insects, when naturally caught, excite the glands to secrete so quickly as they do I know not, but I suppose that the great pressure to which they are subjected forces a little excretion from either extremity of their bodies, and we have seen that an extremely small amount of nitrogenous matter is sufficient to excite the glands. Before passing on to the subject of digestion, I may state that I endeavored to discover with no success the functions of the minute octafid processes with which the leaves are studied, from facts hereafter to be given in the chapters on Aldrovanda and Utricularia. It seemed probable that they served to absorb decayed matter left by the captured insects, but their position on the backs of the leaves and on the footstalks rendered this almost impossible. Nevertheless, leaves were immersed in a solution of 1 part of urea to 437 of water, and after 24 HRS, the orange layer of protoplasm within the arms of these processes did not appear more aggregated than in other speci. Page 301. Mens kept in water. I then tried suspending a leaf in a bottle over an excessively putrid infusion of raw meat to see whether they absorbed the vapor, but their contents were not affected. Digestive power of the secretion. When a leaf closes over any object, it may be said to form itself into a temporary stomach, and if the object yields ever so little animal matter, this serves to use Skiff's expression as a peptogene, and the glands on the surface pour forth their acid secretion, which acts like the gastric juice of animals. As so many experiments were tried on the digestive power of Drosera, only a few were made with Dionea, but they were amply sufficient to prove that it digests. This plant, moreover, is not so well fitted as Drosera for observation. As the process goes on within the closed lobes, insects, even beetles, after being subjected to the secretion for several days, are surprisingly softened, though their chitinous coats are not corroded. Experiment 1. A cube of albumin of 110 of an inch 2 Roman 2000 was placed at one end of a leaf, and at the other end an oblong piece of gelatine 15 of an inch 5 Roman 2000 long, and Dr. W. Roman 1000 Canby of Wilmington, to whom I am much indebted for information regarding Dionea in its native home, has published in the Gardener's Monthly, Philadelphia, August 1868, some interesting observations. He ascertained that the secretion digests animal matter such as the contents of insects, bits of meat, Roman 100, and that the secretion is reabsorbed. He was also well aware that the lobes remain closed for a much longer time when in contact with animal matter than when made to shut by a mere touch or over objects not yielding soluble nutriment, and that in these latter cases the glands do not secrete. The Rev. Dr. Curtis first observed, Boston Journal Nat Hist. Vol. Roman 1 p. 123, the secretion from the glands. I may here add that a gardener, Mr. Knight, is said Kirby, and Spencer's Introduction to Entomology 1818 Vol. Roman 1 p. 295, to have found that a plant of the Dionea, on the leaves of which he laid fine filaments of raw beef, was much more luxuriant in its growth than others not so treated. Page 302. 110 Broad. The leaf was then made to close. It was cut open. After 45 HRS, the albumin was hard and compressed with its angles only a little rounded. The gelatine was corroded into an oval form, and both were bathed in so much acid secretion that it dropped off the leaf. The digestive process apparently is rather slower than in Drosera, and this agrees with the length of time during which the leaves remain closed over digestible objects. Experiment 2. A bit of albumin 110 of an inch square, but only 120 in thickness, and a piece of gelatine of the same size as before were placed on a leaf, which 8 days afterwards was cut open. The surface was bathed with slightly adhesive, very acid secretion, and the glands were all in an aggregated condition. Not a vestige of the albumin or gelatine was left. Similarly sized pieces were placed at the same time on wet moss on the same pot, so that they were subjected to nearly similar conditions. After 8 days, these were brown decayed and matted with fibers of mold, but had not disappeared. Experiment 3. A piece of albumin 320 of an inch 3 Roman 2000 long and 120 broad and thick, and a piece of gelatine of the same size as before were placed on another leaf, which was cut open. After 7 days, not a vestige of either substance was left, and only a moderate amount of secretion on the surface. Experiment 4. Pieces of albumin and gelatine of the same size as in the last experiment were placed on a leaf, which spontaneously opened after 12 days, and here again, not a vestige of either was left, and only a little secretion at one end of the midrib. Experiment 5. Pieces of albumin and gelatine of the same size were placed on another leaf, which after 12 days was still firmly closed, but had begun to wither. It was cut open and contained nothing except a vestige of brown matter where the albumin had lain. Experiment 6. A cube of albumin of 110 of an inch and a piece of gelatine of the same size as before were placed on a leaf, which opened spontaneously after 13 days. The albumin, which was twice as thick as in the latter experiments, was too large for the glands in contact with it were injured, and were dropping off. A film also of albumin of a brown color matted with mold was left. All the gelatine was absorbed, and there was only a little acid secretion left on the midrib. Experiment 7. A bit of half-roasted meat not measured and a bit of gelatine were placed on the two ends of a leaf, which page 303 opened spontaneously after 11 days. A vestige of the meat was left, and the surface of the leaf was here blackened. The gelatine had all disappeared. Experiment 8. A bit of half-roasted meat not measured was placed on a leaf, which was forcibly kept open by a clip, so that it was moistened with the secretion very acid only on its lower surface. Nevertheless, after only 2212 HRS, it was surprisingly softened when compared with another bit of the same meat, which had been kept damp. Experiment 9. A cube of 110 of an inch of very compact roasted beef was placed on a leaf, which opened spontaneously after 12 days. So much feebly acid secretion was left on the leaf that it trickled off. The meat was completely disintegrated, but not all dissolved. There was no mold. The little mass was placed under the microscope. Some of the fibrillae in the middle still exhibited transverse striae, others showed not a vestige of striae, and every gradation could be traced between these two states. Globules apparently of fat and some undigested fibroelastic tissue remained. The meat was thus in the same state as that formerly described, which was half digested by Drosera. Here again, as in the case of albumin, the digestive process seems slower than in Drosera. At the opposite end of the same leaf, a firmly compressed pellet of bread had been placed. This was completely disintegrated, I suppose owing to the digestion of the gluten, but seemed very little reduced in bulk. Experiment 10. A cube of 120 of an inch of cheese and another of albumin were placed at opposite ends of the same leaf. After 9 days, the lobes opened spontaneously a little at the end enclosing the cheese, but hardly any or none was dissolved, though it was softened and surrounded by secretion. 2 days subsequently, the end, with the albumin, also opened spontaneously. Roman 1e 11 days, after it was put on a mere trace, in a blackened and dry condition being left. Experiment 11. The same experiment with cheese and albumin, repeated on another, and rather torpid leaf. The lobes at the end with the cheese, after an interval of 6 days, opened spontaneously. A little. The cube of cheese was much softened, but not dissolved, and but little if at all reduced in size. 12 hours afterwards, the end, with the albumin, opened, which now consisted of a large drop of transparent, not acid, viscid fluid.